Good morning, everyone. We are starting Lake 2022 schools presentations.
You can start. Okay. Good morning to honorable present here. We are from Baptist School Biddi, 8th standard. Myself Ajit, he is Pranav and she is Darshini. We are here to present a topic of production of biopesticides for environmental friendly disease management. As you know, the chemical pesticides pose many long-term threats and risks for living beings on this planet. If you use more chemical pesticides, it can harm our environment, and there will be no growth of plants, and we will not get food, air. And if there are no plants, we won't get even water. So the three main important terms for our survival only will be not there. That's why we are here with the to reduce the amount of chemicals used in the farming. We are here with the biopesticides, which are naturally occurring compounds or agents that are obtained from animals and plants.
We can increase the agricultural productivity in two ways, such as by increasing the crop yield and by providing the manure and by organic based treatments such as by including this biopesticides. These biopesticides are potentially alternatives to synthetic pesticides. Instead of using the synthetic pesticides, you can use these biopesticides.
They are eco-friendly and low specific. That means it doesn't harm the environment and they are friendly to the non-target species. Using of biopesticides, they pose less threat to the environment and human health, and they are less toxic than chemical pesticides.
Sources of biopesticides are easily available, easily biodegradable, exhibit various modes of action. They are less expensive, and they have low toxicity to humans and non-target species. It means that the materials used for our biopesticides can be derived directly from the nature, and anyhow they are biodegradable, so they can be easily biodegradable because they are from the nature, and they have various modes of actions on the pests which harm the plants, and as they are derived from nature, there is no need of no need of buying them or using money for them, and they are less toxic than the chemical pesticide, and they do not harm humans and the non-target species. Botanical pesticides or plant-made pesticides, they are known to repel insects and they suppress oviposition and feeding.
Plants contain some active components that includes phenols, quinols, alkaloids, sterols, terpenes, alcohol, and tannins. These are responsible for the biocidal properties. We can extract the biopesticide on two ways, either by the plant extract or extracting the essential oils. Dried plant parts are preferred as this results in higher yield of active ingredients. That means when our water concentration decreases, the phytochemical concentration increases. That results in higher yield of active ingredients.
Our objectives of the study to produce, to formulate, and to commercialize these biopesticides. To develop the protocol for the production of biopesticides using native plant species. These native plant species are plant species which can be available easily and found in a particular area for a long time. To demonstrate the use of biopesticides and to educate the farmers about these biopesticides, to create awareness among them on biopesticides use and in eco-friendly disease management. And finally, to bring about sustainable practices in the school.
We have chosen the school Wabila School Biddi as a study area. Its coordinates, latitude, and longitude are mentioned in the slide respectively. These are the materials and methods we gone through this process. First, biopesticides were prepared mixing 6 natural ingredients of plants as well as 1 animal product. First, we collected the fresh leaves of neem, guava, custard apple, and papaya. Then these leaves were mixed gently with the distilled water. These leaves were shaved well. With the help of blender, these leaves were powdered. We passed this powder to a sieve to get a uniform size. Next, we crushed 300 grams of neem leaves in 100 ml of carburin. We added a paste of ginger and garlic to the extract. Again, we crushed 200 grams of custard apple leaves, papaya leaf, and guava leaves in water. Then we mixed the both and boiled it for 5 times with some interval till it get half. Then we kept it for 24 hours to get ferment. Then this can be stored in water for 6 months, as we have did now.
The species which we have used in this biopesticides are named in this list. The first is guava leaf. It has the it includes it includes saponins and tannins which have the property of antimicrobial, and it protects the plants from insects. The next is neem leaf. It consists of triterpenoids and steroids which have the property that induces sterility in insects and acts as a repellent.
Next is custard apple leaf, which contains acetogenins and which is proven to be toxic to insects. Next, the ginger, which contains monoterpenoids and sesquiterpenoids, which shows the antibacterial and antifungal property.
Next is garlic. It contains diallyl disulfide, which is a good repellent of insects. Papaya leaves. It contains alkaloids, papain, cyanogenic glycosides, which acts against mosquitoes, caterpillars, and worms. Next, carburin. It contains urea, and it is a natural insecticide, and it's proven that it kills varieties of germs.
To check the presence of the active ingredients in the leaf extract, we did the phytochemical screening. The first one is to check the presence of glycosides. First, we took a mixture containing glacial acetic acid of 2 ml, and we added 2 drops of perchloride. After that, we added 2 ml of concentrated sulfuric acid and with the extract of the papaya leaves. Then we saw a brown ring at the interface. It proves the presence of glycosides.
To check the presence of saponins in the guava leaf extract, we took the guava leaf extract that was diluted with distilled water and was shaken. What we observed was a foam formation, which shows there's the presence of saponins in the guava leaf extract.
Next, to check the presence of tannins in the guava leaf extract, first we took 1 ml of guava leaf extract plus 2 ml of perchloride. Then we observed that the formation of blue, green, and black color, which shows the presence of tannins in guava leaf extract.
Here are the few pics we took while doing the process. First slide contains the collection of leaves. Next, rinsing of leaves. Here we have soaked the leaves inside the water to get cleaned. These are the pictures we took after the complete dried of leaves. Grinding of leaves and extraction with carburin and water. Extraction process. Application of biopesticide in the crop plants of our school garden. Phytochemical screening.
These are the results and discussion of the 4th and 7th day after the application of biopesticides respectively on the watermelon plants. They are found to be effective against sucking pests, pod fruit bearers, and aphids.
These are the result on the latest fingerprint on the 4th and 7th day after the application of the biopesticides respectively. They are found to be effective against the sucking pest, pod, fruit bearers, and aphids.
The indigenously prepared biopesticides are environmentally sound, natural friendly, and economical. It means that they are easily biodegradable and do not harm the environment, and then they can be made at less cost. They can be afforded at less cost. They are eco-friendly since they are easily biodegradable and they therefore do not pollute the environment.
They are friendly to the non-target species and they don't harm any of the useful species. Biopesticides are proven to cause little or no pollution and they are relatively cheaper.
They are usually inherently less toxic than these conventional pesticides. Conventional pesticides are more toxic than these biopesticides. They enhance the crop quality and shelf life of the crop.
Further studies need to be done on the application of these biopesticides to check the effects of the biopesticides on the plants, and then even we have to check the application of biopesticides on the various stages of the plant.
I would like to conclude it by saying they are effective in small quantities and their use promotes sustainable pest management and hence contributes towards a sustainable agriculture. And as many challenges are occurring, still more biopesticides biopesticide is still remaining as a very good suitable alternative to the conventional pesticides.
These are the references which we have gone through while conducting this study.
Thank you.
Very good. You did a good job. Start in the morning. Need a little more energy to do it as a first person. Good. I have one question for you. Theoretically, yes, you have managed it very well. Did you try it on any of the plants and trees that are there in your school campus?
We tried on the lady's finger and watermelon plant.
What was the infection on the leaves or on the fruit?
Sir, there were many pests. Sir, the gardener said us that there were many pests attacking on the plants. That's why we had made the biopesticide and it was very successful.
Good, good. So what was the pest like? What was the problem? Did you identify that? No?
Sir,
You depended on the
we should.
gardener.
Yes, sir.
Any questions from the students? Anyone can ask a question. One question I can give. No one? I will tell you the rules. You get 8 minutes for a presentation. In 7 minutes, I will give you the first bill. In 8 minutes, I will give the final bill. Okay? In 8 minutes, you have to finish your presentation. And after you finish your presentation, you have to come to this desk and do your registration for today. Okay? Thank you. Next, Vidyanikedhan School, Spurti and Sushmita.
Next slide. This is the pointer. Okay. It is easy, simple. It is simple. Wait, I'll just show you.
Good morning, everyone. My name is Sushmita and I'm Smurti. We are from Vidyaniketan School and we're studying in 8th grade. So today we're going to present our study on Ulla Lake and methods for conservation. So Ulla Lake is an urban water body and this lake is an integral part of the ecosystem and it has uses for many recreational activities and environmental attributes. So pollution affects water qualities of lakes and other freshwater resources around the globe. And this lake pollution has affected fishermen, small dairy farmers, and has deprived the economically depressed classes of the society. Not a single lake in Bangalore is fit to be a drinking water source. So our objectives are to study Ulla Lake based on the chemical, physical and biological parameters and to propose low cost, effective and natural methods to purify the lake. Our study area is Ulla Lake, which is situated in the southwestern part of Bangalore city. It falls in the Vrishbhavati Valley within the Bairamangala Lake series and it covers an area of 10 hectares and the perimeter is approximately 1.70 kilometers. So these are our comparative studies. In 2008, the area of the lake was 11.03 hectares and in the year 2016, the area of the lake was 9.833 hectares. So we can see that the lake area has been reducing. So these are the three different sites from which we collected the water samples. Our materials and methods. So the materials we used were total hardness testing kit, alkalinity testing kit, dissolved oxygen testing kit, pH papers, and universal indicators. We collected water from 3 different sample sites and we tested pH values and recorded the physical parameters. We proceeded with the testing of biological parameters using compound microscopes and we identified the different microbes, and then we proceeded with the testing of chemical parameters. So these are our results. In site 1, the color of the water was clear, the temperature was 24 degrees Celsius, and the water was transparent. In site 2, the color of the water was a light sheen green, and the temperature was 23 degrees Celsius, and the water was transparent. In site 3, the color of the water was light brown, it was 25 degrees Celsius, and the water was translucent. Chemical parameters results. So the in site 1, the water sample was 7 pH, indicating that it was neutral. The hardness was 150 parts per million. The alkalinity was 200 ppm, and the dissolved oxygen was 8 milligrams per liter. In site 2, the pH was 9, indicating that the water was basic. The hardness was 200 ppm. The alkalinity was 280 ppm, and the dissolved oxygen was 4 milligrams per liter. And the pH of site 3 was 9, indicating that the water was once again basic. The hardness was 150 ppm, the alkalinity was 225 ppm, and the dissolved oxygen was 2 milligrams per liter. These are the microbes we found. So we found different microbes such as diatoms, chlorococcus, cytomonas, and spirogyra. These are the different plants we observed. And the pictures of us collecting samples and going through with the processes.
Issues faced by the lake. The water spread area of the lake is reducing despite the rains. Storm drains have been blocked because of the development of new roads in residential areas around the lake. Water is being contaminated with sewage water, and there is also an unpleasant odor due to which organisms living inside the lake are being affected. Methods for water purification. Implementation of floating treatment wetlands. Bioremediation can also help reduce the eutrophication which is observed in the lake. And in order to do all of this, we should also urge the local government to recognize these anthropogenic activities and help conserve or take required measures to conserve the lake. These are the reference links we use. Acknowledgement. We are really privileged to be a part of the Lake Symposium 2022 and 23. And we sincerely thank IAC for giving us such a platform. And we also extend our sincere thanks to Vidyanikethan Public School and our respected teachers who guided us throughout this project. Thank you.
You have selected three points to focus on this, right? Any criteria we used that one city, one local, whose answer says that none of the lakes water from that can be usable for drinking and all. There is one lake which TBR has mentioned. For the past 6 years, around the people are using without any purifications. You find out which one. Okay. That I will not tell you. Tell me the criteria of the sample collection. Why you chose that points?
Sure. Let me go to that slide. Ma'am, first of all, the lake which doesn't exactly have an inlet. This lake doesn't have an exact inlet. This is the main part of the lake. This was the picture from the main part of the lake. Here is where the inlet is located, but we didn't take this part of the lake for the sample distance because it is no longer used. But this is the main part of the lake, so we thought we could take different directions. We couldn't actually collect the samples from the lake, but we took samples from different parts of the lake.
Any questions from the students?
My question about the conservation of the lake. What exactly is floating treatment? Floating treatment is where small artificial platforms are created on which plants such as hibiscus, ashwagandha, or cactus are grown. These plants help in breaking
These are they help reduce the biomass present in the water, which in turn reduces the eutrophication. The breakdown of
Next, Jackie Kaur and Sujay Kaur from Bagaji Villas Whitefield.
Presenters will.
A pleasant afternoon to everyone present here. For the past A pleasant morning to everyone present here. For the past few weeks, we were on a research spree on the enormous explorable topic soil. This presentation is presented to you by myself, Suchet Bhatt.
and Japlin Kaur.
We are from grade 8.
Soil is an essential part of life. But from the past few years, we have been harming the soil with poor management, excessive agriculture, and deforestation. Hence, we chose the topic soil to discover a convincing solution for it. Our project is the study of five main parameters: moisture content, nutrient content, soil pH, soil color, and water holding capacity. Our objective is to recommend solutions on the quality of soil.
For the research to run, the main thing essential was soil. So, to get soil, we went to four different places. The first two soil samples, the eggplant soil sample and the rose plant soil sample, were taken from the regions of Kannamangalam. The school soil was taken from the campus of Montevi Villa School, Whitefield, while the urban soil was taken from my apartment, which comes under the region of Woking. The coordinates of the same are shown to you in this slide. As you can see in this slide, here we are collecting the soil samples from all the four places. There were several materials necessary for our project. We mainly use soil from different places, distilled water, microscope, beakers, and much more. We also followed various methods and protocols. For moisture content, we took the difference of the dry and wet weights of the soil and concluded. For soil pH, we made solutions of the soil samples and tested them with pH strips. For soil color, we analyzed the soil thoroughly and concluded about the color. For water holding capacity, we weighed 50 grams of the soil and added water to it until the water started to drip. We measured the amount of water added and concluded. For the nutrient test, we followed the protocols of the soil testing kit and concluded. For microorganisms, we viewed the soil samples thoroughly under a microscope to know about the microorganisms.
As you can see, these are the results of the various parameters we worked on. The moisture content is lowest in school soil. The water holding capacity is lowest in rose soil. The moisture content and water holding capacity is highest in the eggplant soil since it is an agricultural soil. The soil pH of urban school and rose soil is 6, while the soil pH of eggplant soil is 7. The soil color of urban soil is dark brown, while the soil color of the rest three is brown. We also found a microorganism named Bacillus present in the eggplant soil, which is a rod-shaped bacteria. These are some of the other beneficial microorganisms. Rhizobium converts atmospheric nitrogen to water-soluble nitrogen. Nitrobacter and nitrosorbitol
Azotobacter convert atmospheric nitrogen to ammonia. We also research on the nutrient content of the soil. The macronutrients we work on are nitrogen, phosphorus, and potassium. The micronutrients we work on are iron and fluoride. These are the test results of the nutrient content of soil. L represents low, M represents medium, and H represents high. The nitrogen content was highest in eggplant soil. Phosphorus and potassium content was highest in rose and eggplant soil. Iron was highest in rose soil and fluoride was absent in all the four samples. These are the NPK content tests of the eggplant and rose plant soil. These are some of the other observations of the soil color and soil pH of the four samples. On the right hand side of the slide, you can see the iron test, which shows that rose has the highest amount of iron, followed by eggplant, and then the rest.
As we have seen in the previous slide, agricultural soil has the highest moisture content, water holding capacity, and nutrient content as it has the presence of microorganisms and manure. The other soil samples have comparatively less moisture content due to lack of rainfall, manure, and microbes. Also, we have been checking the soil regularly to get accurate results. As we have seen in the previous slide, the nitrogen content of the soil was low. If this decreases further, the plant might get chlorosis or loss of green color due to lack of chlorophyll in the plant. Similarly, the potassium and phosphorus deficiency might decrease the overall production of yield.
After a lot of research, we found out that the soil lacked in microorganisms. And for the microorganism to grow, organic matter should be added in the soil. Constant agriculture and routine check of different parameters of soil is one of the main factors that keeps the soil healthy and should be done regularly. For the moisture content to increase, proper irrigation should be done in the fields. The soil lacks in nitrogen, phosphorus, and potassium, so we recommend NPK fertilizers to be added. As an alternative, alfalfa pellets and worm casting method can also be used. The last 3 points of the slide are the other methods that can enrich the soil with NPK.
Based on the following results, we can conclude that the depletion in soil nutrients of the school soil and urban soil is due to the absence of microorganisms such as rhizobium, which helps to convert the atmospheric nitrogen to water-soluble nitrogen and helps the plant to replenish in nitrogen content. Hence, the conservation of soil is very important in order to produce enough food to reach the needs of the people. Thus, the measures that can be taken include adding manures and nutrient-rich fertilizers to the soil so that the soil fertility and ecological balance can be restored for future generations.
We sincerely thank the IISC team for giving us a glorious opportunity to present our research, thus helping us to grow our capacity of bestowing to the people of the society. We also thank Team BBI for providing us the space and materials necessary for the welfare of our project.
As you can see, these are the websites that we have cited for our research.
Thank you.
When you told that you have NPK in the agricultural field, that is your result showing, right? And in the soil, all nutrients are more in the agricultural soil. Have you made sure that the agricultural land is not given the fertilizer or manure you have already added? Have you tested that?
My question is, agricultural soil, we will be, when you have the agricultural land, you will add manure, nutrients will be added, right? And in the school or urban soil, wherever you have tested, you are not doing anything in that land, right? So have you made sure that the agricultural land, like in 1 week or in 1 month, how many times it is manured or something like that?
No, no, we have not checked that. But since the plant was young and it had not started bearing vegetables, we think that they have not added fertilizers in that stage.
Question from the students.
Good presentation. I just want to check. Somewhere you mentioned about nitrogen. In one soil, nitrogen is less. You said nitrogen and chlorosis you connected it. I just wanted to know.
Yes, ma'am. Nitrogen is the main component of chlorophyll. So
Please, not like it's magnesium. Just check. Not nitrogen.
The ignition is at the center. Yeah, the ignition is at, if it is yellowing the leaf, one piece is magnesium. Nitrogen, how they have connected the nitrogen, maybe chlorophyll is also a protein molecule, chlorophyll pigment. But then actually when you are correlating, it should be magnesium.
thank you.
Next presentation, Vasudevi Vilas, Madhavi, Shruti, Varshini, and Meenakshi.
Mic is on. This is the point if you want to point out.
Good morning, Soman and all present here. I'm Varshini. I'm Shruti. I'm Meenakshi. We are from Vasudevi Vilas Institution, Smart Delhi. The immense pleasure presenting today in front of you all. We all know a lake is a large area of water surrounded by land. Lakes provide us many ecosystems such as fish and aquatic habitats and fresh water. Lakes replenish the groundwater and also help in preserving the biodiversity of the area by providing the necessary habitats. Nowadays, lakes are being depleted and are being polluted at an alarming rate. The various chemicals, factory effluents, and other contaminants released into the water make it almost impossible for the aquatic plants and animals to survive in the lake. To understand this better, we have taken up the history of Kudala Lake. We discuss about its water further in detail. Kundarahatti Lake is located in Bangalore, Karnataka, and is spread around 10 hectares. It used to be a sewage dumping site for the surrounding areas. It is surrounded by big parks, multi-story buildings, and slums. Objectives: to study the quality of water, to check for the presence of microorganisms, to check for the presence of chemicals, and to find ways to purify the lake water to preserve natural habitats and freshwater sources. Study area. This is the satellite view and this is the aerial view of Kundalahalli Lake. The perimeter of Kundalahalli Lake is around 1,500 meters. Its area is around 17 acres and its longitudes and latitudes are shown here. This is a map showing the various inlets and outlets in Kundalahalli Lake. Kundalahalli Lake has 3 main inlets and 1 outlet. There are also 2 STPs installed at different points of the lake. Microorganisms play a vital role in decomposing and breaking down organic matter. However, if conditions are favorable, some of these can reproduce at a very fast rate and they can also promote the growth of harmful algal bloom. When we observed about 1 ml of the water sample from the lake under a microscope, we were able to find the presence of various moving organisms and also other contaminants. This is a table showing the various microorganisms found in the lake. The first one was a type of algae, which is Chlorococcum angustomatis. The number was 11 in the lake and in the sample, and these are single-cell green algae. They are found only in freshwater ponds. They have high amounts of omega-3 polyunsaturated fatty acids. These polyunsaturated fatty acids or PUFAs can harm humans by causing nausea, diarrhea, and heartburn. Next is Cyanidra diatom, which is another type of algae. They were found 6 in number. These are diatoms which have long needle-like cells that exist in single or they radiate in colonies. We were able to find them radiating in colonies. These algae, when conditions are favorable, they become too thick and they also promote the growth of harmful algal bacteria. Algal bloom, sorry. Barnacle larvae, these are type of zooplankton. The number was 2. These attach themselves to a symbiont such as a whale, sea snake, or a crustacean. These can harm the symbiont they're attached to by producing excessive cover. They're also very sharp and they have cuts in them. These can get easily affected and this also harms the symbiont they're attached to. On-site tests. These are the various on-site tests that we have performed and we have listed the lake. These include the pH, temperature, and TDS. These are the various off-site parameters which we have tested in our school R&D lab with the assistance of our teacher. The various tests include alkalinity, hardness, fluoride, fluorine, and iron. The various chemicals or reagents used are mentioned below. We have followed the integrated sampling method and we have collected 2 samples from different points of the lake simultaneously. First sample was collected from the middle point of the lake, while the second sample was collected from the outlet of the lake. These are the results of the various on-site and off-site parameters tests that we have performed. There is also a column showing the BIS standards. We can see here that most of the tests do not match with that of the BIS standards and are higher. Few initiatives taken by the government are in 2016, the BBMP and the local residents had dry-dredged the lake to remove the buildup of sewage from the lake bed. And during the revival of lake, over 1,000 tons of sludge was removed from the lake. And STPs were installed at various points of the lake, and one of its chamber runs entirely on gravity. BBMP has also fenced the lake's perimeter to prevent further dumping of waste into the water. Major problems faced in Kundalahalli Lake. Many living nearby the lake are facing many health hazards due to the excessive dumping of waste into the lake. Kundalahalli Lake used to be a habitat for various birds, but the pollution has put their habitat in danger. During heavy rains, polluted rainwater seeps through the garbage and flow into the lake, making it toxic and threatening aquatic life. Few methods to naturally increase the quality of the water are bioremediation, which is a biotechnical process used to clean up contamination. Here, microorganisms are used. Nualia technology, which is an alternative to algaecide, helps to naturally balance algae and planktons. Plankton net can be used to control excessive phytoplanktons. Artificial intelligence can be used to monitor lakes, and we can provide better security and protection to preserve the wildlife and the aquatic life. Few steps we are taking to bring a change. We have conducted a green assembly and have made posters to make students aware of pollution. We showed a presentation to our apartment manager and asked how the water is being treated in our complex. He informed us that we have a fully functional STP as per government guidelines and water is being treated before going to the sewage. Also, a part of the treated water is used for garden maintenance. We are planning to present a street theatre, also known as Nukkad Natak in the local language, near the lake premises and our school to bring awareness. In conclusion, we can see that the lake water is polluted with various pollutants and effluents due to human activities. Various chemicals and living organisms can be seen in the water sample. The restoration of the lake as a natural habitat can natural and sustainable methods. Lakes are our breathing spaces and our lifeline, so it's our duty to protect, preserve, and refrain from polluting them. These are the various references that we have used to complete this project. We are thankful IAC for giving us this wonderful opportunity. We would also like to thank our school, teachers, parents, and our friends for encouraging, supporting, and motivating us throughout this project. Thank you for giving us this opportunity.
You mentioned artificial intelligence. Can you please elaborate?
Ma'am, IoTs are used in few lakes in Chennai. It is used for monitoring the quality of the water and to take the
Any questions from the students? You mentioned that there are various birds in the lake. Do you have any information of what birds you found there earlier?
Actually, the birds used to be there maybe around 2015 or as such, but when we visited the lake, we were not able to find any. So that's why we had mentioned it as a problem.
Okay. Do you know what birds used to be there?
We weren't able to find it. We didn't have information.
Yeah, references you have mentioned Wikipedia, right? So we have been instructed that first one Wikipedia is incorrect and not the actual reference. So just wanted to point that out.
We will change that.
Next is Radhika Thakur from Bangladesh Jila Gurdu.
Wait.
So, good morning, everybody. I'm Radhika Thakur from grade 8 Bangladesh School, Gurdu, and today I'm here to present my project on birds. So, birds. Birds are warm-blooded vertebrates that belong to the Aves class of the phylum Chordata, and they have a four-chambered heart and heart-shaped eggs, and they have a strong yet lightweight skeleton. So, what is the ecological importance of the birds? So, birds have a great ecological importance as they help in the population control of the insects and rodents, and also they help in the plant pollination and seed dispersal. So birds are the indicators of the nature. If they are in danger, we'll soon know that we too will be in danger. The objectives of my study is to identify the various birds found in my confined geographical area and to study about its various behaviors and characteristics. So this is the study area which includes the Silent City layout, the Madanahari village, and the Kurkuru village. The methodology used are as follows. The duration was for 4 months from July 2022 to November 2022, and the guidance are mentioned. The materials used were binoculars and photography. So these are the various species of birds that are found in my confined geographical area. It includes the river tern, the Achimedia, the Indian sunbird, the cattle egret, jungle myna and the black kite, Asian coil and pied bush shark, the green bee eater, the white-throated kingfisher, Thunderbird and red-whiskered bulbul, the jungle babbler and the Indian roller, which is also called a reef kantha in the common language, the greater cockle and the rock pigeon, and the jungle rat. So these are the results of my study, which includes the scientific name of the birds, the family which it belongs to, the IUCN status, and its feeding habits. These are the results of the rest of the birds. So data analysis. Now, according to the feeding habits, the birds are classified into 4 categories of carnivorous, omnivorous, herbivorous, insectivorous. So these are the insectivorous birds, the omnivorous, the carnivorous, and the nectarivorous. These are the observations of the birds made. So the Asian bee eater is found near the beehives as its feeding habit is the flying insects. The Indian sunbird, the pied bustard, and Ashlepedia, which are parasite birds, are extremely active due to the small size. And these are the rest of the observations. As you can, the rock pigeon has a special characteristic of bobbing its head when it moves forward and backward. Acknowledgements. I would like to thank ISC for providing us with such a platform to present my hobby in the form of a study. These are the websites with it and the links. Thank you.
Can you come to previous slides?
Can go to
Carnivore slide.
wait, huh? Sadika, wait. Okay.
Omnivore. Previous slide. You have mentioned blue rock pigeons. Are they omnivore? What do they feed mainly on?
Sir, actually, they are generalized omnivorous, but they are prefer eating grains. So they can be classified frugivorous as well, but they are generalized omnivorous.
Okay. You know what is omnivore? Omnivore
Yes,
means
they eat insects as well as they eat non-veg as well.
okay. Have you seen pigeons eating insects?
They're generalized omnivorous.
Okay.
Any question for the students? Okay, move on to the next presentation.
ಇನ್ನ ಕಾಲ್ ಮಾಡಿಲ್ಲ ಏನ್ ಸರ್ ತಿಂಡಿಗೆ ಕಾಲ್ ಮಾಡಿಲ್ಲ ಏನಾದ್ರು
Again now.
Okay. Then from KK English School, Shaurya and Geography.
Can start. You can start.
Very happy morning to one and all present. I am Shaurya and she is Geography. We are studying in 8th standard from KK English High School. We are here to discuss about the non-recycling plastics and its effects on the environment. We all know that now plastics are not biodegradable. There are mainly two types of plastic. Some can be recycled and some cannot be recycled. Whatever it is an inseparable part of our life. We start a day with plastics like toothbrush, but I don't because I started using a wooden brush. We cannot live without plastic. We can only reduce, recycle, and reduce. Now, let's see some disadvantages of using non-recyclable plastics. Non-recyclable plastics, of course, cannot be recycled. Hence, they have to find a safe place only in the landfills. Landfills fill up faster. Toxins produced from the landfills can leach into the soil, water bodies, as well as underground water. Plastics are burned and produce toxins like dioxin. This is uncarcinogenic. It increases the amount of garbage burned into the landfills. This increases pollution. Plastic waste can also block drains. By eating the food plastics kills the animals. The main study of our objective is to understand the effect of non-recyclable plastics used in our house and to find an alternative to it. A survey of 9 and 8 standard students to know whether they buy packet milk or get milk from milkman shows that out of 390 students, 290 students buy milk packets at home. This shows that 290 milk packets per day. We had an interaction with transcribed belows of Vartur village and came to know that they don't take empty milk or oil packets because when they store these, they are eaten up by rats and it is a loss for them. Hence, they have to find a safe place only in the landfills. Again, we wanted to find out the number of students, families who buy chili packets at home, and we came to know that out of 390 students, 310 number of students buy chili packets at home. We thought of comparing the price of whole chili and packet chili powder, and we came to know that whole chili is approximately 300 rupees per kg, but whereas 400 rupees per kg, but whereas whole chili is 300 rupees and 30 rupees for powdering it. So the total cost would be 330 rupees, and you would be saving 70 rupees when you buy whole chili and powdering. Actually, we are not concerned about the 70 rupees that we save. We are concerned about the carcinogenic substances that are added into it and the plastics that cause pollution. Now, let's see some disadvantages of using packet chili powder. A minimum of 4 hours going to the landfills monthly, and they cause pollution. Mainly, other than all these, we came to know that they use adulterants mainly such as carcinogenic. And we also came to know that they use colored sodas. Now, let's see whether we have to buy packet chili powder or whole chili. I request all the students, teachers, and judges now to buy packet chili powder. Meanwhile, Jagrati took a survey of 10th, 9th, and 8th standard students to know whether they buy Santosachi or cotton. And we came to know that out of 390 students, 300 students buy Santosachi at home. This shows that 310 sachets per day. Now let's take a family of 4 members and let's hope that they take weekly 3 times their part. So weekly 12 sachets, monthly 48 sachets, and yearly 500 sachets. And this is only the one family that goes into the landfills. And we also let's see. A company that produces anything in sachets is intelligent because it reduces the cost of production and it increases its profit. A common man who buys Santo sachet is also intelligent, thinking that a bottle would be more costly and more wasted and not economic. After I came to know about the pollution caused by the shampoo, I prepared herbal shampoo at home using Shikanai, Sumplat, and Amla. My family and Jagriti's family are using the same now, and we will promise that we will be continuing to use this shampoo. We have created an awareness program to our school teachers and this list shows that the items that are packed in plastics that we buy from the shops. Example: soap, salt, chili powder, turmeric, coriander powder, Santosachi, etc. After I came into this project, I have adopted some green lifestyles such as I use wooden brush instead of plastic brush, and I get milk only from the milkman. And all groceries only wrapped in paper covers, and I also carry steel containers to get any parcels like pani puri from the hotels. But I'm lavender or turmeric. It is always better to buy unpowdered. Try to avoid, if possible, packed items. Instead, you can buy them loose in paper covers from the shops. I also recommend you to adopt green step. It's not a story of Sambu Sati or chili packet. When we look into the grocery list, it's a price. Everything comes in non-recyclable plastics. And all this goes into the landfills. But government is only worried about the plastic carry bags. Instead, the government should pressurize the companies to use biodegradable plastics even to pack things. Or they should take back the plastic that they produce. And let me remind you, there is one and only one beautiful earth to live in. We thank IISc for giving us this wonderful opportunity, and I thank my teachers and friends who supported me.
Nice, very good presentation. You didn't bring your shampoo, herbal shampoo for us. Or how many days since when you are using the herbal shampoo?
Since 15 years old.
3 days only. How will we make out the difference?
Ma'am, it is better to use because you don't have dandruff in your hair.
One more question I have is, for example, you mentioned the carcinogens you told, in packaged food items. How do you test it? From where you want to know? Okay. Check how to test. Next time you come with the test result. Any question from the students?
When you talk about the milk packets, you've taken figures from each family household one packet. Now you've taken in one slide where you talk about the milk packet. So you've taken like you asked your friends, each of your friends about packets, how many of them buy milk packets and how many of them buy from the milk one, right? In this, have you taken into consideration that one family could have multiple milk packets which can be taken?
ಒಂದೇ ಫ್ಯಾಮಿಲಿಯಲ್ಲಿ ನಾವು ಎರಡು ಪ್ಯಾಕೆಟ್ ತಗೊಂತಾರೆ ಇಲ್ಲ ಒಂದೇ
Have you taken into consideration multiple packets?
ಇಲ್ಲಿ ತಗೊಂಡಿದ್ದೆ ಕನ್ಸಿಡರ್
Yeah, but if you take one packet, the figures would be different. Right? Each household will have different requirements.
You had mentioned in your presentation that minimum 4 packets of chili powder go into the tank. Is it per person, per family, or is it per day or per month? You had mentioned that minimum 4 packets of chili packets per 1 month. We'll move on to the next presentation. Then you the public school, Lower, Eastern, and Eastern.
ಯಾವ್ದು ನಿಂದು ಇದು ಗೊತ್ತಿಲ್ಲ ಆಪರೇಟ್ ಮಾಡಿ ಫ್ರಂಟ್ ಬ್ಯಾಕ್ ಓಕೆನಾ PDF ಏನಾ PDF PPT ಬೇಕಲ್ಲ PPT ಇಲ್ಲ ಮ್ಯಾಮ್ ಕಳ್ಸಿರೋದು ಇದು PPTನೂ ಕಳ್ಸಿದ್ವಿ ಮ್ಯಾಮ್ ನೋಡಿ ಇಲ್ಲಿ ಇದೆಯಾ ಅಂತ ಇಲ್ಲ ಪೆನ್ಡ್ರೈವ್ ಇದೆಯಾ ಪೆನ್ಡ್ರೈವ್ ಮೊಬೈಲ್ ಇದೆ ಕಳಿಸ್ಬೋದು ಬೇಗ ಕಳಿಸು
Lakshika and Hemashri Vidyarthi.
ಆಮೇಲೆ ಸ್ವಲ್ಪ ಲೇಟ್ ಮೂವತ್ತರ ಓಕೆನಾ forward back okay pointer
Good morning to one and all present here. Myself Lakshika with my friend Hemashri from Vidyarthi Primary and High School. We can one and all present you for giving us an opportunity to present our study observation and documentation of more diversity in the West Colony. Moths are among the most diverse organisms on the earth, found in many colors, shapes, and sizes. Moths are important among pollinators, particularly they are active after dark when many other pollinating animals have settled down for night. Adult moths and their caterpillars are important food for a wide variety of wildlife, including other insects, spiders, frogs, toads, birds, and bats. In fact, moths make up a major part of diet of bats. Their antennas are feathery and comb-like structure. Moths fold their wings, penetrate over their body, or wrap them around their body. In fact, there could be more than 12 in India. There could be more than 12,000 species of moth, but we don't know the exact number. So to check the moth diversity in Beavers Colony, we did a team of 9 and create an environment in our area so that moths should visit our place, and by that we can do the observation and documentation of moth diversity in Beavers Colony. This is the 9 study sites in where we done our observation and documentation. First cross 9 degree, 84 minutes, 49 seconds north, 77 degree, 58 minutes, 55 seconds east, Namita H Place House, 13 cross, in 9 cross and 12 cross we have 2 study sites, one in north and another one in south. So 13 cross, 12 degree, 84 minutes, 85 seconds north, 77 degree, 58 minutes, 11 seconds east, Hemashri Place House. This is the map of our study area, which belongs to Beavers Colony, Bangalore South, Kodikere Coast, Bangalore, Bangalore 56008. Selected study area is around 2 kilometers square. A light cloth was arranged using a white dhoti. The white cloth of 3 feet into 6 feet, and the top edges were tied. A light was hanging in the middle of the dhoti. A light was kept on throughout the night. Observation was done between morning 5 a.m. to 6:30 a.m. and night 9 p.m. to 10:30 p.m. in the month of July to November 2022, identifying the moths through visual method and also with the help of iNaturalist app, moths of Bangalore 1, and also taking pictures to smartphones provided by our parents. This is the 9 study sites with 9 members who created environment in their houses for moths. We documented totally 37 species of moths belonging to 9 families. The area was November 25. Moth species documented monthly in various study sites. This is a checklist of moth species which we documented in our live study sites. Beak box, Olaria recurvalis, which belongs to Crambidia family. We found this moth in 5 months also. Moss footman, Napitama consolata, it belongs to Erididia family. We found this moth in 5 months. Oleander hawk moth, Franklin snary, it belongs to Springidia family. We found this moth in 4 months. Identified moth families. Bombycidia and Tortucidia family was represented by only one species of moth, and Erididia family was largely represented by 13 species of moth. This is the moth pictures which we observed and documented in our 9 study sites. Moss footman, which is the common moth in our study area. Spotted tiger moth, Oleander hawk moth. Beak moth, it is also one of the common moth in our study area. With this study, we could identify 37 species in the period of 5 months from July to November 2022. At 12th cross north, we found 25 moth species, and in 5th cross, we could able to find 23 moth species in our live tracks. At 7th cross and in 1st cross, we found 9 number of moth species. Among them, moss footman, Tabita consolata 94 in number, and Beakerbo, Stularia recurvalis 51 was the common moth in our study area. Private hawk moth, Spinix registry and bucksilk moth, Bombax mori, were seen only twice in our study area. By creating the environment of having plants near our study place, it is understood that these species of moths are around our area, and our study area has scope for further study also. We thank our head teacher for allowing us to conduct this study. We thank all the 9 members who have created the environment of having plants in their houses for observation and documentation of moths. We thank Energy and Wetland Research Group, Center for Ecology Science, Indian Institute of Science, Bangalore, and Sri Virjulal of CE and Sri Chandra for guiding us. Friends, Moths of Bangalore 1 by Environmental Management and Policy Research Institute, Arishbhat et al. 2015, a brochure of Eyes on Nature, published by Karnataka State Council for Science and Technology. I will actually start Moths of India. For scientific needs, we refer Wikipedia. Thank you.
You said continuum atmosphere by having plants. It gives more sleeping.
ಹಿಂಗಿದೆ ಮಾಡ್ತೀನಿ ಅಲ್ಲ ಚೆಕ್ ಮಾಡಿ ಒಂದ್ಸತಿ ನಡೀತಿದೆ
Why did you find Arabian family for what's from that family?
Because I did most because it belongs to India family. Most women needs family. So it is very well understood to see planet because it Thank you.
Okay, we will wait for snacks. So now the time is 10. So we will reassemble by 10. This one temple.
Students who have not submitted still those are not given your poster PDF and PPT file.
Can you check it? It is PDF. No, this one. Dhruva, right? This is yours, na?
Yes, yes.
Now you copy it.
No, not.
It is there in the pen drive, na?
Yes, yes.
All these to be copied?
No, no, no.
Only one.
Because
they had
by
sent.
miss, yes.
ಇಲ್ಲ ಸರ್
ವಿದ್ಯಾನಿಕೇತನ
ಇದೆಯಲ್ಲ
ಓಪನ್ ನೋಡಿದ್ವಿ ಓಕೆ
ಡಿಲೀಟ್ ಮಾಡ್ಲಾ
ಹಾ
Focus is towards students. If you keep like this, white
Okay.
board is coming.
Okay.
Focus towards. Which one? Where you have sent? Which email you have sent?
Which email?
But then you have to ask them to. Please give it.
Fine. You have a pen drive, na? Okay.
Yes, madam. Yeah, you can just give it. You have the pen drive, or you should.
Mail it. You can mail it, right? I'll tell the email ID. Sir, you have pen drive ಕಳ್ಸಿದ್ದಾರೆ ಇವರು.
Okay. You can. No, or else you can just send it to us. Now it is easy then he has to communicate to us. Then it will take time. No, ಇಲ್ಲಿ ಅಂತ.
Okay. School presidency, right? Okay. Which one? Which one?
Second one.
Second one, nah? Okay. This one is there, nah? You know, top right, right? Front, back, that's it. No much. This one is front, this is back, this is the pointer. Okay. Wait. Okay. Hello, guys. See here, the video has been recording here. Okay. So I think you need to face kind of this way and present. Okay. It's fine. Okay, can go on.
Good morning to all and all present here. My name is Jahan Rao and he is Pradeep. We are from grade 9 Presidency School, Bangalore South. Lakes are an integral part of Bangalore's history and our heritage. Through the study of macrophytes, we can assess the overall health of both the lakes.
For this study, we picked two of the large and reasonably well-kept lakes in South Bangalore, Kotigiri Lake and Begur Lake.
So what are macrophytes? Macrophytes are basically plants that have structures usually more complex, interdependent, and physically substantial.
They are of three types: emergent, submerged, or floating.
Macrophytes as bioindicators. Bioindicators include biological processes, species, or communities and are used to assess the quality of the environment and how it changes over time.
Microphytes respond to these changes and therefore are good bioindicators.
So the objectives of our project. First, to observe plant density by visiting and taking pictures at Kotigiri Lake and Begur Lake.
Identify the various plant species and record their frequency.
Record the area covered by each macrophyte in both the lakes.
Provide a comparison study of the vegetation present in both the lakes.
The materials used for this project were GPS, measuring tape, camera, Google Earth, Google Lens, and the site plantnet.org.
The methods used for this included photography of macrophytes, identification of leaves, and identification through barks.
This is study area 1, which is Kotigiri Lake. It's a prominent lake in South Bangalore, and here you can see the 2017 image versus the 2022 image. You can see the increase in urbanization.
This is study area 2, Begur Lake. Again, you can see a rapid increase in urbanization when you compare the 2017 image and 2022 image.
This is us taking observations at the lake. We had two visits, one on 19th of November and another one on 3rd of December. So this is the area covered by macrophytes in Kotigiri Lake. Here we have used Google Earth to map the area covered by each macrophyte. We have gone to the site and taken data, and according to that, we have recorded it here. So at first glance, you can see water hyacinth is the major macrophyte. Now on to the most frequently found macrophytes at Kotigiri Lake. First is water hyacinth with an area of 64,964 meters square and a percentage area of 54.2. Then we have Scotch grass, which percentage area of 3.94, and biscuit grass and dense flannelweed covered almost similar area. And then we have Parthenium and touch-me-not, which is found around the periphery of Kotigiri Lake. Now on to the lesser frequently found macrophytes in descending order, which is purple fountain grass, Singapore cherry, coconut trees, and bush morning. Then some sparsely found species here and there were trees like mango and teak, and plants like goatweed, wild almond, and asparagus found.
Now this is the map of the area covered by macrophytes at Begur Lake. As you can see, most of the lake is covered by water hyacinth and alligator weed, while a corner of it has slender knotweed. And these are the table
Water hyacinth and alligator weed together combined covered an area of 2,70,114 meters square, while Parthenium and common lantana covered similar area of 3,158 meters square and 3,182 meters square. Then, and these are the less frequent plants. We have Calotropis, carpet weed, fingergrass, and water thyme, while sparsely found species included Indian beech and hackberry.
Now I do a comparison study between both the lakes. First is the percentage of area covered by water hyacinth. In the case of Kotigare Lake, it was 54.2%, while in Baker Lake, it was 55.9%.
The percentage of area covered by all vegetation, including water hyacinth, was at Kotigare Lake it was 66.11%, while at Baker Lake it was 57.49%.
Now to the most prominent species. In Kotigare Lake, it was water hyacinth, Parthenium, touch-me-not, biscuit grass, and scotch grass. While in Baker Lake, we can see some similar species like water hyacinth and Parthenium, and other species like dense flower lordwood and common lantana. This is the gallery of the plants found at Kotigare Lake.
And this is the gallery at Baker Lake.
Now on to the conclusion and discussion. As you can see, both the lakes are being developed and have fence boundaries, though not well kept.
The water is mostly clean at the center, but the inlet and outlet areas were not clean. Diverse aquatic life was also present.
As you can see from our study, diverse vegetation is present both in as well as around the lakes. However, this current vegetation must be preserved and the density of some vegetation must be increased.
From our studies obtained, you can see that more than half of both the lakes is covered by water hyacinth and other weeds. This amount must be reduced to ensure the overall health of the lake. And this is our acknowledgement. We would like to thank IISc for hosting the lake conference. We would like to thank our school and principal ma'am. And we also would like to thank our parents for helping us in data collection. And these are our references. Thank you.
You mentioned that the development of lake. What do you mean by that? What was it?
In Kotigare Lake, recently the bunds had broken and there was flooding. So because of that, there was certain developments due to the bunds
And in Baker Lake, right now renovation is going on. And also there's a lot of work going on around the lake.
Your studies shows that both the lakes are covered with water hyacinth, right? Has it any effect on the fish or the other organisms that to be found in the lake?
The sunlight won't be penetrating the surface of the water hyacinth, and therefore and your oxygen quantity will also decrease. So there may be fish death and eutrophication also taking place.
Have you also observed that around Kotigare Lake, you see people carrying bundles of these water hyacinth back home?
Yeah.
Have you seen them?
Yeah, sometimes.
Yeah, okay. Did you inquire with them as to why they are carrying that water hyacinth?
That's for the clearing of the lake, and also that water hyacinth can also be used.
I think you are throwing away guess on that. Actually, water hyacinth people carry because they use it as a green fodder for cows and buffaloes.
Any question from the students?
You mentioned that we found aquatic animals. How many did you find? Can you name them?
We did not exactly. That's not
the scope of our
we just
project.
found
We found a lot of diverse aquatic life in Kotigare Lake, but that is not the scope of our project.
Bangalore, my school, Mona Shankar.
Maybe wait. People, this one first one, huh? Yeah, that is the this one. I think this one is up forward and it is back. Okay, and you should be facing this side. This camera recording is okay. We can start.
Good morning, everyone. Myself, Mauna Shankar of 9th standard. I'm from Bangalore Place Girls High School. My topic is about air quality in classrooms. So, we the students started bothering about air quality in classrooms during COVID-19 related restrictions. Wearing masks throughout the day and maintaining social distance caused suffocative atmosphere in the classroom. So, I felt classroom air quality might be the less studied topic. And Google search showed that as per familiar fresh air ventilation guidelines, carbon dioxide in indoor spaces should be maintained below 1000 ppm. So I wanted to check what would be the carbon dioxide level in our classrooms and how things could be made better. So my objectives are how to understand how carbon dioxide level varies in different classrooms and in same classroom in different times of the day, and to understand how effective plants are in reducing carbon dioxide, and to check the utility of different indoor plants in reducing carbon dioxide and encouraging students to maintain a green classroom, and to check the utility of or possibility of hydroponics in classroom. So materials and methods. So my materials are green loop pure hydroponic nutrients, plastic bottles, cotton, coriander seeds, thread, pinkbird plus air quality detector, and common indoor plants which are philodendron species. So my study area is classrooms from LKG to 10th standard, and front corridor, cellar, library, lab, etc. And we choose 9th standard, which is my classroom, to keep indoor plants and to check the difference in carbon dioxide level. So these are the pictures of recording carbon dioxide level in different areas of the school. So my method is that we visited different classroom during lunch break to check the carbon dioxide reading, and we set up a simple hydroponic system using plastic bottles so that it can be even we can even reduce the waste also. And we took count of all the plants in the premises, including potted plants, and selected our classroom as the study area, as I said. And we conducted an awareness class for other students and teachers also. So this is the picture of conducting an awareness class. So the expected outcome from our awareness class is that reducing carbon dioxide level in the premises and encouraging all the students to take part in maintaining green classroom and making a healthy atmosphere inside the classroom so that all the students can be healthy and score good grades. So these are the pictures of second seminar. So this is the data of carbon dioxide level before keeping indoor plants in the classroom, which is of carbon dioxide, TVOC and HCHO. This is the data for 5 days. So the other areas are also there, the number of plants, right, and carbon dioxide level. As you can see, the cellar has the HCHO level, which is in poor air quality. So these are the average readings of all the areas in the school. So 8th standard has HCHO level, which is very high. So these are the pictures of setting up hydroponic system. So analysis of changes after setting up indoor plants. So we have kept philodendron species, which is with broad leaves. The number of plants are 20, and we saw a marginal decrease. First, initially the experiment was with 10 plants, and the result was not satisfied. So we added another more 10 plants and continued the measuring. So regularly monitored the levels for 5 days, which is 3 times a day. And even though the weather was cool due to the cyclone, carbon dioxide level was in a marginal decrease. So there are some side effects of carbon dioxide, which is in more carbon dioxide in an indoor space or in a classroom. So it can cause restlessness, increased heart rate, difficulty in breathing, lack of concentration, fatigue, sleepiness, and reduced cognitive functions. So I guess except second and third one, most of the, all of the side effects are experienced by my classmate as I have seen. So the conclusion, the observations are that higher carbon dioxide level during cold weather condition. So the assumption for this observation is that the density of carbon dioxide increases when the temperature gets cooler. So when the temperature gets cooler, the carbon dioxide comes and settles near the ground. So it might be the reason for more carbon dioxide level. And higher carbon dioxide level during midday in two classrooms, which is 8th and 9th standard. It has poor ventilation, like the classroom is linear, not in a square manner. And lower carbon dioxide level in classes with higher strength but larger classroom area. As I said, all the other classes except 8th and 9th standard are in square manner. And lower carbon dioxide level in cellars where there were more plants but HCHO level was found higher. As I said in the beginning data, the cellar had high level of HCHO, and there is an assumption for that since the staff and van drivers park their vehicles in the cellar itself, the automobile exhaust might be the reason for high level of HCHO. So changes my study could make that I got an assurance from the management to fix an air purifier in the two classrooms, which is 8th and 9th standard, and more classroom turning carbon dioxide sensitive and started to grow indoor plants. And students understood about the importance of good ventilation and how it reduces VOC level. And many teachers admitted that they were not knowing how carbon dioxide affects the learning of a student. I think now they will be more considerate towards that. So these are some of the reference links. I sincerely thank my parents, teachers, classmates, and principal for extending all the cooperations needed for the project. And a special thanks to Madhavi of 10th standard for documenting my findings. I would also like to thank CS Department of IISc for giving me an opportunity to present my project. Thank you so much.
Was done for 5 days.
Yes,
at different levels.
Yes,
Do you think it is sufficient to draw conclusion, taking into climatic changes?
It's not like that. It's not like that. We started the project before itself, but suddenly we had 2 weeks of summer. What is it? Dasara holidays and all the sports day competitions, practices for that. We couldn't concentrate more on the project. So we took the 5 days of record.
So the conclusion is based mainly on
Yes. Yes. Thank you.
I have one question. You told me that the cyclone had some effect on carbon dioxide. How people can use why?
So the cyclone effect, it commonly has cold weather. So the cold weather can cause more level of carbon dioxide because the density of carbon dioxide increases. So that might be the reason.
You mentioned about VOC.
Yes.
Can you tell me the full form?
It is total volatile organic compounds, the organic compounds which vaporizes. Thank you.
Viveka.
Next, Bhagwani Vilas Manasali, Anushka and group. Mona, I need you to register here.
Some guest has someone standing there now. I got it. Just see if he wants to come and sit front. You can stand here, okay? Come here. Marathali, right? It's not there. I have any copy immediately. Pen drive. No, don't have. Okay, fine. You go. We'll call you later. Okay. Call you later. Click. Yeah, we'll do. Which school? Trupti, right? Floran. Wait a minute. This one is for changing slides. Okay, this one is for pointer. Slides 1 to back. Back is this one. Okay, this one forward back. Okay, then now you need to stand here. Okay, here it is recording. Okay, so you need to stand here and explain. Okay, all the best.
Namaste, very good morning to one and all present here. My name is Trupti. I'm studying in 9th standard from Kaveri Vidyakshetra Public School.
Myself, Yamuna JV, studying in 9th standard of Kaveri Vidyakshetram Public School. Our topic is about flora found in Kaveri Vidyakshetram campus. What is actually a flora? Flora is all the plant life present in a particular region or time, naturally native plants. Importance of flora. Flora or trees. Trees play an important role in ecosystem. Trees take in carbon dioxide and give out oxygen during photosynthesis. Trees provide shelter and food for both humans and animals. Trees also important source of medicine. Trees also play an important role in preventing soil erosion and floods. They also provide us pleasure. Our main objective was to find flora present in our school campus and medicinal values of them. Materials that we used are mobile measuring tape, pen and rough board. Methods are we use mobile phone to click photos to identify the plants using Google Lens. We use measuring tape to measure the height and girth of the tree. We used pen and rough board to note down the tree's height and girth, which we got by measuring. Our study area is our school campus, which is located in the 12 degree 52 minutes 32 seconds north longitude, 77 degree 39 minutes 6 seconds east latitude. Its perimeter is about 160 meters.
The result we found is we found 49 floras in our school campus, and they all have their own some of the medicinal values. The first is weeping fig. The height of it is 80 centimeter, girth is 13 centimeter, and the species found out the scientific name of it is Ficus benjaminia. The medicinal values are it is used to treat for the skin disorders, inflammation, piles, vomiting, leprosy, malaria, nose diseases, and cancer. And the next is white frangipani. Its height is approximately 3 meters. The girth is 15 centimeters, and the species found are 4. The scientific name of it is Plumeria pudica, and it used to treat for herbs, syphilis, ulcer, diarrhea, etc. The next is Mexican mint. The height is 40 centimeters. The girth is 2 centimeters, and there is only one species found in our school campus. The scientific name of it is Coleus amboinicus. The medicinal values are like it's used to treat for the cold, asthma, constipation, headache, cold, fever, skin diseases. And the next is tomato. Its height is 50 centimeters. The girth is 2 centimeters. The species found is only one. And the scientific name of it is Solanum lepersicum. And their medicinal values are like it's used to prevent eye problems and skin diseases. And the next is light green areca palm. The height is 5 meters and 10 to 30 centimeters. The girth is 30 centimeters, and there are 15 species found. The scientific name of it is Dipsis luxens. Like it's used for the treatment of mental disorder and an eye disorder and mild 4 meter. The girth is 35 centimeter, and there are 15 species found. The scientific name of it is Polythia longifolia. Medicinal values are it is used as an antibiotic agent in the indigenous system of medicine. The next is banana. As you know that banana has many medicinal values, and its height is 3 meter 40 centimeter. The girth is 48 centimeter, and there are seven species found. The scientific name of it is Musa. There are several medicinal values found they got in banana. Like its flower is cooked and gave gave to the patient who is suffering from these diabetes, and other, like it's used to cure for patient who is suffering from leprosy, fever, acute dysentery, etc. Next is neem tree. The height is 7 meter, the girth is 48 centimeter, and the species formed them too. The scientific name of it is Azadia indica. The medicinal values are it is used as an antifungal, antibiotic, antibacterial, antiviral, etc. The next is James calotroph. Its height is 5 meter, girth is 45 centimeter, and there is only one species found. The scientific name of it is Calotropis gigantea. The medicinal values are it is used as an antidote for snake bite, sinus fistula, mumps, burn injuries, and body pain. This is the checklist of our school campus, like the species found in our school campus, their common names and the number of species, the scientific names and the family which they belong to.
Conclusion. We conclude the project by saying that we found 49 flora. Out of them, there were 9 varieties. We conclude the project by informing that most of the trees have their own medicinal values. So we should plant more and more trees to stay connected with the mother nature. We would like to thank IISc committee for giving this opportunity for representing our project. We would like to thank our principal and headmasters for encouraging us to do with this project. We also like to thank our Aruna ma'am and Joshna ma'am for guiding us throughout this project. Our references. Thank you.
Nice presentation. Some of the species, I have 2 questions. Some of the species you found only one number. What have you done to improve the population of that particular plant species diversity? Have you done something? No. You just found it and left it. It's fine, one species also. Okay, another one, when you write the scientific name of a plant or anything, how you should name? How you should write genus and the species name and your example.
I also noticed that you have highlighted both, in the capitals. It has to be italic and small letters. And also the major thing, the problem is you said 49 floras. 49 floras. You're talking about only one flora. Then why are you mentioning the 49 floras everywhere in the slide? Another thing is has many of your teachers gone through the slides? Anybody guided you? Okay. That's a teacher having science background or commerce background or any other subject other than science? Science background. With science background, we are not saying yes or no. Yes or no. And continuously talking.
Answer the question what ma'am is asking. So that if you get understanding.
Nothing wrong. Everything is very good. Nothing to discourage you. Okay. Only things, certain things, keep in mind when you are making presentation, and also how you represent it. You don't need to talk about teacher also. You have a lot of books available in libraries, or Google. Right? Basic concepts should go first into your mind. Then only you should start the search. Everything is fine, nothing to discourage. Your very good effort, okay? Small things here and there. You have to say only 49 species or plants, not floras. You are referring to one single flora. In that, you cannot speak and say 49 floras. It is a collective. These are small things. You need to know the meaning. Everything is very nice. Everything is done properly. Okay.
ಅಲ್ಲಿ ಬನಾನಾ ಸ್ಲೈಡ್ ಅಲ್ಲಿ ಬನಾನಾ ಸ್ಲೈಡ್ ಅಲ್ಲಿ ಬರ್ತಾ ಇದೆ ಬರೀ ಜೀನಸ್ ನೇಮ್ ಬರ್ತೀರಾ ಎಲ್ಲ ಮರಕ್ಕೂ ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಯಾಕೆ ಇದ್ರು ಯಾರಿಗೂ ಬನಾನದು ಬರೀ ಜೀನಸ್ ನೇಮ್ ಇಲ್ಲ ಅಂದ್ರೆ
I just wanted to point out that in your conclusion slide, you said 9 varieties. You should have written varieties all throughout, so we have a clearer understanding about it. Because you found 49 in count, but you found 9 species, right?
Next presentation, Vidya Niketan, submit.
Front back pointer. ಈ camera recording.
Good morning to one and all present here. My name is Samit Singh Shetty, and today I'll be showing my project, which is heavy metal and organic chemical bioaccumulation in my plant. So the objective basically is to estimate the amount of heavy metal and organic chemical bioaccumulation, and then also to determine the rate at which metal bioaccumulation takes place for a specified time. So the principle is basically as follows. Now there are a lot of heavy metals that are present in the air, soil, water, and they are a global issue because of the consequences they have on the human health as well as the animals. So because of their toxicity as well as their accumulative properties and non-biodegradable nature, the heavy metals are very hazardous to terrestrial and aquatic ecosystems. Heavy metals are present in the environment as a result of natural sources and human activities. From natural sources, you have rocks, ore minerals, and volcanoes, and anthropogenic causes are urban development, generation of electricity, and metal industry. So, if we are talking about one of the main part of my project is basically wetlands, and we need to know how important they are. Wetlands, even though they occupy a very small percentage of the Earth's ecosystem, about 6%, they are very unique in which they have carbon sequestration. They have properties which dwarf other ecosystems. So even if they are present in a very small number, they're very important for the human environment. So wetlands have played an important role in purifying the contaminated water, and they've done it for centuries now. However, a majority of them have been exploited, and a lot of heavy metals through anthropogenic activities since the Industrial Revolution have been continuously spilled into these wetlands, and this has made it a very polluted environment. So different metals like cadmium, copper, plumber, zinc, manganese, they're all dissolved in metals. They're all heavy metals. Some of the sources of heavy metals, they come from oil refineries, petrochemical plants, pesticide production, chemical industries, untreated sewage sludge, and so on. If we're talking about sources of organic chemicals, which is again another part of my study, we have these chemicals that come from soil particles, fertilizers, pesticides, and then from grease and oil from cars, trucks, and roads. So the procedure is basically to use artificial intelligence and machine learning to find out how much of these heavy metals are getting accumulated, how much organic chemical bioaccumulation is happening in these wetlands, so that we can predict the health of the wetlands. We can see how well they fare over a course of time, how well they fare over the course of the period, and we can see what steps we can do to prevent this. So the first one is the accumulation of heavy metals. So the time period is taken as 1,000 years, and the copper concentration is taken as 0.05 parts per billion. The simulation is run, and it takes all the natural processes as taken as standard. And the model is built by assigning 2 layers of P. They have an even thickness of 3.4 meters, but then there is a variable thickness at different locations in order to simulate how it is present at a natural location. So this is how the simulation looks. You have a wetland cell which has different widths, and it has different demarcations, and you can see the depth. You can actually increase the depth or decrease it at various positions according to however this one. And this was basically done as random in order to make it as authentic as possible. So we have the time period that is taken here. The input units is basically years, 1,000 years, which translates to about 3 trillion seconds. And then finally, you have the simulation, which is run. So what we get is this graph. Now, this is for copper, and you see that for a period of 1,000 years, the concentration is about 4 to 5 times than it naturally would have been. Now, even though this is for a single heavy metal, there are other metals like cobalt, which almost show the same result. So in the simulation, for example, if you go about 100 years or 200 years, the copper concentration is not too much. But as you start increasing the years, for example, 200 years, 300 years, it starts increasing exponentially. So if you didn't have anthropogenic sources, you would have a relatively copper-free or cobalt-free wetland. But then once the anthropogenic activities are taken into account, you see that the concentrations increase up to 4 to 5 times, which is a huge problem. Now, yeah, so the conclusion for basically this simulation is that over thousands of years, the components in soil solution, they tend to accumulate. They make them zones where major ions and metals are trapped. And according to the simulation, as I said, the metal concentration can increase from 3 to 4 times in comparison to the original values. So next, we have the solution. One of the solutions is constructed wetlands. And this is one of the solutions is constructed wetlands is you can use this solution in order to you can use the solution in order to decrease the stress you have on natural wetlands. And this is basically that is that. And this is how a natural wetland looks. And yeah, so heavy metals, they're major pollutants, they pose threats, and it's important to reduce the bioaccumulation of metals. And these are the references. Yes, so actually, this is my presentation. There are a few slides of it. I don't think. ನಾನು ಮಾಡಿರೋ ಪ್ರೆಸೆಂಟೇಷನ್ ಕಾಪರ್ ಎಷ್ಟು ಅಕ್ಯುಮುಲೇಟ್ ಆಗುತ್ತೆ wetlands ecosystem.
ಅದು ನಮ್ಮ ಆರೋಗ್ಯದಲ್ಲಿ ಏರುಪೇರುಗಳಾಗುತ್ತೆ. ಅದಕ್ಕೆ ಅದು ಎನ್ವೈರನ್ಮೆಂಟಲ್ ಪರಿಸರದಲ್ಲಿ ಎಷ್ಟಿದೆ ಅನ್ನೋದನ್ನ ಅಭ್ಯಾಸ ಮಾಡಲಿಕ್ಕೆ ಅವರು ಕೃತಕ ಬುದ್ಧಿ ಆರ್ಟಿಫಿಷಿಯಲ್ ಇಂಟೆಲಿಜೆನ್ಸ್ ಬುದ್ಧಿಗೆ ಯೂಸ್ ಮಾಡಲ್ಲ ಅಲ್ವಾ, ಅದಕ್ಕೆ ನಾವು ಕೃತಕ ಬುದ್ಧಿ ಕಂಪ್ಯೂಟರ್ ಮೂಲಕ ಅದ್ರ ಮೂಲಕ ಮಾಡ್ತೇವೆ. ಹೊರಗಡೆ ಹಾಕಿದಾಗ ಎಷ್ಟು ವರ್ಷ ತಗೊಳುತ್ತೆ ಅದು
ಇದೊಂದು ಇರೋದಿಲ್ಲ.
ಇಲ್ಲ ನಾನು ಲಾಸ್ಟ್ ಟೈಮ್ ಕೊಟ್ಟಿದ್ದೆ.
ಓಕೆ, ಏನಿಲ್ಲ, ಸುಮ್ಮನೆ ಸಿಂಪಲ್ ಕನ್ನಡದಲ್ಲಿ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡ್ತೀನಿ.
ಅದು ಆ ಸ್ಲೈಡ್ ಕಳಿಸ್ತೀನಿ.
ನೋಡು ಇಲ್ಲಿರೋದು.
Because of anthropogenic impact on the nature, it is 3 to 4 times more than the base point. Otherwise, it would have been nothing accumulated of that. Natural processes with anthropogenic processes with ಅದೆಲ್ಲ ಏನು ಆಗಲ್ಲ. ಅದ್ರ ಬಗ್ಗೆ ಏನಿಲ್ಲ.
I was just indicating that when the
General statement could be 3 to 4 times increase ಆಗುತ್ತೆ because of human interference.
Yeah, the 3 to 4 times the maximum. Like if there was only maximum, it would be somewhere.
ಏನು ಇದನ್ನ
No, it's so much value. Okay, it makes sense.
Say, one of the things is that in the earlier evaluation of I told you, constricted wetland, suppose if this plant goes to the as a father, then we have still a problem. You didn't emphasize that, okay? We should make sure that it is non-palatable wetland plants, okay? That is a good thing. And I'm worried about this 1,000 years message because when it was dumped in the soil, some amount of remediation also happens. So, and the best option is to recover the copper that allows you to be there. So, anyway, you think about it. How it got came out.
Just to take care of your time.
You just say that, okay?
Yes.
You can explain this because it's a we are an expert today. Teacher doesn't require the PPT. Mind you, all never use PPT. I also didn't use PPT. All the youngsters want only the PPT. They don't prepare. Come to the class, put the PPT, talk something, and go. That means you have ready. Yes. You tell. Hey, you have the talent. I know your talent. Do it.
Okay, one more aspect of my research was that I was trying to see the organic chemical bioaccumulation. So in the organic chemical bioaccumulation, we see that, for example, if you use organic chemicals, these are the toxins that are introduced in wetlands. In my study, I used to control wetland and a wetland that was infected or which was introduced to an organic toxin. So basically, when you have these two studies, when you see the control study with a lot of plant species that are present or that are present in a wetland, for example, the diatoms and the animal species, you see that their population varies seasonally, and you can there's a graph that is present in the control study. You don't have any, you have variations, and the population stays almost linear or it reduces. But when there is the introduction of an organic toxin, in my study it was chloropyrifos, and when you introduce that, you see that the population of several species reduces, and a lot of the species are unable to feed on the plants, and the plant species reduce. The properties as well as the carbon sequestration properties of the wetlands they reduce. So that was one more aspect of my research.
How many times you have done the series?
No, at least 12 times for different.
You mentioned it.
Yes, not in this kind of.
That we value.
Next is Aditi and Group. Samir, I need you to register you.
Which school?
One of my teammates is Priya is not present because she's not there.
Archery like this one?
Yeah.
This one first one? Second one, right?
Yes.
This is the presentation, right? Okay. This is for forward, back. focus on front, front, backward. Well, this is the pointer if you want to point, and you should be facing this side. Okay. So it is recording. You just came from here. Facing here.
Good morning to one and all present here. I Aditi. I am Padmini of grade 11 from ACS Magnolia Maruti Public School. We are here to present our project on the study of Arikere Lake. Our school is located near the lake, and as students, we have noticed the lake's deteriorating condition. Therefore, we have selected this lake to assess its current status and put forward the need for its conservation and revival. Starting with the introduction, the lake name is derived from Ari meaning half and Keri meaning lake. It belongs to the Kesi Valley and was possibly a man-made water reservoir. Before encroachment, it covered 37 acres, but now it's estimated to be 25 acres. Our objective here is to study the current status of the lake by analyzing water parameters such as temperature, pH, dissolved oxygen, turbidity, alkalinity, and plankton quantification. These parameters were checked to understand the lake's immediate condition. To study soil parameters such as soil pH and soil humus content, and lastly, creating awareness in government schools and surrounding buildings of the lake. This is our area of study. The latitude and longitude coordinates are shown. The polygon with yellow color represents the boundary of the lake, and the polygon with red represents its encroachments. Extent as per spatial analysis is 7.88 acres. Lake inlet is towards the Banadeka main road, and the lake outlet is towards the Bailey 80 feet road. The coordinates are shown. Methodology is followed: analysis and results of the parameters, conducting surveys, education about the lake's conservation and waste management, biodiversity studies, and suggestions. These are the parameters analyzed. The methods and materials are shown. Along with the parameters, we conducted biodiversity studies of macrophytes, birds, and butterflies. The commonly macrophytes found are shown. The water hyacinth is widely spread across the lake. On the sides of the lake, there were other growing weeds, and are shown now. Study of butterflies was on a week observation from 2nd of December to 9th of December. The total bird species found were 10, and the average count of birds per day was 40. They were found. Here is the checklist of the birds from the previous report, 2016. The total bird species found were 34, but presently there is reduced bird population. The reasons are that there is no sewage treatment plant, leading to nutrition enrichment and macrophyte density increasing, causing low levels of oxygen, thereby reducing the aquatic fauna affecting the bird population. The chart here shows the abundance of insectivore birds present, and during our visits to the lake, there were very less number of insects found, thereby reducing the bird habitat present in the lake. Study of butterflies was also on a week observation, and the total butterfly species found were 8, and the average count of butterflies per day was 20. Moving forward with the water analysis, the sample collection points for sample 1 and 5 from the location where presence of duckweed was more, sample 2 from the periphery, sample 3 was 12 feet from the periphery, and sample 4 from the center. These were the results obtained after the checking of the parameters. We did a periodic comparison through graphs. Starting with the pH, showed the reading between for all samples were from 5 to 8. The lake overall is low in pH, resulting in excessive growth of macrophytes, which causes undesirable growth of macrophytes and planktons. The temperature of the lake is around 19 to 20 and did not vary much to other samples. Turbidity for sample 2, 3, 4, and 5 were less compared to the first, because the reason for more turbidity in the first could be because of deposition of clays and silts from the shoreline erosion and increase of phytoplankton growth. For sample 3, 4, and 5, it was comparatively higher than the first and second, because the first and second did not actually show phenolphthalein indicator. For all the samples, the dissolved oxygen levels were low. The reason is excessive macrophytes growing in the lake. For sample 3, 4, and 5, they had more plankton growth compared to the first and second. In general, for the results of the parameters, there can be variations in different factors affecting them. Along with the water parameters, we did the soil parameters. The reason during our visit to the lake, we found medical and constructional waste dumped. So we thought to analyze the soil pH, basic soil parameters like pH and humus, to understand the decrease in growth of terrestrial plants around the lake. So this soil sample showed acidic pH and soil humus value were found to be between 2 to 4 milliliter. So we compared this soil humus with the garden humus to and it was found to be in similar values. So on this positive note, the lake can be filled with good vegetation and upon the removal and cleaning of the waste around. The Avikere Lake falls under the Class E as per the previous report by RSC, and the lake water is found blackish in color because of unwanted water hyacinth growth and other weeds. These were the newspaper cuttings on the steps taken on elimination of land encroachment, proper fencing, diverting services, resetting, etc., which are presently yet to be done. Steps undertaken during our project, we submitted our report to the PDMP office and mailed it to the SAHAS organization. The vendors around the lake were educated about the effects of dumping garbage. Awareness drives were conducted in government schools, and a survey was conducted in the surrounding locality and within our school, and responses were received. These are the lake survey analysis and responses we received. Number of questions were asked, and majority of the responses by the residents and students were that they can help in simple contributions by voluntary programs that can be set up. In conclusion, the lake is covered with excessive growth of macrophytes, due to which further sample collection was not possible. The water parameters that were analyzed were not following the BIS standards. This clearly states that the lake is highly polluted and there is decrease in number of fish and other aquatic biota. The lake is still contaminated by sewage and the vegetable vendors still dump their waste. The impact is that the fishermen whose livelihood is dependent on this are facing several problems. This was based on an interview with the boatmen. Suggestions that can be done as shown. After all the analysis done, we realized that the most important objective was creating awareness. Awareness programs that can be conducted periodically will greatly help people understand the importance, protection, and conservation of the lakes. Before concluding, I would like to give an example of Saraki Lake. This lake was also once contaminated by sewage inflow and also had become a place of garbage dump. But with the joint effort of both the government and community, the lake was restored to its former glory. So we as students are inspired, and we would also like to continue to work on this project with proper resources, funding, and support. We will be able to implement these conservation measures for the lake. Through this forum, we would request the ISC team to take this message of Avikere Lake rejuvenation to appropriate higher authorities. Thank you. Thank you to ISC for providing us this following meeting. References taken as shown.
ನಾವು ಈ ಪ್ರಾಜೆಕ್ಟ್ ಏನಕ್ಕೆ ಮಾಡಿದ್ವಿ ಅಂದ್ರೆ ಆ ಕೆರೆ ನಮ್ ಸ್ಕೂಲ್ ಹತ್ರ ಇದೆ. ಅದು ತುಂಬಾ ಕಲುಷಿತವಾಗಿದೆ. ಅದಕ್ಕೆ ನಾವು ಸುತ್ತಮುತ್ತ ಯಾರ ಜನ ಇದ್ದಾರೆ ಅವ್ರಿಗೆಲ್ಲ ತಿಳಿಸಿ ನಮ್ಗೆ ಹೆಲ್ಪ್ ಮಾಡಕ್ಕೆ, ಈ ಕೆರೆನ ಕ್ಲೀನ್ ಮಾಡಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡಕ್ಕೆ ಈ ಪ್ರಾಜೆಕ್ಟ್ ಮಾಡಿದ್ವಿ. ನಾವು ಆ ಕೆರೆಯಲ್ಲಿ ನೀರು ಮತ್ತೆ ಮಣ್ಣು ಹೆಂಗಿದೆ ಅಂತ ಚೆಕ್ ಮಾಡಿದ್ವಿ.
You have it is a very elaborate work you have done, very good. And you have compared there are so many questions I have that I will ask you later. The insectivorous plants you have compared. How did you compare from the past 2016?
This is report by IC, which was already submitted. So we just put it and compare
My question is, you have done the November and December, right?
December.
And what is IIC's report? And I think that is 2 years they have posted. So this 2 years, how do you compare with the 2 months in February?
In that, we found more valuable parts actually. But in February, I know but still found very less number than what we did. But since we were given less amount of time to research, like whatever research like time we had, we did.
Yeah, if you
there may be difference also. There may be huge difference actually, but right now we can say there is actually reduced birth population.
So if you can do it, like if you are continuing,
Yes,
then
yes,
you can compare.
Yeah, sure. Yes, ma'am. Yes, ma'am. Not periodic.
You have restricted bringing awareness only to government schools or something like that.
There is the lake is there, like there is a school behind the lake, and there is surrounding people also. So government school students they actually know that the lakes condition about the lakes condition. So I thought to teach them and educate them about the proper disposal of waste in these conditions, so that they can tell the future generation and family friends peers around to not do garbage.
You said just now. Children who study in a government school, ಸರ್ಕಾರಿ ಶಾಲೆಯಲ್ಲಿ ಓದೋ ಮಕ್ಕಳು ಲೇಕ್ ಅನ್ನ ಪಲ್ಯೂಟ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಅಂತ ಡೋಂಟ್ ಜನರಲೈಸ್. Those children are better. Who's polluting the lake? You know who's polluting the lake? All the other people who are living in those high-rise buildings. Please see.
Yes.
The people who are living in those high society apartments surrounding the lake, they are the polluters. Their sewage is getting into the Arkera lake.
Yes.
Okay, they carry their waste that is produced at home. Bag alladhu ತುಂಬಿಸ್ಕೊಂಡು ಬಂದು ಕಾರಲ್ಲಿ ಹೋಗ್ತಾ ಹೋಗ್ತಾನೆ, ಟೂ ವೀಲರ್ ಅಲ್ಲಿ ಹೋಗ್ತಾ ಹೋಗ್ತಾನೆ, ಕೆಳಗೆ ಹಿಂಗೆ ಎಸ್ಬಿಟ್ಟು ಹೋಗ್ತಾರೆ. Okay, they need awareness, not the government school children.
Yes.
Okay, they are better. Okay.
So we also
Thank you.
talked to the surrounding localities. We had a direct interaction with them. But we'll carry forward.
Yeah. Okay.
Yes. Thank you.
Next presentation will be from Vidyaniketan School. Varun.
Varun Singh. Yes. Varun. Karnataka.
I know Kannada.
Okay.
But I'll present in English.
You can present in English. At the end, you just summarize. Okay. The people who are sitting back.
But I don't know the technical words like
Not technical
verification.
words. What happened or analyzed or something. Water analysis for the day. So.
First time, yeah.
Okay. The end. Okay. This is the pointer and this is for forward and this is for back. That's it.
Sorry, this is the forward.
This is for back. Varun, here. Since it is recording.
Okay. Greetings everyone, I'm Varun Singh from Vidyaniketan and I'm here for the Lake Symposium presentation on the topic of sustainability of peri-urban lakes. You may be wondering what is peri-urban lakes? Well, peri-urban city is the city which is situated at an outskirts of an urban area. So peri-urban lakes are the lakes which is situated at the outskirts of an urban area. Urban lake is an integral part of any urban city which has recreational and also environmental attributes. Lakes are also accompanied by parks which harbor various types of flora and fauna, and also lakes are vulnerable for the developments and also to the government policies which define lakes as only recreational attributes. It is very important to take in consideration that lakes have to be considered as environmental attribute. It was really important. Next, values of lakes are various: ecological, economical, cultural, social, and political. And these are the driving forces for the sustainability of the lake. Objectives and study area. Our main objective is to understand and identify the different water parameters of Ulla Lake, and also to suggest few measures for the sustainability of the lake, and also for the identify the driving forces for the sustainability of the lake. This is a geospatial image. You can see the upper part is the stream and the lower part is the lake. The upper part is stream inlet and outlet is the yellow marker. Down is the lake inlet at the right side, and the bottom right corner. Bottom left corner is the lake outlet. You can identify there's a large amount of peri-urban settlements, houses over there, and also you can identify the vegetation of the urban lake at the perimeters is also good. Methods and materials. We collected the sample once and we did the chemical analysis within 48 hours. We took the location to take the samples quite near to the inlet of the water, in which the water consisted a recent flow of the water and also the pre-existing water. And we took this sample only once. These are the lab experiments that we conducted. We took two sources to identify the different water parameters. One is from Sneha Guest House, which gave us the readings through their experiments. And another one, I conducted myself the different readings in Vidyaniketan, which we did with various procedures. The procedures in this book. These are the results that we got. We collected the sample at 5 p.m. and the temperature was 22 degrees Celsius. And these are other parameters that we found. Turbidity got as 10.9 nefrometric turbidity unit. This is well under limit and shows that the characterization of the surface of the water is not all that bad, and the visibility for the fish is very good. Alkalinity in terms of CaCO3, we got 260.4 milligrams per liter. This is well below 600 milligrams per liter, and this shows that the lake has the ability to neutralize acids on bases and also to overcome manures or moss growth. Dissolved oxygen. Well, dissolved oxygen is the amount of oxygen present in free oxygen and also in non-compound forms. While doing a fieldwork in the lake, we identified aquabirds like cranes, kingfishers. This shows that there is fishes living in the sea, living in the lake. So, as we got the dissolved oxygen of 4.3 milligrams per liter, this is well below 6.5 to 8 milligrams per liter. This shows that the fishes living in the lake are in a stress because it's pretty low. But if you consider the above 3 milligrams per liter, they are not in hypoxia or suffocating. Fishes still survive there, but they are in a stress. Chemical oxygen demand. Chemical oxygen demand is the amount of oxygen required for the chemical oxidization of the organic pollutants in a non-toxic form. This is about 64.89 milligrams per liter. This is well in control, even though this is seepage flow into the water. And we did several tests to identify the presence of carbon dioxide. We didn't find any detection of free carbon dioxide in the water. Biochemical oxygen demand. Biochemical oxygen demand is the amount of oxygen required for the microbes to break down the organic pollutants into non-toxic forms. We got it as 24.1 milligrams per liter. This is well below the 30 milligrams per liter limit, but it is very high. So, even though there is algae in the river, algae and other weeds floating in the river, there is very less amount of organic pollutants suspended in the lake. And there is a high typical sources for a high BOD like dead plants, failing septic systems, wastewater treatment plants, etc. As the BOD is very high, the impact is this high biochemical oxygen demand results in the increased amount of oxygen utilized for the microbes to degrade organic pollutants. This shows that there is lesser amount of dissolved oxygen because the oxygen is mainly utilized by the microbes. Measurements and impact hardness, fluoride and fluorides. Hardness, we got it as 225 parts per million, which shows that the water is very hard and it is very hard for the fishes to survive too. And fluorides, we did not detect any fluoride in the water. And fluoride, we got it as 180 milligrams per liter. This is well below 250 milligrams per liter, which does not result in a higher amount of toxicity in the organism. This doesn't have a big impact on the water. Next are the microbes. We did the microbial analysis and we got Euglena in the first one. It is dividing. We can see two of the Euglenas over there. And the others are stock attached penetrate diatoms. These are the diatoms attached into the leaf of the sample that we got. These are many were there, but these are the pictures that we got. These diatoms were attached to the leaf. Conclusion. As per the 2015 publication in the internet, the study shows that there will be increased amount of fluctuation during the winter season in the water quality in the water quality index. So we got this sample to be we got the sample during the beginning of the winter, and we did the analysis. So as the dissolved oxygen is very low, we can conclude that the health of the water, health of the lake is quite affected, and it is quite polluted. So for this, we can improve the purification methods and filtration methods, analyze them and implement them on the foreign materials that is being introduced into the river, into the lake. Next is bioremediation techniques. Due to the bioremediation is a process in which the environmental for the microbes which take part in the breakdown of chemical oxidization to break down organic pollutants, we can include bioremediation. So this alters the environment for the microbial growth to increase the bioremediation, thereby decreasing the biochemical oxygen demand, thereby increasing the dissolved oxygen, which will help in the betterment of the flora and fauna of the lake too. Overall, with the current study, we can say that the Ullal Lake can sustain for a few more years. These are the references that we do. Acknowledgement. I would really like to thank the school management, the school teachers to assisting me through the work, and also Sneha Testot in Chandralord in Bengaluru to helping me out figuring out the chemical parameters. And also I would really thank the Indian Institute of Science for giving us a good opportunity to present this research. Thank you. ಸರಿ ಸರಿ ನಾವು ಇಲ್ಲಿ ಉಳ್ಳಾಲ್ ಕೆರೆ ಬಗ್ಗೆ ವಾಟರ್ ಅನಾಲಿಸಿಸ್ ಮಾಡಿದೀವಿ. ಇದರಿಂದ ನಮ್ಗೆ ಸಿಕ್ಕಿದಿರುವ ರಿಸಲ್ಟ್ಸ್ ಏನಂದ್ರೆ ಉಳ್ಳಾಲ್ ಲೇಕ್ ಇನ್ನೂ ಸ್ವಲ್ಪ ವರ್ಷಗಳು ಸಸ್ಟೈನ್ ಆಗಬಹುದು. ಆಮೇಲೆ ಅದಕ್ಕೆ ಬೇಕಾದ ಪ್ಯೂರಿಫಿಕೇಶನ್ ಮೆಥಡ್ಸ್ ಎಲ್ಲ ಪ್ರಾರಂಭ ಮಾಡಬಹುದು.
ಇಲ್ಲಿ ಇನ್ನೊಂದು ವಿಷಯ ಏನಂತಂದ್ರೆ ಉಳ್ಳಾಲ್ ಲೇಕ್ ಇವರು ಶಾಲೆ ಪಕ್ಕದಲ್ಲೇ ಇದೆ. ಹಾಗಾಗಿ ಶಾಲೆಯ ಒಂದು ವಿದ್ಯಾನಿಕೇತನ ಶಾಲೆಯವರು ಕೆರೆಯನ್ನ ಅವ್ರು ಅಡಾಪ್ಟ್ ಮಾಡ್ಕೊಂಡಿದ್ದಾರೆ. ಇದು ತುಂಬಾ ಮುಖ್ಯ. ಎಲ್ಲಾ ಶಾಲೆಗೂ ಯಾರಾದ್ರೂ ಶಾಲೆ ಹೋಗ್ತಾವು, ತಮ್ಮ ಶಾಲೆ ಪಕ್ಕದಲ್ಲಿ ಯಾವ್ದಾದ್ರು ಕೆರೆ ಇದೆ ಅಂತಂದ್ರೆ ಅದನ್ನ ದಯವಿಟ್ಟು ಅಡಾಪ್ಟ್ ಮಾಡಿಕೊಳ್ಳೋದ್ರಿಂದ ಎರಡು ಅನುಕೂಲ ಆಗುತ್ತೆ. ಒಂದು, ಕೆರೆ ಸಂರಕ್ಷಣೆ ಆಗುತ್ತೆ. ಎರಡನೇದು, ಮಕ್ಕಳಿಗೆ ಕೆರೆ ಬಗ್ಗೆ, ನೀರಿನ ಬಗ್ಗೆ, ಪಕ್ಷಿಗಳ ಬಗ್ಗೆ, ಆಮೇಲೆ ಪ್ರದೂಷಿತವಾಗುವಂತಹ ವಸ್ತುಗಳ ಬಗ್ಗೆ, ಕೆಮಿಕಲ್ಸ್ ಬಗ್ಗೆ ಎಲ್ಲ ಬಗ್ಗೆನೂ ಸಂಶೋಧನೆಗಳು ಸತತವಾಗಿ ಆ ಮಕ್ಕಳೇ ಮಾಡಿ ಅದನ್ನು ಪರಿಣಾಮ ಪಡಿಬಹುದು. ಅವರ ಮುಂದೆ ಫರ್ದರ್ ಎಜುಕೇಶನ್ಗೆ ಅದು ತುಂಬಾ ಅನುಕೂಲ ಆಗುತ್ತೆ. ನೋಡಿ, ಉಳ್ಳಾಲ್ ಶಾಲೆಯವರು ವಿದ್ಯಾನಿಕೇತನ ಶಾಲೆಯವರು ಉಳ್ಳಾಲ್ ಲೇಕ್ ಮೇಲೆ ಒಂದು 15-20 ಪ್ರಾಜೆಕ್ಟ್ಸ್ ಮಾಡಿರ್ಬೇಕು ಆಲ್ ಟುಗೆದರ್. ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಅವ್ರ ಪ್ರೆಸೆಂಟೇಷನ್ ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಪ್ರತಿ ಲೇಕ್ ಸಿಂಪೋಸಿಯಂ ಅಲ್ಲಿ ಆ ಕೆರೆ ಇರುತ್ತೆ. ಅದೇ ರೀತಿಯಲ್ಲಿ ನಾವು KK ಶಾಲೆ ವರ್ತೂರು, ಅವರು ಹಾಗೇನೆ ಊರ ಪಕ್ಕದಲ್ಲಿದೆ, ಶಾಲೆ ಪಕ್ಕದಲ್ಲಿ ಕೆರೆ ಇದೆ ಅಂತ ಅದನ್ನ ಸತತವಾಗಿ 1998ರಿಂದ ಮಾಡ್ಕೊಂಡು ಬಂದಿದ್ದಾರೆ. ಹಾಗಾಗಿ ನಾವು ಶಾಲೆಯಲ್ಲಿ ಇರೋರು ಆತರ ಮಾಡ್ಕೊಂಡ್ರೆ ಅನುಕೂಲ ಆಗುತ್ತೆ.
Next one is Vidyaniketan School, Yadunamani.
ಯಾವ್ದಿದ್ರೆ ನೆಕ್ಸ್ಟ್ ಒಂದು ನಿಮಿಷ ಮಾತಾಡೋಣ. ಈ ಕಡೆ ಈ ಕಡೆ
A very good morning to everyone present. For this world to coexist with peace and harmony, we need sustainable development and environmental education. I am Gauri, this is Yaganandan and this is Vishnu, and we are here for Lake 2022. The global temperature is rising by 0.2 degrees Celsius every decade. This might seem like a negligible number, but it's a huge threat that needs immediate action. Global warming can have harmful effects, such as the rise in sea level, and it can completely imbalance the equilibrium in nature. We have dual objectives. Our first objective is to come up with easily adaptable solutions for sustainable development that can be followed by everyone. And our second objective is to spread awareness about environmental education. The principles that we follow are the usage of solar energy and to prove it as the best form of energy. To compare the carbon emissions by an IC, internal combustion engine, and an EV, which is electric vehicle. We also aim to bring awareness and educate people and also state some of the measures taken by the Karnataka government. So the energy that we all receive and the energy that we use comes from various resources, out of which fossil fuels is used the most to generate electricity. These fossil fuels generate a lot of carbon dioxide, which can have harmful effects. Solar energy, we prefer solar energy because it's inexpensive, clean, and it is very easy to harness from the photovoltaic technology, which produces almost zero carbon dioxide emissions. These are the reasons as to why we don't prefer the other kinds of resources to generate electricity or other kinds of energy.
So, as stated by Gauri earlier, the global rise in temperature, it's not a simple issue that can be ignored, right? 0.2 degrees is not a small amount. So, what is our main objective and what is our ultimate goal here is to compare the carbon emissions of an EV and an IC engine car and ultimately conclude that the carbon emissions from an IC engine is far greater when compared to that of an EV. So, this is a standard engine manufacturing process which has been adopted by auto manufacturers throughout the world. And here, this table states the components which are required in an IC engine, the ratios in which they need to be present, and the CO2 emissions in order to acquire them. So, adding up all the values, we get a total of 1,799 kg of CO2 just to manufacture the engine. And in order to manufacture an engine, not only raw materials, even electricity is required. In order to generate the electricity, also CO2 emissions are used. So, in this slide, the amount of CO2 emissions in order to generate the electricity is shown. Adding the values from the previous slide and this slide, we get a total of 4,900 kg of CO2 being released in order to manufacture one engine. Similarly, a plot has been plotted for the EV system, and the general requirements are as follows. Adding up the value, we get a total of 11,400 kg of CO2 being released. Now, this may contradict what we are trying to do here, but this is later explained in our presentation. So, this is a graphical comparison between the CO2 emissions during the manufacturing and the acquiring raw material acquiring processes. These are the specs comparison between both the systems. The main point to be noted in this slide is that the people's preference in India is 65% towards IC systems. So the people are still conservative towards IC systems. The mindset is conservative. This is a detailed analysis that has been done by us through simple maths, which shows the amount of CO2 which is released per kilometer and the total amount of fuel and electricity which is required by both the systems to be run throughout the lifespan. So the total value that we get for an IC system is 106,000 kg of CO2 throughout the lifespan, whereas for an EV, it's only 11,500 kg. This is a similar plot. And this is the total CO2 comparison between both the systems. As you can see, 90% is from an IC engine and only 10% is from an EV. So, we have ultimately concluded that an EV is better. But even this system has its own set of problems, right? So, the major challenges being that it offers limited range, it takes time to charge the batteries, and as stated earlier, people's mindset is still conservative towards IC engines. They don't want to try something new. So, what are we going to do about it? We emphasize on the idea of swap back. It is a seamless process wherein a charged battery can be replaced by an empty battery by the user itself. So I'm sure all of you have cupboards in your homes, right? And each cupboard will have a drawer. So imagine this as a huge cupboard, and it will have several drawers. Each drawer will contain one charge battery. When your vehicle runs out of charge, you can approach this docking station, replace your empty battery with a charged one, and you can continue your journey. Meanwhile, the old battery will be charging. It can be used by someone else. And the advantages of this system, the major advantage being it is eco-friendly and pocket-friendly, both to the consumer and the end user. It is time-saving as you need not charge the battery, right? And it is a one-of-a-kind product that harnesses renewable energy to its fullest potential. And if you already have an existing car and you don't want to spend your money on a new electric vehicle, so what are you going to do? You can just convert your existing car into an EV. So this is a detailed analysis of how much capital that you would require in order to compare a car and the efficiency of the converted car. Here we can see that an EV is far more efficient when compared to both of them, but the converted car is also efficient when compared to an ICE engine. And this is the graphical representation of the carbon emissions. We have completed the two aspects of our principle. Moving on to the third one.
Thank you, Vishnu. So, environmental education. It's educating our own people about how they can conserve the ecosystem in their capacity and also take responsible decisions. So now we know what sustainable development means. It's the development that meets the needs of the present generation without compromising the needs of the future generation. By adopting sustainable development, we can reduce problems like global warming that results in the optimal usage of resources. So our constitution itself states that it's the duty of every citizen to put his or her efforts in ecological conservation, and the other steps taken by the government are put in this way. Electric buses. We've already seen a lot of e-buses in the streets, so it's our own public transport that are evolving into EVs. So our Karnataka state government has planned to launch an all-electric fleet throughout the state by the year 2030. And the advantages of e-buses include less greenhouse gas emissions, more mileage, and much more. So we interviewed an official from the Karnataka Forest Department, Isar Krishna Kumar, and he gave us many such valuable information that contributed towards our project. And these are some of the solutions suggested by him and the steps taken by the Karnataka Forest Department. So to analyze the impact of environmental education, what we did is that we conducted a seminar to a group of 40 students of grade 5, and we gave them pre and post seminar questionnaires. And we noticed how their perspectives changed during the seminar, and the data regarding the same is put in this slide. So result that our result is that first we found out that an IC vehicle produces 9 times more carbon when compared to an EV. And second, that we found from the survey data analysis that students resolve to conserve the ecosystem consciously. Now, to conclude this, we now know that sustainable development is very important for a country that wishes to go towards development. And for that, we need innovative ideas like swap bags that can be acceptable by all. And that little effort from everyone is very important. If every individual in this country has that kind of mentality, this world can be changed. These are the picture galleries of the seminar. The link of the video, our references, and the references of the particular slide is put in the notes section. And our acknowledgement, we would like to thank IISc for giving us this opportunity and the whole Vidyaniketan management, and also Mr. Krishna Kumar for his valuable time.
We will continue to make this place and this world a better place, sea to sea, person to person, community to community, until this entire world is embraced with resilience, love, and life. Thank you.
We would also like to tell that we also took the liberty to calculate the monetary aspects surrounding this entire proposition to show how much cost efficient the battery manufacturing and usage would be. So you can please look at the slides.
And the links for each data is pinned in the note section under each slide.
Thank you.
I like to sum up the whole thing in Kannada. So beginning with ಜಗತ್ತಲ್ಲಿ ಗ್ಲೋಬಲ್ ವಾರ್ಮಿಂಗ್ ಅಂದ್ರೆ ಜಾಗತಿಕ ತಾಪಮಾನ ಏನಿದೆ ಅದು ಹೆಚ್ಚಾಗ್ತಾ ಇದೆ. ಸೊ ಅದಕ್ಕೆ ಮೇನ್ ಸೋರ್ಸ್ ಅಂದ್ರೆ ಮೇನ್ ಮೂಲ ಏನಂದ್ರೆ ಐಸಿ ವೆಹಿಕಲ್ಸ್ ಏನಿದೆ, ಪೆಟ್ರೋಲ್ ಮತ್ತು ಡೀಸೆಲ್ ಇಂದ ಚಾಲನೆ ಆಗುವ ವೆಹಿಕಲ್ಸ್ ಏನಿದೆ, ವಾಹನಗಳು, ಪೆಟ್ರೋಲ್ ಕಂಬಷನ್ ವೆಹಿಕಲ್ಸ್, ಅದರಿಂದ ಅದು ಹೊರಸೂಸುವ ಹೊಗೆಯಿಂದ ಅದರಲ್ಲಿ ತುಂಬಾ ಹಾನಿಕರ ಅಂಶಗಳಿರುತ್ತೆ, ಅದು ಕೂಡ ಗ್ಲೋಬಲ್ ವಾರ್ಮಿಂಗ್ ಕಾಸ್ ಆಗುತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ನಾವೇನ್ ಮಾಡ್ಬೇಕು, ಇವಿಗೆ ತಗೊಂಡು ಹೋಗೋದು. ಸೊ ಇವಿ ಅಂದ್ರೆ ವಿದ್ಯುತ್ ಮೂಲಕ ಚಾಲನೆ ಆಗುವ ಕ್ಯಾಲ್ಕುಲೇಷನ್ ಒಂದು ಪೆಟ್ರೋಲ್ನಿಂದ ಹೋಗುತ್ತೆ 9 ಪಟ್ಟು ಜಾಸ್ತಿ ಕಾರ್ಬನ್ ಎಮಿಷನ್ ಮಾಡುತ್ತೆ when compared to an EV. And so that's EVs ನ ಹೋಗ್ಬೇಕಂದ್ರೆ ಅದಕ್ಕೂ ಒಂದು ಪ್ರಾಬ್ಲಮ್ಸ್ ಇದೆ. ಅದನ್ನ ಓವರ್ಕಮ್ ಮಾಡೋಕೆ ನಮ್ಮ ಐಡಿಯಾ ಇದೆ ಸ್ವಾಪ್ ಬ್ಯಾಟ್ಸ್, ಅದನ್ನ ನಾವು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತೀವಿ. ಸೊ ಈ ಐಡಿಯಾಸ್ ನಾವು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ರೆ ಸಾಕಾಗಲ್ಲ, ಜನರಿಗೆ ಒಂದು ಅರಿವು ಮೂಡಿಸಬೇಕು, ಅವರಿಗೆ ಶಿಕ್ಷಣ ನೀಡಬೇಕಾಗುತ್ತೆ. ಸೊ ಎನ್ವೈರನ್ಮೆಂಟಲ್ ಎಜುಕೇಶನ್ ಅಂದ್ರೆ ಪರಿಸರದ ಬಗ್ಗೆ ಶಿಕ್ಷಣ ಇದೆ, ಅದು ಕೂಡ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ನಮ್ಮ ಕನ್ಕ್ಲೂಷನ್ ಏನಂದ್ರೆ ಸುಸ್ಥಿರ ಅಭಿವೃದ್ಧಿ ಅಂದ್ರೆ ಸಸ್ಟೈನಬಲ್ ಡೆವಲಪ್ಮೆಂಟ್ ಏನಿದೆ ಅದಕ್ಕೆ ಒಂದು ಇನ್ನೋವೇಟಿವ್ ಐಡಿಯಾಸ್ ಅಂದ್ರೆ ಅದರ ಐಡಿಯಾಸ್ ನಮಗೆ ಏನಿದೆ ಸಸ್ಟೈನಬಲ್ ಸ್ವಾಪ್ ಬ್ಯಾಟ್ಸ್ ಬಗ್ಗೆ ಅಂತದ್ದು. ಅಂಡ್ ಇನ್ನೊಂದು ಪರಿಸರದ ಬಗ್ಗೆ ಶಿಕ್ಷಣ ಜಗತ್ತಿಗೆ ಅರಿವು ಮೂಡಿಸುವ ಮೂಲಕ ನಾವು ನಮ್ಮ ಮುಂದಿನ ಪೀಳಿಗೆಗೆ ಒಂದು ಸುರಕ್ಷಿತ ಪ್ರಪಂಚವನ್ನು ಸೃಷ್ಟಿಸಬಹುದು.
ಆಪರ್ಚುನಿಟಿ ನಮಗೆ ಕನ್ನಡದಲ್ಲಿ ಪ್ರಶ್ನೆ ಕೇಳಬಹುದು. ಕನ್ನಡದವರು ಕೇಳಿ, ಕನ್ನಡದಲ್ಲಿ ಕೇಳಿ. ಅವ್ರು ಹೇಳಿರೋದು ಈಗ ಅರ್ಥ ಆಯ್ತಾ? ಹೌದು, ನನ್ ಕೇಳಿದ್ರೆ ಏನ್ ಅನ್ಕೊಂತಾರೋ, ನಗ್ತಾರೋ ಅಥವಾ ಹೇಳ್ತಾರೋ ಇಲ್ವೋ ಅಂತ ಸಂಕೋಚ ಪಡ್ಬೇಡಿ. ನೀವ್ ಕೇಳಿದ ಪ್ರಶ್ನೆ ಏನೇ ಇರಬಹುದು, ಎಷ್ಟು ಚಿಕ್ಕದಾಗಿರ್ಬಹುದು, ರೈಟ್? ಪ್ರಶ್ನೆ ಕೇಳಿದ್ರೆ ಕಲೀರಿ.
ನೀವು ಇವಿ ಅದ್ರ ಜೀವನ ಪೂರ್ತಿ ಎಷ್ಟು ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಎಮಿಟ್ ಮಾಡುತ್ತೆ ಅನ್ನೋದನ್ನ ನಾವು ನೋಡಿದ್ರಲ್ಲ, ಆ ಇದ್ರಲ್ಲಿ ನೀವು ಎಲೆಕ್ಟ್ರಿಸಿಟಿ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡಕ್ಕೆ ಏನು ಕಲ್ಲಿದ್ದಲಿಂದ ಮಾಡ್ತಾರೆ ಅದನ್ನ ಕನ್ಸಿಡರ್ ಮಾಡಿದ್ರೆ ಅಥವಾ ಬರೀ ಸೋಲಾರ್?
ತುಂಬಾ ಒಳ್ಳೆ ಕ್ವೆಶ್ಚನ್. ನಾವು ಎರಡನ್ನೂ ಕನ್ಸಿಡರ್ ಮಾಡ್ತೀವಿ. ಒಂದ್ ವೇಳೆ ನೀನು ಕೋಲ್ ಇಂದ ಜನರೇಟ್ ಆಗಿರೋ ಎಲೆಕ್ಟ್ರಿಸಿಟಿ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಎಷ್ಟು ಕಾರ್ಬನ್ ಎಮಿಷನ್ ಬರುತ್ತೆ, ಅದೇ ಸೋಲಾರ್ ಪವರ್ ಇಂದ ಜನರೇಟ್ ಮಾಡಿರೋ ಎಲೆಕ್ಟ್ರಿಸಿಟಿ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಎಷ್ಟು ಕಾರ್ಬನ್ ಎಮಿಷನ್ ಆಗುತ್ತೆ, ಎರಡು ಡೀಟೇಲ್ ಆಗಿ ಕಂಪೇರ್ ಮಾಡ್ತೀವಿ. ಎಲೆಕ್ಟ್ರಿಸಿಟಿ ಇಂದ ಜನರೇಟ್ ಮಾಡಿದ್ರೆ ಎಷ್ಟು ಬರುತ್ತೆ, ಅದೇ ನಾವು ಸೋಲಾರ್ ಪವರ್ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಇನ್ನೂ ಕಮ್ಮಿ ಬರುತ್ತೆ ಕಾರ್ಬನ್ ಎಮಿಷನ್. ಇದು ಇನಿಷಿಯೇಟಿವ್ ಆಟೋ ಮ್ಯಾನುಫ್ಯಾಕ್ಚರ್ಸ್ ಯೂಸ್ ಮಾಡ್ತಿದ್ದಾರೆ. ಅವರ ಐಡಿಯಾ ಆ ಸ್ವಾಪ್ ಬ್ಯಾಟ್ ಬರೀ ಕೋಲಿಂದ ಜನರೇಟ್ ಆಗಿರೋ ಎಲೆಕ್ಟ್ರಿಸಿಟಿ ಮಾತ್ರ ಅಲ್ಲ, ಸೋಲಾರ್ ಪವರ್ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಇನ್ನೂ ಎಫಿಷಿಯಂಟ್ ಇರುತ್ತೆ ಅಂತ ಪ್ರತಿಷ್ಠಿಸ್ತೀವಿ.
How that you use this photovoltaic cell to harness the solar energy? Please let me know what it like photovoltaic cell is made up of and what is its capacity. Please let me know.
So, this photovoltaic is usually made from silicon. The silicon atoms are used to make this technology, as it can reflect more light. And as you all know, it's semiconductor. It is a good conductor for electricity generation, and this is the procedure that is mentioned in this slide. It has a threshold frequency. The frequency of light is easy to obtain from the sunlight. Hence, the solar energy is very efficient.
And coming to the capacity, we have used 10,000 kilowatt solar panel.
The material capacity, like we are expressing in some units, right?
Kilowatt.
That is what the energy what we are getting from the photovoltaic cell. Now I am asking the silicon, like they are the semiconductors made up of silicon and germanium, right? So in between that we have some electron like valence gap between that. Please just you need to check that. Okay, so
to grow that solar.
ಈ ಪ್ರಶ್ನೆ ನೀವು ಲಂಚ್ ಟೈಮ್ ಅಲ್ಲಿ ಹೇಳಿ ಅವ್ರಿಗೆ. ಓಕೆ, ನಿಮ್ಮ ಸಜೆಷನ್ ಕೊಡಿ ಅವ್ರಿಗೆ, ಈಸಿ ಆಗುತ್ತೆ. ಲಂಚ್ ಟೈಮ್ I will give you.
Sure. Yeah. Thank you so much.
We are running out of time.
Thank you, sir. Thank you all.
Thank you all.
Next is from Vidya Niketan, Aditya and Abu.
ಇಲ್ಲಿ ಕ್ಯಾಮ್ರಾ ರೆಕಾರ್ಡ್ ಆಗ್ತಿದೆ ಈ ಕಡೆ
ಶುರು ಮಾಡ್ಬೋದಾ? Good afternoon, Dr. So myself Aditya and my teammate name is Amol. Today we are going to present the topic waste management. So let me give you a brief introduction about waste management. It's nothing but collecting, segregating and managing the waste components and properly recycled, which ensures that the resources of the environment are protected. So our objective is very simple: to analyze the waste generated in the locality and to come up with preventive measures which can aid for waste management.
So basically what we have done is taken Govindaraj Nagar as the basis of a hotspot on the basis of the garbage being generated. We also approached the nearest BBMP office to collect information of the same. We also took some pictures of the area where garbage is being carelessly disposed by the BBMP office. And we also took the soil from the same area and we collected few test results. So this is the area where the garbage is being carelessly dumped. This rectangular area is around 3 to 4 acres of land where the garbage is being dumped by the BBMP. Upon asking them why it's being done, they're saying that it's being thrown there so that it can be further taken into recycling, but we don't know for sure if they're following it or not. So after collecting the soil samples from the same area, we first checked for the presence of microplastic, and we were able to detect under the 10x magnification and 40x magnification 40 microplastics. We were able to confirm that this is microplastics by shining light upon them. After shining light upon them, we see the purple color which is reflected, which indicates that it is plastic.
And we also did the analysis of the soil, as you can see. So find the pH level of the soil, what we did was we used demineralized water. Specifically, why we use demineralized water? Because the normal tap water consists of chlorine and other minerals, which might affect the pH value. So we use the universal indicator as well. And surprisingly, we found that the pH value is 1, that is pink in color. And generally, the pH value of soil ranges between 3.5 and 10, but this was found to be very highly acidic. And we also did some acid radical detection test where we took the soil solution and we added some ammonia monoxide solution. We added few drops of concentrated HNO3. So the yellow precipitate was obtained, as you can see in the picture, and hence we came to a conclusion that phosphate is. And we also approached the nearest BBMP office and collected the data of the waste generated in the ward of Govindaraj Nagar. So nearly 350 grams to 450 grams of waste is generated by each house per day in the locality where I stay. And on further analysis, we went to each of our neighboring houses, collected the waste, and this procedure was done for 1 week. And what we did was we took the average waste that is generated and found out that it is 430 grams approx, and nearly 122,000 kgs of waste is generated in my ward, where it is generated by nearly 3.5 to 3.6 lakh houses. So the statistics of the waste generated, which are collected from BBMP office, as is shown here. So it is mainly classified into 3 types: the household waste, commercial waste, as well as the street sweeping waste. In terms of percentage, it's 68.5%, 28.3%, and 3.1 to 3.2%. In terms of kg per month, it's nearly 84,000 kgs generated per month, and the commercial waste comes up to nearly 34,700 kgs, and the street sweeping waste is at 3,800 kgs.
So due to hyperconsumerism of products in the area, there is inevitably garbage waste being generated. So we asked the BBMP what mitigations they have taken. And they told us that there are around 7 wet waste processing plants, biomethane gas plants, and 1 sanitary landfill. Despite all these present, that only 20 to 25% of the total waste that is being generated is being recycled.
So as you got to know that nearly 20 to 25% of the waste generated is only being recycled. So there are some people who have come up with innovative ideas like turning plastic waste into roads. So this was done in Bangalore itself by two scientists named Mr. Ramat Khan and Dr. They tied up with RV Engineering College and created a substance called as polyblend. So what they did was they mixed the recycled plastic with this polyblend and generally they use it to lay the road. So generally why they use this because the normal lifespan of a road is between 3 to 4 years. But if they use a polymerized bitumen and usually a road, it extends up to 6 to 7 years. One of the prominent reasons is because of the melting point of the plastic. It's between 130 to 140 degrees Celsius, whereas for just a bitumen it's 60 to 70 degrees Celsius. So we can clearly see that the plastic roads are even more efficient and can withstand very, very high temperature and corrosive weather.
So by now we all are familiar with Adamya Chetana, which was established by Tejaswini Ma'am. So I'll give a brief, but since we already know more about it. Food we've been eating in the past days was provided and cooked in the zero garbage kitchen of Adamya Chetana. So as the name suggests, there is absolutely no garbage being generated by this. What they basically do is instead of LPG, they use liquid fuels, and the gas which is generated in the same is sent to the farmers to be used as manure. And the vegetable waste which are being generated, this is sent to nearby goshalas so that they can be grazed upon. This indeed is proving to be an efficient, sustainable model.
Let's talk about an NGO. So here stands the Mumbai-based organization where they generally tie up with IT companies. And recently, in 2018, they conducted a survey or a project with the IT company in Bangalore called MIT, where they educated the youngsters about replacing the plastic cutleries with the bamboo cutleries. So these bamboo cutleries are more sustainable, and if thrown, they can be recycled properly. So the office which used to produce nearly 250 kg of waste, the IT company now produces less than 7 kg of waste. So we can really see the contribution of the NGOs in the society.
So here we can see that we have been doing composting from the wet waste which is generated at the house. And also some of the pictures taken here are from the Gnanabharati ward, where the residential area associations have approached the PBMP office and ensured that the cleaning is done around places. Some places where black spots, in the places of roads where the garbage is being carelessly disposed by the people, are being cleaned, and then rangolis are being thrown around the area, which ensures that the garbage will not be thrown on that area again. This has been done in the past 3 to 4 months, and it's been proved really effective. I would like to thank, first of all, I would like to thank Shikuna and all the other teachers who supported us and guided us throughout the presentation. We would also like to give my gratitude to IISc as well as the school management for giving us such an opportunity to present our thoughts and ideas and also to
So what to conclude that our venture will not end here. Surely in future we will try to come up with more and more innovative ideas in our research, and we will not try, we will surely do, because try means you do means we will surely do. Thank you. ಮೂಲತಃ ತ್ಯಾಜ್ಯ ನಿರ್ವಹಣೆ ವೇಸ್ಟ್ ಮ್ಯಾನೇಜ್ಮೆಂಟ್ ಬಗ್ಗೆ ಹೇಳಿದ್ವಿ. ಮತ್ತೆ ತುಂಬಾ ವಿಜ್ಞಾನಿಗಳು ಮತ್ತೆ ತುಂಬಾ ಸರ್ಕಾರೇತರ ಸಂಸ್ಥೆಗಳ ಕೊಡುಗೆಗಳನ್ನು ಇಲ್ಲಿ ತೋರಿಸಿದ್ವಿ. ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಏರಿಯಾ ತಗೊಂಡ್ವಿ. ಸೊ ಆ ಜಾಗಗಳಲ್ಲಿ ಹೇಗೆ ಕಸಗಳನ್ನ ಕೇರ್ಲೆಸ್ಲಿ ಹೆಂಗ್ ಬಿಸಾಕ್ತಿದ್ದಾರೆ, ಆಮೇಲೆ ಅದನ್ನ ಹೇಗೆ ಸರಿಪಡಿಸ್ಬೋದು, ಮತ್ತೆ ಅಲ್ಲಿರೋ ಮಣ್ಣಲ್ಲಿ ಯಾವ್ಯಾವ ಸತ್ವಗಳಿದೆ, ಯಾವ್ಯಾವ ಸತ್ವಗಳು ಹೋಗಿದ್ದಾವೆ, ಮತ್ತೆ ಪ್ಲಾಸ್ಟಿಕ್ ಎಷ್ಟಿದೆ, ಅದ್ರ ಬಗ್ಗೆ ನಾವು ಕೆಲವು ಸಂಶೋಧನೆಗಳನ್ನು ಮಾಡಿ ಮುಂದೆ. ಆಮೇಲೆ ತುಂಬಾ ವಿಜ್ಞಾನಿಗಳು, ಸರ್ಕಾರೇತರ ಸಂಸ್ಥೆಗಳ ಕೊಡುಗೆಗಳನ್ನು ನಾವು ಹೇಳಿದ್ದೀವಿ. This one, the feeling with it, we from positive to an average.
why she is asking that question is, her school children have already done this project sometime back. In the beginning, I think it was in 2012, if I'm not wrong. And in their school, they collected in 1 year the non-biodegradable plastic less than 40 micron, 1,650 kgs in 1 year. And it was given to the same company who used it in asphalting Taban Road and Old Airport Road. The plastic collected from her school was used to asphalt those two roads. That's it.
It's in Yelchanal. Okay, if you have not visited, then I don't have that question. I wanted to ask if you are telling this plastic roads are so effective. So effective. Why don't our roads are done in plastic?
Because politicians cannot make the money. They have to make the roads repair, etc. Every now and then, so that they are taking. That could be the only reason, and maybe the scaling up will be
The university, Bangalore University, they had actually made this road like around 30 years ago. Right now it was gone, and now they have just put regular filament road and they have put.
When Modi came to visit,
That is the time when they changed it everything. And
see, in India, we have a very good brains, very good policies, very good acts, and very good judiciary. But implementation of all this is zero. That is the problem. So anything we do, we can discuss, we can come to conclusion. Implementation is not doing.
Students any questions? So that plastic roads thing, I don't think I got it properly. If we make plastic roads, we usually plant trees on the roadside. So we
You are worried about water circulation. Suppose you use plastic in road, then how the water gets circulated? What about the trees that are surrounding the area? Right? The water will flow down and trees will be on the sides of the road. They'll get enough of water.
so like what the plastic toxins
That is very, very minimal compared to
no, they actually mix it with polymer. Polymer called
You get it in, like in a pencil that is there.
Yes, yeah.
It's a bigger size.
Yeah.
We have seen this. We have gone to the, you should visit.
the polymer name is styrene butadiene styrene. Also, that does not allow the plastic to
Keep continuing with it.
Yes, ma'am, of course.
Time ಮಾಡಬೇಕು.
So next, Pratham from CBSA industry.
ಪ್ರಥಮದ ಇದಲ್ಲ ಕೊಡಿ ಇಲ್ಲಿ ನಿಂತ್ಕೊ PDF ಆಯ್ತು ನೀನು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿ ನಾ ಇಲ್ಲೇ ಇರ್ತೀನಿ ಓಕೆನಾ ನಾನು ಮಾಡ್ತೀನಿ ತಗೋ ಇನ್ನು ಮುಂದೆ ಹೋಗೋದು ಯಾವಾಗ ಅಂತ ಹೇಳು ನಾನು ಹೋಗ್ತೀನಿ ಓಕೆನಾ ಒಂದು ನಿಮಿಷ ತಾಳು ಇಲ್ಲ ಇಲ್ಲ ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ಸ್ಟಾರ್ಟ್ ಮಾಡು.
ನನ್ನ ಹೆಸರು ಪ್ರಥಮ ಗೌಡ. ನಾನು 8ನೇ ತರಗತಿಯಲ್ಲಿ ಕಲಿಯುತ್ತಿದ್ದೇನೆ. ನಮ್ಮ ಶಾಲೆ ಶ್ರೀನಿವಾಸ. ನನ್ನೂರು ಒಳಕೋಡಿ. ಇದು ಕುಮಟಾದ ಹೃದಯ ಭಾಗದಿಂದ 4 ಕಿಲೋಮೀಟರ್ ದೂರದಲ್ಲಿರುವ ಅರವಿ ಸಮುದ್ರದ ತೀರದಲ್ಲಿದೆ. ಇದು ವಿಶಾಲವಾದ ಹಾಗೂ ಸುಂದರವಾದ ತೀರವನ್ನು ಹೊಂದಿದೆ. ಅರವಿಕೋಡಿ ಈರುಳ್ಳಿ ಎಂದರೆ ಸಿಹಿಯಾದ ರುಚಿಯಾದ ಕಾರವಿಲ್ಲದ ಕಡಿನಲ್ಲಿ ನೀರು ಬರಿಸಿದ ಈರುಳ್ಳಿ ಎಂದರೆ ಇದು ಅರವಿಕೋಡಿ ಈರುಳ್ಳಿ. ಈರುಳ್ಳಿಯು ಸಹ ಒಂದು ಔಷಧಿ ಸಸ್ಯ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಅಲೆನ್ಸೆಫಾ. ಇದರ ಸಿಪ್ಪೆಯನ್ನು ನೆನೆಸಿ ಆರಿಸಿ ದಿನಕ್ಕೆ 2 ವೇಳೆಯಲ್ಲಿ ಕುಡಿಯುತ್ತಾ ಬಂದರೆ ಕಣ್ಣಿನ ಹೋಗುತ್ತದೆ. ನಾನು ಸಂದರ್ಶಿಸಿದ ಕೆಲವು ವ್ಯಕ್ತಿಗಳ ದೃಶ್ಯದ ತುಣುಕುಗಳು. ಇವರು ನನಗೆ ಅನೇಕ ಮಾಹಿತಿಗಳನ್ನು ತಿಳಿಸಿದ್ದಾರೆ. ಇವರಿಗೂ ಸಹ ನಾನು ಧನ್ಯವಾದಗಳನ್ನು ಹೇಳುತ್ತೇನೆ. ಎರಡು ಪಾತಾಳ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ರೋವೆಲ್ಫಿಯಾ ಸರ್ಪೆಂಟಿನ. ಸಂಸ್ಕೃತದಲ್ಲಿ ಸತ್ವಗಂಧ ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ ಒಟ್ಟೊಂದು ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಕರಾವಳಿ ಪ್ರದೇಶಗಳಲ್ಲಿ ಹಾಗೂ ಪಶ್ಚಿಮ ಘಟ್ಟಗಳಲ್ಲಿ ವಿಶೇಷವಾಗಿ ಕಂಡುಬರುತ್ತದೆ. ಇದರಲ್ಲಿ 2 ವಿಧವಿದೆ, ಕೆಂಪು ಮತ್ತು ಬಿಳಿ. ಇದರ ಬೇರನ್ನು ನಾವು ಬಣ್ಣದಲ್ಲಿ ಹಾಕಿದ್ದಾರೆ. ಇದರ ಬೇರಿನ ಶಕ್ತಿ ಹೆಚ್ಚಾಗುತ್ತದೆ ಎಂಬ ಆಯುರ್ವೇದದ ನಂಬಿಕೆಯೂ ಸಹ ಇದೆ. ಸರ್ಪಸುತ್ತಿಗೆ. ಇದರ ಬೇರನ್ನು ನಾವು ಕಲ್ಲು ತೆಗೆಯುವ ಕಲ್ಲಿನಲ್ಲಿ ತೆಗೆಯುವಾಗ ಹೆಚ್ಚಬೇಕು. ಕಿರಾತಕ ಕಡ್ಡಿ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಎಂಡೋಗ್ರಾಫಿಸ್ ಪೆನಿಕೆರೇಟಾ. ಎಲೆ ಮತ್ತು ಕಾಂಡ ಔಷಧೀಯ ಮೂಲ. ಯಾವುದೇ ಹೊಟ್ಟೆ ನೋವು ಇದ್ದರೆ ಇದರ ಕಷಾಯ ಸೇವಿಸಿದರೆ ಅದು ಗುಣಮುಖ. ಸಕ್ಕರೆ ಕಾಯಿಲೆ ಇದ್ದವರಿಗೂ ಇದು ರಾಮಬಾಣ. ಬಳ್ಳಿ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಟಿನೋಸ್ಪೊರಾ ಕಾರ್ಡಿಫೋಲಿಯಾ. ಕಾಂಡವನ್ನು ಜಜ್ಜಿ ರಸವನ್ನು ತೆಗೆದು ಜೇನುತುಪ್ಪದೊಂದಿಗೆ 6 ಸೇವನೆಗೂ ಮುಂಚೆ ಸೇವಿಸಿದರೆ ಸ್ತ್ರೀದೋಷಗಳು ಅಂದರೆ ವಾತಪಿತ್ತ ಕಫ ನಿವಾರಣೆ ಆಗುವುದು. ಬಿಳಿ ಅಕ್ಕಿ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಎಲೆಟ್ರಾಫಿಸ್ ಪೆನಿಕೆರೇಟಾ. ಸಂಸ್ಕೃತದಲ್ಲಿ ಅರ್ಕ ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ ಟೆಕ್ ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಕಜ್ಜಿಗೆ. ಬಿಳಿ ಅಕ್ಕಿಯ ಹಾಲು ವ್ಯಕ್ತಿಗೆ ಹಾಗೂ ಬಿಳಿ ಅಕ್ಕಿಗೆ ಲೇಪಿಸಿದರೆ ಅದು ಗುಣವಾಗುವುದು. ಕಲ್ಲುಬಾಳೆ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಬೂಸಾ ಸೂಪರ್. ಸಂಸ್ಕೃತದಲ್ಲಿ ಜಂಗಲಿ ಕೇಲು ಹಾಗೂ ಸಂಸ್ಕೃತದಲ್ಲಿ ಪೀನಕದಲ್ಲಿ ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ ಜಂಗಲಿ ಕೇಲು ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಸಹ್ಯಾದ್ರಿ ಕಾಡುಗಳಲ್ಲಿ ಹಾಗೂ ಪಶ್ಚಿಮ ಘಟ್ಟಗಳಲ್ಲಿ ವಿಶೇಷವಾಗಿ ಕಂಡುಬರುತ್ತದೆ. ಇದರ ಬೀಜ ಔಷಧಿ ಮೂಲ. ಆಮಶಂಕಿಗೆ. ಇದರ ಬೀಜವನ್ನು ತೆಗೆದುಕೊಂಡು ನೀರಿನಲ್ಲಿ ಹಾಕಿ ಪಾಲುಗಳನ್ನು ಬೆರೆಸಿ ಕುಡಿಯಬೇಕು. ಎಮ್ಮೆ ಮೊಸರಿನಲ್ಲಿ ಬಿಸಿಯನ್ನು ಊಟ ಮಾಡಬೇಕು. ವೀಳ್ಯದೆಲೆ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಪೈಪರ್ ಬೀಟಲ್. ವೀಳ್ಯದೆಲೆ ರಸ ಮುಕ್ಕಳಿಸಿ ದಿನಕ್ಕೆ 2 ವೇಳೆಯಂತೆ ಕುಡಿಯುವುದೆಂದರೆ ಬಾಯಿಯ ದುರ್ವಾಸನೆ ಹೋಗುವುದು. ಮುಖವನ್ನು ವೀಳ್ಯದೆಲೆ ಕಷಾಯದಿಂದ ತೊಳೆಯುವುದರಿಂದ ಮುಖದ ಕಲ್ಲು ಹೆಚ್ಚಾಗುತ್ತದೆ. ಬ್ರಾಹ್ಮಿ. ಇದರ ಮತ್ತೊಂದು ಹೆಸರು ಮಂಜೆಲ್ಲ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಸೆಂಟೆಲಾ ಐಸಿಯಾಟಿಕಾ. ನೆನಪಿನ ಶಕ್ತಿಯನ್ನು ಒದಗಿಸುತ್ತದೆ. ದೇಹದ ಉಷ್ಣತೆಯನ್ನು ನಿಯಂತ್ರಿಸುತ್ತದೆ. ಪತ್ರೆಗಿಡ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಏಗನ್ ಮರ್ಮುಲಸ್. ಕನ್ನಡದಲ್ಲಿ ಬಿಲ್ಪತ್ರೆ ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ ಬೇಲ, ಸಂಸ್ಕೃತದಲ್ಲಿ ಬಿಲ್ವ ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಉಷ್ಣ ಸುಮಣಿಕೆ. ಇದರ ಎಲೆಯನ್ನು ತೆಗೆದುಕೊಂಡು ಜಜ್ಜಿ ಒಂದು ಸೇರು ನೀರಿನಲ್ಲಿ ಹಾಕಿ ಕುದಿಸಿ ಕುಡಿಯಬೇಕು. ಜ್ವರ. ಪತ್ರೆಗಿಡದ ಕಾಯಿಯನ್ನು ಅಥವಾ ಎಲೆಯನ್ನು ತೆಗೆದುಕೊಂಡು ನೀರಿನಲ್ಲಿ ಹಾಕಿ ಅಥವಾ ಹಾಲಿನಲ್ಲಿ ಹಾಕಿ ಕುಡಿಯಬೇಕು. ಈ ಮಾಹಿತಿಯನ್ನು ನಮಗೆ ತಿಳಿಸಿದವರು ಮೀನಾಕ್ಷಿ ಪಡ್ವಾರ್ ಅರಳಿಕೋಡಿ. ಬಿಳಿ ಚಿತ್ರಮೂಲ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಪ್ಲಂಬೇಗೋ ಸೈಲೆಂಕ್ಯಾ. ಸಂಸ್ಕೃತದಲ್ಲಿ ಚಿತ್ರಕ ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ ಫಂಡರ ಚಿತ್ರಕ ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಗಂಟಲೊಳಗೆ ಆದ ಹುಣ್ಣು. ಇದರ ಬೇರನ್ನು ಅಕ್ಕಚ್ಚಿನಲ್ಲಿ ತೆಗೆದು ಆವಾಗ ಕೆತ್ತಬೇಕು. ಈ ಮಾಹಿತಿಯನ್ನು ನಮಗೆ ನೀಡಿದವರು ಸರದನೆ ಕಳೆಯಕೋಡಿ. ಬಿಳಿ ದಾಸವಾಳ. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಹಿಬಿಸ್ಕಸ್ ರೋಸಾ. ಕನ್ನಡದಲ್ಲಿ ದಾಸವಾಳ, ದಾಸನವು, ದಾಸನಿಗೆ ದಾಸವಾಳ ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಹಿಂದಿಯಲ್ಲಿ ಗುಡಲ. ಆಮಶಂಕಿಗೆ. ಇದರ ಹೂವು ಅಥವಾ ಮೊಗ್ಗನ್ನು ಕಾಲುತು ಮತ್ತು ಜೀರಿಗೆ ಪುಡಿ ಸೇರಿಸಿ ನಯವಾಗಿ ಅರೆದು ಕುಡಿಯಬೇಕು. ಬೇಳೆ ಉಡಲು. ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಜೆಟ್ರೋಫಾ ಕರ್ಕಸ್. ಕನ್ನಡದಲ್ಲಿ ಬೇಳೆ ಉಡಲು ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ
ಬಾಂಗ್ರೆಡ್ ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಮಲೆನಾಡಿನ ಪ್ರದೇಶಗಳಲ್ಲಿ ಹಾಗೂ ಕರಾವಳಿ ಜಿಲ್ಲೆಗಳಲ್ಲಿ ವಿಶೇಷವಾಗಿ ಕಂಡುಬರುತ್ತದೆ. ಇದರ ಎಲೆಯನ್ನು ಕಿತ್ತಾಗ ಬಿಳಿಯ ಹಾಲನ್ನು ಬಿಡುವುದು. ಆ ಹಾಲನ್ನು ನಾವು ಹಲ್ಲು ನೋವು ಇರುವ ಜಾಗಕ್ಕೆ ಹಚ್ಚಿದರೆ ಹಲ್ಲು ನೋವು ನಿವಾರಣೆ ಆಗುವುದು. ಈ ಗಿಡದಿಂದ ಜೈವಿಕ ಇಂಧನವೂ ಸಹ ಮಾಡಬಹುದು. ತುರಿಕೆ ಮತ್ತು ಕಜ್ಜಿ, ಇದರ ಬೇರನ್ನು ನಿಂಬೆ ರಸದಲ್ಲಿ ತೆಗೆದು ಗಂಧವನ್ನು ಬಿಸಿ ಮಾಡಿ ಲೇಪಿಸಬೇಕು. ಇದನ್ನು ಹಳ್ಳಿ ಜನರು ಬಟ್ಟೆಕನ ಗಿಡ ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಈ ಮಾಹಿತಿಯನ್ನು ನಮಗೆ ನೀಡಿದವರು ಸರದಾನ ಕಳುಕೊಡಿ. ಕಹಿಬೇವು, ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಅಜಿಟಿರೆಕ್ಟಾ ಇಂಡಿಕಾ. ಬೇವಿನೆಣ್ಣೆ ಕಷಾಯ ಮತ್ತು ಎಳ್ಳೆಣ್ಣೆ ಲೇಪನದಿಂದ ಚರ್ಮದ ತುರಿಕೆ ಹೋಗುತ್ತದೆ. ಬೇವಿನ ಎಣ್ಣೆ ಮತ್ತು ತುಳಸಿ ಎಲೆ ಹಾಕಿದ ನೀರಿನ ಸ್ನಾನದಿಂದ ಕಟ್ಟಿ ಮತ್ತು ಮಾಯವಾಗುತ್ತದೆ. ತುಂಬೆ ಗಿಡ, ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಲ್ಯೂಕೇಸೆಸ್ಟ್ರಾ. ಇದರ ಎಲೆ ಮತ್ತು ಔಷಧಿ ಮೂಲ. ತುಂಬೆ ಗಿಡದ ಎಲೆಯನ್ನು ರಸವನ್ನು ಹಾವು ಕಚ್ಚಿರುವ ಜಾಗಕ್ಕೆ ಹಚ್ಚುವ ಪದ್ಧತಿ ಇದೆ. ಹಾವಿನ ವಿಷ ರಕ್ತದಲ್ಲಿ ಸೇರಿಕೊಳ್ಳದೆ ಸಾವು ಸಂಭವಿಸುವ ಸಾಧ್ಯತೆ ಕಡಿಮೆ ಇದೆ. ಮಾಹಿತಿ ನೀಡಿದವರು ಈ ಗಿಡದ ಬಗ್ಗೆ ಮಾಹಿತಿ ನೀಡಿದವರು ನಮಗೆ ಸುನಂದ ಪಡ್ಗರ್ ಕೊಡಿ. ನೋಡಿ ಸರ, ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಅಲೋವೆರಾ. ಸಕ್ಕರೆ ಸಮಸ್ಯೆಗೂ ಪರಿಹಾರ ಅಲೋವೆರಾದಲ್ಲಿದೆ. ತಾಜಾ ಅಲೋವೆರಾ ಲೋಳೆಯನ್ನು ಕುಡಿದರೆ ಜೀರ್ಣಕ್ರಿಯೆ ಸುಧಾರಣೆ ಆಗುವುದು ಮತ್ತು ಅಸಿಡಿಟಿಯಿಂದ ಬಳಲುತ್ತಿರುವ ಜನರಿಗೆ ಪರಿಹಾರ ನೀಡುವುದು. ಚರ್ಮದ ಆರೈಕೆ ಮಾಡುವುದು, ಮೊಡವೆಯನ್ನು ನಿವಾರಿಸುವುದು, ತಲೆಹೊಟ್ಟನ್ನು ನಿವಾರಿಸುವುದು, ಚರ್ಮಕ್ಕೆ ಕಾಂತಿಯನ್ನು ನೀಡುವುದು. ಮಜ್ಜಿಗೆ ಹುಲ್ಲು, ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಸಿಂಗೋಫೋಗನ್ ಸಿಟ್ರೇಟಸ್. ಇಂಗ್ಲಿಷ್ ನಲ್ಲಿ ಮಲಬಾರ್ ಗ್ರಾಸ್ ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ ಪರಿಚಯ ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಒಣಕೆಮ್ಮಿಗೆ ಮಜ್ಜಿಗೆ ಹುಲ್ಲು 4 ತೊಲೆ ತೆಗೆದುಕೊಂಡು ನೀರಿನಲ್ಲಿ ಹಾಕಿ ಕುದಿಸಿ ಸೇವಿಸಬೇಕು. ನೆಗಡಿಯಿಂದ ಮೂಗಿನಲ್ಲಿ ಗಟ್ಟಿಯಾದ ಸಿಂಬಳ ಬರುತ್ತಿದ್ದರೂ ಸಹ ಕಡಿಮೆ ಆಗುವುದು. ಪಪ್ಪಾಯಿ, ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಕ್ಯಾಲಿಕಾ ಪಪ್ಪಾಯಿ. ಸಾಮಾನ್ಯವಾಗಿ ಎಲ್ಲಾ ಕಡೆಗಳಲ್ಲಿ ದೊರೆಯುವ ಅಪಾರ ಔಷಧಿ ಗುಣ ಹೊಂದಿರುವ ಈ ಸಸ್ಯವಾಗಿದೆ. ಇದರ ಎಲೆಯನ್ನು ಜಜ್ಜಿ ಸೋಸಿ ರಸಕ್ಕೆ ಜೇನುತುಪ್ಪ ಸೇವಿಸಿದರೆ ಡೆಂಗ್ಯೂ, ಮಲೇರಿಯಾ ಜ್ವರ ನಿವಾರಣೆ ಆಗುವುದು. ಸೋಮವಾರದ ಗಿಡ, ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಕ್ರೋಟಾನ್ ಒಬ್ಲೊಂಗಿಫೋಲಿಯಸ್. ಸಂಸ್ಕೃತದಲ್ಲಿ ಭೂತಾಳವೇರಿ ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ ಹೊಕ್ಕುಂ ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಕರಾವಳಿ ಪ್ರದೇಶಗಳಲ್ಲಿ ಹಾಗೂ ಸಹ್ಯಾದ್ರಿ ಕಾಡುಗಳಲ್ಲಿ ವಿಶೇಷವಾಗಿ ಕಂಡುಬರುತ್ತದೆ. ಸ್ವಲ್ಪ ಜೊತೆಗೆ ಸೋಮವಾರದ ಬೇರನ್ನು ನಿಂಬೆ ರಸದಲ್ಲಿ ತೆಗೆದು ಆವಕ್ಕೆ ಹಚ್ಚಬೇಕು. ಹಿಪ್ಪಲಿ, ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಪೈಪರ್ ಲಾಂಗಮ್. ಇದನ್ನು ಹಿಪ್ಪಲಿ ಎಂದು ಕರೆಯುವರು. ಅಸ್ತಮಕ್ಕೆ ಉತ್ತಮ ಗಿಡಮೂಲಿಕೆ. ಗಂಟಲು ನೋವಿನಿಂದ ಬಳಲುತ್ತಿರುವವರಿಗೆ ಇದೊಂದು ವರದಾನ. ಜೇನುತುಪ್ಪದಲ್ಲಿ ತೆಗೆದು ರಸವನ್ನು ಲೆಕ್ಕಿಸಬೇಕು. ನೆಲನಲ್ಲಿ, ಇದರ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಫಿಲೆಂಥಸ್ ನೀರುಳಿ. ಕಾಮಲೆಗೆ ನೆಲನಲ್ಲಿ ಉತ್ತಮ ಔಷಧಿ. ಹೊಟ್ಟೆ ನೋವು ಅಜೀರ್ಣದಿಂದ ಹೊಟ್ಟೆ ನೋವಾದಲ್ಲಿ ನೆಲನಲ್ಲಿ ಸೊಪ್ಪಿನ ಕಷಾಯ ತಯಾರಿಸಿ ತುಪ್ಪದಲ್ಲಿ ಹುರಿದ ಇಂಗಿನ ಪುಡಿ ಬೆರೆಸಿ ಕಷಾಯವನ್ನು ಸೇವಿಸಬೇಕು. ಈ ಗಿಡದ ಬಗ್ಗೆ ನಮಗೆ ಮಾಹಿತಿ ನೀಡಿದವರು ಶಾರದಾ ಕಳೆಯಕೊಡಿ. ನಾನು ಸಂದರ್ಶಿಸಿದ ವ್ಯಕ್ತಿಯ ಹೆಸರು, ವಿಳಾಸ, ಮನೆಮದ್ದು, ಉಪಯೋಗಿಸಿದ ಸಸ್ಯಗಳು, ಸ್ಥಳೀಯ ಹೆಸರು, ವೈಜ್ಞಾನಿಕ ಹೆಸರು. ನಾವು ಅನೇಕ ಮನೆಮದ್ದು ಗಿಡಗಳ ಮಾಹಿತಿ ಕಲೆಯಕ್ಕೆ ಔಷಧಿ ಗುಣವಿರುವ ಸಸ್ಯ ಮತ್ತು ಬೇರೆ ಬೇರೆ ಕಾಯಿಲೆಗೆ ಬರುವುದೆಂದು ತಿಳಿದುಕೊಂಡೆನು. ಕೋವಿಡ್-19 ಸಮಯದಲ್ಲಿ ಮನೆ ಮದ್ದು ತಿಂದರೆ ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿ ಹೆಚ್ಚಿಸಿಕೊಳ್ಳಬಹುದು. ಆರೋಗ್ಯ ಸಂಪತ್ತು ಇರುವ ದೇಹದಲ್ಲಿ ಆರೋಗ್ಯಪೂರ್ಣ ಮನಸ್ಸು ಇರುತ್ತದೆ ಎನ್ನುತ್ತಾ ಈ ಯೋಜನೆಯನ್ನು ಪ್ರಸ್ತುತಪಡಿಸಲು ಅವಕಾಶ ನೀಡಿದ ಭಾರತೀಯ ವಿಜ್ಞಾನ ಸಂಸ್ಥೆಗೆ ಹಾಗೂ ಮಾರ್ಗದರ್ಶನ ಮಾಡಿದ ನಮ್ಮ ಗುರುಗಳಿಗೆ ಕೃತಜ್ಞತೆಗಳು. ಧನ್ಯವಾದ.
ಅದನ್ನು ಮಾಡಿದ್ರೆ
ಆಗ ನೀನು ಅದು ಯಾವ್ದೋ ಇದೇ ಇರ್ತಾನೆ ಅದು ಅದರ ನೀರು ಮಾಡಿ ತಗೊಳ್ಕೊಂಡ್ರೆ ಅದು ಮೂಢನಂಬಿಕೆಯ ನಿಜ ವೈಜ್ಞಾನಿಕವಾಗಿ ಸಾಕ್ಷಿ ಇದೆಯಾ? I'm not doubting it. I'm just asking whether it's a superstition. Does it work every time? Does it work?
ಲಾಸ್ಟ್ ಕ್ವಶನ್ ನೋಡೋ ಸರ್ ಕೇಳ್ತಾರೆ.
ಮೊದಲಿಗೆ ನೀನು ಕನ್ನಡದಲ್ಲಿ ಪ್ರೆಸೆಂಟೇಷನ್ ಪ್ರಸ್ತುತಪಡಿಸಿದ್ದಕ್ಕೆ ನನ್ನ ಪರವಾಗಿ ತುಂಬಾ ಧನ್ಯವಾದಗಳು. ಬಹಳಷ್ಟು ಸಸ್ಯಗಳು ಬಳ್ಳಿಗಳ ಬಗ್ಗೆ ಮಾಹಿತಿ ವೀಳ್ಯದೆಲೆ ಕೊತ್ತಂಬರಿಗೆಲ್ಲ ನಿಮ್ಮ ಹಳ್ಳಿ ಸುತ್ತಮುತ್ತ ಅಡಿಕೆ ಮರನ ಬೆಳೀತಾರಲ್ಲ, ಅದನ್ನು ಸೇರಿಸ್ಕೊಂಡಿದ್ರೆ ನಿಮ್ಮ ಪ್ರೆಸೆಂಟೇಷನ್ ಅತ್ಯುತ್ತಮವಾಗಿ ಆಗ್ತಿತ್ತು ಅನ್ನೋ ನನ್ನ ಸಲಹೆ ಇರ್ತದೆ. ನಾನು ನಿಮ್ಗೆ ಪ್ರಶ್ನೆ ಕೇಳಬೇಕು ಅಂತೇನಿಲ್ಲ. ನೆಕ್ಸ್ಟ್ ಏನಾದ್ರು ಪ್ರೆಸೆಂಟೇಷನ್ ಕೊಡುವಾಗ ಅಡಿಕೆ ಮರನು ಸೇರಿಸ್ಕೊಂಡು ಅದರಿಂದ ಏನು ಉಪಯೋಗ ಆಗ್ತಿದೆ ಅಂತ ಸೇರಿಸ್ಕೊಂಡು ನಿಮ್ಮ ಪ್ರೆಸೆಂಟೇಷನ್ ಅನ್ನು ಕೊಡಿ ಅಂತ ನಾನು ಕೇಳ್ಕೊಳ್ತೇನೆ. ಥ್ಯಾಂಕ್ ಯು.
Next participant, Dhanya from Dhanya Monastery from Vanali Kumta.
College, school name? Right, no, nothing is registered here, right? I just need to see here and ಬನ್ನಿ. ಮೇಲ್ ಮಾಡಿದ್ದೀರಾ ಇಲ್ಲ ಪೆನ್ ಡ್ರೈವ್ ಎಲ್ಲ ಹಾಕಿಟ್ಟಿದ್ದೀರಾ? ನಮ್ಮ ಟೀಚರ್ ಇರೋದಾ ಅವ್ರಿಗೆ ಕೇಳ್ತಾರಿದ್ದೀರಾ? ಅಲ್ಲ ಪೆನ್ ಡ್ರೈವಾ ಹೆಸರು ಏನಿದೆ ಇದರಲ್ಲಿ?
ಗವರ್ಮೆಂಟ್ ಹೈಸ್ಕೂಲ್ ವನ್ನಳ್ಳಿ ಅಂತ ಇದೆ ನೋಡಿ.
ಇದಾ? ಅದಕ್ಕೆ ಆಗ ಗೊತ್ತಾಗಲಿಲ್ಲ. ಅದಕ್ಕೆ ಯಾವುದು ಇದೇನಾ?
ಪಿಪಿಟಿನೂ ಕೂಡ ಇತ್ತು ಪ್ರಥಮದು PDF.
ಅದು ಇದು ಚೇಂಜ್ ಆಗಿತ್ತು, ಫಾಂಟ್ ಎಲ್ಲ ಚೇಂಜ್ ಆಗಿತ್ತು, ಅದಕ್ಕೆ ನಾನು ಇದು ಮಾಡಿದೆ. ಒಂದು ನಿಮಿಷ ಹೇಳ್ತೀನಿ. ಇದೆ ಅಲ್ಲ ಮುಂದೆ ಈ ಕಡೆ ಸೈಡ್ ಬನ್ನಿ, ಇಲ್ಲಿ ಬನ್ನಿ. ಓಕೆನಾ? ಏನು ಟೆಸ್ಟ್ ಮಾಡಕ್ ಹೋಗ್ಬೇಡಿ. ಓಕೆ, ಇಲ್ಲಿ ರೆಕಾರ್ಡಿಂಗ್ ನಡೀತಿದೆ, ಅದಕ್ಕೆ ಈ ಕಡೆ ಸೈಡ್ ಹೇಳಿ ನಿಮ್ಗೆ ತೋರಿಸ್ಕೊಂಡು. ಓಕೆನಾ? ಚೆನ್ನಾಗಿ ಮಾಡಿ ಆಲ್ ದ ಬೆಸ್ಟ್. ಒಟ್ಟು ಹೇಳಲ್ಲ, ಇಲ್ಲಿ ಒಟ್ಟು ಇದು ಹಿಂದೆ ಅಷ್ಟೇ ಸಿಂಪಲ್. ಈ ಕಡೆ ಹಂಗೆ ಹಿಡ್ಕೊ, ಹಂಗಲ್ಲ ಹಿಂಗೆ. ಹಾಂ ಕರೆಕ್ಟ್. ಸರಿ.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು ಧನ್ಯ ಗಣಪತಿ ನಾಯ್ಕ್. ನನ್ನ ಹೆಸರು ನಮ್ಮ ವಿಷಯವೇನೆಂದರೆ ನಮ್ಮ ಗ್ರಾಮದಲ್ಲಿ ದೊರಕುವ ವನ್ಯ ಸಸ್ಯಗಳ ಪರಿಚಯ. ಇದರ ಉದ್ದೇಶ ನಮ್ಮ ಗ್ರಾಮದಲ್ಲಿ ದೊರಕುವ ಕಾಡು ಹಣ್ಣುಗಳ ಮಾಹಿತಿ ಮತ್ತು ಕಾಡು ಸಸ್ಯಗಳನ್ನು ಸಂಗ್ರಹಿಸುವುದು ಹಾಗೂ ಅವುಗಳಲ್ಲಿರುವ ಪೌಷ್ಟಿಕಾಂಶಗಳ ಬಗ್ಗೆ ಅರಿಯುವುದು. ವಿಧಾನ ನಾವು ಸರ್ವೆ ಮುಖಾಂತರ ವನ್ನಳ್ಳಿ ಹಾಗೂ ಮಂಗೋಡ್ಲ ಗ್ರಾಮಗಳಿಗೆ ತೆರಳಿ ಅಲ್ಲಿರುವ ವನ್ಯ
ಭಾಗಗಳ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ಸಂಗ್ರಹಿಸಿದ್ದೇವೆ. ಮೊದಲನೇದಾಗಿ ಕೆಸುವಿನ ಎಲೆ. ಇದನ್ನು ಕೊಲೋಕಾಸಿಯ ಅಲ್ಸೆಟ್ರನ ಎಂದು ಕರೆಯುತ್ತೇವೆ. ಕೆಸುವಿನ ಸಸ್ಯದಲ್ಲಿ ಕರಿಕೆಸ, ಕಾಡುಕೆಸ, ಮರಕೆಸ, ಮೊಟ್ಟೆಕೆಸ ಎಂಬೆಲ್ಲ ವಿಧಗಳಿವೆ. ಮಳೆಗಾಲದಲ್ಲಿ ಹೆಚ್ಚಾಗಿ ಬೆಳೆಯುತ್ತದೆ. ತಂಪಿನ ಪ್ರದೇಶದಲ್ಲಿ ಇದು ಹೆಚ್ಚು ಕಾಲ ಇರುತ್ತದೆ. ಕೆಸುವಿನ ಎಲೆ, ಗಡ್ಡೆ, ಬಳ್ಳಿ, ಕಾಂಡಗಳು ಇವೆಲ್ಲವೂ ಖಾದ್ಯ ಭಾಗಗಳಾಗಿವೆ. ಇವುಗಳಲ್ಲಿ ಕಾಡುಕೆಸ, ಮರಗೆಸ, ಮೊಟ್ಟೆಕೆಸ ಇವೆಲ್ಲವೂ ತಾನಾಗಿ ಬೆಳೆದುಕೊಳ್ಳುತ್ತವೆ. ಕೆಸುವಿನ ಎಲೆಯಿಂದ ಪತ್ರೊಡೆ ಎಂಬ ರುಚಿಕರ ತಿಂಡಿಯನ್ನು ಮಾಡುತ್ತಾರೆ. ಅಲ್ಲದೆ ಚಟ್ನಿ, ಕರ್ಕಿಯನ್ನು ಸಹ ತಯಾರಿಸಲಾಗುತ್ತದೆ. ಬೆಟ್ಟದ ನೆಲ್ಲಿಕಾಯಿ, ಇದನ್ನು ಎಂಬ್ಲಿಕಾ ಅಫಿಷನಾಲಿಸ್ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಬೆಟ್ಟ ಹಾಗೂ ಕಾಡಿನಲ್ಲಿ ದೊರೆಯುವ ಹುಳಿ ಒಗರು ರುಚಿಯನ್ನು ಇದು ಹೊಂದಿರುತ್ತದೆ. ನೆಲ್ಲಿಕಾಯಿಯಲ್ಲಿ ಸಿ ಜೀವಸತ್ವ ಇದ್ದು ಇದನ್ನು ನೇರವಾಗಿ ತಿನ್ನಬಹುದು. ನೆಲ್ಲಿಕಾಯಿಗಳನ್ನು ಬಿಸಿಲಿನಲ್ಲಿ ಒಣಗಿಸಿ ಇದನ್ನು ಡಬ್ಬದಲ್ಲಿ ಸಂಗ್ರಹಿಸಿಡಬಹುದು. ಇದನ್ನು ನೆಲ್ಲಿ ಚಟ್ಟು ಎಂದು ಕರೆಯಲಾಗುತ್ತದೆ. ಈ ಚಟ್ಟನ್ನು ಬೇಕಾದಾಗ ನೀರಿನಲ್ಲಿ ನೆನೆಸಿ 4 ಚಮಚ ಜೀರಿಗೆ, ಕಾಯಿ ತುರಿಯೊಂದಿಗೆ ರುಬ್ಬಿಕೊಂಡು ಕಂಬಳಿ ರೂಪದಲ್ಲಿಯೂ ಸೇವಿಸಬಹುದು. ಇದರಿಂದ ಉಪ್ಪಿನಕಾಯಿಗಳನ್ನು ಸಹ ಮಾಡುತ್ತಾರೆ. ಇದರ ಹಿಂಡಿಯನ್ನು ಮಕ್ಕಳಿಗೆ ಶೀತವಾದಾಗ ಕೊಡುತ್ತಾರೆ. ಉಪಯುಕ್ತ ಗುಣಗಳೇನೆಂದರೆ ಸಿ ಜೀವಸತ್ವ ಹೇರಳವಾಗಿದ್ದು ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿ ಹೆಚ್ಚಿಸುತ್ತದೆ ಹಾಗೂ ಪಿತ್ತ ನಿವಾರಕವಾಗಿದೆ. ಕಂದರಕಾಯಿ, ಕ್ಯಾರಿಸ್ಕಾ ಕ್ಯಾರಂಡಸ್, ಬೆಟ್ಟದಲ್ಲಿ ಕಂಡು ಬರುವ ಮುಳ್ಳುಗಳಿಂದ ಕೂಡಿದ ಪೊದೆಯಂತಹ ಸಸ್ಯ ಇದಾಗಿದೆ. ಈ ಸಸ್ಯದ ಕಾಯಿ ದುಂಡಾಗಿದ್ದು ಹಸಿರಾಗಿರುತ್ತದೆ ಮತ್ತು ಹುಳಿ ರುಚಿಯನ್ನು ಇದು ಹೊಂದಿರುತ್ತದೆ. ಈ ಹಣ್ಣನ್ನು ನೇರವಾಗಿ ತಿನ್ನಬಹುದು. ಇಲ್ಲವಾದಲ್ಲಿ ಚಟ್ನಿ, ಉಪ್ಪಿನಕಾಯಿ, ಕಂಬಳಿ ರೂಪದಲ್ಲಿಯೂ ಸೇವಿಸಬಹುದು. ಬಿಳಿ ಮುಳ್ಳನ್ನು, ಜಿಜಿಫಿಸ್ ರುಗೋಸಾ, ಮುಳ್ಳನ್ನು ಹೊಂದಿರುವ ಸುಮಾರು 5 ರಿಂದ 8 ಮೀಟರ್ ಎತ್ತರ ಇದು ಬೆಳೆಯುತ್ತದೆ. ಈ ಸಸ್ಯದ ಕುಡಿಗಳನ್ನು ನೀರಿನಲ್ಲಿ ಕಿವುಚಿದರೆ ಮಂದವಾದ ಲೋಳೆಯಂತ ದ್ರವ ದೊರೆಯುತ್ತದೆ. ಈ ದ್ರವವನ್ನು ಬೇಸಿಗೆ ಕಾಲದಲ್ಲಿ ಆಯಾಸವನ್ನು ಪರಿಹರಿಸಲು ಸೇವಿಸಬಹುದು. ಇದರಿಂದ ಕಣ್ಣು ಉರಿ, ನೆತ್ತಿ ಉರಿ, ಪಾದದ ಉರಿ ನಿವಾರಣೆ ಆಗುತ್ತದೆ. ಪರಿಗೆ ಹಣ್ಣು, ಇದು ಕೂಡ ಮುಳ್ಳಿನಿಂದ ಕೂಡಿದ ಪೊದೆಯಂತ ಸಸ್ಯವಾಗಿದೆ. ಇದು ಅಕ್ಟೋಬರ್ ತಿಂಗಳಿನಲ್ಲಿ ಹೂ ಬಿಡುತ್ತದೆ ಹಾಗೂ ಮಾರ್ಚ್ ಏಪ್ರಿಲ್ ತಿಂಗಳಲ್ಲಿ ಹಣ್ಣಾಗುತ್ತದೆ. ಇದರಲ್ಲಿ ಸಿ ಜೀವಸತ್ವ ಇದ್ದು ನಾರಿನ ಅಂಶ ಹೆಚ್ಚಿರುತ್ತದೆ. ಕುಸುಮಾಲೆ ಸಸ್ಯ, ಕುಸುಮಾಲೆಯನ್ನು ಕೇಸ್ಕಾರು, ಹೊಳೆ ದಾಸವಾಳ ಮುಂತಾದ ಹೆಸರಿನಿಂದ ಇದನ್ನು ಕರೆಯಲಾಗುತ್ತದೆ. ಮಲೆನಾಡು ಮತ್ತು ಕರಾವಳಿಯ ಬೆಟ್ಟ ಕಾಡಿನಲ್ಲಿ ಇದು ಕಂಡು ಬರುತ್ತದೆ. ಹಳದಿ ಮತ್ತು ಕೆಂಪು ಬಣ್ಣದ ಹೂಗಳನ್ನು ಬಿಡುವ 2 ಜಾತಿಯ ಸಸ್ಯಗಳಿವೆ. ಈ ಹೂಗಳು ಗೊಂಚಲಾಗಿ ಬೆಳೆದುಕೊಂಡಿರುತ್ತದೆ. ಇದನ್ನು ನೇರವಾಗಿ ತಿನ್ನಬಹುದು. ಇಲ್ಲವಾದಲ್ಲಿ ಕಂಬಳಿ, ಚಟ್ನಿ, ಸಾಂಬಾರ್ ರೂಪದಲ್ಲಿಯೂ ಸೇವಿಸಬಹುದು. ಇದರ ಉಪಯುಕ್ತ ಗುಣಗಳೇನೆಂದರೆ ಉರಿ ಮೂತ್ರ ನಿವಾರಣೆ ಮಾಡುತ್ತದೆ. ನೆರಳು ಸಸ್ಯ, ಸೈಸಿಯಂ ಕ್ಯುಮಿನಿ, ಇದೊಂದು ನಿತ್ಯ ಹರಿದ್ವರ್ಣ ಸಸ್ಯವಾಗಿದ್ದು ಇದರಲ್ಲಿ ನಾಯಿ ನೇರಳೆ ಹಾಗೂ ಜಂಬು ನೇರಳೆ ಎಂಬ 2 ವಿಧಗಳಿವೆ. ನೇರಳೆ ಹಣ್ಣಿನ ಜ್ಯೂಸ್ ಮಕ್ಕಳ ಹೊಟ್ಟೆ ನೋವನ್ನು ನಿವಾರಣೆ ಮಾಡುತ್ತದೆ ಹಾಗೂ ನೇರಳೆ ಬೀಜವನ್ನು ಒಣಗಿಸಿ ಮೆಂತ್ಯ ಜೊತೆ ಬೆರೆಸಿ ಪೌಡರ್ ಮಾಡಿ ಬೇಕಾದ ಸಮಯದಲ್ಲಿ ಬಳಸಬಹುದು. ನೇರಳೆ ಎಲೆಯ ಕಷಾಯ, ಮಣ್ಣಿನ ಮಡಿಕೆಯಲ್ಲಿ 1/2 ಲೀಟರ್ ನೀರಿಗೆ 4 ರಿಂದ 6 ನೇರಳೆ ಎಲೆಯನ್ನು ಹಾಕಿ ಹಸಿಶುಂಠಿ, ಕಾಳುಮೆಣಸಿನೊಂದಿಗೆ ಇದನ್ನು 8 ರಿಂದ 10 ನಿಮಿಷ ಕುದಿಸಿ ಕಷಾಯ ರೂಪದಲ್ಲಿ ಸೇವಿಸಬಹುದು. ಇದರಿಂದ ಸಿಹಿಮೂತ್ರ ರೋಗ, ಬಿಪಿ, ಸ್ಥೂಲತೆ ಮುಂತಾದ ರೋಗಗಳು ನಿವಾರಣೆ ಆಗುತ್ತದೆ. ಅಗಸೆ ಗಿಡ, ಕ್ಯಾಸಿಯಾ ತೋರಾ, ರಸ್ತೆ ಬದಿಗಳಲ್ಲಿ ಗುಂಪು ಗುಂಪಾಗಿ ಬೆಳೆದುಕೊಂಡಿರುವ ಸಸ್ಯ ಇದಾಗಿದೆ. ಇದರ ಎಲೆಗಳನ್ನು ಪಲ್ಯ, ಚಟ್ನಿ, ಕಂಬಳಿ ರೂಪದಲ್ಲಿಯೂ ಸೇವಿಸಲಾಗುತ್ತದೆ. ಇದರ ಉಪಯುಕ್ತ ಗುಣಗಳೇನೆಂದರೆ ಚರ್ಮ ರೋಗಗಳಿಗೆ ಇದು ರಾಮಬಾಣ. ರಿಂಗ್ವರ್ಮ್, ಎಕ್ಸಿಮ್, ರಕ್ತದೋಷ, ಕುರುಬಾಧೆ, ಮೊಡವೆಗಳು ಉಪಚಾರಕ್ಕೆ ಇದರ ಔಷಧಿಗಳು ಬಹು ಪ್ರಸಿದ್ಧ. ಕನ್ನೆಕುಡಿ, ಇದರ ಎಳೆಯ ಸೊಪ್ಪುಗಳನ್ನು ನೀರಿನಲ್ಲಿ ತೊಳೆದು ತುಪ್ಪದಲ್ಲಿ ಮೃದುಟ್ಟುಕೊಳ್ಳಬೇಕು. 4 ಚಮಚ ಜೀರಿಗೆ, 5 ರಿಂದ 6 ಕಾಳುಮೆಣಸು, ಕಾಯಿ ತುರಿಯೊಂದಿಗೆ ರುಬ್ಬಿ ಹುಳಿ ಮಜ್ಜಿಗೆಗೆ ಸೇರಿಸಿ ರುಚಿಗೆ ತಕ್ಕಷ್ಟು ಉಪ್ಪನ್ನು ಸೇರಿಸಿ ಇದನ್ನು ಸೇವಿಸಬೇಕು. ಇದರಿಂದ ದೇಹದ ಉಷ್ಣ ನಿಯಂತ್ರಿಸುತ್ತದೆ ಹಾಗೂ ಉಷ್ಣದಿಂದ ಬರುವ ಹೊಟ್ಟೆ ನೋವು ಸಹ ನಿವಾರಣೆ ಆಗುತ್ತದೆ. ಹತ್ತಿ ಚಕ್ಕೆ, ಫೈಕಸ್ ಸೆಸಿಮೋಸಾ, ಇದೊಂದು ಮರವಾಗಿದ್ದು ಇದರ ಚಕ್ಕೆಯನ್ನು ತೆಗೆದುಕೊಂಡು ಹಸಿ ಶುಂಠಿ, ಕಾಳುಮೆಣಸು, ಜೀರಿಗೆಯೊಂದಿಗೆ ಜಜ್ಜಿ ಈ ಮಿಶ್ರಣವನ್ನು ಕುದಿಸಬೇಕು. ನಂತರ ಇದಕ್ಕೆ ಹಾಲು ಸೇರಿಸಿ ಟೀ ರೂಪದಲ್ಲಿ ಇದನ್ನು ಸೇವಿಸಬಹುದು. ಮಾವಿನ ಹಣ್ಣು, ಮ್ಯಾಂಜಿಫೇರಾ ಇಂಡಿಕಾ, ಇದು 10 ರಿಂದ 15 ಮೀಟರ್ ಎತ್ತರದಲ್ಲಿ ಬೆಳೆಯುತ್ತದೆ. ಕರ್ನಾಟಕದ ದಕ್ಷಿಣದ ಎಲ್ಲಾ ಭಾಗಗಳು ಹಾಗೂ ಮಲೆನಾಡು ಮತ್ತು ಕರಾವಳಿಯಲ್ಲಿ ಇದು ಕಂಡು ಬರುತ್ತದೆ. ಇದರ ಎಲೆ, ಮಿಡಿ, ಕಾಯಿ, ಹಣ್ಣು ಎಲ್ಲವೂ ಖಾದ್ಯ ಭಾಗಗಳಾಗಿವೆ. ಮಾವಿನಕಾಯಿಯನ್ನು ಚಿತ್ರಾನ್ನ, ಅಪ್ಪೆ ಹುಳಿ, ಕಂಬಳಿ, ಚಟ್ನಿ, ಸಾಂಬಾರ್ ರೂಪದಲ್ಲಿಯೂ ಸೇವಿಸಲಾಗುತ್ತದೆ. ಇದರ ಮಾವಿನ ಎಲೆಯನ್ನು ತೋರಣ ಮಾಡಲು ಸಹ ಬಳಸುತ್ತಾರೆ. ಇದರ ಉಪಯುಕ್ತ ಗುಣಗಳೇನೆಂದರೆ ನಮ್ಮ ದೇಹದ ಜೀರ್ಣಶಕ್ತಿಯನ್ನು ಹೆಚ್ಚಿಸುತ್ತದೆ ಹಾಗೂ ಇದರಲ್ಲಿ ಸಿ ಜೀವಸತ್ವ ಹೇರಳವಾಗಿರುತ್ತದೆ. ಹಾಲೆಬಳ್ಳಿ, ಸೈಕ್ಲಿಯಾ ಪೆಲ್ಟಾ, ಇದೊಂದು ಬಳ್ಳಿಯಾಗಿದ್ದು ಮರಗಿಡ ಆಶ್ರಯಿಸಿ ಬೆಳೆಯುತ್ತದೆ. ಇದರ ಔಷಧಿಗೆ ಉಪಯುಕ್ತ ಭಾಗಗಳು ಎಲೆ, ಕಾಂಡ, ಬೇರು, ಎಲೆ, ಕಾಯಿ, ಬೇರು. ಇದು ಪಿತ್ತ, ಹೊಟ್ಟೆ ನೋವು, ಚಿಕ್ಕ ಮಕ್ಕಳಲ್ಲಿ ಜಂತು, ತಲೆಯುರಿ, ಕಣ್ಣು ಉರಿ ನಿವಾರಣೆಗೆ ಬಳಸುತ್ತಾರೆ. ಚಿಕ್ಕ ಮಕ್ಕಳಿಗೆ ಅಥವಾ ದೊಡ್ಡವರಿಗೆ ಹೊಟ್ಟೆ ನೋವು ಆರಂಭವಾದಾಗ ಇದರ ಹಳೆಬಳ್ಳಿಯ ಬೇರನ್ನು ಹುಳಿ ಮಜ್ಜಿಗೆಯಲ್ಲಿ ತೆಗೆದು ಚಿಕ್ಕ ಮಕ್ಕಳಿಗೆ 1 ಚಮಚ, ದೊಡ್ಡವರಿಗೆ 2 ಚಮಚ 3 ದಿನ ಬೆಳಿಗ್ಗೆ ಖಾಲಿ ಹೊಟ್ಟೆಯಲ್ಲಿ ಸೇವಿಸುವುದರಿಂದ ಹೊಟ್ಟೆ ನೋವು ನಿವಾರಣೆ ಆಗುತ್ತದೆ. ಮುರುಗಲ ವ್ಯಾರ್ಜಿನಿಯಾ ಇಂಡಿಕಾ ಸುಮಾರು 4 ರಿಂದ 5 ಮೀಟರ್ ಎತ್ತರ ಇದು ಬೆಳೆಯುತ್ತದೆ. ಡಿಸೆಂಬರ್ನಲ್ಲಿ ಹೂ ಬಿಟ್ಟು ಮಾರ್ಚ್ ಏಪ್ರಿಲ್ ತಿಂಗಳಲ್ಲಿ ಇದು ಹಣ್ಣಾಗುತ್ತದೆ. ಇದರ ಉಪಯುಕ್ತ ಭಾಗ ಹಣ್ಣು. ರಾತ್ರಿ 1 ಲೋಟ ನೀರಿಗೆ 4 ರಿಂದ 5 ಮುರುಗಲ ಸಿಪ್ಪೆ ಹಾಕಿ ಬೆಳಿಗ್ಗೆ ಖಾಲಿ ಹೊಟ್ಟೆಯಲ್ಲಿ ಬೆಲ್ಲ ಸೇರಿಸಿ ಇದನ್ನು ಸೇವಿಸಬೇಕು. ಹಣ್ಣಿನ ರಸದಿಂದ ಕೋಕಂ ತಯಾರಿಸುತ್ತಾರೆ. ಬೇಸಿಗೆಯಲ್ಲಿ ದಣಿವಾರಿಸಲು ಪೇಯವಾಗಿದೆ. ಇದರ ಪ್ರಯೋಜನ ಪಿತ್ತ ವಿಕಾರಗಳು ನಿವಾರಣೆಯಾಗುತ್ತದೆ. ಈಚಲು ಫಯೋನಿಕ್ಸ್ ಸಿಲ್ವೆಸ್ಟ್ರಿಸ್ ಹಕ್ಕಿ ಪಕ್ಷಿಗಳಿಂದ ಬೀಜ ಪ್ರಸಾರವಾಗಿ ಬೆಟ್ಟ ಗುಡ್ಡಗಳಲ್ಲಿ ತಾನಾಗಿ ಬೆಳೆದುಕೊಳ್ಳುವ ಸಸ್ಯ ಇದಾಗಿದೆ. ಇದು ಹಸಿರು ಬಣ್ಣದ ಕಾಯಿ ಹಳದಿ ಬಣ್ಣಕ್ಕೆ ತಿರುಗಿ ಕಾಫಿ ಬಣ್ಣದಲ್ಲಿ ಇದು ಹಣ್ಣಾಗುತ್ತದೆ. ಇದರಲ್ಲಿ ಪ್ರೋಟೀನ್, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್, ಫಾಸ್ಫರಸ್, ವಿಟಮಿನ್ ಸಿ, ಕ್ಯಾಲ್ಸಿಯಂ ಇರುತ್ತದೆ. ಕಬ್ಬಿಣಾಂಶ ಇರುವುದರಿಂದ ರಕ್ತಹೀನತೆಗೆ ಇದು ಔಷಧಿಯಾಗಿದೆ. ಇದರಿಂದ ನೀರ, ವೈನ್, ಜೆಲ್ಲಿಯನ್ನು ಸಹ ತಯಾರಿಸಲಾಗುತ್ತದೆ. ಕಳಲೆ ಬ್ಯಾಂಬೋಸ್ ಅರುಣಿನೇಸಿಯಾ ಬಿದುರಿನ ಚಿಗುರು ಅಥವಾ ಬಿದುರಿನ ಮೊಗ್ಗುಗಳಲ್ಲಿ ಬಿದುರಿನ ಜಾತಿಯ ತಿನ್ನಬಹುದಾದಂತಹ ಕುಡಿಗಳಾಗಿವೆ. ಮಳೆಗಾಲದಲ್ಲಿ ಮಲೆನಾಡು ಹಾಗೂ ಕರಾವಳಿ ಪ್ರದೇಶದಲ್ಲಿ ಇದು ಕಂಡು ಬರುತ್ತದೆ. ಕಳಲೆಯನ್ನು ಚಿಕ್ಕದಾಗಿ ತುಂಡರಿಸಿ 3 ದಿನ ನೀರಿನಲ್ಲಿ ನೆನೆಸಿಡಬೇಕು. ನಂತರ ಆ ನೀರನ್ನು ಹೊರಚೆಲ್ಲಿ ಆ ಕಳಲೆಯನ್ನು ಬೆಳೆಸಬಹುದು. ಇದರಿಂದ ಸಾಂಬಾರು, ಹುಳಿ, ಪಲ್ಯ, ಉಪ್ಪಿನಕಾಯಿ ಮುಂತಾದ ಖಾದ್ಯಗಳನ್ನು ತಯಾರಿಸಲಾಗುತ್ತದೆ. ಮಳೆಗಾಲದಲ್ಲಿ ದೇಹದ ಉಷ್ಣವನ್ನು ಕಾಪಾಡುತ್ತದೆ.
ಹಲಸಿನ ಹಣ್ಣು. ಇದರ ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಆರ್ಟೋಕಾರ್ಪಸ್ ಹೆಟೆರೋಫಿಲ್ಲಸ್. ಇದರ ಕಾಯಿ 5 ಕೆಜಿ 40 ಕೆಜಿ ಇರುತ್ತೆ. ಈ ಹಣ್ಣಿನ ಒಳಗೆ ತುಂಬಾ ಸೊಳೆಗಳಿರುತ್ತೆ. ತುಂಬಾ ಸಿಹಿಯಾಗಿರುತ್ತೆ. ಹಾಗೆ ಈ ಹಲಸಿನ ಹಣ್ಣಿನಿಂದ ಸಾಂಬಾರ್ ಮಾಡ್ತಾರೆ, ಹಾಗೆಯೇ ಪಲ್ಯ ಮಾಡ್ತಾರೆ, ಚಟ್ನಿ ಮಾಡ್ತಾರೆ, ಪಾಯಸ ಮಾಡ್ತಾರೆ, ಜಾಮ್ ಮಾಡ್ತಾರೆ, ಜೆಲ್ಲಿ ಕೂಡ ಮಾಡ್ತಾರೆ. ಹಾಗೆ ಈ ಹಲಸಿನ ಕಾಯಿಯಿಂದ ಚಿಪ್ಸ್ ಕೂಡ ಮಾಡ್ತಾರೆ. ಹಾಗೆ ಈ ಹಲಸಿನ ಬೀಜದಿಂದ ಪಲ್ಯ ಮಾಡ್ತಾರೆ, ಸಾಂಬಾರ್ ಮಾಡ್ತಾರೆ, ಹೋಳಿಗೆ ಕೂಡ ಮಾಡ್ತಾರೆ. ಹಾಗೆ ಇದರ ಉಪಯುಕ್ತ ಗುಣ ಹೇಳ್ಬೇಕಂದ್ರೆ ವಿಟಮಿನ್ ಸಿ ಹೆಚ್ಚಾಗಿರೋದ್ರಿಂದ ವೈರಸ್ ಆಗಿರ್ಬೋದು, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಆಗಿರ್ಬೋದು ಅದು ನಮ್ಮಿಂದ ದೂರ ಇರುತ್ತೆ. ಹಾಗೆ ಗೇರು. ಇದರ ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಅನಕಾರಿಯಂ ಅಸಿಡೆಂಟಲ್. ಗೋಡಂಬಿ ಬೀಜ ಇದು ಜನಪ್ರಿಯ ಕುರುಕುಲ ತಿಂಡಿ ಆಗಿದೆ. ಇದನ್ನು ಸಿಹಿ ತಿಂಡಿಗಾಗಿ ಅಥವಾ ಸುಕ್ಕಗಳಲ್ಲೂ ಕೂಡ ಬಳಸುತ್ತೇವೆ. ಇದರಲ್ಲೂ ಕೂಡ ಕಡಿಮೆ ಕೊಬ್ಬಿನಾಂಶ ಕೂಡ ಹೊಂದಿದ್ದು ಜೀವಸತ್ವಗಳಾದ B2, B3, ಮ್ಯಾಗ್ನೀಷಿಯಂ ಅಂಶ ಹೆಚ್ಚಾಗಿರೋದ್ರಿಂದ ತಲೆ ಕೂದಲು ಕಪ್ಪಾಗ್ಲಿಕ್ಕೆ ಅಥವಾ ತಲೆ ಕೂದಲು ಕೂಡ ಗಟ್ಟಿಯಾಗ್ಲಿಕ್ಕೆ ಇದು ಕೂಡ ಸಹಕಾರಿಯಾಗಿದೆ. ಅಲ್ಲದೆ ಮುಖದ ಹೊಳಪನ್ನು ವೃದ್ಧಿಸಲು, ತೂಕ ಹೆಚ್ಚಿಸಲು, ನೆನಪಿನ ಶಕ್ತಿ ಕೂಡ ಹೆಚ್ಚಿಸಲು ಇದು ಒಂದು ಸಹಕಾರಿಯಾಗಿದೆ. ಹಾಗೆ ಹುಣಸೆ ಮರ. ಇದರ ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಟ್ಯಾಮರಿಂಥಸ್ ಇಂಡಿಕಾ. ಹುಣಸೆ ಮರ ಇದು 25 ಅಂತ 30 ಮೀಟರ್ ಎತ್ತರದಲ್ಲಿ ಬೆಳೆಯುತ್ತದೆ. ಇದರಿಂದ ಪಾನಕ, ಸಾರು ಕೂಡ ಮಾಡ್ತಾರೆ. ಹಾಗೆ ಔಷಧಿ ಗುಣ ಇರ್ಬೇಕಂದ್ರೆ ಈ ಹಣ್ಣಿನ ರಸದಿಂದ ಗಂಟಲಿನ ನೋವಾಗಿರ್ಬೋದು, ಮಲಬದ್ಧತೆ ಆಗಿರ್ಬೋದು ಅದು ಕೂಡ ನಿವಾರಣೆ ಮಾಡುತ್ತೆ. ಹಾಗೆ ಈ ಎಲೆಯ ರಸದಿಂದ ಉರಿಮೂತ್ರ ಆಗಿರ್ಬೋದು, ಜ್ವರ ಕೂಡ ನಿವಾರಣೆ ಮಾಡುತ್ತೆ. ಹಾಗೆ ಬೊಗರೆ ಹಣ್ಣು. ಇದರ ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಇಂಡಿಯನ್ ಜುಜುಬೆ. ಇದು ಮುಳ್ಳಿನಲ್ಲಿ ಬೆಳೆಯುವ ಪೊದೆಯಂತ ಸಸ್ಯ ಇದಾಗಿದೆ. ಇದು ಬೇಸಿಗೆ ಕಾಲದಲ್ಲಿ ಹೂ ಬಿಟ್ಟು ಕಾಯಿ ಆಗ್ತದೆ. ಇದು ಕಾಯಿ ಹಸಿರಾಗಿದ್ದು ಹಣ್ಣಾದಾಗ ಕೆಂಪು ಬಣ್ಣದಲ್ಲಿ ಬರುತ್ತೆ. ಇದರ ರುಚಿ ಹುಳಿ ಒಗರು ಹೊಂದಿದೆ. ಹಾಗೆ ಇದರ ಸಿಹಿ ಜ್ಯೂಸ್ ಕೂಡ ಹೇಳಲಾಗಿದೆ.
ನಮ್ಮ ಊರಿನಲ್ಲಿ ವನ್ಯ ಸಸ್ಯಗಳ ಅಧ್ಯಯನದಿಂದ ನಮಗೆ ತಿಳಿದು ಬಂದದ್ದೇನೆಂದರೆ ಕಚ್ಚಾ ಹಣ್ಣನ್ನು ಸೇವಿಸುವ ಸಸ್ಯ 40% ಹಾಗೂ ಸಂಸ್ಕರಿಸಿ ಸೇವಿಸುವ ಆಹಾರ ಸಸ್ಯ 60%. ಇನ್ನು ಸ್ವರೂಪದ ಆಧಾರದ ಮೇಲೆ ಶೇಕಡ ಪ್ರಮಾಣ ಗಿಡಗಳು 10%, ಮರಗಳು 40% ಹಾಗೂ ಪೊದೆಗಳು 50%ಗಳಾಗಿ ವರ್ಗೀಕರಿಸಲಾಗಿದೆ. ಕಾಡು ಹಣ್ಣುಗಳ ಸವಿ ಬಲ್ಲವರೇ ಬಲ್ಲವರು. ವಿಷರಹಿತವಾದ ನೈಸರ್ಗಿಕವಾದ ಈ ಹಣ್ಣುಗಳು ಆರೋಗ್ಯಕ್ಕೂ ಒಳ್ಳೆಯದು. ನಮ್ಮ ಸುತ್ತಮುತ್ತಲಿನಲ್ಲಿ ಕಾಡುಗಳಲ್ಲಿ ಇಂತಹ ಕಾಡು ಹಣ್ಣುಗಳನ್ನು ಪರಿಣಾಮಕಾರಿಯಾಗಿ ಸುಸ್ಥಿರವಾಗಿ ಬಳಸಿಕೊಂಡರೆ ನಾವು ವೈದ್ಯರ ಬಳಿಗೆ ಹೋಗದೆ ನಮ್ಮ ಆರೋಗ್ಯವನ್ನು ನಾವೇ ಕಾಪಾಡಿಕೊಳ್ಳಬಹುದು. ನಮ್ಮ ಊರಿನಲ್ಲಿ ಇರುವ ನಮಗೆ ಗೊತ್ತಿರದ ಎಷ್ಟೋ ಕಾಡು ಹಣ್ಣುಗಳ ಸಸ್ಯಗಳ ಪರಿಚಯ ನಮಗಾಯಿತು ಹಾಗೂ ನಾವು ಅದರ ಉಪಯೋಗಗಳನ್ನು ತಿಳಿದುಕೊಂಡೆವು. ಈ ಯೋಜನೆಯನ್ನು ಪ್ರಸ್ತುತಪಡಿಸಲು ಅವಕಾಶ ನೀಡಿದ ಭಾರತೀಯ ವಿಜ್ಞಾನ ಸಂಸ್ಥೆಗೆ ಹಾಗೂ ನನಗೆ ಮಾರ್ಗದರ್ಶನ ನೀಡಿದ ನನ್ನ ಗುರುಗಳಿಗೆ ಕೃತಜ್ಞತೆಗಳು ಹಾಗೂ ನನಗೆ ಗೊತ್ತಿಲ್ಲದ ಎಷ್ಟೋ ಕಾಡು ಸಸ್ಯಗಳ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಿದ ನನ್ನ ಊರ ನಾಗರಿಕರಿಗೂ ಸಹ ಧನ್ಯವಾದಗಳು.
ಲೈವ್ ಆಗಿ ತಂದಿದ್ದೀನಿ ತೋರಿಸ್ತೀನಿ. ಗಿಡ ಇದು ಈಚಲು, ಇದು ಈಚಲಿನ ಎಲೆ, ಇದು ಈಚಲಿನ ಎಲೆ, ಇದು ಕುಸುಮಾಲಿ ಸಸ್ಯದ ಎಲೆ, ಇದು ಮುಳ್ಳು ಹಣ್ಣು, ಇದು ಹಸಿರು ಬಣ್ಣದಲ್ಲಿರುತ್ತೆ, ನಂತರ ಇದು ಹಣ್ಣಾದಾಗ ಕಪ್ಪು ಬಣ್ಣಕ್ಕೆ ತೆರಳುತ್ತದೆ. ಇದು ಗೇರುಬೀಜ ಸಸ್ಯ, ಇದು ನೆಲ್ಲಿಕಾಯಿ ಗಿಡ, ಇದು ನೆಲ್ಲಿಕಾಯಿ ಗಿಡ, ಇದು ಕೆಸುವಿನ ಸಸ್ಯ, ಇದು ನೇರಳೆ ಸಸ್ಯ, ಇದು ಮುರುಗಿಲಿನ ಸಸ್ಯ, ಇದು ಮೊಟ್ಟೆ ಕೆಸ, ಇದು ಈಚಲಿನ ಕಾಯಿಗಳು, ಇದು ಬೊಗರೆ ಹಣ್ಣು, ಇದು ಮುಳ್ಳಹಣ್ಣು, ಇದು ಗೋಡಂಬಿ, ಇದು ಕೊಸೆಕಾಯಿ. ಇದನ್ನ ಉಪ್ಪು ನೀರಲ್ಲಿ ನೆನೆಸಿಡ್ಬೇಕು. ಇದು ಸುಮಾರು ವರ್ಷದ ತನಕ ಬರುತ್ತೆ. ಇದು ನಾವು ಮುರುಗಲನ್ನ ಬಿಸಿಲಲ್ಲಿ ಒಣಗಿಸಿಡ್ಬೇಕು. ಇದನ್ನು ಸಾಂಬಾರ್ಗಳಲ್ಲಿ ಸಹ ಬಳಸಲಾಗುತ್ತೆ. ಇದನ್ನ ನೆಲ್ಲಿ ಚೆಟ್ಟು ಎಂದು ಕರೆಯುತ್ತೇವೆ.
One point I want to make here. What all she showed just now, this specimen, they have collected it from the forest right in the forest area. Okay, it is not available anywhere else. ಅದು ಕಾರಣವಾಗಿ ಕಲೆಕ್ಟ್ ಮಾಡಿ ಅದು ಯಾವುದು ಅಂತ ಗುರುತಿಸಿ ಅದನ್ನ ಐಡೆಂಟಿಫೈ ಮಾಡಿ ಲೇಬಲ್ ಮಾಡಿ ತಂದಿದ್ದಾರೆ ಮಕ್ಕಳು. ಪ್ರಶ್ನೆ ಇದೆಯಾ ಯಾರ್ದಾದ್ರು? We take one question from the students. Huh? Thank you. Thank you.
Yeah, that I think poster is there. Yes. What about poster? ಇದು ಇನ್ನೊಂದು ಟೀಚರ್ಸ್ ಅದಲ್ಲ. ವೇಟ್ ನನಗೆ ಸ್ವಲ್ಪ ನೀಡಿ ಇಲ್ಲ ಕೇಳ. ಹಾ ಅವನಿಗೆ ಅಲ್ಲೇ ಅಲ್ಲೇ ಅಲ್ಲ ಸರ್ ಇಲ್ಲ ಅದು ಅವ್ರ ಲ್ಯಾಪ್ಟಾಪ್ ಅಲ್ಲಿ ಇರಬೇಕು, ನಾವು ಬೇರೆ ಲ್ಯಾಪ್ಟಾಪ್ ಇರಬೇಕು. ಸಿಟ್ಸ್ ಇದೊಂದೇ ಇರೋದಲ್ಲ, ಅದೊಂದೇ, ಅದು ಓಪನ್ ಆಗ್ತಿಲ್ಲ, ಅದು ಕನ್ನಡದಲ್ಲಿ ಇರೋದಿಲ್ಲಲ್ಲ, ಅದಕ್ಕೆ ಇವಾಗ ಇದು ಚೇಂಜ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. ಅದಕ್ಕೆ ಅವ್ರು ಕೊಟ್ಟಿದ್ದಾರೆ, ನೋಡ್ತೀನಿ ತೊಗೊಳ್ಳಿ. ಯಾವ ಸ್ಕೂಲು? ಹಾಂ
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು ರುಚಿತಾ ಬಾಬು ದೇಶಪಾಂಡೆ. ನಾನು ಉತ್ತರ ಕನ್ನಡ ಜಿಲ್ಲೆಯ ಹೊನ್ನಾವರ ತಾಲೂಕಿನ ಸರ್ಕಾರಿ ಪ್ರೌಢಶಾಲೆ ಕರ್ಕೇಶ್ವರಹಳ್ಳಿಯಲ್ಲಿ 9ನೇ ತರಗತಿಯಲ್ಲಿ ವ್ಯಾಸಂಗ ನಡೆಸುತ್ತಿದ್ದೇನೆ. ಪ್ರತಿ 2 ವರ್ಷಕ್ಕೊಮ್ಮೆ ಬರುವ ಇಂಡಿಯನ್ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಫ್ ಸೈನ್ಸ್ ಅವರ ವತಿಯಿಂದ ನಡೆಯುವ ಹಳ್ಳಿ ಆಹಾರ ಹಾಗೂ ಔಷಧಿ ಸಸ್ಯದ 2 ಥೀಮ್ಗಳಲ್ಲಿ ಇಂದು ನಾನು ಹಳ್ಳಿ ಆಹಾರ ಮತ್ತು ಔಷಧಿ ಸಸ್ಯಗಳ ಕುರಿತು ಪರಿಚಯವನ್ನು ನೀಡಲಿದ್ದೇನೆ. ನಮ್ಮ ದೇಶದಲ್ಲಿರುವ ಪ್ರತಿಯೊಂದು ಗಿಡವು ತನ್ನದೇ ಆದ ಔಷಧಿ ಗುಣವನ್ನು ಹೊಂದಿರುತ್ತದೆ. ಆದರೆ ಈ ವಿಷಯ ಮನುಷ್ಯನಿಗೆ ಮಾತ್ರ ತಿಳಿದಿರುವುದಿಲ್ಲ. ಒಮ್ಮೆ ಮನುಷ್ಯನ ದೇಹದಲ್ಲಿ ಯಾವುದಾದರೂ ಕಾಯಿಲೆಗಳು ಸಂಭವಿಸಿದ್ದಲ್ಲಿ ಮನುಷ್ಯ ನೇರವಾಗಿ ಹೋಗುವುದು ಆಸ್ಪತ್ರೆಗೆ ಹೋದ ನಂತರ ಮಾತ್ರೆಗಳನ್ನು ತಿಂದು 1-2 ದಿನಗಳ ಕಾಲ ತನ್ನ ಆರೋಗ್ಯವನ್ನು ಸರಿಯಾಗಿರಿಸಿಕೊಳ್ಳುತ್ತಾನೆ ಮತ್ತು 2 ದಿನಗಳ ನಂತರ ಮತ್ತೆ ಆರೋಗ್ಯದಲ್ಲಿ ಏರುಪೇರು ಕಾಣಿಸಿಕೊಳ್ಳುವುದು ಸಾಮಾನ್ಯ. ಆದರೆ ಔಷಧಿ ಗಿಡಗಳ ಶಕ್ತಿ ಏನೆಂದರೆ, ಔಷಧಿ ಗಿಡಗಳಿಂದ ತಯಾರಿಸಿದ ಔಷಧಿಯನ್ನ 5 ದಿನಗಳ ಕಾಲ ಹಾಗೂ 7 ದಿನಗಳ ಕಾಲ ಸೇವಿಸಿದರೆ ಯಾವುದೇ ರೀತಿಯ ಕಾಯಿಲೆಗಳು ಮತ್ತೆ ಮಾನವನ ದೇಹದಲ್ಲಿ ಬರುವುದಿಲ್ಲ, ಮತ್ತೆ ಆ ಕಾಯಿಲೆಗಳು ಮುಂದುವರಿಯುವುದಿಲ್ಲ. ಇದು ಔಷಧಿ ಗಿಡಗಳ ಪ್ರಮುಖ ಶಕ್ತಿ. ಈಗ ನಾನು ಮೊದಲನೆಯದಾಗಿ ಮಾಲಿಬಂದಿ ಗಿಡ. ಈ ಮಾಲಿಬಂದಿ ಗಿಡದ ನಾವು ಬೇರು ಹಾಗೂ ಕಾಂಡ ಮತ್ತು ಇದಕ್ಕಾಗುವ ಹೂಗಳನ್ನು ಸಹ ಬಳಕೆ ಮಾಡುತ್ತೇವೆ. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಲಿಟ್ಸಿಯಾ ಘಟಿಕಾ. ಇದರ ಮಹತ್ವ, ಮಾನವನ ದೇಹದಲ್ಲಿ ಎಲ್ಲಾದರೂ ಮೂಳೆ ಮುರಿದಲ್ಲಿ 5 ದಿನಗಳಿಂದ 7 ದಿನಗಳ ಕಾಲ ನಿರಂತರವಾಗಿ 3 ದಿನ ಸೇವಿಸುವುದರಿಂದ ಮೊಟ್ಟೆಯಲ್ಲಿ ಅಥವಾ ತುಪ್ಪದಲ್ಲಿ ಹಾಕಿ ಇದನ್ನು ಸೇವಿಸುವುದು ಸೂಕ್ತ. ಏಕೆಂದರೆ ಮಾಡಿದ ಕೆಲಸ ಸರಿಯಾಗಿ ಪರಿಣಾಮ ಬೇಕಾದರೆ ನಮಗೆ ಬೇಕಾದ ವಸ್ತುಗಳನ್ನು ನಾವು ಬಳಸಿ ಔಷಧಿಯನ್ನು ತಯಾರಿಸಿಕೊಳ್ಳುವುದು ಸೂಕ್ತ. ಹಾಗಾಗಿ 7 ದಿನಗಳಲ್ಲಿ ಮೂಳೆಯು ಗಟ್ಟಿಯಾಗಿ ಸದೃಢತೆಯನ್ನು ಹೊಂದುತ್ತದೆ. ನನಗೆ ಇದನ್ನು ಸಂದರ್ಶಿಸಿದವರು ಶಾರದಾ ದೇಶಪಾಂಡೆ. 2ನೇದು ಅಂಡ್ಲಿ ಮರ. ಈ ಅಂಡ್ಲಿ ಮರದ ಸಣ್ಣ ಬೇರಿನ ತುಂಡು ಹಾಗೂ ಕಾಂಡವನ್ನು ನಾವು ಬಳಕೆ ಮಾಡಬಹುದು. ಇದರಿಂದ ಹೊಟ್ಟೆ ನೋವು ಹಾಗೂ ವಾಂತಿ ಭೇದಿಗೂ ಇದು ಪರಿಣಾಮವನ್ನು ಬೀರುತ್ತದೆ ಮತ್ತು ಮನುಷ್ಯನ ದೇಹದಲ್ಲಿ ಸುಮಾರು 50% ಸಕ್ಕರೆ ಕಾಯಿಲೆಯು ಸಹ ನಿವಾರಣೆಯಾಗುತ್ತದೆ. ಇದನ್ನು ನನಗೆ ಸಂದರ್ಶಿಸಿದವರು ಭಾಗೀರಥಿ ದೇಶಪಾಂಡೆ. 3ನೇದು ಬಿಳಿ ಕುಸುಮಲೆ ಗಿಡ. ಇದರ ಬಿಳಿ ಕುಸುಮಲೆ ಗಿಡಕ್ಕೆ ಆಗುವಂತಹ ಹಣ್ಣಿನ ಬೀಜಗಳು ಮತ್ತು ಬಿಳಿ ಕುಸುಮಲೆ ಗಿಡದ ಬೇರನ್ನು ನಾವು ಬಳಕೆ ಮಾಡಬಹುದು. ಮನುಷ್ಯನಲ್ಲಿ ಜ್ವರದಿಂದಾಗಲಿ ಅಥವಾ ಬೇರೆ ಯಾವುದೇ ಕಾಯಿಲೆಯಿಂದಾಗಲಿ ಮನುಷ್ಯ ಅಶಕ್ತತೆಗೆ ಒಳಗಾಗಿದ್ದಲ್ಲಿ ಇದನ್ನು ನೇರವಾಗಿ 1ರಿಂದ 3 ದಿನಗಳ ಕಾಲ ನಿರಂತರವಾಗಿ ನೀಡುವುದರಿಂದ ವ್ಯಕ್ತಿಯ ಅಶಕ್ತತೆಯು ಕಡಿಮೆಯಾಗುತ್ತದೆ ಹಾಗೂ ಹೃದಯ ಸಂಬಂಧಿ ಕಾಯಿಲೆಗಳು ಸಹ ನಿವಾರಣೆಯಾಗುತ್ತದೆ. ಸೋಮವಾರದ ಗಿಡ. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಕ್ರೋಟೋನಾ ಆಂಬ್ಲೋಬಿಫಲಸ್. ಇದರ ಅಕ್ಕಿ ತೊಳೆದ ನೀರು ಹಾಗೂ ಗಿಡದ ಬೇರು ಮತ್ತು ಕಾಂಡವನ್ನು ನಾವು ಬಳಕೆ ಮಾಡಿಕೊಳ್ಳಬಹುದು. ಇದರಿಂದ ತುರಿಕೆಯು ನಿವಾರಣೆಯಾಗುತ್ತದೆ. ಇನ್ನು ಔಪತ್ತಿ ಮರ. ಇದರ ಮರಕ್ಕೆ ಆಗುವಂತಹ ಚಕ್ಕೆ ಹಾಗೂ ಮರದ ಬೇರನ್ನು ನಾವು ಬಳಕೆ ಮಾಡಿಕೊಳ್ಳಬಹುದು. ಇದರಿಂದ ಮಾರಣಾಂತಿಕ ಕಾಯಿಲೆಯಾದ ಸಿಡುಬು ರೋಗವನ್ನು ನಾವು ಪರಿಹರಿಸಿಕೊಳ್ಳಬಹುದು. ವಾಯಂಗಳ ಬಳ್ಳಿ. ಈ ಬಳ್ಳಿಯ ಬೇರು ಹಾಗೂ ನಾವು ಕಾಂಡವನ್ನು ಬಳಕೆ ಮಾಡಿಕೊಳ್ಳಬಹುದು. ಇದು ಸಹ ಹೊಟ್ಟೆ ನೋವಿಗೆ ಪರಿಣಾಮವನ್ನು ಬೀರುತ್ತದೆ, ಅಂದರೆ ಮನುಷ್ಯನಲ್ಲಾದ ಹೊಟ್ಟೆ ನೋವು ನಿವಾರಣೆಯಾಗುತ್ತದೆ. ಇನ್ನು ಕಾಡು ಬಾಳೆ. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಮೂಸಾ ಎಕ್ಯುಮಿನಿಟಾ. ಇದರ ಕಾಡು ಬಾಳೆಯ ಬಾಳೆ ಗಿಡದ ಚರ್ಮ ಹಾಗೂ ಇದರ ಬೇರನ್ನು ಸಹ ಬಳಕೆ ಮಾಡಿಕೊಳ್ಳಬಹುದು. ಇದರಿಂದ
ಮನುಷ್ಯನ ದೇಹದಲ್ಲಿ ಉಷ್ಣತೆ ಹೆಚ್ಚಾಗಿದ್ದಲ್ಲಿ ದೇಹವು ತಂಪುಗೊಳ್ಳುವುದು ಸಾಮಾನ್ಯ. ಮುಟ್ಟಿದರೆ ಮುನಿಸಸ್ಯ ಅಂದರೆ ನಾಚಿಕೆ ಮುಳ್ಳು. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಮಿಮುಸಾ ಪುಡಿಕಾ. ಇದನ್ನು ನಾವು ಸಸ್ಯದ ಬೇರನ್ನು ನಾವು ಬಳಕೆ ಮಾಡಿಕೊಳ್ಳಬಹುದು. ಇದು ಮನುಷ್ಯನಲ್ಲಿ ಮೂಲವ್ಯಾಧಿ ರೋಗವು ಕಂಡು ಬಂದಲ್ಲಿ ಈ ಔಷಧಿಯನ್ನು ನಿರಂತರವಾಗಿ 7 ದಿನಗಳ ಕಾಲ ಬೆಳಿಗ್ಗೆ ಹಾಗೂ ಸಂಜೆ ನೀಡುವುದರಿಂದ ಮೂಲವ್ಯಾಧಿ ರೋಗವು ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಇನ್ನು ಉತ್ತರಾಣೆ ಗಿಡದ ಬೇರು ಹಾಗೂ ಕಾಂಡವನ್ನು ನಾವು ಬಳಕೆ ಮಾಡಿಕೊಳ್ಳಬಹುದು. ಇದರಿಂದ ಮೂತ್ರಾಂಗ ವ್ಯೂಹದ ಯಾವುದೇ ರೋಗಗಳು ನಿವಾರಣೆಯಾಗುತ್ತದೆ. ಇನ್ನು ಹಾಡಿ ಎಲೆ. ಈ ಗಿಡದ ಎಲೆಗಳು ಹಾಗೂ ಈ ಗಿಡವನ್ನು ಪೂರ್ತಿಯಾಗಿ ನಾವು ಬಳಕೆ ಮಾಡಿಕೊಳ್ಳಬಹುದು. ಇದರಿಂದ ತಯಾರಾಗುವಂತಹ ಲೋಳೆಯಂತಹ ಪದಾರ್ಥವನ್ನು 10 ರಿಂದ 15 ನಿಮಿಷಗಳ ಕಾಲ ತಲೆಯ ಮೇಲೆ ಇರಿಸಿಕೊಂಡರೆ ತಲೆನೋವು ನಿವಾರಣೆ ಆಗುತ್ತದೆ. ಒಟ್ಟಾರೆ ಇದರ ಪ್ರಮುಖ ಉದ್ದೇಶವೇನೆಂದರೆ ಪರಿಸರ ಸಂರಕ್ಷಣೆಯಿಂದಾಗುವ ಪ್ರಯೋಜನವನ್ನು ನಾವು ಪಡೆಯುವುದು. ಪ್ರತಿಯೊಂದು ಗಿಡಕ್ಕೂ ಸಹ ಔಷಧಿ ಗುಣವಿರುತ್ತದೆ ಎಂಬುದನ್ನು ತಿಳಿಯುವುದು. ಒಟ್ಟಾರೆ ಔಷಧಿ ಗಿಡಗಳಿಂದ ನಿರ್ಮಾಣವಾಗುವುದು ಆರೋಗ್ಯ, ನೆಮ್ಮದಿಯೇ ಮನುಷ್ಯನ ಜೀವನದ ಪ್ರಮುಖ ಧ್ಯೇಯ. ಆರೋಗ್ಯವೇ ಭಾಗ್ಯ ಎಂಬುದೇ ಔಷಧಿ ಗಿಡದ ಬಗ್ಗೆ ನನ್ನ ಒಂದು ಅಭಿಪ್ರಾಯ. ಧನ್ಯವಾದಗಳು.
ಯೂನಿಟ್ ಡೆವಲಪ್ಮೆಂಟ್ ಏನು ಪ್ರಶ್ನೆ ಇಲ್ಲ ಅಂತ ಹೇಳ್ಬೋದು. ಹಾಗಾಗಿ ವೆರಿ ಗುಡ್. After medicinal plants, I think most of you are feeling hungry now. You need some medicine for the stomach. Okay, you can leave your belongings here and you can go down now. We break up lunch. Half an hour. Just check the time. Time is now 1 o'clock. By 1:30, we have to be back. What I have found during the breakfast time in the morning, first you stand there in groups, start discussing. You meet some new people, you start talking to them. So finally you forget the time, right?
Layashri, are you able to hear us?
Yes, I'm able to hear.
Okay. Okay.
Just
Yeah.
a
While.
moment, sir.
Okay.
Yes, sir. So can I begin?
Layashri, can you wait for another 5 to 10 minutes?
Sure, sir. Sure.
Once we finish that, then we have poster presentation. You carry your poster, you can put up your poster here. You have a board, you can explain. Or if you have a PPT in your poster, you can also you can use both the means. Either the poster you can hang it in the top or through your PPT. Right? So what can we do? We continue.
Yes, sir.
Okay.
ಅಲ್ಲ ಇದು ಇವಾಗ ಒಂದೇ ಒಂದು ಆನ್ಲೈನ್ ಪ್ರೆಸೆಂಟೇಷನ್ ಅಂತ ಬೇಕು.
Okay, we continue.
Layashri?
Yes,
Hello?
sir. Yes, sir. I'm present.
Okay. So you can switch on the camera and start presenting. Okay.
Sure, sir.
I'll be
Thank
sharing
you.
the screen.
Yes, sir. May I start?
Yes, you can start.
Yes, sir. So, greetings to one and all present here. I am Laya from grade 11th. I am here today to present my research on the topic of assessing the impact of diets and agriculture on the environment and health. So, moving ahead, let's go to the introduction slide. So, we're going a lot goes into feeding our ever-growing population, for which we use commercial agriculture. So today we're going to try to understand how diets and agriculture affect the environment and the overall health status of a person that consumes the diet. At the end, we will also be exploring environmentally friendly agricultural alternatives such as food forestry and permaculture through a case study. So the area of study, the next slide. So the area of study chosen for this is Aranya Forest and Sanctuary, which is an alternative method of farming. Next slide. Yes, so my major objectives for the research study was first to study the impact of diversified and non-diversified diets on the environment and overall health by assessing and quantifying the impact on set criteria. And my second objective was exploring viable alternatives to commercial agriculture such as food forestry and permaculture. And my third and one is a very interesting objective being is to create awareness on nutrition and environmental conservation. Next slide. Yeah. So let's take a look at the methods that were employed for the data collection through reliable sources. So the first one was an online survey with urban respondents. The second one was a rural area survey and interaction, which was conducted in the rural villages of Tamil Nadu, Karnataka, and Andhra Pradesh. And the third method that was employed was, assessing the biodiversity and environmental status of Aranya Forest and Sanctuary data through official sources. Next one. Yes. So generally, when we take our food, we don't give much thought into it. So let's take a look at the criteria set by professionals on what a nutritious diet should consist of. So the first one being 50% or less grain, second one being 20% or more of seasonal produce like vegetables and fruits, and the third one is it should not have more than 30% of highly refined or processed foods. And the fourth one is an essential amount of nutrients like dietary fiber, proteins, complex carbohydrates, and others. Next one. Yes, so let's look at similar criteria that was taken for the study of the impact on diets, on health. So the criteria that was employed here was first, history of chronic illness, second one was physical mobility, and third one was mental health and emotional well-being. A thing to note is that the age group surveyed here is 15 to 70 years old. Now we use the above criteria as a metric to compare and contrast our findings and results. Next slide. So this is the interesting part. I would like to present to you the results and the data analysis. So to your left, we see the diversified diet, which has an overall higher proportion of vegetables and fruits and a good amount of grains and pulses and dairy products, which is considered to be a diversified diet, which is mostly prominent in rural areas. And to our right, we have a non-diversified diet, which is mostly prominent in urban areas. So it mostly, as you can see, if you compare to yourself, you can see that it has a higher proportion of processed foods like refined oils and fats, etc. In between, we have a comparative chart on what an ideal diet should look like. So if you take some time to look at it, we find that the diversified diet prominent in rural areas is the closest to this metric. Next one. So in all the three criteria that was said, that is chronic illness, physical mobility, and also mental health and emotional well-being, people who consumed a diversified diet outperformed those who consumed a non-diversified diet in all the three set criteria. 2 slides ahead. One more slide ahead. Yes. So now that we have seen the results and the findings of both for, on agriculture and health of the diets and the criteria, let's take a look at the impact of diversified and non-diversified diets and agriculture on the environment. As we have seen that diversified diets are mostly composed of seasonal and local produce such as vegetables, fruits, pulses, millets, etc. Therefore, they have lesser food miles, which is basically a concept that assesses the overall carbon footprint of a food it takes before it reaches our plate, that is the consumer. And the advantages that this presents to us is a better nutrition, second one being environmentally friendly, and also it is economical. So if you take a look at the environmental benefits in particular, it provides us lesser transportation, no packaging, lesser food waste, and overall lower carbon footprint. Next one. So now before we delve into the interesting part that is exploring alternatives, I would like to present to you a very interesting food, which I would like to call it a superfood, that is millets. I would like to call it a superfood both in terms of nutrition, as it's very nutritionally dense. It contains a good amount of complex carbohydrates, dietary fibers, vitamins and minerals, but also it's also environmentally friendly. It is not a water intensive crop. It can also be grown in areas that are prone to drought, and it doesn't require as much inputs. So 2023 is declared as the International Year of Millets, and India is at the forefront of this initiative. Next slide. So now that we have taken a good look at the results, the data analysis, and the impact, what can be done? Can we really switch commercial agriculture to alternatives such as permaculture and food forestry? Is it a viable alternative really to feed our ever-growing population? That is what we're going to see through this exploring alternatives section, that is food forestry and permaculture. We've taken the example of Aranya Forest, which was a 100-acre barren land, as you can see in the before picture. And today it is a regenerated indigenous forest with thriving biodiversity. Some of the principles that were adopted in this conversion was use of permaculture principles and use of alternative renewable energies, soil and moisture conservation measures, and various others. Next slide. So a thing to note before we discuss the biodiversity of Aranya is that it is a tropical dry evergreen forest type spanning over 100 acres. So a staggering 251 species was recorded in the last few decades after this development was happening, and over 240 bird species, both migratory and resident in nature, was also seen. And also some wildlife was spotted like the civet, jackal, the jungle cat, the fox, and various others. Next slide. So you can see the array of the amazing biodiversity at Aranya. This is what happens when we give back to nature and use better alternatives. We can see there are various types of frogs and toads, butterflies, and through trap cameras they have also pictured the scats of wildlife. Next one. As we can see, pug marks and scats of wildlife animals, which shows us that it's a thriving ecosystem. Next one. So these are some of my pictures from the field visit that I did in the rural areas and interaction, as you can see. Next one. So now that we have gotten a good idea of what this research was about, let's derive some interesting conclusions. So the first conclusion is that diversified diet is directly linked to an overall lower carbon footprint impact of agriculture on the environment, because the diversified diet is constituted with local and seasonal produce. And the second one is that consumption of a diversified diet ensures optimal nutrition and helps in maintaining the overall health of a person. Third one is that commercial agriculture negatively impacts the environment, and alternatives like food forestry and permaculture can be used and looked into to conserve biodiversity, maintain soil health, and also to have an overall lower carbon footprint. Next one. So now we have also looked at the conclusions that we derived, but what can be done? So a cliche that we get in this is awareness. So awareness is very essential, especially in terms of nutrition, agriculture, and making our planet a better place. So that's why I run my own initiative, my own non-profit organization called as Earth Guardians Bangalore, which I'll be using as a platform to further put my research ahead and bring some real world change. Next one. So these are some pictures. I have committed myself to over 100 plus hours of volunteering and community service and composting and various other activities. Next one. I've also conducted few awareness programs both in my school and my nearby government schools. Next one. So before I would like to end my presentation here, I would like to show you a very interesting finding on research which might interest all of us here. So in the course of my data analysis and field visits, I asked people, do you think research is only for solving big problems? Sadly enough, 65% of them said yes, and followed by a second question, that is, do you think it can be used to solve everyday problems? And many of them answered no. So this shows that we as a scientific community are often misunderstood for the work we do. Work in the field of AI, artificial intelligence, spacecrafts is very well applauded. So I think through my research, I also want to show people and break this misconception and give equal importance to research done in the fields of environment and millets and etc. Next one. So yes, that concludes my presentation. I would really like to thank IISc very much for providing me this opportunity to share my insights, especially in the virtual mode, which is very beneficial. And a huge thank you to D. Saravanan sir from Aranya Forest and Sanctuary for coordinating with me throughout the project and giving me first-hand data and insights. I would also like to thank our director, Dr. Seema Goyal ma'am and management. A huge thank you to my mentor, Gayatri ma'am, and a special mention to everyone who took part in the survey actively and also the interactions. Next one. So yes, these are the sources that I've used in doing my research. So that completes my presentation. Thank you very much. It's a great opportunity for me to be on the virtual mode and still present. So I would like to once again thank for this opportunity.
Students, do you have any questions?
Actually, I would like to know the background of the people who you have interviewed, because that also matters, giving their answers, their experiences. Like science is research is meant for solving big problems, such questions. Their background also matters when they answer.
Ma'am, can you repeat the question? I'm actually not able to hear it.
You interviewed some people now to get the feedback on whether the research is being conducted only to solve big problems and how much it can solve such questions. And you showed me some percentages, around 40 and all. I would like to know the background of these subjects whom you have interviewed.
Yes, ma'am, sure. So for this, I had composition of both rural, urban, educated, non-educated. So my thing was to get a metric which.
Yes, can you repeat?
Yes, ma'am. So the people that I, surveyed for the last two questions that I showed you, excuse me, sorry. Yes, ma'am. I'll just repeat myself. So the people that I surveyed for getting the last research that I got on research was people from both urban and rural areas who were both educated and non-educated. Yes, ma'am. So yes, ma'am. So it had a composition of both rural and urban respondents, and also various other factors like education, employment type, where they currently work, and their belief systems, etc. Hello.
Hello. One more question. You showed something like the diversified diet.
Yes, ma'am.
One can have a healthy, health will be maintained in a very good, and they will be strong, etc. And also you showed this Aranya forest related things.
Yes.
I am not getting how these two are related.
So you're asking like how the diversified diet is linked with the exploring alternatives study of Aranya?
Yes.
Yes, ma'am. So basically, as I showed, a diversified diet consists of a variety of local and seasonal produce. For example, in urban areas, we consume food from various parts of the world and our own local community. But whereas if the diet is local and seasonal, a transportation is cut, and as the benefits I was talking. So how I linked it with the case study is that Aranya was a barren land. So then they converted it into a indigenous local forest. So in which a part of it is a food forest, right? So the locals are also benefited from this. So so basically I wanted to show that by eating a diversified diet that I showed in the data analysis, we can like have like forest conservation and also fodder for like the cattle, and also the locals can also be greatly benefited by these greening activities. So this is the link I wanted to show between the case study and the diversified diet, ma'am.
The diversified diet of this population, and before the Aranya, which is covered with the food plantation, whatever, vegetation. How you do draw the data before and after this Aranya development?
Yes, ma'am. So basically, Aranya is right now, the area has not been studied. So I'll be doing in-person field work in the summer this time. But then I do have a before and after picture of how it looked. So basically, Aranya didn't even have enough soil cover. Like topsoil was totally washed away. It had visibly no vegetation at all except a few tamarind trees scattered here and there. So from that, it has developed to this today.
Actually, I understand. Introducing this local indigenous food, producing plants there and helping the local population. But my question is, now you have shown this diversified diet leading to a good health of the population, and this since you immediately, I think you cannot relate it to this Aranya study. Maybe after some time, if you have the data of before Aranya development and after few years, health conditions of the population in and around, then I think you will be able to conclude, in a data-based conclusion.
Yes, ma'am. Yes. Yes, ma'am. So that is a very good addition. I will definitely look into it. And I think I have already mentioned in the before thing, I will actually go in person to it, and I'll update my work on the Aranya part of my presentation in the next Lake conference.
Okay, thank you.
Thank you, ma'am. Thank you very much.
Next, Suvarna and Sanika.
School. CHS, Sirsi. ಸೆಕೆಂಡರಿ ಹೈಸ್ಕೂಲ್ ಹೇಳುತ್ತೆ. ಯಾವುದು? ಅಲ್ಲ ಸುವರ್ಣ ಅಲ್ಲ? ನಾ ಮಾಡ್ತೀನಿ. ಮಾಡ್ತೀನಿ. ಮಾಡ್ತೀನಿ ನೋಡಿ, ನೀವು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿ, ಬೇಕಾದ್ರೆ ಯೂಸ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಇಲ್ಲಿ ನೋಡಿ, ಈ ಕಡೆ ಸೈಡ್ ನಿಂತ್ಕೊಂಡ್ ಮಾತಾಡಿ, ಕ್ಯಾಮ್ರಾ ಇಲ್ಲಿದೆ ಅಂತ. ಇಲ್ಲ ಮಾಡ್ತೀನಿ ನಾನು, ನೀವು ಕ್ಯಾಮ್ರಾ ಇಲ್ಲಿದೆ, ಈ ಕಡೆ ಸೈಡ್ ನೋಡ್ಕೊಂಡ್ ಮಾತಾಡ್ಬೇಕು, ಈ ಕಡೆ ಮಾತಾಡ್ಬೇಕು ಸರ್, ಅವ್ರಿಗೆ ಅಷ್ಟೇ ಅಲ್ಲ. ಸ್ಟಾರ್ಟ್ ಮಾಡಿ.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು ಸುವರ್ಣ ಮತ್ತು ಇವಳು ಸಾನಿಕಾ. ಇಂದು ನಾವಿಬ್ಬರೂ ಸೇರಿ ಔಷಧಿ ಸಸ್ಯಗಳ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ತಿಳಿಸುತ್ತಿದ್ದೇವೆ. ನಮ್ಮ ಗ್ರಾಮದಲ್ಲಿ ದೊರೆಯುವ ಔಷಧಿ ಸಸ್ಯ ಮತ್ತು ಸಸ್ಯೋತ್ಪನ್ನಗಳು, ಅವುಗಳ ಉಪಯೋಗ ಮತ್ತು ಅವುಗಳ ಮನೆಮದ್ದುಗಳ ಬಗ್ಗೆ ಇಂದು ತಿಳಿಸುತ್ತಿದ್ದೇವೆ. ನನ್ನೂರಿನ ವೈಶಿಷ್ಟ್ಯತೆ. ಉತ್ತರ ಕನ್ನಡ ಜಿಲ್ಲೆಯ ಕುಮಟಾ ತಾಲೂಕಿನ ಹಿರೇಗೊತ್ತಿಯು ಅಂಕೋಲ ಮತ್ತು ಕುಮಟಾದ ಮಧ್ಯದಲ್ಲಿ ಬರುವ ರಾಷ್ಟ್ರೀಯ ಹೆದ್ದಾರಿ 66ರಲ್ಲಿ ಬರುವ ಉಲ್ಲೇಖಗೊಂಡಿರುವಂತೆ ಹಿರೇಗುತ್ತಿ ಪ್ಲೇಟ್ ಎಂದು ಕರೆಯಲ್ಪಡುವ ತಾಮ್ರಶಾಸನದಲ್ಲಿ 5ನೇ ಶತಮಾನದಲ್ಲಿ ಉಲ್ಲೇಖಗೊಂಡಿರುವಂತೆ ಭೋಜರಾಜರು ಇಲ್ಲಿ ಆಳ್ವಿಕೆ ನಡೆಸಿರುವ ಮಾಹಿತಿಯನ್ನು ತಿಳಿಸುತ್ತದೆ. ಇವರ ಅರಸನಾದ ಅಂಕಿತವರ್ಮನು ಶ್ರೀ ಮಹಾಲಿಂಗೇಶ್ವರ ದೇವಸ್ಥಾನದ ಸಮುಚ್ಚಯದಲ್ಲಿ ಬೌದ್ಧ ಧರ್ಮಶಾಲೆಯನ್ನು ಕಟ್ಟಿಸಿದ ಎಂದು ಹೇಳಲಾಗಿದೆ. ಹಿರೇಗುತ್ತಿಯ ಸಮೀಪದಲ್ಲಿರುವ ನುಷಿಕೋಟೆಯ ಕಾಂಡ್ಲವನ ಜೀವಿಗಳ ಉಳಿವಿಕೆಗೆ ಕಾರಣವಾಗಿದೆ. ಒಂದು ಕಡೆ ಕದಡಿಯ ವಿಹಂಗಮ ನೋಟ, ಇನ್ನೊಂದು ಕಡೆ ಗುಡ್ಡ ಬೆಟ್ಟ, ಇವುಗಳ ನಡುವೆ ಹಿರೇಗುತ್ತಿಯ ವೈಭವ. ನಮ್ಮ ಊರಿನ ಪ್ರತಿ ಮನೆಯಲ್ಲೂ ಕನಿಷ್ಠ 10 ರಿಂದ 15 ಗಿಡಗಳನ್ನು ನೆಟ್ಟು ಅವುಗಳ ಪೋಷಣೆ ಮಾಡುತ್ತಾರೆ. ಅಷ್ಟೇ ಅಲ್ಲದೆ ಪ್ರಸ್ತುತ ಕೊರೋನಾ ಪ್ಯಾಂಡಮಿಕ್ ಸನ್ನಿವೇಶ ಇದ್ದುದರಿಂದ ಔಷಧಿ ಸಸ್ಯಗಳನ್ನು ಬೆಳೆಸಿ ಅವುಗಳನ್ನು ಸಂಖ್ಯೆ ಹೆಚ್ಚಾಗಿದೆ. ನಾನು ನನ್ನ ಗ್ರಾಮದ ಸುಮಾರು 30ಕ್ಕೂ ಹೆಚ್ಚು ಕುಟುಂಬಗಳನ್ನು ಭೇಟಿ ಮಾಡಿ 30ಕ್ಕೂ ಹೆಚ್ಚು ಗಿಡಗಳ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ಸಂಗ್ರಹಿಸಿದ್ದೇನೆ. ಇದು ನಾವು ಸಂಗ್ರಹಿಸಿದ ಸಸ್ಯದ ತುಣುಕುಗಳು. ಲೋಳೆ ಸರ, ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಅಲವೇರಾ. ಸಕಲ ಸಮಸ್ಯೆಗೂ ಪರಿಹಾರ ಅಲವೇರಾದಲ್ಲಿದೆ. ಅಲವೇರಾದ ಲೋಳೆಯನ್ನು ಕೂಡಿದರೆ ಜೀರ್ಣಕ್ರಿಯೆ ಚೆನ್ನಾಗಿ ಆಗುತ್ತದೆ. ಮೂಲವ್ಯಾಧಿ ಹಾಗೂ ಅಲ್ಸರ್ನಂತಹ ಕಾಯಿಲೆಗಳಿಗೂ ಕೂಡ ಅಲವೇರಾ ಔಷಧಿಯಾಗಿದೆ. ಅಷ್ಟೇ ಅಲ್ಲದೆ ಚರ್ಮದ ಆರೈಕೆಗೆ ಹಾಗೂ ಎಲ್ಲಾ ರೀತಿಯದಲ್ಲೂ ಅಲವೇರಾ ಔಷಧಿಯಾಗಿದೆ. ಮೊಡವೆಗಳನ್ನು ನಿವಾರಿಸುತ್ತದೆ, ತಲೆ ಹೊಟ್ಟನ್ನು ನಿವಾರಿಸುತ್ತದೆ, ತಲೆ ಉರಿಗೂ ಕೂಡ ಇದು ಔಷಧಿಯಾಗಿದೆ. ನಾಚಿಕೆ ಮುಳ್ಳು, ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಮಿಮಸ್ ಪುಡಿಕ. ಬಳಸುವ ವಿಧಾನ ಏನಂದರೆ ಇದನ್ನು ಗಿಡ ಸಮೇತವಾಗಿ ಅರಿತು ಅದನ್ನು ಊತವಾದ ಜಾಗಕ್ಕೆ ಹಚ್ಚಿ ಬಟ್ಟೆಯಿಂದ ಕಟ್ಟಿದರೆ ಊತ ಬೇಗನೆ ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಈ ಗಿಡದ ಎಲೆ ಮತ್ತು ಬೇರನ್ನು ಜಜ್ಜಿ ರಸ ತೆಗೆದು 2 ಅಥವಾ 3 ಚಮಚ ರಸಕ್ಕೆ 1 ಲೋಟ ನೀರು ಬೆಳೆಸಿ ಕುಡಿಯುವುದರಿಂದ ಮಲಬದ್ಧತೆ ನಿವಾರಣೆ ಆಗುತ್ತದೆ. ನಾಚಿಕೆ ಮುಳ್ಳಿನ ಎಲೆಯನ್ನು ಜಜ್ಜಿ ಗಾಯಕ್ಕೆ ಹಚ್ಚಿದರೆ ಕೂಡಲೇ ರಕ್ತ ಸೋರಿಕೆ ಕಡಿಮೆಯಾಗಿ ಗಾಯವು ಬೇಗನೆ ವಾಸಿಯಾಗುತ್ತದೆ. ಈ ಗಿಡದ ರಸವನ್ನು ಚರ್ಮಕ್ಕೆ ಹಚ್ಚುವುದರಿಂದ ಚರ್ಮದಲ್ಲಿನ ತುರಿಕೆ ಹಾಗೂ ಇತರ ಚರ್ಮ ರೋಗಗಳು ಕೂಡ ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಲಕ್ಕಿ ಗಿಡ, ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ವೈಟೆಕ್ಸ್ ನಿಕುಂಡ. ಇದನ್ನು ನಾವು ಸಂಸ್ಕೃತದಲ್ಲಿ ನಿರ್ಗುದಿ, ಹಿಂದಿಯಲ್ಲಿ ಸಂಬಾಲು ಎಂದು ಕರೆಯುತ್ತೇವೆ. ಅಷ್ಟೇ ಅಲ್ಲದೆ ತೆಲುಗುವಲ್ಲಿ ತೆಲ್ಲವಾವಿಲ ಎಂದು ಕೂಡ ಕರೆಯುತ್ತಾರೆ. ಹಲವಾರು ಭಾಷೆಯಲ್ಲಿ ಹಲವಾರು ಹೆಸರಿನಿಂದ ಕರೆಯುತ್ತಾರೆ. ಇದರ ಬೇರು ಎಲೆ ಔಷಧದ ಮೂಲವಾಗಿದೆ. ಕಿವಿಯಲ್ಲಿ ಕ್ರಿಮಿ ಕೀಟ ಹೊಕ್ಕಿದರೆ ಇದರ ಬೇರಿನ ರಸವನ್ನು ತೆಗೆದು ಹಾಕುವುದರಿಂದ ಆ ಕ್ರಿಮಿ ಕೀಟಗಳು ಅಲ್ಲಿಯೇ ನಾಶವಾಗಿ ಹೋಗುತ್ತದೆ. ದನಗಳಿಗೆ, ಉಳುವ ಎತ್ತುಗಳಿಗೆ ಹೆಗಲು ಹುಣ್ಣು ಆದಾಗ ಇದನ್ನು ನಾವು ಔಷಧಿಯನ್ನಾಗಿ ಬಳಸುತ್ತೇವೆ. ಅರಸಗೆ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಜಸ್ಟಿಕ್ ಅರಸದೆ. ಇದು ಉದ್ದವಾದ ಎಲೆಯನ್ನು ಹೊಂದಿದ್ದು ಎಲೆ ಔಷಧಿಯ ಗುಣ ಹೊಂದಿದೆ. ಕಫಕ್ಕೆ ಮುಯಿದರೆ 10 ರಿಂದ 15 ಎಲೆಗಳನ್ನು ಒಂದು ಬಟ್ಟೆ ತುಂಡೆಯಲ್ಲಿ ಇಟ್ಟು ಗಂಟು ಕಟ್ಟಿ ಉಗಿಯಲ್ಲಿ ಬೆಚ್ಚಗೆ ಮಾಡಿ ಎದೆಯ ಭಾಗಕ್ಕೆ ಇಟ್ಟುಕೊಂಡರೆ ಕಫ ಕರಗುತ್ತದೆ. ಸೈನಸ್, ಇದೇ ಬೆಚ್ಚಗಿನ ಗಂಟನ್ನು ಹಣೆಯ ಕೆನ್ನೆಯ ಭಾಗಕ್ಕೆ ಇಟ್ಟುಕೊಂಡರೆ ನೋವು ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಇದರ ಹದವಾದ ಉಗಿಯನ್ನು ಮೂಗಿನಿಂದ ಹೊರಗೆ ತೆಗೆದುಕೊಂಡರೆ ಮೂಗು ಕಟ್ಟಿಕೊಂಡಿದ್ದು ಕೂಡ ಹತೋಟಿಗೆ ಬರುತ್ತದೆ. ಬಿಳಿ ಎಕ್ಕೆ ಗಿಡ, ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಕಾಲೋಟ್ರೋಪಿಸ್ ಗಿಗಂಟಿಯಾ. ಇದನ್ನು ಸಂಸ್ಕೃತದಲ್ಲಿ ಅರ್ಕ, ಹಿಂದಿಯಲ್ಲಿ ಎಕ್ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಇದು ಪೊದೆಗಳ ರೂಪದಲ್ಲಿದ್ದು 4-5 ಅಡಿ ಎತ್ತರದಲ್ಲಿ ಬೆಳೆಯುತ್ತದೆ. ಇದನ್ನು ನಾವು ಕಚ್ಚಿಗೆ ಔಷಧಿಯನ್ನಾಗಿ ಬಳಸುತ್ತೇವೆ. ಬಿಳಿ ಎಕ್ಕೆ ಹಾಲು 1 ತೊಲೆ, ಸಾಸಿವೆ ಎಣ್ಣೆ 1 ತೊಲೆ ಮತ್ತು ಅರಿಶಿನ ಪುಡಿ 2 ತೊಲೆ ಎಲ್ಲವನ್ನು ಒಟ್ಟಿಗೆ ಬೆರೆಸಿ ಕಾಯಿಸಿ ಕಜ್ಜಿಗೆ ಲೇಪಿಸಬೇಕು. ಇದರಿಂದ ಕಜ್ಜಿ ಅತಿ ಕಡಿಮೆ ಅವಧಿಯಲ್ಲಿ ಕಡಿಮೆಯಾಗುತ್ತದೆ. ವೃತ್ತಿಗೆ ಮತ್ತು ಬಿಳಿ ಚಿಬ್ಬಿಗೂ ಕೂಡ ಇದನ್ನು ಔಷಧಿಯನ್ನಾಗಿ ಬಳಸುತ್ತೇವೆ. ಸುಟ್ಟ ಗಾಯಕ್ಕೂ ಕೂಡ ಇದರ ಎಲೆಯನ್ನು ಪುಡಿ ಮಾಡಿ ಹಚ್ಚುವುದರಿಂದ ಸುಟ್ಟ ಗಾಯವು ಅತಿ ಕಡಿಮೆ ಅವಧಿಯಲ್ಲಿ ಕಡಿಮೆಯಾಗುತ್ತದೆ. ತೊಂಡೆಕಾಯಿ, ಇದು ಮಧುಮೇಹ ಹಾಗೂ ಕಾಮಲೆ ರೋಗಕ್ಕೆ ಬರುತ್ತದೆ. ಎಲೆಗಳ ರಸ ತೆಗೆದು 20 ರಿಂದ 30 ಮಿಲಿನಷ್ಟು ಖಾಲಿ ಹೊಟ್ಟೆಯಲ್ಲಿ ಸೇವಿಸುವುದರಿಂದ ಹೀಗೆ ಮಾಡಿದರೆ 3 ತಿಂಗಳಿನಲ್ಲಿ ಸಕ್ಕರೆ ಮಟ್ಟ ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಅಣಲೆಕಾಯಿ, ಇದನ್ನು ನಾವು ವೈಜ್ಞಾನಿಕವಾಗಿ ಟರ್ಮಿನಾಲಿಯಾ ಸೆಬುಲಾ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಇಲ್ಲಿ ನೋಡಿ, ಇದೇ ಅಣಲೆಕಾಯಿ, ಇದೇ ಹಣ್ಣು. ಇದರ ಚೂರ್ಣ ಜಿಗಿದು ತಿನ್ನುವುದರಿಂದ ಜೀರ್ಣಶಕ್ತಿ ಹೆಚ್ಚಾಗುತ್ತದೆ. ಅದರೇ ಅಷ್ಟೇ ಅಲ್ಲದೆ ಅದನ್ನು ಆಹಾರವಾಗಿ ಸೇವಿಸುವುದರಿಂದ ನಮ್ಮ ಬುದ್ಧಿಶಕ್ತಿ ಕೂಡ ಬೆಳವಣಿಗೆ ಆಗುತ್ತದೆ. ಇದನ್ನು ಹುರಿದು ತಿನ್ನುವುದರಿಂದ ತ್ರಿದೋಷಗಳಾದ ವಾತ, ಪಿತ್ತ, ಕಫಗಳು ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಇದರ ಬಗ್ಗೆ ನಮಗೆ ಮಹೇಶ್ ನಾಯ್ಕ್ ಇವರು ಮಾಹಿತಿ ನೀಡಿದರು. ಚಿತ್ರಮೂಲ, ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಪ್ಲಂಬಗೋಸಿಯಾಲನಿಕ. ಇದನ್ನು ಸಂಸ್ಕೃತದಲ್ಲಿ ಚಿತ್ರಕ ಮತ್ತು ಹಿಂದಿಯಲ್ಲಿ ಪಾಂಡಾರ ಚಿತ್ರಕ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಕರಾವಳಿ ಹಾಗೂ ಮಲೆನಾಡು ಜಿಲ್ಲೆಗಳಲ್ಲಿ ಇದನ್ನು ನೆಟ್ಟು ಬೆಳೆಸುತ್ತಾರೆ. ಇದರಲ್ಲಿ 2 ವಿಧವಿದೆ, ಒಂದು ಕೆಂಪು ಚಿತ್ರಮೂಲ, ಮತ್ತೊಂದು ಬಿಳಿ ಚಿತ್ರಮೂಲ. ಗಂಟಲೊಳಗೆ ಹುಣ್ಣು ಆದಾಗ, ಅಂದರೆ ಉಷ್ಣತೆಯಿಂದ ಗಂಟಲು ಬಿರುಕು ಉಂಟಾಗಿ ನಮಗೆ ಮಾತನಾಡಲು ಕಷ್ಟವಾಗುತ್ತದೆ. ಅಂತಹ ಸಂದರ್ಭದಲ್ಲಿ ಇದನ್ನು ನಾವು ಆಕಳ ಅಪ್ಪಚ್ಚಿನಲ್ಲಿ ತೆಗೆದು ಲೇಪಿಸುವುದರಿಂದ ಅಥವಾ ಸೇವಿಸುವುದರಿಂದ ಅದು ಅತಿ ಕಡಿಮೆ ಅವಧಿಯಲ್ಲಿ ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಹೊರಗೆ ಗಂಟಲು ಬಾವು ಇದ್ದರೂ ಕೂಡ ಇದನ್ನು ಲಿಂಬೆರಸದಲ್ಲಿ ತೆಗೆದು ಹಚ್ಚುವುದರಿಂದ ಗಂಟಲು ಬಾವು ಕೂಡ ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಪಪಾಯ, ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಕ್ಯಾಲಿಸ್ ಪಪಾಯ. ಸಾಮಾನ್ಯವಾಗಿ ಎಲ್ಲ ಕಡೆ ದೊರೆಯುವ ಮತ್ತು ಅಪೋಲ ಔಷಧಗಳು ಹೊಂದಿರುವ ಸಸ್ಯ ಪಪಾಯವಾಗಿದೆ. ಔಷಧಿಯ ಗುಣ ಏನಂದ್ರೆ ವಯಸ್ಸಾದಂತೆ ಕೂವ ಜೀರ್ಣಶಕ್ತಿಯನ್ನು ವೃದ್ಧಿಸಲು ಸಹಕರಿಸುತ್ತದೆ. ಮೂಳೆಯ ಸವಿತ ಹೃದಯ ಕಾಯಿಲೆಗಳಿಗೆ ಸೂಕ್ತವಾಗಿದೆ. ಇದರ ಎಲೆಯನ್ನು ಜಜ್ಜಿ ಸೋಸಿ ರಸಕ್ಕೆ ಜೇನುತುಪ್ಪದೊಂದಿಗೆ ಸೇವಿಸಿದರೆ ಡೆಂಗ್ಯೂ, ಮಲೇರಿಯಾ ಜ್ವರಗಳು ಕೂಡ ನಿವಾರಣೆಯಾಗುತ್ತದೆ. ಕಹಿಬೇವು, ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಅಸಾಡಿರೆಕ್ಟಾ ಇಂಡಿಕಾ. ಕಹಿಬೇವಿನ ಎಲೆಯನ್ನು ಸೇವಿಸುವುದರಿಂದ ನಮಗೆ ಅಜೀರ್ಣತೆ ಮತ್ತು ಹೊಟ್ಟೆ ನೋವಿನಂತಹ ಕಾಯಿಲೆಗಳು ದೂರವಾಗುತ್ತದೆ. ಆದ್ದರಿಂದ ವರ್ಷಕ್ಕೆ 1 ಬಾರಿ ಆದರೂ, ಅಂದರೆ ದೀಪಾವಳಿ ಹಬ್ಬದಂದು ನಾವು ಕಹಿಮದ್ದನ್ನು ಔಷಧಿಯನ್ನಾಗಿ ಪಡೆಯುತ್ತೇವೆ. ಇದರ ಬೇವಿನ ಎಣ್ಣೆ ಹಾಗೂ ತುಳಸಿ ಎಲೆ ಹಾಕಿದ ನೀರಿನಿಂದ ಸ್ನಾನ ಮಾಡಿದರೆ ಚರ್ಮದ ಎಲ್ಲಾ ರೋಗಗಳು ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಒಂದೆಲಗ, ಇದನ್ನು ಸೆಂಟೆಲಾ ಎಸ್ಟೇಟಿಕ್ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಇದು ನೆನಪಿನ ಶಕ್ತಿಯನ್ನು ವರ್ಧಿಸುತ್ತದೆ. ಅಷ್ಟೇ ಅಲ್ಲದೆ ದೇಹದ ಉಷ್ಣತೆಯನ್ನು ಕಡಿಮೆ ಮಾಡುತ್ತದೆ.
ಚಿಕ್ಕ ಮಕ್ಕಳ ಬುದ್ಧಿಶಕ್ತಿ ವರ್ಧನೆಯಾಗಲು ಕೂಡ ಇದು ಸಹಾಯಕವಾಗುತ್ತದೆ. ತುಳಸಿ ಗಿಡ ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಆಸಿಮಾಮ್ ಸ್ಕ್ಯಾಂಕ್ಟಮ್. ಜ್ವರ, ಕೆಮ್ಮು, ನೆಗಡಿಯಂತಹ ರೋಗವನ್ನು ಕಡಿಮೆ ಮಾಡುತ್ತದೆ. ಅಷ್ಟೇ ಅಲ್ಲದೆ ತುಳಸಿ ಎಲೆಯನ್ನು ಹಾಕಿದ ನೀರಿನಿಂದ ಉಗಿ ತೆಗೆದುಕೊಂಡರೆ ಯಾವುದೇ ದೊಡ್ಡ ಮಹಾ ಥಂಡಿಯಾಗಿದ್ದರೂ ಅದು ಅತಿ ಕಡಿಮೆ ಅವಧಿಯಲ್ಲಿ ಗುಣವಾಗುತ್ತದೆ. ಕರಿಬೇವು ವೈಜ್ಞಾನಿಕ ನೈಸರ್ಗಿಕ ಗಿಡಮೂಲಿಕೆಯಾದ ಬೇವಿನಲ್ಲಿಯೂ ಸಮೃದ್ಧವಾದ ಪೋಷಕಾಂಶಗಳು ಒಳಗೊಂಡಿರುತ್ತದೆ. ಹಾಗಾಗಿ ಇದನ್ನು ಪೋಷಕಾಂಶಗಳ ಆಗಲೇ ಎನ್ನಲಾಗುವುದು. ಈ ಪುಟ್ಟೆಯಲ್ಲಿ ವಿಟಮಿನ್ ಎ, ಬಿ, ಸಿ ಮತ್ತು ಬಿ ಅಧಿಕವಾಗಿರುತ್ತದೆ. ಅಲ್ಲದೆ ಕಬ್ಬಿಣ ಮತ್ತು ಕ್ಯಾಲ್ಸಿಯಂ ಪ್ರಮಾಣ ಅಧಿಕವಾಗಿರುವುದರಿಂದ ನಿತ್ಯ ಇದನ್ನು ಆಹಾರದಲ್ಲಿ ಕಡ್ಡಾಯವಾಗಿ ಸೇವಿಸಬೇಕು. ಇದರ ಸೇವನೆಯಿಂದಾಗಿ ದೇಹದಲ್ಲಿ ಶೇಖರಣೆಯಾದ ಧನೆಗಟ್ಟು ಕೊಬ್ಬು ಕರಗುತ್ತದೆ, ಅಜೀರ್ಣ ಸಮಸ್ಯೆ ನಿವಾರಿಸುವುದು, ವಾಕರಿಕೆ ಹೋಗಲಿಸುವುದು ಮತ್ತು ಸುಟ್ಟ ಗಾಯವನ್ನು ಕೂಡ ಇದು ಗುಣಪಡಿಸುತ್ತದೆ. ಬಿಳಿ ದಾಸವಾಳ ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಹಿಬಿಸ್ಕಸ್ ರೋಸಾ. ಇದನ್ನು ಕನ್ನಡದಲ್ಲಿ ದಾಸವಾಳ, ದಾಸಣಿಗೆ ಮತ್ತು ಹಲವಾರು ಹೆಸರಿನಿಂದ ಕರೆಯುತ್ತಾರೆ. ಆದರೆ ಹಿಂದಿಯಲ್ಲಿ ಗುಡಲ್ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಇದು ನಮ್ಮ ದೇಶದ ಎಲ್ಲಾ ಭಾಗಗಳಲ್ಲಿ ಕಂಡು ಬರುತ್ತದೆ. ಇದರ ಎಲೆ ಮತ್ತು ಹೂವು ಔಷಧದ ಮೂಲವಾಗಿದೆ. ಆಮಶಂಕೆಗೆ ಇದರ ಹೂವು ಅಥವಾ ಮೊಗ್ಗನ್ನು ಹತ್ತು ತೆಗೆದುಕೊಂಡು ಮತ್ತು ಜೀರಿಗೆ ಪುಡಿ ಹಾಗೂ ಆಕಳ ಹಾಲಿನೊಂದಿಗೆ ಸೇರಿಸಿ ಕಷಾಯವನ್ನು ಮಾಡಿ ಕುಡಿಯುವುದರಿಂದ ಆಮಶಂಕೆ ಅತಿ ಕಡಿಮೆ ಅವಧಿಯಲ್ಲಿ ಕಡಿಮೆಯಾಗುತ್ತದೆ. ತುಂಬೆ ಗಿಡ ವೈಜ್ಞಾನಿಕ ಸಂಸ್ಕೃತದಲ್ಲಿ ಇದನ್ನು ದ್ರೋಣಪುಷ್ಟಿ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಸಮುದ್ರ ಮಟ್ಟದಿಂದ ಹಿಡಿದು 3000 ಅಡಿಗಳ ಎಲ್ಲಾ ಪ್ರದೇಶವನ್ನು ಕಾಣುತ್ತವೆ. ಮರುಗಿದ ಪ್ರದೇಶಗಳಲ್ಲಿ ಗದ್ದೆಗಳಲ್ಲಿ ರಸ್ತೆ ಇಬ್ಬದಿ ಗಿಡವಾಗಿ ಬೆಳೆಯುತ್ತದೆ. ಇದು ಗಿಡ ಸಾಮಾನ್ಯವಾಗಿ 30 ರಿಂದ 60 ಸೆಂಟಿಮೀಟರ್ ಎತ್ತರ ಬೆಳೆಯುತ್ತದೆ. ಇದರ ಎಲೆ ಔಷಧಿಯ ಮೂಲ. ಹಲ್ಲುನು ತುಂಬೆ ಗಿಡದ ಬಹಿರನ್ನು 5 ಗ್ರಾಂನಂತೆ ಜಜ್ಜಿ ಕೊಬ್ಬರಿ ನೀರು ಹಾಕಿ ಚೆನ್ನಾಗಿ ಕುದಿಸಬೇಕು. ಆಳದ ಮೇಲೆ ಸೋಸಿ ಅದರಿಸಿರುವಾಗ ನೋವಿನ ಭಾಗದ ವಿರುದ್ಧ ಕಿವಿಗೆ, ಅಂದರೆ ಎಡಬದಿಗೆ ಹಲ್ಲು ನೋವಾದರೆ ಬಲ ಕಿವಿಯಲ್ಲಿ ಹಲ್ಲು ನೋವಾದರೆ ಎಡ ಕಿವಿ 4 ಹನಿ ಬಿಡಬೇಕು. ದಿನಕ್ಕೆ 1 ಬಾರಿ 1 ದಿವಸ ಹೀಗೆ ಮಾಡಿದರೆ ಹಲ್ಲು ತುಂಬೆ ಹಲ್ಲು ಕಡಿಮೆ ಆಗುತ್ತದೆ. ಅಮೃತ ಬಳ್ಳಿ ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಟಿನೋಸ್ಪೊರಾ ಕಾರ್ಡಿಫೋಲಿಯಾ. ಇದನ್ನು ಸಂಸ್ಕೃತದಲ್ಲಿ ಗುಡುಚಿ, ಸೋಮವಲ್ಲಿ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಹಿಂದಿಯಲ್ಲಿ ಗುಲಾಂಚ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಇದರ ಕಾಂಡವನ್ನು ಜಜ್ಜಿ ರಸವನ್ನು ತೆಗೆದು ಜೇನುತುಪ್ಪದೊಂದಿಗೆ ದಿನಕ್ಕೆ 3 ಬಾರಿ ಆಹಾರ ಸೇವನೆಗೂ ಮುಂಚೆ ಸೇವಿಸುವುದರಿಂದ ಪಿದೋಷಗಳಾದ ವಾತ, ಪಿತ್ತ, ಕಫಗಳು ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಜ್ವರ ನಿಯಂತ್ರಣಕ್ಕೆ ಬರುತ್ತದೆ. ಸೋಮಲಕ್ಷಿ ವೈಜ್ಞಾನಿಕ ಈಯಣ್ಣಿನ ಪಾನಕಕ್ಕೆ ಸ್ವಲ್ಪ ಸೇರಿಸಿದರೆ ದಣಿವು ದೂರವಾಗಿ ಶರೀರದ ಊತ ಮತ್ತು ತಾಪಗಳು ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಮೂದ ಅಥವಾ ಪಿತ್ತರಸ ಸೇವನೆಗೆ ದಿವ್ಯೌಷಧವಾಗಿದೆ. ಇದನ್ನು ಹರಿಹಣ್ಣಿನ ರಸಕ್ಕೆ ಕಾಳುಮೆಣಸು, ಜೀರಿಗೆ, ಕಲ್ಲು ಸಕ್ಕರೆ ಹಾಕಿ ಪಾಕ್ ಮಾಡಿ ದಿನಕ್ಕೆ 2 ಬಾರಿ ಸೇವಿಸಿದರೆ ಕೆಮ್ಮು, ಕಫ ನಿವಾರಣೆಯಾಗುತ್ತದೆ. ಟಿಂಬೋಫೋಗಸ್ ಸಿಟ್ರೇಟಸ್ ಅಂದರೆ ಮಜ್ಜಿಗೆ ಹುಲ್ಲು. ಇದನ್ನು ಇಂಗ್ಲಿಷ್ನಲ್ಲಿ ಮಲಬಾರ್ ಗ್ರಾಸ್ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಇದು ಹುಲ್ಲಿನ ಜಾತಿಗೆ ಸೇರಿದ ಸಸ್ಯವಾಗಿದೆ. ಒಣ ಕೆಮ್ಮೆಗೆ ಮಜ್ಜಿಗೆ ಹುಲ್ಲನ್ನು ಕುತ್ತಲೆ ತೆಗೆದುಕೊಂಡು ಅದನ್ನು 1 ಸೇರು ನೀರಿನೊಂದಿಗೆ ತೆಗೆದುಕೊಂಡು ಅರ್ಧ ಸೇರು ಕಾಯಿಸಿ 1/4 ಸೇರನ್ನು ಕಷಾಯವಾಗಿ ಸೇವಿಸುವುದರಿಂದ ಒಣ ಕೆಮ್ಮು ಕಡಿಮೆಯಾಗುತ್ತದೆ. ಬೆಟ್ಟ ನೆಲ್ಲಿಕಾಯಿ ರಸವನ್ನು ಅರಿಶಿನೊಂದಿಗೆ ದಿನನಿತ್ಯ ಸೇವಿಸುವುದರಿಂದ ಮಧುಮೇಹ ನಿಯಂತ್ರಿಸಬಹುದು. ಇದು ಬೆಟ್ಟ ನೆಲ್ಲಿಕಾಯಿ ಚೂರ್ಣ. ಇದನ್ನು ರಕ್ತಸ್ರಾವದಲ್ಲಿ ಇದರ ಚೂರ್ಣವನ್ನು ಜೇನುತುಪ್ಪದೊಂದಿಗೆ ನೀಡಬೇಕು. ಇದರಲ್ಲಿ ವಿಟಮಿನ್ ಸಿ ಹೇರಳವಾಗಿದೆ. ಇದನ್ನು ಆಯುರ್ವೇದಿಕ್ ಔಷಧಿಯಲ್ಲಿ ಬಳಸುತ್ತಾರೆ. ದಾಲ್ಚಿನ್ನಿ ಸಿನೆಮನ್ ವೆರಮ್ ಎಂದು ಇದನ್ನು ಕರೆಯುತ್ತಾರೆ. ಕೊಲೆಸ್ಟ್ರಾಲ್ ಮಟ್ಟವನ್ನು ಕಡಿಮೆ ಮಾಡುತ್ತದೆ, ರಕ್ತದೊತ್ತಡವನ್ನು ನಿಯಂತ್ರಿಸುತ್ತದೆ. ಅಷ್ಟೇ ಅಲ್ಲದೆ ದಾಲ್ಚಿನ್ನಿಯಲ್ಲಿ ಸಿನಿಮಾಲ್ಡಿಹೈಡ್ ಎಂಬ ಸಕ್ರಿಯ ಅಂಶವು ವಿವಿಧ ರೀತಿಯ ಸೋಂಕಿನ ವಿರುದ್ಧ ಹೋರಾಡುತ್ತದೆ. ಇದರ ಬಗ್ಗೆ ನಮಗೆ ಮಹೇಶ್ ನಾಯ್ಕ್ ಇವರು ಮಾಹಿತಿ ನೀಡಿದರು. ಆರೋಗ್ಯ ಭಾಗ್ಯವೆಂಬಂತೆ ನಮ್ಮ ಆರೋಗ್ಯವನ್ನು ನಾವು ಕಾಪಾಡಿಕೊಂಡು ಮಹಾಭಾಗ್ಯವನ್ನು ತಂದುಕೊಳ್ಳಬೇಕು. ಸಂಪತ್ತು ಇದ್ದರೇನು, ಆರೋಗ್ಯ ಸರಿ ಇಲ್ಲದ ಮೇಲೆ ಏನೇ ಇದ್ದರೂ ಕೂಡ ಅದು ಕಡಿಮೆ. ಏಕೆಂದರೆ ನಮ್ಮ ಸುತ್ತಮುತ್ತಲಿನಲ್ಲಿರುವ ಗಿಡಮೂಲಿಕೆಗಳನ್ನು ನಾವು ಸುಸ್ಥಿರವಾಗಿ ಪರಿಣಾಮಕಾರಿಯಾಗಿ ಅವುಗಳ ಉಪಯೋಗವನ್ನು ಪಡೆದುಕೊಂಡು ನಮ್ಮ ಆರೋಗ್ಯವನ್ನು ಚೆನ್ನಾಗಿ ಇಟ್ಟುಕೊಳ್ಳಬೇಕು. ಯಾರು ಆರೋಗ್ಯವಂತರಾಗಿರುತ್ತಾರೆಯೋ ಅವರೇ ನಿಜವಾದ ಸಿರಿವಂತರು. ಸುಸ್ಥಿರವಾಗಿ ಈ ಗಿಡಗಳನ್ನು ಬಳಸಿಕೊಳ್ಳುವುದರಿಂದ ನಾವು ನಮ್ಮ ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿಯನ್ನು ಹೆಚ್ಚಿಸಿಕೊಳ್ಳಬೇಕು ಎಂದು ಹೇಳುತ್ತಾ ಈ ಯೋಜನೆ ಪ್ರಸ್ತುತಪಡಿಸಲು ಅವಕಾಶ ಮಾಡಿಕೊಟ್ಟ ಎಲ್ಲರಿಗೂ ಕೃತಜ್ಞತೆಗಳು. ಈಗ ನಾವು ಮಾಹಿತಿ ನೀಡಿದ್ವಲ್ಲ, ಅದರಲ್ಲಿ ಆ ಎಲೆಗಳನ್ನೆಲ್ಲ ಸಂಗ್ರಹಿಸಿಕೊಂಡು ಅದರಲ್ಲಿ ನಾವು ಈಗ ಮಾರ್ಬಲ್ ಮಾಡಿದ್ದೇವೆ. ಇಲ್ಲಿ ನೋಡಿ, ಅಣಲೆಕಾಯಿ ಆಗ ತೋರಿಸಿದ್ನಲ್ಲ, ಇದೆ ಅಣಲೆಕಾಯಿ, ಅದರ ಎಲೆ ಇದು. ಅದನ್ನ ನಾವು ಹಗ್ಗದಲ್ಲಿ ಸಂಗ್ರಹಿಸಿಟ್ಕೊಂಡು ಒಣಗಿಸ್ಕೊಂಡು ನಂತರ ಈ ರೀತಿಯಾಗಿ ಲ್ಯಾಮಿನೇಷನ್ ಮಾಡಿದ್ದೇವೆ. ಅಲೋವೆರಾ, ಇದು ಅಲೋವೆರಾದ ಎಲೆನ ನಾವು ಅಲೋವೆರಾದ ಎಲೆನ ಅದೇ ರೀತಿ ಒಣಗಿಸ್ಕೊಂಡು ಈ ರೀತಿ ಮಾಡಿದ್ದೇವೆ. ನಂತರ ಇದು ಬಿಳಿ ಚಿತ್ರಮೂಲ, ಆಗ ಹೇಳಿದ್ನಲ್ಲ, ಇದರಲ್ಲಿ 2 ರೀತಿ ಇದೆ, ಕೆಂಪು ಚಿತ್ರಮೂಲ, ಬಿಳಿ ಚಿತ್ರಮೂಲ. ಬೇವಿನ ಎಲೆ, ಕೈ ಬೇವಿನ ಎಲೆ, ಇದು ಒಂದೆಲಗ, ಇದನ್ನ ನಾವು ಬ್ರಾಹ್ಮಿ ಅಂತಲೂ ಕರೀತೇವೆ. ಇದು ತುಳಸಿ ಗಿಡ, ಸಾಮಾನ್ಯವಾಗಿ ತುಳಸಿ ಗಿಡ ಎಲ್ಲರ ಮನೆಯಲ್ಲೂ ಕಂಡು ಬರ್ತದೆ, ಎಲ್ಲರೂ ನೋಡಿರ್ತಾರಲ್ಲ. ಇದು ಮಜ್ಜಿಗೆ ಹುಲ್ಲು. ದಾಲ್ಚಿನ್ನಿ, ಇದು ತುಂಬೆ ಗಿಡ, ಮಧುಮೇಹ ಮತ್ತು ಕಾಮಾಲೆ ರೋಗಕ್ಕೆ ಇದು ತೊಂಡೆಕಾಯಿ, ಇದು ಬೆಟ್ಟದ ನೆಲ್ಲಿಕಾಯಿ ಹಾಗೂ ಇದು ಅದರ ಚೂರ್ಣ, ಮತ್ತು ಇದು ಅಮೃತ ಬಳ್ಳಿ, ಇದು ಜಟ್ಲಪ್ಪ. ಇದನ್ನು ಕನ್ನಡದಲ್ಲಿ ಬಿಳಿ ಔದರಿ ಎಂದು ಕರೆಯುತ್ತಾರೆ.
ಮೊದಲು ಇಂಪಾರ್ಟೆಂಟ್ ಉಪಯೋಗ ಕೂದಲು ಬೆಳೆಯುತ್ತೆ ಓಕೆ ಕರಿಬೇವಿಂದು ಒಂದು ಉಪಯೋಗನ ಬಿಟ್ಬಿಟ್ಟಿದ್ಯಾ ಎಲ್ಲರೂ ಹೇಳ್ತಾರೆ ಅದನ್ನ ಬಿಟ್ಟರೆ ಕೂದಲು ಉದುರೋದಿಲ್ಲ ಮತ್ತೆ ಕೂದಲು ಆಗುತ್ತೆ
ಅದು ಸುಮಾರು ಜನ ಅವರು ಮಾಡಿರ್ತಾರೆ.
ನಿಮ್ಗೆ ಯಾರು ಫೀಡ್ಬ್ಯಾಕ್ ಕೊಟ್ಟರು ಅವ್ರು ಹೇಳಿ ಸರಿ ನಿಮ್ಗೆ
Sobita and Kavna from Government High School.
ಮ್ಯಾಮ್ ಇದೇ ಸೇಮಾ ಬೇರೆ ಸ್ಕೂಲಾ GHS ಅದು ಹೊನ್ನಳ್ಳಿ ಯಾವ್ದು ನೋಡಿ ಕವನ ಯಾವ್ದು ಇದು ನೋಡಿ ಕವನ ಕೆರೆ ಅಲ್ಲ ಒಂದ್ ನಿಮಿಷ ಈ ಕಡೆ ಸೈಡ್ ಫೇಸಿಂಗ್ ಮಾಡ್ಬೇಕು ಈ ಕಡೆ ಆಯ್ತಾ ಈ ಕ್ಯಾಂಪ್ ನಡೀತಿದೆ ಈ ಕಡೆ ನೋಡ್ಕೊಂಡು ಹಿಂಗ್ ಮಾತಾಡಿ ಬರೋ ಜಡ್ಜಸ್ಗೆ ಅಷ್ಟೇ ಅಲ್ಲ ಓಕೆನಾ ಹಿಂಗ್ ಮಾತಾಡಿ ನಾನು ಆಪ್ರೇಟ್ ಮಾಡ್ಲಾ ನೀವ್ ಮಾಡ್ತೀರಾ ನಾ ಮಾಡ್ತೀನಿ ಸ್ಟಾರ್ಟ್ ಮಾಡಿ
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು ಕವನ ಸುಬ್ರಾಯ ನಾಯ್ಕ್. ನನ್ನ ಹೆಸರು ಶೋಭಿತಾ ಕಾಂತು ನಾಯ್ಕ್. ನಮ್ಮ ವಿಷಯ ಏನಂದ್ರೆ ಹೊನ್ನಳ್ಳಿ ಗ್ರಾಮದ ಮನೆ ಮದ್ದುಗಳು. ಔಷಧೀಯ ಗಿಡಮೂಲಿಕೆಗಳು ಎಂದು ಕರೆಯಲ್ಪಡುವ ಔಷಧೀಯ ಸಸ್ಯಗಳು ಇತಿಹಾಸ ಪೂರ್ವ ಕಾಲದಿಂದಲೂ ಸಾಂಪ್ರದಾಯಿಕ ಔಷಧಿ ಅಭ್ಯಾಸಗಳಲ್ಲಿ ಪತ್ತೆಯಾಗಿದೆ. ಆಯುರ್ವೇದದಲ್ಲೂ ಗಿಡಮೂಲಿಕೆಗಳ ಬಳಕೆ ಇದ್ದು ನೈಸರ್ಗಿಕವಾಗಿ ಸಿಗುವಂತಹ ಮೂಲಿಕೆಗಳನ್ನು ಬಳಸಿ ಔಷಧಿ ತಯಾರಿಸಲಾಗುತ್ತಿದೆ. ರಾಸಾಯನಿಕಗಳಿಂದ ತಯಾರಿಸಲ್ಪಟ್ಟ ಔಷಧಿಗಳಿಗಿಂತ ಇದು ಹೆಚ್ಚು ಪರಿಣಾಮಕಾರಿಯಾಗಿದೆ. ಈ ಯೋಜನೆಯ ಉದ್ದೇಶ ಏನಂದ್ರೆ ನಮ್ಮ ಗ್ರಾಮದಲ್ಲಿ ದೊರಕುವ ಔಷಧಿ ಸಸ್ಯಗಳನ್ನ ಗುರುತಿಸುವುದು, ಔಷಧಿ ಸಸ್ಯಗಳ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನ ಸಂಗ್ರಹಿಸುವುದು, ಔಷಧಿ ಸಸ್ಯಗಳ ಔಷಧಿಯ ಗುಣಗಳನ್ನ ತಿಳಿದುಕೊಳ್ಳುವುದು ಮತ್ತು ಜನರಲ್ಲಿ ಔಷಧಿ ಸಸ್ಯಗಳ ಬಗ್ಗೆ ಜಾಗೃತಿಯನ್ನ ಮೂಡಿಸುವುದೇ ಇದರ ಉದ್ದೇಶ. ನಮ್ಮ ಹೊನ್ನಳ್ಳಿಯು ಉತ್ತರ ಕನ್ನಡ ಜಿಲ್ಲೆಯ ಕುಮಟಾ ತಾಲೂಕು ಕೇಂದ್ರದಿಂದ 3 ಕಿಲೋಮೀಟರ್ ದೂರದಲ್ಲಿರುವ ಸಮುದ್ರದ ತಟದಲ್ಲಿರುವ ಊರೇ ನಮ್ಮ ಹೊನ್ನಳ್ಳಿ ಊರು. ಇದು ನಾವು ಇಷ್ಟು ಭಾಗವನ್ನು ಆರಿಸಿಕೊಂಡು ಹೊನ್ನಳ್ಳಿ ಬಹುಮಠ ಸುಶಿಷ್ಟ ಭಾಗವನ್ನು ಆರಿಸಿಕೊಂಡು ಅಲ್ಲಿನ ಜನರನ್ನ ಭೇಟಿ ಮಾಡಿ ಅವರಿಂದ ಔಷಧಿ ಸಸ್ಯಗಳ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನ ಸಂಗ್ರಹಿಸಿದೆವು. ಸಂಗ್ರಹಿಸಿದ ಮಾಹಿತಿಯನ್ನ ದಾಖಲಿಸಿದೆವು. ಅವುಗಳನ್ನ ನಿಮ್ಮ ಮುಂದೀಗ ಪ್ರಸ್ತುತಪಡಿಸುತ್ತಿದ್ದೇನೆ. ಮೊದಲನೆಯದಾಗಿ ನೆಲನೆಲ್ಲಿ. ಇದರ ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಫಿಲಾಂಥಸ್ ನೀರುರಿ. ಇದರ ಎಲೆ ಕಾಂಡವನ್ನ ಅರೆದು ಚೆನ್ನಾಗಿ ಚಿಟಿಕೆ ಜೀರಿಗೆಯೊಂದಿಗೆ ಅರೆದು 2 ಲೋಟ ನೀರಿಗೆ ಹಾಕಿ ಅದು 1 ಲೋಟ ಬರುವವರೆಗೂ ಕುದಿಸಿ ಕುಡಿಯುವುದರಿಂದ ಲಿವರ್ ಸೋಂಕು, ಅಸಿಡಿಟಿ ಅಂತಹ ರೋಗ ಇದ್ರೆ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಇನ್ನು ಎರಡನೇದಾಗಿ ಅಮೃತವಳ್ಳಿ. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರೇನಂದ್ರೆ ಟಿನೋಸ್ಪೊರಾ ಕಾರ್ಡಿಫೊರಾ. ಇದರ ಕಾಂಡವನ್ನ 1/4 ಚಮಚ ಜೀರಿಗೆ, 5 ರಿಂದ 6 ಕಾಳು ಮೆಣಸು, 1/4 ಚಮಚ ಕೊತ್ತಂಬರಿಯೊಂದಿಗೆ ಜಜ್ಜಿ ರಸವನ್ನು ತೆಗೆದು ಸೇವನೆ ಮಾಡುವುದರಿಂದ ಅಪಾಯಕಾರಿಯಾದಂತಹ ಜ್ವರಗಳಾದ ಡೆಂಗ್ಯೂ, ಹಂದಿ ಜ್ವರದಂತಹ ರೋಗ ಇದ್ರೆ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಹಾಗೂ ಈಗ ಸದ್ಯ ಬಂದಿರುವಂತಹ ಕೊರೋನಾಗೂ ಕೂಡ ಇದು ದಿವ್ಯ ಔಷಧವಾಗಿದೆ. ಕೊರೋನಾ ಸಂದರ್ಭದಲ್ಲಿ ನಾವು ಈ ಮೂಲಿಕೆಯನ್ನ ಜಾಸ್ತಿಯಾಗಿ ಬಳಸಿದ್ದೀವಿ. ಇನ್ನು ಗಾಳಿ ಗಿಡ. ಇದನ್ನು ನಮ್ಮೂರಿನಲ್ಲಿ ಹೆಚ್ಚಾಗಿ ಬಳಸ್ತಾರೆ. ಇದು ಲಕ್ವ ರೋಗಕ್ಕೆ ಮದ್ದಾಗಿದೆ. ಲಕ್ವ ರೋಗ ಯಾವ ಸಂದರ್ಭದಲ್ಲಿ ಬರುತ್ತೆ ಅಂತ ಸೂಚನೆ ಕೊಟ್ಟು ಬರೋದಿಲ್ಲ. ಅಂತಹ ಸಂದರ್ಭದಲ್ಲಿ ಏನ್ ಮಾಡ್ತೀವಿ ಅಂದ್ರೆ ಇದರ ರಸವನ್ನ ಅವರ ಮೈಗೆ ಲೇಪನ ಮಾಡ್ತೀವಿ. ಇದರಿಂದ ಅದು ಜಾಸ್ತಿಯಾಗಿ ಎಫೆಕ್ಟ್ ಆಗುದಿಲ್ಲ, ಸ್ವಲ್ಪವಾದ್ರೂ ನಿಯಂತ್ರಣದಲ್ಲಿರುತ್ತೆ ಅನ್ನುವಂತ ಸಲುವಾಗಿ ಗಾಳಿ ಗಿಡವನ್ನ ಬಳಸ್ತೀವಿ. ಇನ್ನು 4ನೇದಾಗಿ ಭದ್ರಮುಷ್ಟಿ. ಇದರ ನೀರಿಗೆ ಭದ್ರಮುಷ್ಟಿ, ಖರ್ಜೂರ, ಒಣದ್ರಾಕ್ಷಿಯನ್ನ ಹಾಕಿ ಕುದಿಸಿ ಕುಡಿಯುವುದರಿಂದ ಒಳಜ್ವರ ಇದ್ರೆ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಇನ್ನು 5ನೇದಾಗಿ ಕನ್ನೆ ಕುಡಿ. ಇದನ್ನ ನಮ್ಮೂರಿನಲ್ಲಿ ಸ್ವಾಲೆ ಕುಡಿ ಅಂತ ಕರೀತಾರೆ. ಇದು ತಂಪು ಪ್ರದೇಶದಲ್ಲಿ ಬೆಳೆಯುವಂತದ್ದು. ಇದನ್ನ ನಾವು ಚೆನ್ನಾಗಿ ತೊಳೆದು ತುಪ್ಪ, ಜೀರಿಗೆ, ಕಾಳು ಮೆಣಸು ಹಾಕಿ ಹುರಿದಿಟ್ಟುಕೊಂಡು ನಂತರ ಅದಕ್ಕೆ ಕಾಯಿ ತುರಿ ಹಾಕಿ ರುಬ್ಬಿ ಮಜ್ಜಿಗೆ ಸೇರಿಸಿ ತಂಬಳಿ ಮಾಡಿ ಸೇವನೆ ಮಾಡುವುದರಿಂದ ದೇಹದ ಉಷ್ಣವನ್ನ ನಿಯಂತ್ರಣ ಮಾಡುತ್ತೆ ಮತ್ತು ಉಷ್ಣದಿಂದ ಬರುವಂತಹ ಹೊಟ್ಟೆ ನೋವನ್ನು ಕೂಡ ಇದು ನಿವಾರಣೆ ಮಾಡುತ್ತೆ. ಇನ್ನು ಹತ್ತಿ ಚಕ್ಕೆ. ಇದರ ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಫೈಕಸ್ ರೆಸಿಮೋಸ್. ಇದೊಂದು ಮರವಾಗಿದ್ದು ಇದರ ಚಕ್ಕೆಯನ್ನು ತೆಗೆದು ಚೆನ್ನಾಗಿ ತೊಳಿಬೇಕು. ನಂತರ ಅದನ್ನ ಕಾಳು ಮೆಣಸು, ಜೀರಿಗೆ, ಶುಂಠಿಯನ್ನ ಹಾಕಿ
ಜಜ್ಜಿ ರಸವನ್ನ ತೆಗೆದು ಅದನ್ನ ಅರ್ಧ ಲೀಟರ್ ನೀರಿಗೆ ಹಾಕಿ ಅದು 1/4 ಲೀಟರ್ ಬರುವವರೆಗೂ ಕುದಿಸಿ ಕುಡಿಯುವುದರಿಂದ ದೇಹದ ಉಷ್ಣವನ್ನ ನಿಯಂತ್ರಣ ಮಾಡುತ್ತೆ, ಉಷ್ಣದಿಂದ ಬರುವಂತ ಹೊಟ್ಟೆ ನೋವನ್ನು ನಿವಾರಣೆ ಮಾಡುತ್ತೆ ಮತ್ತು ಒಣ ಕೆಮ್ಮನ್ನ ಇದು ನಿವಾರಣೆ ಮಾಡುತ್ತೆ. ಇನ್ನು 7ನೇದಾಗಿ ಹಾಡೆ ಬಳ್ಳಿ. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರೇನೆಂದ್ರೆ ಸೈಕ್ಲಿಯಾ ಫೆಲ್ಟ್ರಿಟಾ. ಇದೊಂದು ಬಳ್ಳಿ ಆಗಿದ್ದು ಮರ ಗಿಡದ ಆಶ್ರಯಿಸಿ ಬೆಳೆಯುತ್ತೆ. ಇದರ ಎಲೆ, ಕಾಯಿ, ಬೇರು ಮೂರು ಕೂಡ ಔಷಧೀಯ ಗುಣವನ್ನ ಹೊಂದಿರುವಂತದ್ದು. ಇದು ಚಿಕ್ಕ ಮಕ್ಕಳಿಗೆ ಜಂತು, ಪಿತ್ತ, ಹೊಟ್ಟೆ ನೋವು, ವಾಯು ಹಾಗೂ ಕಣ್ಣುರಿ, ತಲೆಯುರಿ ಬಂದಂತಹ ಸಂದರ್ಭದಲ್ಲಿ ಇದರ ಕಷಾಯವನ್ನ ಸೇವನೆ ಮಾಡ್ತೀವಿ. ಉಷ್ಣತೆಯಿಂದ ತಲೆ ಉರಿ, ಕಣ್ಣುರಿ ಬಂದಂತಹ ಸಂದರ್ಭದಲ್ಲಿ ಏನ್ ಮಾಡ್ಬೇಕಂದ್ರೆ ಇದರ 8-10 ಎಲೆಗಳನ್ನ ತೆಗೆದು ನೀರಿನಲ್ಲಿ ಹಾಕಿ ಕಿವುಚಿ 10 ನಿಮಿಷಗಳ ಕಾಲ ಹಾಗೆ ಇಡ್ಬೇಕು. ಅದು ನಂತರ ಗಟ್ಟಿ ಆಗತ್ತೆ. ನಂತರ ಅದನ್ನ ತಲೆಗೆ ಲೇಪನ ಮಾಡುವುದರಿಂದ ತಲೆ ಉರಿ, ಕಣ್ಣುರಿ ಇದ್ರೆ ನಿವಾರಣೆ ಆಗತ್ತೆ. ಇನ್ನು ನಾಚಿಕೆ ಮುಳ್ಳು. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರೇನೆಂದ್ರೆ ಮೈಮೋಸಾ ಪುಡಿಕಾ. ಇದು ಕಳೆಗಳ ಮಧ್ಯೆ ಬೆಳೆಯುವಂತದ್ದು. ಇದು ಮುಟ್ಟಿದರೆ ಮುದುಡಿಕೊಳ್ಳುತ್ತೆ. ಇದನ್ನು ನಾವು ಕಳೆ ಅಂತ ಕಿತ್ತೆಸಿತೀವಿ, ಆದ್ರೆ ಇದರಲ್ಲೂ ಕೂಡ ಔಷಧಿಯ ಗುಣ ಇರುತ್ತೆ. ಗಿಡದ ಎಲೆಯ ಬೇರನ್ನ ಜಜ್ಜಿ ರಸವನ್ನ ತೆಗೆದು 2-3 ಚಮಚದಷ್ಟಕ್ಕೆ 1 ಲೋಟ ನೀರು ಹಾಕಿ ಕುದಿಸಿ ಕುಡಿಯುವುದರಿಂದ ಮಲಬದ್ಧತೆ ಇದ್ರೆ ನಿವಾರಣೆ ಆಗುತ್ತೆ. 9ನೇದಾಗಿ ಕಮಲಾಕ್ಷಿ. ಇದರ ಹಣ್ಣಿನ ಪಾನಕ ಅಥವಾ ಶರಬತ್ತನ್ನ ಕುಡಿಯುವುದರಿಂದ ದಣಿವು ದೂರವಾಗ್ತದೆ ಮತ್ತು ಕಫ ಗಟ್ಟಿಯಾಗಿದ್ರೆ, ಕೆಮ್ಮಿದ್ರೆ ಇದರ ಪಾನಕವನ್ನ ಮಾಡಿ ಕುಡಿತೀವಿ. ಈ ಹಣ್ಣಿನ ರಸಕ್ಕೆ ಕಾಳು ಮೆಣಸು, ಜೀರಿಗೆ, ಕಲ್ಸಕ್ಕರೆಯನ್ನ ಹಾಕಿ ಕುದಿಸಿ ಕುಡಿಯುವುದರಿಂದ ಕೆಮ್ಮು, ಕಫ, ಜ್ವರಗಳಿದ್ರೆ ನಿವಾರಣೆ ಆಗತ್ತೆ. ಇನ್ನು 10ನೇದಾಗಿ ಸದಾಪುಷ್ಪ. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರೇನೆಂದ್ರೆ ಕ್ಯಾಥರಾಂಥಸ್ ರೋಸಿಯಸ್. ಇದರ ಎಲೆಗಳ ಕಷಾಯ ಸೇವನೆಯಿಂದ ಮಧುಮೇಹ ನಿಯಂತ್ರಣದಲ್ಲಿರುತ್ತೆ. 1 ಹಿಡಿ ನಿತ್ಯ ಪುಷ್ಪದ ಎಲೆಯ ರಸ ತೆಗೆದು ಸ್ವಲ್ಪ ಅಕ್ಕಿ ಹಿಟ್ಟನ್ನ ಸೇರಿಸಿ ಗಾಯಗಳಿಗೆ ಹಚ್ಚುವುದರಿಂದ ರಕ್ತ ಸೋರುವಿಕೆ ನಿಲ್ಲುತ್ತೆ. ಆಡುತೊಗೆ. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರೇನೆಂದ್ರೆ ಅಡಕಡ ವಾಸಿಕ. ಇದನ್ನ ಚೆನ್ನಾಗಿ ತೊಳೆದು ಬಟ್ಟೆಯಲ್ಲಿ ಕಟ್ಟಬೇಕು. ನಂತರ ಅದನ್ನ ಬಿಸಿ ಪಾತ್ರೆಯ ಮೇಲೆ, ಬೆಚ್ಚಗಿನ ಪಾತ್ರೆಯ ಮೇಲೆ ಬಟ್ಟೆ ಇಟ್ಟು ಅದ್ರ ಮೇಲೆ ಅದನ್ನ ಇಟ್ಟು ಬೇಯಿಸ್ಬೇಕು. ನಂತರ ಅದನ್ನ ಎದೆ ಭಾಗ, ಬೆನ್ನಿನ ಭಾಗಕ್ಕೆ ಇಟ್ಕೊಂಡ್ರೆ ಕಫ ಗಟ್ಟಿಯಾಗಿದ್ರೆ, ಉಸಿರಾಟದ ತೊಂದರೆಗಳಿದ್ರೆ ಅದು ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಇನ್ನು 12ನೇದಾಗಿ ಗರಿಕೆ ಹುಲ್ಲು. ಇದರ ಬೇರು ಮತ್ತು ಗುಪ್ತ ಕಾಂಡವನ್ನ ಚೆನ್ನಾಗಿ ಅರೆದು ಅದನ್ನ ಕಷಾಯ ಮಾಡಿ ಸೇವನೆ ಮಾಡುವುದರಿಂದ ಮೂತ್ರವಿಸರ್ಜನಕಾಂಗದ ಸಮಸ್ಯೆ, ಗುಪ್ತಾಂಗಗಳ ಸಮಸ್ಯೆ ಇದ್ರೆ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಹಾಗೂ ರಕ್ತ ಸೋರ್ತಾ ಇರುವಂತಹ ಸಂದರ್ಭದಲ್ಲೂ ಕೂಡ ಇದರ ರಸವನ್ನ ಹಾಕ್ತೀವಿ. ಉಗುರು ಸುತ್ತಿ ಆದಾಗ ಇದರ ರಸಕ್ಕೆ ಅರಿಶಿನ ಮತ್ತು ಸುಣ್ಣವನ್ನ ಬೆರೆಸಿ ಉಗುರು ಸುತ್ತಿಗೆ ಹಚ್ಚುವುದರಿಂದ ಉಗುರು ಸುತ್ತಿ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ತುಂಬೆ ಗಿಡ. ಇದರ ತುಂಬೆ ಎಲೆಯನ್ನ ಜಜ್ಜಿ ರಸವನ್ನ ತೆಗೆದು ಅದನ್ನ ಕುಡಿಯುವುದರಿಂದ ಥಂಡಿ, ಕೆಮ್ಮಿದ್ರೆ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಮತ್ತು ಗಂಟಲು ನೋವಿಗೆ ತುಂಬೆ ಸೊಪ್ಪು, ಕಾಳು ಮೆಣಸು ಹಾಗೂ ಬೆಳ್ಳುಳ್ಳಿ ಹೆಸರನ್ನ ಜಜ್ಜಿ ಅದನ್ನ ಗಂಟಲಿಗೆ ಲೇಪನ ಮಾಡಿ ಇಟ್ಕೊಳ್ಳೋದ್ರಿಂದ ಗಂಟಲು ನೋವಿದ್ರೆ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ದೊಡ್ಡಪತ್ರೆ. ಇದನ್ನ ನಾವು ಸಾಂಬ್ರಾಣಿ ಎಲೆ, ಸಾವಿರ ಸಂಬಾರು ಎಲೆ ಅಂತಾನೂ ಕರೀತಾರೆ. ಇದರ 5-6 ಎಲೆಗಳನ್ನ ತೆಗೆದುಕೊಂಡು ಒಂದು ಬದಿ ಮಾತ್ರ ಬಿಸಿ ಮಾಡ್ಬೇಕು. ನಂತರ ಅದರ ರಸವನ್ನ ತೆಗೆದು ಕುಡಿಯುವುದರಿಂದ ಥಂಡಿ, ಕೆಮ್ಮು, ಶ್ವಾಸಕೋಶದ ಸಮಸ್ಯೆಗಳಿದ್ರೆ ದೂರ ಆಗುತ್ತೆ. ಇನ್ನು ತುಳಸಿ. ಇದೆಲ್ಲ ನಿಮ್ಗೆ ಗೊತ್ತಿರುವಂತದ್ದೇ. ಇದರಲ್ಲಿ ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿ ಹೆಚ್ಚಾಗಿರೋದ್ರಿಂದ ದಿನಕ್ಕೆ 1ರಂತೆ 1 ಎಲೆಯನ್ನ ತಿಂದ್ರೆ ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿಯು ಹೆಚ್ಚಾಗುತ್ತೆ ಮತ್ತು ಬಾಯಿಯ ದುರ್ಗಂಧ ವಾಸನೆಗಳಿದ್ರು ಕೂಡ ದೂರ ಆಗುತ್ತೆ. ಇದರ ವೈಜ್ಞಾನಿಕ ಹೆಸರು ಅಜಟೇರಿಯಸ್ ಇಂಡಿಕಾ. ಈ ಬೇವಿನ ಎಲೆ ಹೊಗೆಯಿಂದ ಸೊಳ್ಳೆ ಮತ್ತು ಕೀಟಗಳನ್ನ ಓಡಿಸಬಹುದು ಮತ್ತು ಇದನ್ನ ಧಾನ್ಯಗಳಲ್ಲಿ ಹುಗಿದಿಡುವುದರಿಂದ ಧಾನ್ಯಗಳಿಗೆ ಕೀಟಗಳು ಬರುವುದಿಲ್ಲ. ಮತ್ತು ಇದರ ಎಲೆಯನ್ನ ಕುದಿಸಿ ಎಲೆಯನ್ನ ತೆಗೆದು ನೀರಿನಿಂದ ತಲೆ ತೊಳೆದ್ರೆ ತಲೆ ತುರಿಕೆ, ಹೊಟ್ಟುಗಳಿದ್ರೆ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಹಾಗೆ ಬ್ರಾಹ್ಮಿ. ಇದರ ಸೈಂಟಿಫಿಕ್ ನೇಮ್ ಸೆಂಟೆಲ್ಲಾ ಏಸಿಯಾಟಿಕಾ. ಇದನ್ನ ನಾವು ನಮ್ಮ ಗ್ರಾಮೀಣ ಭಾಷೆಯಲ್ಲಿ ಒಂದೆಲ್ಗ ಅಂತ ಕರಿತೀವಿ. ಇದು ತಂಪು ಪ್ರದೇಶದಲ್ಲಿ ಚೆನ್ನಾಗಿ ಮತ್ತು ಹೆಚ್ಚಾಗಿ ಬೆಳೆಯುತ್ತೆ. ಇದನ್ನ ನಾವು ಹಿಂದಿನ ಕಾಲದಿಂದಲೂ ಒಂದು ಔಷಧಿ ಗಿಡವನ್ನಾಗಿ ಬಳಸ್ಕೊತಾ ಬಂದಿದ್ದೀವಿ. ಇದನ್ನ ಬಳಸುವ ವಿಧಾನ ಕಷಾಯ ಅಥವಾ ತಂಬಳಿ ರೂಪದಲ್ಲಿ ಕೂಡ ಸೇವನೆ ಮಾಡಬಹುದು. ಇದರಿಂದ ನೆನಪಿನ ಶಕ್ತಿ ಹೆಚ್ಚಾಗುತ್ತೆ ಮತ್ತು ದೇಹದಲ್ಲಿರುವ ಉಷ್ಣತೆ ಕೂಡ ನಿಯಂತ್ರಣದಲ್ಲಿರುತ್ತೆ. ಇನ್ನು ಅರಿಶಿನ. ಒಂದು ಚಮಚ ಅರಿಶಿನ ಪುಡಿಗೆ ಕಡಲೆ ಹಿಟ್ಟನ್ನ ಮಿಕ್ಸ್ ಮಾಡಿ ಮುಖಕ್ಕೆ ಹಚ್ಚುವುದರಿಂದ ಮುಖದ ಕಾಂತಿ ಹೆಚ್ಚಾಗತ್ತೆ. ಮತ್ತು 1 ಇಂಚು ಅರಿಶಿನ ಕೊಂಬಿಗೆ ತೆಂಗಿನ ತುರಿ, 4 ಮೆಣಸು, ಹುಳಿ ಮತ್ತು ಉಪ್ಪನ್ನು ಮಿಶ್ರಣ ಮಾಡಿ ಸೇವನೆ ಮಾಡುವುದರಿಂದ ನಂಜು ಕೂಡ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಹಾಗೆ ಪೇಲೆ ಪುಡಿ. ಈ ಎಳೆ ಪುಡಿಯನ್ನ ಬಾಯಲ್ಲಿ ಒಗೆಯುವುದರಿಂದ ಬಾಯಿಯ ಹುಣ್ಣುಗಳು ಕೂಡ ನಿವಾರಣೆ ಆಗುತ್ತೆ. ಹಾಗೆ ಅದಕ್ಕೆ ತೆಂಗಿನ ತುರಿ, ಜೀರಿಗೆ, ಕಾಳು ಮೆಣಸನ್ನು ಹಾಕಿ ರುಬ್ಬಿ ಅದಕ್ಕೆ ಮಜ್ಜಿಗೆ, ಬೆಲ್ಲ ಮತ್ತು ಉಪ್ಪನ್ನು ಸೇರಿಸಿ ಸೇವನೆ ಮಾಡುವುದರಿಂದ ಅತಿಸಾರ ಭೇದಿ ಕೂಡ ನಿಯಂತ್ರಣದಲ್ಲಿರುತ್ತೆ. ಉತ್ತರಾಣಿ. ಈ ಎಲೆಯನ್ನ ರಸವನ್ನ ತೆಗೆದು ಗಾಯಗಳಿಗೆ ಹಚ್ಚುವುದರಿಂದ ಗಾಯಗಳ
ತೆಗೆದು ಅದನ್ನ ಪುಡಿ ಮಾಡಿ ಅದಕ್ಕೆ ಕಾಳು ಮೆಣಸು ಮತ್ತು ಜೇನುತುಪ್ಪವನ್ನು ಹಾಕಿ ಸೇವನೆ ಮಾಡೋದ್ರಿಂದ ಕೆಮ್ಮು ಕೂಡ ನಿರ್ವಹಣೆ ಆಗುತ್ತೆ. ಹಾಗೆ ಅಲ್ವೇರಾ, ಈ ಅಲ್ವೇರಾದ ಲೋಳೆಗಳನ್ನ ಕಣ್ಣು ಮತ್ತು ನೆತ್ತಿಯ ಮೇಲೆ ಇಟ್ಟುಕೊಳ್ಳುವುದರಿಂದ ಕಣ್ಣು ಮತ್ತು ನೆತ್ತಿಯ ಉರಿ ಕೂಡ ಕಡಿಮೆ ಆಗತ್ತೆ. ಹಾಗೆ ಇದನ್ನು ಸೇವಿಸುವುದರಿಂದ ಜೀರ್ಣಕ್ರಿಯೆ ಕೂಡ ಸುಧಾರಣೆ ಆಗತ್ತೆ. ಇದರಿಂದ ತಿಳಿದು ಬರುವುದೇನೆಂದ್ರೆ 75% ಜನರು ಔಷಧಿಯನ್ನ ಬಳಸಿ ವೈದ್ಯರ ಬಳಿಗೆ ಹೋಗದೆ ತಮ್ಮ ಆರೋಗ್ಯವನ್ನು ಕಾಪಾಡಿಕೊಳ್ತಾ ಇದ್ದಾರೆ. ಇನ್ನು 25% ಜನರು ಅವುಗಳನ್ನ ಮೊದಲು ಬಳಸಿ ನಂತರ ವೈದ್ಯರ ಬಳಿಗೆ ಹೋಗ್ತಾ ಇದ್ದಾರೆ. ಇನ್ನು ಬಳಕೆಯಾಗುವ ಸಸ್ಯಗಳ ಭಾಗಗಳ ಶೇಕಡಾ ಪ್ರಮಾಣವನ್ನು ನೋಡೋದಾದ್ರೆ 75% ಎಲೆಯ ಭಾಗ, 10% ಪೂರ್ಣ ಸಸ್ಯ, 5% ಕಾಂಡ, ಕಾಯಿ ಹಣ್ಣು 10%. ಇನ್ನು ರೋಗಗಳಿಗೆ ಬಳಕೆಯಾಗುವ ಮೂಲಿಕೆಗಳ ಶೇಕಡಾ ಪ್ರಮಾಣವನ್ನು ನೋಡೋದಾದ್ರೆ ಚರ್ಮ ರೋಗಕ್ಕೆ 10%, 25% ಹೊಟ್ಟೆ ಸಮಸ್ಯೆಗೆ, 25% ಶ್ವಾಸಕೋಶದ ಸಮಸ್ಯೆಗೆ, 5% ಮೂತ್ರಜನಕಾಂಗದ ಸಮಸ್ಯೆಗೆ, 15% ಜ್ವರದ ಸಮಸ್ಯೆಗೆ ಹಾಗೂ 20% ಕುಷ್ಠ ನಿಯಂತ್ರಣಕ್ಕಾಗಿ ಬಳಸ್ತಾ ಇದ್ದೇವೆ. ಕೊನೆಯದಾಗಿ ನಮ್ಮೂರಿನಲ್ಲಿ ಇರುವ ನಮಗೆ ಗೊತ್ತಿರದ ಎಷ್ಟೋ ಔಷಧಿ ಸಸ್ಯಗಳ ಪರಿಚಯ ನಮಗಾಯಿತು ಮತ್ತು ಅವುಗಳ ಉಪಯೋಗವನ್ನು ನಾವು ತಿಳಿದುಕೊಂಡೆವು. ನಮ್ಮ ಸುತ್ತಮುತ್ತಲಿನಲ್ಲಿ ಇಂತಹ ಗಿಡಮೂಲಿಕೆ ಸಸ್ಯಗಳನ್ನು ಪರಿಣಾಮಕಾರಿಯಾಗಿ ಹಾಗೂ ಸುಸ್ಥಿರವಾಗಿ ಬಳಸಿಕೊಂಡು ವೈದ್ಯರ ಬಳಿಗೆ ಹೋಗದೆ ನಮ್ಮ ಆರೋಗ್ಯವನ್ನು ನಾವು ಕಾಪಾಡಿಕೊಳ್ಳಬಹುದು. ಈ ಯೋಜನೆಯನ್ನು ಪ್ರಸ್ತುತಪಡಿಸಲು ಅವಕಾಶ ನೀಡಿದ ಭಾರತೀಯ ವಿಜ್ಞಾನ ಸಂಸ್ಥೆಗೆ ಹಾಗೂ ನನಗೆ ಇದರ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಿದ ನನ್ನೆಲ್ಲ ಗುರುವೃಂದದವರಿಗೆ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತೇನೆ ಮತ್ತು ಈ ಗಿಡಗಳ ಉಪಯೋಗವನ್ನು ತಿಳಿಸಿದ ನನ್ನ ಊರಿನ ಸಮಸ್ತ ನಾಗರಿಕರಿಗೂ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತೇನೆ ಮತ್ತು ನಾಟಿ ವೈದ್ಯರಾದ ಗೌರಿ ಶಾಂತರಾಮ್ ನಾಯ್ಕ್ ಇವರಿಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತೇನೆ. ಧನ್ಯವಾದಗಳು.
ನೀವೆಲ್ಲ ಹೇಳಿದ್ರಿ
ಜನರಲ್ಲಿ ಜಾಗೃತಿಯನ್ನು ಮೂಡಿಸಬೇಕು.
ನಾವು ಅಲ್ಲಿ ಫುಡ್ ಅರೇಂಜ್ ಮಾಡಿದ್ಮೇಲೆ ಆರ್ಗ್ಯಾನಿಕ್ ಫುಡ್ ಇದೆಲ್ಲ ಮೂಲಕನೂ ಅದ್ರಲ್ಲಿ ಹಾಕಿದ್ವಿ. ಅಂದ್ರೆ ನೀವು ಅದನ್ನ ಸರಿಯಾಗಿರುತ್ತೆ. ನಾನು ಅಲ್ಲಿ ಊಟ ಮಾಡೋ ನೋಡಿದ್ನಲ್ಲ, ಅದಕ್ಕೋಸ್ಕರ ಏನ್ ಮಾಡಿದೀನಿ ಅಂದ್ರೆ ಅಲ್ಲಿ ಸಿರಿ ಪೊಂಗಲ್ತಿಲ್ಲ ಅಲ್ಲಿ ಗಿಡ ತಿಂದಿದ್ದೀನಿ. ಸಾಯಂಕಾಲ ಹೋದಾಗ ಎಲ್ಲರೂ ಅಲ್ಲಿ ಹೋಗಿ ಖಾಲಿ ಮಾಡ್ಬೇಕು. ಜಾಪತ್ರ ಇದೆ, ದ್ರಾಕ್ಷಿ ಇದೆ, ಗೋಡಂಬಿ ಇದೆ, ಬೆಲ್ಲ ಇದೆ, ಆರ್ಗ್ಯಾನಿಕ್ ಬೆಲ್ಲ, ಇನ್ನು ಏನೆಲ್ಲ ಇದೆ
ಇದನ್ನು ನಾವು ಲೈವ್ ಆಗಿ ತಂದಿದ್ದೀವಿ.
ನೀವು 15 ನಿಮಿಷ ಆಲ್ರೆಡಿ ತಗೊಂಡಿದ್ದೀರ. ಅಲ್ಲಿ ಇಟ್ಕೊಂಡಿದ್ದೀರಿ, ಕೌಂಟರ್ ಇದೆಯಲ್ಲ, ಈಗ ಹೋದಾಗ ಪ್ರಶ್ನೆಗಳಿದೆಯಪ್ಪ ಯಾರಿಗಾದ್ರು? ಯಾರು ಸಮಾವೇಶ ಮಾಡಕ್ಕೆ ಯಾರು ಲೇಟ್ ಬಂದವರು ಯಾರು? ನಾನು ಊಟ ಮಾಡುವಾಗ ಕೆಲವರು ಹೇಳಿದೆ ಮೊಸರನ್ನ ಜಾಸ್ತಿ ತಿನ್ಬೇಡಿ, ನನ್ ಮಾತು ಕೇಳಿಲ್ಲ. ಮೊಸರನ್ನ ತಿಂದು ಚೆನ್ನಾಗಿದೆ ಅಂತ. ಓಕೆ, ಏನ್ವೇ ಎಕ್ಸರ್ಸೈಸ್ ಮಾಡೋಣ. ಶತ್ರು ಅದೇ ಹೇಳ್ತಿದ್ದಾರೆ ಸರ್, ಸಮರೈಸ್ ಮಾಡೋದು ಹೇಳ್ಬೇಕು, ನಿದ್ದೆ ಮಾಡ್ತಿದ್ದಾರೆ ಕೆಲವರೆಲ್ಲ ಅಂತ. ಯಾಕಂದ್ರೆ ಚಪ್ಪಾಳಿ ಶಬ್ದ ಬರಲೇ ಇಲ್ಲ. ರೋಲ್ ಮಾಡೋಣ.
ಅಲ್ಲಿ ಸೈನ್ ಹಾಕ್ಬಿಟ್ಟಿ ಹೋಗಿ. ಪರಾಸ್
Krishna Deepak from Government High School.
ಯಾವ ಸ್ಕೂಲ್ ಪುಟಿ ಇದೆಯಲ್ಲ? 1 hour ಪ್ರಜ್ನಾ ದೀಪಕ್. ಓಕೆ, ಇಲ್ಲಿ ನಿಂತ್ಕೊಂಡ್ ಮಾಡ್ಬೇಕು. ಓಕೆನಾ? ಇಲ್ಲಿ ನಿಂತ್ಕೊಂಡೆ, ಕ್ಯಾಮ್ರಾ ಇದೆ, ಈ ಕಡೆ ನೋಡ್ಕೊಂಡ್ ಮಾತಾಡ್ಬೋದು ಒಬ್ಬರಿಗೆಲ್ಲ. ಓಕೆ. ಓಕೆನಾ? ಇದು ಆಪರೇಟ್ ಮಾಡ್ತೀಯಾ ಇಲ್ಲ ಮಾಡ್ಲಾ? ನೀವು ಮಾಡ್ಕೊತೀರಾ? ಓಕೆ.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನನ್ನ ಹೆಸರು ಪ್ರಜ್ಞಾ ದೀಪಕ್ ನಾಯ್ಕ್. ನಾನು ಉತ್ತರ ಕನ್ನಡ ಜಿಲ್ಲೆಯ ಹೊನ್ನಾವರ ತಾಲೂಕಿನ ಹೊತ್ಕೆಶೀರೂರು ಗ್ರಾಮದ ಸರ್ಕಾರಿ ಪ್ರೌಢಶಾಲೆ ಹೊತ್ಕೆಶೀರೂರು ಕಡ್ಡೀರಿನಲ್ಲಿ 9ನೇ ತರಗತಿಯಲ್ಲಿ ವ್ಯಾಸಂಗವನ್ನು ನಡೆಸುತ್ತಿದ್ದೇನೆ. ಹೊತ್ಕೆಶೀರೂರು ಭಾರತದ ಕರ್ನಾಟಕ ರಾಜ್ಯದ ಉತ್ತರ ಕನ್ನಡ ಜಿಲ್ಲೆಯ ಹೊನ್ನಾವರ ತಾಲೂಕಿನಲ್ಲಿರುವ ಒಂದು ಚಿಕ್ಕ ಕುಗ್ರಾಮ. ಇದು ಹೊತ್ಕೆಶೀರೂರು ಪಂಚಾಯತ್ ವ್ಯಾಪ್ತಿಗೆ ಬರುತ್ತದೆ. ಇದು ಬೆಳಗಾವಿ ವಿಭಾಗಕ್ಕೆ ಸೇರಿದ್ದು. ಇದು ಜಿಲ್ಲಾ ಕೇಂದ್ರ ಕಾರವಾರದಿಂದ ದಕ್ಷಿಣಕ್ಕೆ 85 ಕಿಲೋಮೀಟರ್ ದೂರದಲ್ಲಿದೆ. ಪಿನ್ ಕೋಡ್ 581338 ಆಗಿದೆ. ಹೊತ್ಕೆಶೀರೂರು ಗ್ರಾಮದ ನೆರೆಯ ಗ್ರಾಮ ಚಂದಾವರವಾಗಿದ್ದು ಇದು ಪ್ರಸಿದ್ಧ ಶ್ರೀ ಹನುಮಂತ ದೇವರ ದೇವಸ್ಥಾನಕ್ಕೆ ಪ್ರಸಿದ್ಧವಾಗಿದೆ. ಹೊತ್ಕೆಶೀರೂರು ಉತ್ತರಕ್ಕೆ ಕುಮಟ, ದಕ್ಷಿಣಕ್ಕೆ ಭಟ್ಕಳ ತಾಲೂಕು, ಪೂರ್ವಕ್ಕೆ ಸಿದ್ದಾಪುರ ತಾಲೂಕು ಹಾಗೆ ಉತ್ತರಕ್ಕೆ ಅಂಕೋಲ ತಾಲೂಕಿನಿಂದ ಸುತ್ತುವರಿದಿದೆ. ಶಿರಸಿ, ಸಾಗರ, ಕಾರವಾರ, ಕೋಟ ಹೊತ್ಕೆಶೀರೂರಿಗೆ ಸಮೀಪದಲ್ಲಿರುವ ನಗರಗಳಾಗಿವೆ. ಆರೋಗ್ಯವೇ ಮಹಾಭಾಗ್ಯ. ಆದರೆ ಇಂದಿನ ಆಧುನಿಕ ಯುಗದಲ್ಲಿ ಆರೋಗ್ಯವೇ ಮಹಾಭಾಗ್ಯ ಎಂಬಂತಾಗಿದೆ. ನಮ್ಮ ದೇಶವು ವಿಶೇಷವಾಗಿ ಸಂಸ್ಕೃತಿ, ಭಾಷೆ, ಉಡುಗೆ ತೊಡುಗೆ ಹಾಗೂ ಆಹಾರದಲ್ಲೂ ವೈವಿಧ್ಯತೆಯನ್ನ ಹೊಂದಿದೆ. ಭಾರತ ಆಹಾರ ಪದ್ಧತಿಯಲ್ಲಿ ಆರೋಗ್ಯದ ಹಿತದೃಷ್ಟಿಯಿಂದ ವೈಜ್ಞಾನಿಕತೆಯನ್ನು ಕೂಡ ಅಳವಡಿಸಲಾಗಿದೆ. ಋತುಮಾನಗಳಿಗನುಗುಣವಾಗಿ ಸ್ಥಳೀಯ ವೈವಿಧ್ಯತೆಗಳಿಗನುಗುಣವಾಗಿ ನಮ್ಮ ದೇಶದ ಆಹಾರ ಪದ್ಧತಿ ಜಾರಿಯಲ್ಲಿರುತ್ತದೆ. ಮಾತು ಬಲ್ಲವನಿಗೆ ಜಗಳವಿಲ್ಲ, ಊಟ ಬಲ್ಲವನಿಗೆ ರೋಗವಿಲ್ಲ. ಅದೆಷ್ಟೋ ದೊಡ್ಡ ಕಾಯಿಲೆಗಳನ್ನ ಆಹಾರದಿಂದಲೇ ಗುಣಪಡಿಸುವ ಶಕ್ತಿಯನ್ನ ಹೊಂದಿದೆ. ಅಂತಹ ಕೆಲವು ಆಹಾರ ಪದಾರ್ಥವನ್ನ ನಮ್ಮ ಸ್ಥಳೀಯ ಪ್ರದೇಶದಲ್ಲಿ ಸಾಕಷ್ಟು ಜನರನ್ನ ಸಂದರ್ಶಿಸಿ ಮಾಹಿತಿಗಳನ್ನ ಕಲೆ ಹಾಕಿದ್ದೇನೆ. ಅದನ್ನ ಇದೀಗ ನಿಮ್ಮ ಮುಂದೆ ಪ್ರಸ್ತುತಪಡಿಸಲು ಬಯಸುತ್ತೇನೆ. ಮೊದಲನೇದಾಗಿ ಮರಕೆಸು. ಇದರಿಂದ ನಾವು ಪತ್ರೊಡೆಯನ್ನ ತಯಾರಿಸ್ತೇವೆ. ಈ ಪತ್ರೊಡೆಯನ್ನ ತಯಾರಿಸಲಿಕ್ಕೆ ನಾವು ಮರಕೆಸುವಿನ ಎಲೆಯನ್ನ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ. ಇದನ್ನ ಸೇವಿಸುವುದರಿಂದ ನಮ್ಗೆ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ, ಇದನ್ನ ಬಳಕೆ ಮಾಡೋದ್ರಿಂದ ಆರೋಗ್ಯವು ಉತ್ತಮವಾಗಿರ್ತದೆ. ಈ ಮರಕೆಸು ಹೊಟ್ಟೆಯಲ್ಲಿರುವಂತಹ ಕಲ್ಮಶಗಳನ್ನ ಹೊರಹಾಕ್ತದೆ. ಹೇಗೆಂದ್ರೆ ಇದರಲ್ಲಿ ಸೆಲ್ಯುಲೋಸ್ ಇರುತ್ತದೆ. ಇದರಲ್ಲಿ ಆರೋಗ್ಯಕರವಾದ ಪೋಷಕಾಂಶಗಳಿದ್ದು ಇದು ದುರ್ಬಲತೆಯನ್ನ ಕಡಿಮೆಗೊಳಿಸುತ್ತದೆ. ಹಾಗೆ ಜೀರ್ಣಕ್ರಿಯೆಯ ಶಕ್ತಿಯನ್ನು ಕೂಡ ಹೆಚ್ಚಿಸುತ್ತದೆ. ಮಹಿಳೆಯರಲ್ಲಿ ಹಾರ್ಮೋನ್ಗಳ ಸಂಖ್ಯೆಯನ್ನು ಕೂಡ ಹೆಚ್ಚಿಸುತ್ತದೆ. ಇದು ಕೆಂಪು ರಕ್ತ ಕಣಗಳನ್ನು ಕೂಡ ಹೆಚ್ಚಿಸುತ್ತದೆ. ಹಾಗೆ ಇದರಲ್ಲಿ ವಿಟಮಿನ್ ಎ ಹೇರಳವಾಗಿದೆ. ಹಾಗೆ ಇದು ಕಣ್ಣಿನ ಆರೋಗ್ಯಕ್ಕೆ ತುಂಬಾ ಉತ್ತಮವಾಗಿದೆ. ಇದು ಕ್ಯಾನ್ಸರ್ ನಂತಹ ಕಣಗಳನ್ನ ದೂರ ಮಾಡ್ತದೆ. ಹಾಗೆ ಇದು ಮಧುಮೇಹ ಕಾಯಿಲೆಗಳಿಗೆ ರಾಮಬಾಣವಾಗಿದೆ. ಎರಡನೇದಾಗಿ ಕಳಲೆ ಬಾಜಿ. ಇದನ್ನ ನಾವು ತಯಾರಿಸಲಿಕ್ಕೆ ಕಳಲೆಯನ್ನ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ. ಇದನ್ನ ನಾವು ಸೇವಿಸುವುದರಿಂದ ನಮ್ಗೆ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ, ಇದನ್ನ ಸೇವಿಸುವುದರಿಂದ ಸಾಕಷ್ಟು ಪ್ರಮಾಣದ ಪ್ರೋಟೀನ್, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್, ನಾರಿನಾಂಶ, ಖನಿಜಗಳು ಸಿಗುತ್ತದೆ. ಇದರಲ್ಲಿ ಕೊಬ್ಬು ಮತ್ತು ಸಕ್ಕರೆಯ ಪ್ರಮಾಣ ಕಡಿಮೆ ಇರುವುದರಿಂದ ಮಧುಮೇಹಿಗಳಿಗೆ ಇದು ಅತ್ಯುತ್ತಮ ಆಹಾರ ಪದಾರ್ಥವಾಗಿದೆ. ಇದು ಕರುಳಿನ ಸಮಸ್ಯೆ ನಿಭಾಯಿಸಲು ಸಹಾಯ ಮಾಡ್ತದೆ. ಹಾಗೆ ಕ್ಯಾನ್ಸರ್ ಅನ್ನು ಕೂಡ ತಡೆಗಟ್ಟುತ್ತದೆ. ಇದು ದೇಹಕ್ಕೆ ಅಗತ್ಯವಾದ ವಿಟಮಿನ್ ಎ, ವಿಟಮಿನ್ ಇ, ವಿಟಮಿನ್ B6, ಮೆಗ್ನೀಸಿಯಂ ಅನ್ನು ಒಳಗೊಂಡಿದೆ. ಇದು ನಿತ್ಯ ಆಹಾರದಲ್ಲಿ ಸೇವಿಸಿಕೊಂಡರೆ ಮಾರಣಾಂತಿಕ ಕಾಯಿಲೆಗಳಿಂದ ದೂರ ಉಳಿಯಬಹುದು. ಹಾಗೆ ಹೃದಯ ಸಂಬಂಧಿ ಕಾಯಿಲೆಗಳಿಗೂ ಕೂಡ ಇದು ಉತ್ತಮವಾಗಿದೆ. ಶ್ವಾಸಕೋಶದ ಆರೋಗ್ಯಕ್ಕೂ ಕೂಡ ಉತ್ತಮವಾಗಿದೆ. ಗರ್ಭಾಶಯದ ತೊಂದರೆ ಇರುವವರು ಇದನ್ನ ಸೇವಿಸುವುದರಿಂದ ಆರೋಗ್ಯವನ್ನ ಕಾಪಾಡ್ಕೊಳ್ಬಹುದು. ಮೂರನೇದಾಗಿ ಅಣಬೆ ಸಾರು. ಇದನ್ನ ನಾವು ತಯಾರಿಸಲಿಕ್ಕೆ ಅಣಬೆಯನ್ನ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ. ಇದನ್ನ ನಾವು ಸೇವಿಸುವುದರಿಂದ ನಮ್ಗೆ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ, ಇದು ದೇಹದ ತೂಕವನ್ನ ಕಡಿಮೆ ಮಾಡ್ತದೆ. ಹಾಗೆ ದೇಹಕ್ಕೆ ಉತ್ತಮ ನ್ಯೂಟ್ರಿಷನ್ ಅನ್ನ ನೀಡ್ತದೆ. ವಿಟಮಿನ್ ಡಿ ಇದರಲ್ಲಿ ಹೆಚ್ಚಾಗಿರುವುದು ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿಯನ್ನು ಕೂಡ ಹೆಚ್ಚಿಸ್ತದೆ. ಕೊಲೆಸ್ಟ್ರಾಲ್ ಅನ್ನ ಕಡಿಮೆ ಮಾಡ್ತದೆ. ಮಧುಮೇಹ ನಿಯಂತ್ರಣ ಮಾಡ್ತದೆ. ಕ್ಯಾನ್ಸರ್ ಅನ್ನು ನಿಯಂತ್ರಿಸ್ತದೆ. ಹಾಗೆ ಲೈಂಗಿಕ ಆರೋಗ್ಯವನ್ನು ಕೂಡ ಕಾಪಾಡ್ತದೆ. ಐದನೇದಾಗಿ ಒಂದೆಲಗ ಚಟ್ನಿ. ಇದನ್ನ ನಾವು ಮಾಡ್ಲಿಕ್ಕೆ ಬಳಸಿಕೊಂಡ ಭಾಗದ ಹೆಸರು ಒಂದೆಲಗದ ಎಲೆ. ಇದು ದೇಹಕ್ಕೆ ಹಾಗೆ ಮನಸ್ಸಿಗೆ ತಂಪು ತರುವುದು ಮಾತ್ರವಲ್ಲದೆ ಚಿಕ್ಕ ಮಕ್ಕಳಲ್ಲಿ ಸ್ಮರಣ ಶಕ್ತಿಯನ್ನ ಕೂಡ ಹೆಚ್ಚಿಸುತ್ತದೆ. ಒತ್ತಡ ಹಾಗೂ ಆತಂಕವನ್ನ ಕೂಡ ನಿಯಂತ್ರಿಸುತ್ತದೆ. ನೆನಪು, ಏಕಾಗ್ರತೆಯನ್ನು ಕೂಡ ವೃದ್ಧಿ ಮಾಡ್ತದೆ. ಅಪಸ್ಮಾರ ಸ್ಥಿತಿಯನ್ನು ಕೂಡ ನಿವಾರಿಸುತ್ತದೆ. ಹಾಗೆ ಮನಸ್ಸಿಗೆ ಶಾಂತಿ ಹಾಗೂ ವಿರಾಮವನ್ನು ಕೂಡ ನೀಡುತ್ತದೆ. ಇದು ನರ ವ್ಯವಸ್ಥೆಯನ್ನು ಶಾಂತವಾಗಿರುವುದರ ಜೊತೆಗೆ ಹೃದಯದ ಮತ್ತು ಶ್ವಾಸಕೋಶದ ಆರೋಗ್ಯವನ್ನು ಕೂಡ ಕಾಪಾಡುತ್ತದೆ. ಮೆದುಳಿನ ಅಂಗಾಂಶಗಳಿಗೆ ಶಕ್ತಿಯನ್ನ ನೀಡುತ್ತದೆ. ಹಾಗೆ ಖಿನ್ನತೆ ದೂರ ಮಾಡುವ ಲಕ್ಷಣ ಕೂಡ ಇದರಲ್ಲಿದೆ. ದೈಹಿಕ ಹಾಗೂ ಮಾನಸಿಕ ಆರೋಗ್ಯಕ್ಕೆ ಇದು ತುಂಬಾ ಉತ್ತಮವಾಗಿದೆ. 6ನೇದಾಗಿ ಉಪ್ಪಾಗಿ ತುಪ್ಪ. ಇದನ್ನ ನಾವು ತಯಾರಿಸಲಿಕ್ಕೆ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ ಅಂತ ಹೇಳಿದ್ರೆ ಉಪ್ಪಾಗಿ ಬೀಜವನ್ನ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ. ಇದನ್ನ ನಾವು ಸೇವಿಸೋದ್ರಿಂದ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ ಮಾನವ ದೇಹದ ಕೊಬ್ಬು ಮತ್ತು ಬೊಜ್ಜನ್ನ ಕರಗಿಸಲು ಸಹಾಯ ಮಾಡ್ತದೆ. ಇದು ಹೈಡ್ರಾಕ್ಸಿ ಅಂಶವನ್ನ ಒಳಗೊಂಡಿದೆ. ಇದು ಸೇವಿಸುವುದರಿಂದ ಮಲಬದ್ಧತೆ, ಮುಟ್ಟಿನ ತೊಂದರೆ ಹಾಗೂ ಜಂತುಹುಳದ ಬಾಧೆಯನ್ನು ಕೂಡ ನಿವಾರಿಸುತ್ತದೆ. ಈ ಹಣ್ಣಿನ ಬೀಜವು ಕೊಬ್ಬಿನಂಶವನ್ನ ಹೊಂದಿದ್ದು ಇದರಿಂದ ತುಪ್ಪವನ್ನ ತೆಗೆಯಲಾಗ್ತದೆ. 7ನೇದಾಗಿ ಮುರುಗಲು ಸಾಂಬಾರು. ಇದನ್ನ ನಾವು ಈ ಪದಾರ್ಥವನ್ನ ತಯಾರು ಮಾಡಲು ನಾವು ಮುರುಗಲನ್ನ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ. ಇದನ್ನ ಸೇವಿಸುವುದರಿಂದ ನಮ್ಮ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ ಇದು ಪಿತ್ತ ದೋಷವನ್ನ ಕಡಿಮೆ ಮಾಡ್ತದೆ. ಹಾಗೆ ಇದು ಪಿತ್ತಕ್ಕೆ ತುಂಬಾ ಶಮನಕಾರಿಯಾಗಿದೆ. ಇದು ಹಲವಾರು ಪೌಷ್ಟಿಕಾಂಶಗಳನ್ನ ಒಳಗೊಂಡಿದೆ. ಹೇರಳವಾಗಿ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಗಳನ್ನ ಹೊಂದಿದೆ. ನಾರಿನಾಂಶ ಕೂಡ ಇದರಲ್ಲಿ ಹೇರಳವಾಗಿದೆ. 8ನೇದಾಗಿ ಕಾಡು ಬಸಳೆಯ ಚಟ್ನಿ. ಇದನ್ನ ನಾವು ಮಾಡ್ಲಿಕ್ಕೆ ಬಳಸ್ಕೊಂಡ ಭಾಗ ಯಾವ್ದಂತ ಹೇಳಿದ್ರೆ ಕಾಡು ಬಸಳೆ ಎಲೆ. ಇದನ್ನ ನಾವು ಸೇವಿಸುವುದರಿಂದ ನಮ್ಮ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ ಮೂತ್ರಪಿಂಡದ ಸಮಸ್ಯೆಯನ್ನ ತಡೆಗಟ್ಟುತ್ತದೆ, ಮಧುಮೇಹ ನಿವಾರಕ, ಜೀರ್ಣಾಂಗ ವಿಭಾಗದ ಸಮಸ್ಯೆಗಳಿಗೆ ರಾಮಬಾಣ, ಹಾಗೆ ಇದು ಸೋಂಕನ್ನ ಕೂಡ ನಿವಾರಿಸುತ್ತದೆ, ರಕ್ತದೊತ್ತಡದ ಸಮಸ್ಯೆಯನ್ನ ಕಡಿಮೆ ಮಾಡ್ತದೆ. ಕಾಡು ಬಸಳೆಯ ಎಲೆಯನ್ನು ಹಣೆಗೆ ಹಚ್ಚುವುದರಿಂದ ತಲೆನೋವನ್ನು ಕೂಡ ಕಡಿಮೆ ಮಾಡ್ತದೆ. ಹಾಗೆ ಹೃದಯವು ಆರೋಗ್ಯಯುತವಾಗಿರುತ್ತದೆ. ಮೂತ್ರದಲ್ಲಿನ ರಕ್ತ ಮತ್ತು ಕೀವು ಮುಂತಾದ ಸಮಸ್ಯೆಗಳಿಗೆ ಇದು ರಾಮಬಾಣವಾಗಿದೆ. ಹಾಗೆ 9ನೇದು ತೊಗಟೆ ಸೊಪ್ಪಿನ ಪಲ್ಯ. ಇದನ್ನ ತೊಗಟೆ ಸೊಪ್ಪನ್ನ ನಾವು ಈ ಪದಾರ್ಥವನ್ನ ತಯಾರು ಮಾಡ್ಲಿಕ್ಕೆ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ. ಇದರಿಂದ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ ಕಿಬ್ಬೊಟ್ಟೆ ನೋವನ್ನ ಹೋಗಲಾಡಿಸ್ತದೆ, ಹಾಗೆ ರಕ್ತವನ್ನು ಕೂಡ ಶುದ್ಧೀಕರಿಸ್ತದೆ, ಬೊಜ್ಜನ್ನ ಕರಗಿಸ್ತದೆ, ಹಾಗೆ ತ್ವಚೆಯ ಆರೋಗ್ಯಕ್ಕೂ ಒಳ್ಳೆಯದು, ಆಮಶಂಕೆ ಕಾಯಿಲೆಯನ್ನು ಕೂಡ ಇದು ನಿವಾರಿಸ್ತದೆ. ಹಾಗೆ 10ನೇದಾಗಿ ಬೇವಿನ ಎಲೆಯ ಚಟ್ನಿ. ಇದನ್ನ ನಾವು ತಯಾರಿಸಲಿಕ್ಕೆ ಬಳಸ್ಕೊಂಡ ಭಾಗದ ಹೆಸರು ಬೇವಿನ ಎಲೆ. ಇದನ್ನ ಸೇವಿಸುವುದರಿಂದ ನಮ್ಗೆ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ ರಕ್ತದಲ್ಲಿನ ಸಕ್ಕರೆಯ ಮಟ್ಟವನ್ನ ನಿಯಂತ್ರಿಸುತ್ತದೆ, ಹೊಟ್ಟೆಯ ಹುಣ್ಣುಗಳನ್ನು ಕೂಡ ಕಡಿಮೆಗೊಳಿಸುತ್ತದೆ. ಇದು ಅಸಿಡಿಟಿ, ಮೂತ್ರ ಹಾಗೂ ಚರ್ಮ ರೋಗಿಗಳಿಗೆ ಅತ್ಯುತ್ತಮ ಆಹಾರವಾಗಿದೆ. ಇದು ಜೀರ್ಣಕ್ರಿಯೆಯನ್ನ ಸುಧಾರಿಸುತ್ತದೆ, ಆಯಾಸವನ್ನ ನಿವಾರಿಸುತ್ತದೆ, ಹಾಗೆ ಗಾಯಗಳನ್ನು ಕೂಡ ನಿವಾರಿಸುವ ಶಕ್ತಿ ಇದರಲ್ಲಿದೆ. 11ನೇದಾಗಿ ಬೆಟ್ಟದ ನೆಲ್ಲಿಕಾಯಿಯ ಉಪ್ಪಿನಕಾಯಿ. ಬೆಟ್ಟದ ನೆಲ್ಲಿಕಾಯಿಯನ್ನ ನಾವು ಉಪ್ಪಿನಕಾಯಿ ತಯಾರು ಮಾಡ್ಲಿಕ್ಕೆ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ. ಇದನ್ನ ಸೇವಿಸುವುದರಿಂದ ನಮ್ಗೆ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ ಇದರಲ್ಲಿ ದೇಹಕ್ಕೆ ಅಗತ್ಯವಾದ ಕ್ಯಾಲ್ಸಿಯಂ ಹೇರಳವಾಗಿರ್ತದೆ, ಮಲಬದ್ಧತೆಯನ್ನು ನಿವಾರಣೆ ಮಾಡ್ತದೆ, ಕೇಶ ಸುಂದರವಾಗಿ ಮತ್ತೆ ಉದ್ದವಾಗಿ ಬೆಳೆಯಲು ಕೂಡ ಇದು ಸಹಾಯ ಮಾಡ್ತದೆ. 12ನೇದಾಗಿ ವಿಟಾಮಿನ್ ಸೊಪ್ಪಿನ ಪಲ್ಯ. ವಿಟಾಮಿನ್ ಸೊಪ್ಪನ್ನ ಬಳಸ್ಕೊಂಡಿದ್ದೇವೆ ನಾವು. ಇದರಿಂದ ಆರೋಗ್ಯದ ಮೇಲೆ ಏನ್ ಮಹತ್ವ ಅಂತ ಹೇಳಿದ್ರೆ ಇದರಲ್ಲಿ ವಿಟಮಿನ್ ಅಂಶ ಹೆಚ್ಚಿರುತ್ತದೆ, ದೇಹದ ಉಷ್ಣಾಂಶವನ್ನ ಕಡಿಮೆಗೊಳಿಸ್ತದೆ, ಹಾಗೆ ಬಾಯಾರಿಕೆಯನ್ನು ಕೂಡ ಕಡಿಮೆ ಮಾಡ್ತದೆ. ಆರೋಗ್ಯವೇ ಮಹಾಭಾಗ್ಯ ಎಂಬಂತೆ ಸಕಲ ರೋಗಗಳಿಗೆ ಆಹಾರವೇ ಮದ್ದು. ಸುಖಿ ಜೀವನಕ್ಕೆ ಆಹಾರ ಕ್ರಮವೇ ಸೂತ್ರ. ಕೇವಲ ಸಂತುಲಿತ ಆಹಾರವನ್ನು ಸೇವಿಸುವುದರ ಮೂಲಕ ಆರೋಗ್ಯಯುತವಾದ ಜೀವನ ಶೈಲಿಯನ್ನ ನಡೆಸಬಹುದು ಎಂಬ ಆಸೆಗಳೊಂದಿಗೆ ನನಗೆ ಸಿಕ್ಕ ಅವಕಾಶಕ್ಕಾಗಿ ವಂದಿಸುತ್ತಾ ನನ್ನ ಈ ಪ್ರೆಸೆಂಟೇಷನ್ಗಾಗಿ ಪೂರ್ಣ ವಿರಾಮವನ್ನ ನೀಡ್ತೇನೆ.
Again, there are also scientific name, genus and species name, species name, capital.
ಇಟಾಲಿಕ್ಸ್ ಅಂತ ಬರುತ್ತೆ, ಇಟಾಲಿಕ್ಸ್ ಅಲ್ಲಿ ಬರ್ಬೇಕು.
ಮೊದಲನೇದು ಕ್ಯಾಪಿಟಲ್ ಲೆಟರ್ ಶುರು ಮಾಡುತ್ತೆ. 2ನೇ ಪದ ಇದೆಯಲ್ಲ, ಸೆಕೆಂಡ್ ವರ್ಡ್ ಸೆಕೆಂಡ್ ನೇಮ್, ಅದು ಸ್ಮಾಲ್ ಲೆಟರ್ಸ್ ಅಲ್ಲಿ ಇರ್ಬೇಕು. ಅದು ಕರೆಕ್ಟ್ ವೇ ಆಫ್
ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದಾಗ ಆ ಥರ ಚೇಂಜ್ ಮಾಡ್ಕೊಂಡು. ಓಕೆ, ನೋ ಕ್ವೆಶ್ಚನ್. ಒಂದು ಕ್ವೆಶ್ಚನ್.
ವಿಟಾಮಿನ್ ಸೊಪ್ಪು ಅಂತ ಹೇಳಿದ್ರೆ ಅದನ್ನ ಕನ್ನಡದಲ್ಲಿ ಏನದು?
ವಿಟಾಮಿನ್ ಸೊಪ್ಪು ಅಂತಾನೆ ನಮ್ ಕಡೆ ಕರೀತಾರೆ.
ಉಪ್ಪಾಗೆ ತುಪ್ಪ ಅಂತ ಮಾಡಿದ್ರಲ್ಲ, ಅದು ಉಪ್ಪಾಗೆ ಮತ್ತೆ ಕೋಕಮ್ ಇದೆಯಲ್ಲ, ಅದು ಎರಡಕ್ಕೂ ಒಂದೇ ಸೈಂಟಿಫಿಕ್ ಹೆಸರು ಹೇಳಿದ್ರಲ್ಲ,
ಅದೆರಡು ಬೇರೆ
ಉಪ್ಪಾಗಿದ್ದು
ವ್ಯಾಕ್ಸಿನಿಯಾ ಕಾಂಬೋಜಿಯಾ
ಗ್ಲಾಸನಿಯಾ ಗಮ್ಮಿಗುಟ್ಟ.
ಹೆಸರು ಅದ್ರ ಹೆಸರು ತಪ್ಪಾಗಿದೆ, ಅದು ಒಂದ್ಸರಿ ಕರೆಕ್ಟ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಮತ್ತೆ ನೀವು ಇದನ್ನ ಹೇಳಿದ್ದೀರಲ್ಲ ವಿಟಮಿನ್ ಸೊಪ್ಪು ಅಂತ, ಅದಕ್ಕೆ ಚಕ್ರಮಣಿ ಅನ್ನೋ ಸೊಪ್ಪು ಅಂತ ಹೆಸರು ಇರ್ಬೋದು.
One of the friends, but this is part of. Okay, that's it. Keep it near, na? When you come this way, camera is here. You should point to the camera.
I am Monica of grade 8 from Ideal International Techno School, Chennai. With me, Manogna of grade 8 is here. Today we are here to present the document about butterflies of Ideal Campus. I chose this topic because I am interested in butterflies. Butterfly is a flying insect with a small body, large and colorful wings. Like all other insects, butterflies have 6 legs and 3 main body parts: head, thorax, and abdomen. Butterflies have closely related to moths, which also have colorful wings. And which also have colorful wings. Of the moths are also large in proportion to their bodies and antennas. Our study area: to study the butterflies, different species of butterflies, to analyze the butterflies. Materials and methods. Study area: Ideal Campus. The study area is located in Garga village, which is 5 kilometers away from the Maluk headquarters, Chennai. Our study is mainly dominated with areca plantations and corn fields. Materials: we use butterflies of Bengaluru and South Indian butterflies and butterflies of India. Field guide to identify butterflies. Methods: we use it to random samples to all-out search method to observe the butterflies. We also collected few dead ones to identify the names of the butterflies. As you can see in the graph, totally we documented 25 species of butterflies. Here, swallowtail we documented 5 butterflies, and whites and yellows we documented 5 butterflies. Blues we documented 5 butterflies. Brush-footed we documented 9 butterflies, and swallow and skippers we documented only 1 butterfly, that is common grass dart. Out of 25 species, common grass, yellow, and plain tiger are very common. Common grass dart is the only one skipper, and it is also our rarest butterfly for us. Recommendations: our study area has been planted with more rose plants to attract more butterflies. Based on my studies, I conclude like this. My recommendations are: we have to plant more rose and nectar plants to attract butterflies. And we should not use pesticides and insecticides. Conclusion: our study gave us total list of 25 species of butterflies. Our school campus hosts a fair number of butterfly species. Our school campus is potential enough to develop a butterfly garden. Here, the reference books are here. I thank Dr. T. Ramachandran sir for the opportunity to present here. Second, I will thank to our Ideal Management and Chennai sir. Third, I thank to all faculties and friends who have supported me, and also my family members. Thank you.
one question here. How big is your school campus? What is the area that you have in your school campus? Roughly. Maybe 2 acres? Hey? No, I have come here, that's why I'm asking you. Okay. So have you started tree plantation work in your school campus? You have to start now. Okay, okay. Because it's a new school that's coming up in a new area. A new building is coming up. Is your building work over? No, it is still under construction. Okay, that's why many butterflies did come to you. So you plant more trees, right? Most plants, automatically butterflies will come to your place. Okay, any questions? One question.
You had mentioned that you found dead wings of the butterflies, and I want to know what you identified from that, and how did you find the dead wings? Was it just on the floor, or did you actually see the butterflies?
Thank you.
Meeting, closure, and earlier from ideal school.
Good afternoon.
Waiting.
Good afternoon to you all. Myself, Vidya Patel, from grade 8. With me, Abhinav of grade 7 and Lochan of grade 8 from Ideal International School. We are all interested in birds, so we are taken. Today, we are here to present the birds of Garga Lake. Birds are warm-blooded organisms with four chambered hearts. Birds are the only organisms to have the feathers and beaks. They have all-shaded legs. We choose this topic to study the bird diversity in the Durga Lake. Many birds are seen around the lake and few on the water surface itself.
Object. Objectives: to study the bird diversity in the Garga, to analyze the bird species abundance in the study site, to understand the occurrence status of birds, that is migratory and resident.
Materials and methods. Study area. Garga Lake. The study area is located in Garga village, which is just one kilometer away from the Ideal School. Our study site is mainly dominated with areca farm and farm fields. Materials we used earth and wild wetland birds of Bangalore by Vijayalal MV and Chatvesh Shat are birds of Indian subcontinent by Grimmett.
Hmm. Wait a minute. Entire screen.
No, no, no.
No, skip. Hmm? Skip. Can you यहाँ से
Why it's that?
What?
अटक गई स्टक
यहाँ से टूट रहा है वहाँ से
यहाँ पे नहीं हो रहा है
sometimes it's working, sometimes it's not.
the system is stuck
अगर दिखा रहा है यहाँ पे लेकिन सिर्फ नहीं हो रहा हाँ फेस In YouTube it is showing, but the slides are not moving. In YouTube it is working. We have. Yes, sir.
It was coming. हाथ ही लगा
किसमें?
It's not moving.
In YouTube it is showing. The slides are not moving.
Huh?
In YouTube it is showing. YouTube. Will begin. Correct na? Unmuted.
ಇದರ ಆರೋಗ್ಯಕ್ಕೆಂದು ನಮ್ಮ ಅಜ್ಜಿ ಸ್ವಾದಿಷ್ಟ ತಂಬಳಿಯನ್ನು ನೋಣ ಬೆಳೆಸಿದಳು. ಇದು ಯಾವುದರದ್ದು ಅಜ್ಜಿ ಎಂದು ಕೇಳಿದಾಗ ನಮ್ಮ ಮನೆಯ ಬೇಲಿ ಅಂಚಿನಲ್ಲಿ ಅಲ್ಲಲ್ಲಿ ಚಿಗುರುತ್ತಿರುವ ನೆಲನೆಲಿ ಗಿಡ ಎಂದು ತೋರಿಸಿಕೊಟ್ಟರು. 2022 ಸೆಂಟರ್ ಫಾರ್ ಇಕಾಲಜಿಕಲ್ ಸೈನ್ಸ್ ಮುಂತಾದವರು ನೀಡಿದ ಪ್ರಾಜೆಕ್ಟ್ ಬಗ್ಗೆ ಚಿಂತಿಸುತ್ತಿದ್ದ ನನಗೆ ಇಂತಹ ಕಾಡು ಸಸ್ಯಗಳನ್ನು ಗುರುತಿಸಿ ಪಟ್ಟಿ ಮಾಡಿ ಜನರಿಗೆ ಏಕೆ ತಿಳಿಸಬಾರದು ಎಂಬ ವಿಚಾರ ನನ್ನಲ್ಲಿ ಮೊಳೆಯಿತು. ಈ ವಿಷಯದಲ್ಲಿ ಆಸಕ್ತಿ ತಳೆದ ನಾವಿಬ್ಬರು ವಿಜ್ಞಾನ ಶಿಕ್ಷಕರ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಕೈಗೊಂಡ ಯೋಜನೆಯೇ ಆರೋಗ್ಯ ಸಂವರ್ಧನೆಗೆ ಆಹಾರದ ಭಾಗವಾಗಿ ಕಾಡು ಸಸ್ಯಗಳ ಬಳಕೆ. ಈ ಯೋಜನೆಯ ಬಗ್ಗೆ ಕಾರ್ಯಾಚರಣೆ ಕೈಗೊಂಡ ಮೊಟ್ಟ ಮೊದಲಿಗೆ ಜನರಲ್ಲಿರುವ ಪ್ರಾಥಮಿಕ ಮಾಹಿತಿಯನ್ನು ತಿಳಿದುಕೊಳ್ಳಲು ಈ ರೀತಿಯ ನಿಗದಿತ ನಮೂನೆಯಲ್ಲಿ ಸೆಪ್ಟೆಂಬರ್ 10ರಿಂದ 30ರವರೆಗಿನ ಅವಧಿಯಲ್ಲಿ ಮುಸ್ಲಿಂ ಪಂಚಾಯತದ ಬೆಂಡೆಗದ್ದೆ, ಶಿಂಗನಹಳ್ಳಿ, ಗಣೇಶ್ ನಗರ ಮುಂತಾದ ಹಳ್ಳಿಗಳಿಗೆ ಭೇಟಿ ಕೊಟ್ಟು ಈ ರೀತಿಯಲ್ಲಿ ಮಾಹಿತಿ ಸಂಗ್ರಹಿಸಿದ್ದೇವೆ. ನಾವು ಭೇಟಿ ಮಾಡಿದ ಮನೆಗಳ ಸಂಖ್ಯೆ 105, ಒಟ್ಟು ಜನಸಂಖ್ಯೆ 445, ಕುಟುಂಬದ ವಿಧಗಳಲ್ಲಿ ಕೃಷಿಕರು 68, ಕೃಷಿಯೇತರು 37. ಅಡುಗೆಯಲ್ಲಿ ಕಾಡು ಸಸ್ಯಗಳ ಬಳಕೆ ಕುಟುಂಬ ಪ್ರತಿನಿತ್ಯ ಹಾಲು, ವಾರಕ್ಕೊಮ್ಮೆಯಾದರೂ 57 ಕುಟುಂಬ. ಆಹಾರದ ಮೂಲವಾಗಿ ಬಳಕೆಯಾಗುವ ಕಾಡು ಸಸ್ಯಗಳಲ್ಲಿ ಕನಿಷ್ಠ 10ಕ್ಕಿಂತ ಹೆಚ್ಚಿನ ಕಾಡು ಸಸ್ಯಗಳ ಪರಿಚಯ ಇರುವ ಕುಟುಂಬ 13, ಇಲ್ಲದ ಕುಟುಂಬ 92. ಈ ಮಾಹಿತಿಯನ್ನು ವಿಶ್ಲೇಷಣೆಗೆ ಒಳಪಡಿಸಿದಾಗ ಕೃಷಿಕರು 65%, ಕೃಷಿಯೇತರು 35%. ಕಾಡು ಸಸ್ಯಗಳ ಬಳಕೆಯಲ್ಲಿ ಭಾಗದ ಉಪಯೋಗದ ಮಾಹಿತಿ ಇರುವವರು 50%, ತೊಗಟೆ 17%, ಹೂವು 14%, ಬೇರು 12%, ಕಾಯಿ ಅಥವಾ ಹಣ್ಣು 75%. ಈ ಮಾಹಿತಿ ಕ್ರೋಢೀಕರಣದಿಂದ ನಮಗೆ ತಿಳಿದು ಬಂದ ವಿಷಯವೇನೆಂದರೆ ಸಾಕಷ್ಟು ಜನರಿಗೆ ಎಲ್ಲಾ ಗಿಡಗಳ ಪರಿಚಯವಾಗಿಲ್ಲ ಮತ್ತು ಆರೋಗ್ಯದ ಮೇಲೆ ಅವುಗಳ ಮಹತ್ವತೆ ಸಂಪೂರ್ಣವಾಗಿ ತಿಳಿದಿಲ್ಲ ಎಂಬುದು. ಅಕ್ಟೋಬರ್ 2ರಂದು ನಾವು ಸ್ಥಳೀಯವಾಗಿ ಸಿಗುವ ಆಹಾರಕ್ಕಾಗಿಯೇ ಬಳಕೆಯಾಗುವ ಕಾಡು ಸಸ್ಯಗಳನ್ನು ಕಲೆ ಹಾಕಲು ಮೊಟ್ಟ ಮೊದಲಿಗೆ ಅರಿಶಿಣ ಕಾಮಲೆ ಮತ್ತು ಕೌಂಟಿ ಔಷಧಿ ನೀಡುವ ಗಂಗಜ್ಜ ಮನೆಯವರಿಗೆ ಭೇಟಿ ನೀಡಿ ಮಕ್ಕಳಿಗೆ ನೆನಪಿನ ಶಕ್ತಿ ಹೆಚ್ಚಿಸುವ ಒಂದೆಲಗ, ಜಂಡಿಸ್ಗೆ ಬಳಸುವ ಎಲವರಿಗೆ, ರೋಗ ನಿರೋಧಕ ಶಕ್ತಿಗೆ ಬಳಸುವ ನೆರನಲ್ಲಿ ಹೀಗೆ 12 ಸಸ್ಯಗಳ ಭಾಗಗಳನ್ನು ತೆಗೆದುಕೊಂಡು ಅವುಗಳ ಲೋಕಲ್ ನೇಮ್ ಮತ್ತು ಉಪಯೋಗಗಳೊಂದಿಗೆ ನೋಟ್ ಮಾಡಿಕೊಂಡು ಈ ರೀತಿಯಲ್ಲಿ ಹೈಪೇರಿಯಂ ತಯಾರಿಸಿದ್ದೇವೆ. ಹೀಗೆ ಅಕ್ಟೋಬರ್ ರಜೆಯಲ್ಲಿ 8 ದಿವಸ ಬೆಂಡೆಗದ್ದೆ, ಶಿಂಗನಹಳ್ಳಿ, ಹೊಸಳ್ಳಿ ಮುಂತಾದ ಹಳ್ಳಿಯ ಸ್ಥಳೀಕರ ಜೊತೆ ಬೆಟ್ಟಕಾಡುಗಳಲ್ಲಿ ಓಡಾಡಿ 55ಕ್ಕೂ ಹೆಚ್ಚಿನ ಕಾಡು ಸಸ್ಯಗಳನ್ನು ಕಲೆ ಹಾಕಿದ್ದೇವೆ. ಕೆಲವು ಒಣಗಿಸಿ ಇರುವ, ಸಂರಕ್ಷಿಸಿರುವ ಆಹಾರ ಬಗ್ಗೆ ಬಳಕೆಯಾಗುವ ಕಾಡು ಸಸ್ಯಗಳನ್ನು ಈ ರೀತಿಯಲ್ಲಿ ಸಂಗ್ರಹಿಸಿ ಅರಣ್ಯ ಕಾಲೇಜಿನ ಬಯೋಟೆಕ್ನಾಲಜಿ ಪ್ರೊಫೆಸರ್ ಆದ ಅನಿಲ್ ಹೆಗಡೆ ಅವರಿಗೆ ತೋರಿಸಿ ಅವರ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಅವರಿಗೆ ತೋರಿಸಿ ಅವುಗಳ ವೈಜ್ಞಾನಿಕ ಹೆಸರಿನೊಂದಿಗೆ ಉಪಯೋಗಗಳ ಬಗ್ಗೆ ಮಾಡಿದ್ದೇವೆ. ಈ ರೀತಿಯಲ್ಲಿ ಸಂಗ್ರಹಿಸಿದ ಮಾಹಿತಿಯನ್ನು MS ಕಾಲೇಜಿನ ಬಯೋಟೆಕ್ನಾಲಜಿ ಪ್ರೊಫೆಸರ್ ಆದ ಅನಿಲ್ ಹೆಗಡೆ ಅವರಿಗೆ ತೋರಿಸಿ ಅವರ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಬಳಕೆಗೆ ತಕ್ಕಂತೆ ಈ ರೀತಿಯಲ್ಲಿ ವಿವರಿಸಿದ್ದೇವೆ. ಒಣಗಿಸಿ ಬಳಸುವ ಪದಾರ್ಥಗಳು ಹಲಸು, ಮೂಡಾಗಿಲು, ಮುರುಗಲು, ವಾಟೆ, ಉಪ್ಪಗೆ. ತಂಬಳಿ ಪದಾರ್ಥಗಳು ನೆಲ್ಲಿಪುಡಿ, ನಾಚಿಕೆ ಪುಡಿ, ಮೆಣಸು ಇತ್ಯಾದಿ. ಚಟ್ನಿಯಾಗಿ ಬಳಸುವ ಪದಾರ್ಥಗಳು ಪತ್ರೆ, ಕರಿಬೇವು, ಕೆಸ, ನೆಲ್ಲಿ ಇತ್ಯಾದಿ. ಪಲ್ಯ ಪದಾರ್ಥಗಳು ಚಗಟೆ, ಚೌತಿಕುಂಡಿಗೆ, ಕರಡಿ ಸೊಪ್ಪು, ಕಡಲೆ, ಹಲಸು. ದೋಸೆ ಅಥವಾ ತಿಂಡಿ ಪದಾರ್ಥಗಳು ಹಲಸು, ಹೆಬ್ಬಲಸು, ಕೆಸಗಂಟಿನ ದೋಸೆ ಇತ್ಯಾದಿ. ಉಪ್ಪಿನಕಾಯಿ ಕಾಡು ಸಸ್ಯಗಳು ಮಾವು, ನೆಲ್ಲಿ, ಮಕ್ಕಳ ತಾಯಿ ಬೇರು, ಕಳಲೆ, ಜುಮ್ಮ. ಹಣ್ಣಿನ ಪದಾರ್ಥಗಳು ಗೇರು, ಗುಡ್ಡೆಗೇರು, ಹಲಗೆ ಹಣ್ಣು, ದುರುಕಲು, ನೇರಳೆ, ಬಿಳಿಮುಳ್ಳೆ ಹಣ್ಣು ಮತ್ತು ಕರಿಮುಳ್ಳೆ ಹಣ್ಣು, ಗಂಜಲು, ಬಿಕ್ಕಿ ಹಣ್ಣು, ಚಳ್ಳೆ ಹಣ್ಣು. ದ್ರವ ಪದಾರ್ಥ ಜ್ಯೂಸಿಯಾಗಿ ಬಳಕೆ ಹುಳುಗಳು, ವಾಟೆ, ಸೋರೆ ಪುಡಿ, ಚಟ್ನಿ ಇತ್ಯಾದಿ. ಕಷಾಯವಾಗಿ ಬಳಕೆ ಒಂದೆಲಗ, ಹಾಲು ಬಳ್ಳಿ ಬೇರು, ಅರಳಿನ ಇತ್ಯಾದಿ. ಚಿತ್ರಾನ್ನದ ಪಲಾವುಗಳಿಗೆ ಕರಿಬೇವು, ಲವಂಗ, ದಾಲ್ಚಿನ್ನಿ, ಕಾಳು ಕೊತ್ತಂಬರಿ ಇತ್ಯಾದಿ. ತುಪ್ಪವಾಗಿ ಬಳಕೆ ಉಪ್ಪಗೆ ಹಾಗೂ ಮಂಗಲು. ಪಲ್ಯದ ಕಾಳು ಸಸ್ಯಗಳು ಚಗಟೆ ಸೊಪ್ಪಿನ ಪಲ್ಯ, ಬಾಳೆ ಎಲ್ಲಿ ಪಲ್ಯಕ್ಕೆ ಹಾಕುವ ಒಗ್ಗರಣೆಯನ್ನು ಹಾಕಿ ಹುರಿಯುತ್ತ ಎಣ್ಣೆಯೊಂದಿಗೆ ಚಗಟೆ ಸೊಪ್ಪನ್ನು ಹಾಕಿ ಹುರಿದ ಸೊಪ್ಪು, ಬೆಲ್ಲ, ಹುಳಿ ಪುಡಿ ಸೇರಿಸುವುದು. ಲಾಭ ಬಹಳವಿದೆ. ಉಪಯೋಗ: ಇದು ಉತ್ತಮ ಜೀರ್ಣಕಾರಿ ವಸ್ತು, ಕಣ್ಣಿನ ಕಾಂತಿಯನ್ನು ಹೆಚ್ಚಿಸುತ್ತದೆ ಇತ್ಯಾದಿ. ಮಾಹಿತಿ ನೀಡಿದವರು ಲೋಲಾ ಭಟ್ಟನಿಮ್ಮನೆ. ಉಪ್ಪಿನಕಾಯಿ ಕಾಳು ಸಸ್ಯಗಳು ಮಾವು, ಮಾವಿನ ಅಪ್ಪೆ ಹುಳಿ, ಮಾವಿನಕಾಯಿ ಅಪ್ಪೆ ಹುಳಿ, ಮಾವಿನಕಾಯಿಯ ಗೊಜ್ಜು, ಮಾವಿನಕಾಯಿ ತೊಕ್ಕು, ಮಾವಿನಕಾಯಿಯ ಉಪ್ಪಿನಕಾಯಿ, ಮಾವಿನಕಾಯಿಯ ಚಟ್ನಿ. ಉಪಯೋಗಗಳು: ಉತ್ತಮ ವಿಟಮಿನ್ ಸಿ ಹೊಂದಿರುವುದರಿಂದ ಆಹಾರ ಪದಾರ್ಥ, ಹೊಟ್ಟೆಗೆ ಶೋಧೆಯನ್ನು ನಿವಾರಿಸುತ್ತದೆ. ಹಣ್ಣಿನ ಪದಾರ್ಥಗಳು: ನೇರಳೆ, ಹಣ್ಣಿನ ಕೂಡಿನ ತಂಬಳಿ. ಇದರ ಪುಡಿಯನ್ನು ಜೇನಿನೊಂದಿಗೆ ಹುರಿದು ಕಾಯಿ ತುರಿ ಹಾಕಿ ಬೀಸಿದರೆ ಮತ್ತು ಮಜ್ಜಿಗೆಯನ್ನು ಸೇರಿಸಿದರೆ ತಂಬಳಿ ಸಿದ್ಧವಾಗುತ್ತದೆ. ಉಪಯೋಗ: ಸಕ್ಕರೆ ಅಂಶವನ್ನು ನಿಯಂತ್ರಿಸುತ್ತದೆ. ಅಶ್ವಗಂಧ ಕಾಳು ಸಸ್ಯಗಳು: ಒಂದೆಲಗ, ಇದನ್ನು ಬ್ರಾಹ್ಮಿ ಎಂದು ಕರೆಯುತ್ತಾರೆ. ಇದರ ತಂಬಳಿ ಮಾಡುತ್ತಾರೆ, ಕಷಾಯ ಮಾಡುತ್ತಾರೆ. ಒಂದೆಲಗ ಲೇಹ. ಉಪಯೋಗ: ಜ್ವರ, ಕೆಮ್ಮು, ನೆಗಡಿಗೆ ದಿವ್ಯ ಔಷಧಿ. ತುಪ್ಪ ತಯಾರಿಕೆಯ ಕಾಳು ಸಸ್ಯಗಳು: ಉಪ್ಪಾಗೆಯ ಉಪಯೋಗ: ಶಕ್ತಿವರ್ಧಕವಾಗಿ ಬಳಕೆ. ಹಬ್ಬ ಹರಿದಿನಗಳಲ್ಲಿ ಕಾಳು ಸಸ್ಯಗಳ ಬಳಕೆ: ಶಿವರಾತ್ರಿಯಲ್ಲಿ ಕಾಳಮೆಣಸಿನ ಹಿಟ್ಟಿನ ಹಾಲೇರು, ಯುಗಾದಿಯಲ್ಲಿ ಬೇವು ಬೆಲ್ಲ, ನಗರ ಪಂಚಮಿಯಲ್ಲಿ ಹಲಸಿನ ತೊಳೆಯ ಒಬ್ಬಟ್ಟು, ಮಾರಿಕೇಶನ ಕುತ್ತೊಡೆ, ಕೃಷ್ಣಾಷ್ಟಮಿಯಲ್ಲಿ ಹಲಸಿನ ತೊಳೆಯ ಉಂಡೆ, ಭೂಮಿ ಹುಣ್ಣಿಮೆಯಲ್ಲಿ ಅತ್ಯಂಬರ ಸೊಪ್ಪಿನ ಪಲ್ಯ, ಆರೇಶಿನ ಎಲೆಯಲ್ಲಿ ಕಡುಬು, ಗಣೇಶ ಚತುರ್ಥಿಯಲ್ಲಿ ಹತ್ತಿರಸ ಕಣಗಿಲೆ ರಸದಲ್ಲಿ ಚಕ್ಕುಲಿ ಇತ್ಯಾದಿ. ಋಷಿ ಪಂಚಮಿಯಲ್ಲಿ ಕಿವಿ ತಂಬಳಿ, ದೀಪಾವಳಿಯಲ್ಲಿ ಕಾಡು ಬಾಳೆಯಲ್ಲಿ ಕಡುಬು, ಲವಂಗದ ಎಲೆ, ಶ್ರಾದ್ದದಲ್ಲಿ ಬಾಳೆ ದಿಂಡಿನ ಉಳಿಲು, ಹಲಸಿನ ಎಲೆ ಒಬ್ಬಟ್ಟಿನ ಇಡ್ಲಿ, ಮದುವೆ ಮುಂಜಾನೆ ಹಲಸಿನ ಹಪ್ಪಳ, ಚಿಪ್ಸ್ ಇತ್ಯಾದಿ. ವಿಆರ್ ಭಟ್ಟನ ಮನೆ ಹಾಗೂ ರವಿಯಂಗಡಿ ಬೆಂಡೆಗದ್ದೆ ಅವರು ಅತ್ಯಂಬರ ಸೊಪ್ಪು, ಇಲಿಕಿವಿ, ಅಂಡಿ ಹಣ್ಣು, ಹಲಗೆ ಹಣ್ಣು, ಹಪ್ಪಳದಾಯಿ ನಶಿಸುತ್ತಿರುವ ಕಾಳು ಸಸ್ಯಗಳನ್ನು ತಿಳಿಸಿಕೊಡ್ತಾರೆ. ನಾವು ಕಾಲಗಳ ಪ್ರಕಾರದಲ್ಲಿ ಈ ರೀತಿಯಲ್ಲಿ ವಿಂಗಡಿಸಿ: ಎಲ್ಲಾ ಕಾಲದಲ್ಲೂ ಎಲೆವರೆಗೆ ಹತ್ತಿ ಕುಡಿ, ಬೆಳ್ಳೆಹೂ, ಒಂದೆಲಗ ಇತ್ಯಾದಿಗಳು. ಮಳೆಗಾಲದಲ್ಲಿ ಎಲೆ ಬಿದಿರು, ಬೆಟ್ಟದ ಅಣಬೆ, ಸೋರಳೆ ಕುಡಿ ಇತ್ಯಾದಿ. ಚಳಿಗಾಲದಲ್ಲಿ ನೆಲ್ಲಿ, ಕಾಡು ಮೆಣಸು, ಕಾಡರ್ಶಿನ ಇತ್ಯಾದಿ. ಬೇಸಿಗೆಗಾಲದಲ್ಲಿ ಮಾವು, ಸಂಪಿಗೆ, ಗುಡ್ಡೆಗೇರು, ಹಲಸು, ನುಗ್ಗುಗಳು ಇತ್ಯಾದಿ. ನವೆಂಬರ್ 3ರಂದು ನಮ್ಮ ಶಾಲೆಯಲ್ಲಿ ಪಾಲಕರಿಗೆ ಕಾಡು ಸಸ್ಯಗಳ ಬಳಕೆ ವಿಚಾರ ಸಂಕಿರಣ ಏರ್ಪಡಿಸಿದೆವು. ನವೆಂಬರ್ 5ರಂದು ಸ್ಥಳೀಯ ಶ್ರೀಶಕ್ತಿ ಸಂಘದವರಿಗೆ ಕೆಲವು ತಂಬಳಿ ಪಲ್ಯ ಮಾಡಿ ತೋರಿಸುವ ಕಾರ್ಯಕ್ರಮವನ್ನು ಹಮ್ಮಿಕೊಂಡಿದ್ದೆವು. ಹಾಗೂ ನವೆಂಬರ್ 10ರಿಂದ 30ರವರೆಗೆ ಇಂತಹ ಪಾಂಪ್ಲೆಟ್ಸ್ ಅನ್ನು ಹಂಚಿ ಮನೆ ಮನೆಗೆ ತೆರಳಿ ಕಾಡು ಸಸ್ಯಗಳನ್ನು ಆಹಾರದ ಭಾಗವಾಗಿ ಮತ್ತು ನಶಿಸುತ್ತಿರುವ ಕಾಡು ಸಸ್ಯಗಳನ್ನು ಸಂರಕ್ಷಿಸಲು ಜಾಗೃತಿ ಕಾರ್ಯಕ್ರಮವನ್ನು ಹಮ್ಮಿಕೊಂಡಿದ್ದೆವು. ಹಲಸಿನ ಹಪ್ಪಳ ಮಾರಾಟಕ್ಕೆ ಸಿದ್ಧತೆ ಮಾಡುತ್ತಿರುವ ಗಂಗಾ ಭಟ್ಟರು. ನೆಕ್ಸ್ಟ್ ಪುನರ್ಸಂಗ್ರಹ ಸೂಚನೆ ವಿಶ್ಲೇಷಣೆ. ಡಿಸೆಂಬರ್ 1ರಿಂದ 15ರವರೆಗೆ ನಾವು ಪುನರ್ಸಂರಚನೆ ಮಾಡಿದ್ದೇವೆ. ಮಾಹಿತಿ ಕ್ರೋಢೀಕರಣ ಸಂದರ್ಭದಲ್ಲಿ ಎಲೆ 50, ಪುನರ್ಸಂರಚನೆ ಸಂದರ್ಭದಲ್ಲಿ ಎಲೆ 500, ಹಾಗೆ ತೊಗಟೆ 17, ತೊಗಟೆ 20, ಹೂ 14, ಹೂ 25, ಹಣ್ಣು ಕಾಯಿ 75, ಹಣ್ಣು ಕಾಯಿ 100, ಬೇರು 12, ಬೇರು 22. ಹಾಗೂ ನಮ್ಮ ಪ್ರಾಜೆಕ್ಟ್ ಮೊದಲಿಗೆ ನಮ್ಮ ಪ್ರಾಜೆಕ್ಟ್ ಹಾಗೂ ನಂತರದಲ್ಲಿ ನೋಡಿದಾಗ ನಮ್ಮ ಪ್ರಾಜೆಕ್ಟ್ ಅನ್ನು ಪ್ರಗತಿಯಲ್ಲಿ ಹೋಲಿಕೆಯನ್ನು ನೋಡಿದಾಗ ನಮ್ಮ ಪ್ರಾಜೆಕ್ಟ್ ಜನರಲ್ಲಿ ಸಕಾರಾತ್ಮಕತೆಯನ್ನು ಹೋಲಿಕೆಯನ್ನು ಕಾಣುತ್ತದೆ. ಹಾಗೂ ತೋಟಗಾರಿಕೆ ಇಲಾಖೆ ಹಾಗೂ ಅರಣ್ಯ ಇಲಾಖೆಯವರು ನಮ್ಮ ಪ್ರಾಜೆಕ್ಟ್ ನ ನೋಡಿ ಪ್ರದರ್ಶಿಸಿದಾಗ ಅವರು ಪ್ರಶಸ್ತಿನ ಪತ್ರ ಕೂಡ ಕೊಟ್ಟಿದ್ದಾರೆ. ಡಯಾಬಿಟಿಸ್ ನಿಂದ ಬಳಲುತ್ತಿರುವ ವಿಆರ್ ಭಟ್ ತಂಡವನೆ ಹಾಗೂ ಧನುರ್ವಾದದಿಂದ ಬಳಲುತ್ತಿರುವ ಕೃಷ್ಣ ಭಂಡಾರಿ ಅವರು ತಮ್ಮ ಆಹಾರದಲ್ಲಿ ಹೊಸತಿದ್ದನ್ನು ತಂದಕ್ಕಾಗಿ ಹೇಳಿದ ಧನ್ಯವಾದಗಳು. ನಮಗೆ ಈ ಪ್ರಾಜೆಕ್ಟ್ ನಲ್ಲಿ ಮತ್ತಷ್ಟು ಮುಂದುವರೆಯಲು ನೀಡಿದ ಟಾನಿಕ್ ಆಗಿದೆ ಎನ್ನುತ್ತಾ ಪೂರ್ಣ ವಿರಾಮ.
Next, Amina from Bangalore. Please.
Friend, this is back. Okay, so back. Can check once. Okay. Camera is here. Should be taking. Okay. Can start.
Good morning, everyone. Myself, Amina Kausar. I'm from 7th standard. Good afternoon, everyone. Myself, Amina Kausar. I'm from 7th standard. Myself, Amsika from 7th standard. I'm here from Bangalore Girls Society. And my topic name is paper-based institutions. In India, literacy rate is high, so the demand of paper is also high. And when the demand of paper is high, it is estimated that in 2025, the demand of paper would be 2.5 crore metric tons. And this is a very difficult task for the Indian paper industries also. One side because they have to deal with the deforestation and pollution, and other hand side they have to deal with the increasing demand of paper. In India, 3 million tons of paper is recycled, but it's only the 20% of the total. We know that plastic is harming the environment, but it is like paper is also harming the environment as like plastic. And these are the problems we face from paper waste. First is deforestation. About 10,000 A4 sheet size paper need a cost of 3, which is 45 feet useful trunk and a diameter of 8 inches. The tons of paper waste every year decides thousands of trees and landfill volume. Nearby 10 to 11 million tons ends up in landfill. Litter. When paper waste is littering, it makes the place very unattractive for the tourists and business owners. And this happens in schools and offices that faulty prints, wrong contents, mistakes, etc. will all lead to waste of money. Why should we save or reduce paper waste? Recycling 1 ton of paper can help in saving about 70% raw materials, saving along with coal of 60% and energy of 43% and water of 70%, which is used to make paper from wood. My objective is to understand how students can contribute to saving environment by altering certain habits. To cooperate with the school for the reduction of paper waste in classrooms. We learn how paper waste can help in reduction of environment. This project was taken as an eco-class assignment. My methods and material are study area. Classes from first standard to 10th standard. Sample collection method. We requested IS to keep the paper waste so that we can check the weight. Method. First 1 week we observe the quantity of waste generated and next week went to the awareness campaign. Take overall weight of the testing which was collected and kept. Use spring scale to measure the total paper waste which was given by our helper staff. Maintain a manual record. These are my materials. Spring scale to measure the total waste and book to maintain manual record. Results. This is the total paper waste find from the classrooms. These are the total waste find from 8 to 10. This is the total paper waste before I went to the awareness campaign. These are the pictures of when I went to the awareness campaign. We requested students to not take paper unnecessary, to put the paper in separate bin which was kept in the corridor, and requested others to not mix the paper waste with the food waste so that it could go for separately recycling. This is total paper waste generated after I went to the awareness campaign, and the difference is 700 grams, which is 1.7 kg. And observation are, despite of awareness campaign, some classes like especially 4th and 10th will continue to produce almost the same amount of paper waste, and this was seen in higher classes, and in some lower classes teacher was strict, and also they produced a less amount of paper waste. Conducted and second awareness campaign where waste was generated more. Discussion. Method of waste disposal in our school. The school gives the drivers to the BBMP power chemicals weekly once. On an average, if our school produces 5 kg per week, the total production will be 1.5 kg, which is a really big amount from our school. Estimated waste generation by schools on an average. A search in Google shows that there are 1,391 schools, including higher and lower primary. Considering a minimum of at least 2 kg per week, the annual production will be 83,460 kg. Any paper which is stained or has patches of oil or soil patches, it cannot be recycled. I think that paper as found in the classroom might have one or other stain, but it could go directly in the landfill. Recommendation for the other school, which was followed by our school, to go paperless. Our school communicated parents through WhatsApp, and even circulars are sent from soft copy. Activities are given in books, not in the paper, so that we can check. We can save the paper. Previous handwritten books are also allowed as rough notes. And the main point, we have reduced the number of notebook issued, because this year if we buy the more amount of books, next year again we have to buy the same amount. And that last year paper would be waste, books will be waste. So our school has stopped this. I conclude that though any institution takes up measure, individuals' cooperation matters. Even first standard LKG and LKG student can help in saving the environment. And for any good work, awareness campaign is required. I thought this is the right platform to communicate with the students. Together we make a difference. Reference. These are some of the reference. My acknowledgement. I sincerely thank to the students, teachers, especially Krishna Madam and Principal of our school for cooperating and helping me doing this project. I sincerely thank to the team of CES IASC for giving me an opportunity to present this. Thank you.
That was a good presentation on saving and conserving forest and paper waste. Yeah, that's a good idea. But in a study, I just saw that though you created an awareness among your own friends in your own school, class 4 and class 7 students did not reduce the wastage of paper. Did you try to find out what could be the reason?
It happens when teachers are not strict also.
Oh, and you there are no, please make a note. Class 4 and class 10 teacher. Yeah. Okay, that's a good one. Okay. Any questions?
So,
There's one. Yeah.
if teachers are strict, if teachers should be strict, then what is the use of your awareness program? Your awareness program should give the punch to the students. There should be a behavioral change. Behavioral change should be there in your friends. Such awareness program should be given to your friends.
Teacher, is my experience. At times, the teacher's strictness also plays a very important role. You have to be. Otherwise, no, any kind of awareness you create and whatever you do, that mindset change doesn't come so easily. Right? One question that there are students. You take one question. There at the back. What is there at the back?
It's not a question, it's a correction. You have calculated the total number of schools and multiplied it with a number. Can you go to that slide? Follow there.
Next.
This one.
No, it's our schools next. No further. You have mentioned that at least 2 kgs per week is produced. But why have you multiplied it with 30? It's month. I have 4 weeks in a month. Maybe it's here. 4 weeks plus 30, it is 30 days. Yes. Okay, what did you do with the paper waste that you have collected? Here. What did you do with the paper waste that you have collected? Collected during your survey.
We was giving to BBMP.
Oh, you come to me and tell you very much. You should not give to BBMP. That is that dealers, they will take it. There are several ways that you need for papers. You have to take it because we have money.
Thank you.
Let's go to correction and moving on to the next presentation. That is Ruba, Nishchal and Nishant from Vijay Leaders. Amina, can you please come and register?
Hey, Michael, how are you? Oh, you. And Michael, go back.
Warm good afternoon to one and all present here. Myself Nisha, he is Nissal, he is Dhruva, and we are from Vidyaniketan Public School, and we will be doing a presentation on the topic limnology and aquatic ecology and aspects of biology. Lakes in Bangalore. We chose this topic about limnology and biomonitoring because we felt that lakes in Bangalore were extremely polluted, and we found out in basic data over here. We found out that 1,452 lakes were present in 1985. Now in 2021-2022, it was reduced to just 183. We were shocked to see such a drastic change, and we chose this topic. These are the literature. We did a lot of literature study, and we found out that most of the lakes are encroached. The lake we chose was Dubasipalya Lake, which is located in Kengeri. It lies in the Vrishabhavati Valley. The lake area has been reduced by literally 20 to 30 meters from each side. So you can tell how much encroachment has occurred. So it is also in the BDA West Engineering Division. BBMP had recommended that the lake should be cleaned in 2014, but till in 2022 it hasn't been cleaned at least once. Our objective is to study the inland ecosystem of Dubhasipalya Lake and try to provide suggestions for conserving the lake. Our methodologies include: we collected water samples from inlet, outlet, and center and tested it for various chemical and biological parameters in each week. We recorded different microphages and plant species present in different spots of the lake. We also did on-spot check of temperature, physical parameters, dissolved oxygen, etc. Now my friend Dhruva.
So we use the study area as Dubhasipalya Lake. So we found the perimeter as 1,194 meter and area as 58 meter square. So we use the Google Earth help and we found the historical images of the lake. So we can see from 2017 the eutrophication like growth of algae has increased in the lake. So this is the inlet of the map where we collected the sample, and this is the outlet of the lake. So even we can see the dug up area for the sewage from the residents which are led to the inlet to the lake. The residents' wells are left in the drainage directly to the inlet of the lake, which also pollutes the lake, and this is the main factor. Next, the sample collection. We collected samples from 3 different areas: the inlet, outlet, and the center of the lake. This is the inlet, this is the outlet, and this is the center. These are the 3 bottles of sample we collected. We identified various species during a time of sample collection. These are the images what we found through the help of a compound microscope. And we found Arthrospara and highly coiled structure and diatoms in the inlet of the lake. We weren't able to identify the highly coiled structure. In the center of the lake, we found Chlorococcum, Euglena, Anaverina, and Spirogyna. In outlet of the lake, also we found some organisms, Paramecium diatoms, and circular organisms which we weren't able to identify, and Chetiphodus. And these are the physical parameters. The color in inlet it is pale grayish, in center it is blackish, and outlet it is pale white. From this we can conclude that as most of the animals live in the center of the lake and it is blackish, visibility is not possible. Temperature is 20 degrees Celsius in inlet, 16 degrees Celsius in center, and 24 degrees Celsius in outlet. This is because in inlet the water is let in from sewage and homes, so it will be at room temperature. In center, it will be in direct contact with the atmosphere, and in outlet it will be 24 degrees Celsius because it's in our lake the outlet is connected to the inlet, so we took it as a point where it was connected for a lot of time, so it's 24 degrees. 24 degrees Celsius. The transparency was translucent for inlet. It was completely opaque as the color itself states in center. Outlet was slightly translucent. The order was very pungent in each of them. These are the chemical parameters. The pH we found out was 7 pH in inlet, 9 pH in center, and 8 pH in outlet. From this we can tell that the water is quite basic. Total solids, we found 0.1 milligram per liter in each of them. Dissolved oxygen 2.96 milligram per liter was found in inlet, and 2.16 milligram per liter was found in outlet. They were well below the BIS range of 4 milligram per liter. From this, we can conclude that as not much of oxygen is present for respiration of organisms are finding it difficult to thrive. Chloride, we found it at 260.800 milligram per liter in inlet, which also tells us that it's quite normal, but it's just a bit more. Now, my friend, next. And the aspects of biomonitoring, here we try to take the samples of the lake and test it weekly. We have also we saw in the previous slides, and we thought of setting up CCTV cameras around the lake so that the inhabitant or species in and around the lake can be seen, and people don't and see to that people don't dispose any garbage or anything into the lake. Observation: due to presence of less dissolved oxygen, we observed that life was not able to survive, and due to the presence of microorganisms like Spirogyna, Euglena, we observed that life was sewage water which was get through. And we observed that water was emanating from the lake. Conclusion, we can conclude by this time, the lake is heavily polluted and algal bloom has been increased, and organisms are unable to survive in the lake. And actions by the citizens promoting awareness, we can promote awareness to the people and let them know how the lakes are declining, or make them aware that we should conserve the lakes, and we see to that regulate the waste disposal. These are some of the references, and we express our gratitude to our teachers and our school management for giving us opportunity to participate in this, and we thank IAC for giving us a wonderful opportunity to lead our journey of learning and discovery. Thank you. How do you test?
Not temperature, we tested it using laboratory thermometer. Yes, ma'am, on site. We took the laboratory thermometers and we tested it on site. Ma'am, we took the bottles, ma'am. We first rinsed it to avoid cross contamination from the inlet, outlet, and center. Yeah, both. We took a boat. The fishermen thrive over there, ma'am. So we took their help and we dissolved oxygen through Brinkler's method, titration. 54,864 meters square. No, 54,800. Thank you, sir.
Stand here, face to the camera here, okay? This is for operation friends, back. Okay, want the pointer, it's here.
A very good afternoon to one and all present here. I'm Anushka. I'm Advaya. And I'm Dhanushri. And we are from grade 9 Vagdala School, Marutali. And today we're going to present about geoinformatics and ecosystem management. Geoinformatics, also called as GIS, is a combination of two words, geography and informatics. Geoinformatics, also known as smart maps, is one of the powerful technologies used to support basic scientific inquiry, as well as address the complex social environmental challenges through data analysis. It collects and organizes the data and then analyzes it through computation and geovisualization. The main objectives of the presentation we'll be discussing about gathering water resource data management, and we'll discuss a few problems related to human-animal interaction and the solutions to these problems. The study name for this presentation is Rohali Forest. It is located about 20 kilometers from Bangalore in Kanakapura Road. The area is 590 acres. The latitude is 12.8 degree north and the longitude is 77.5 degree east. Gathering water resource data. As we know, water plays a very crucial role in maintaining the balance and harmony in nature. Lack of it can cause an imbalance in environmental and ecological spheres. GIS helps to gather water resource data by capturing a high contrast picture of a particular field of view and sending it to the state for continuous monitoring. As we can see this graph, as population is increasing, the demand for water is also increasing. Slide 1. As we know, in olden times there was greenery and vegetation present almost everywhere. But due to increase in population, people cut down the trees and animals living there were forced to move away into a new locality. But in the new locality, there was not a lot of natural resources like food, water, and shelter like they expected, so they have no other option than to come back. As you can see in this chart,
In 1973, there was greenery and vegetation present throughout Bangalore, but over the years, due to increase in population, the vegetation has decreased and buildings and architectures has increased. By the year of 2020, there's almost no greenery and vegetation present. As a result, the animals come and mix with the human habitation. And here's a scenario where a leopard has entered a school premises in the year of 2016, and these type of incidents has occurred many over the years.
This is another scenario of the situation. A leopard was recently spotted in an apartment complex in Banagata Road. The CCTV camera caught this animal sneaking into the apartment the second I know. This incident happened in the year 2014, and unfortunately, as you can see, while it was trying to cross the road, it got hit by the vehicle and died. As we can see here, 4 leopards were recently spotted in the Bangalore city near Turahalli Forest. The forest department is still trying to catch these leopards.
This is an image of a leopard which killed a 23-year-old woman in Mysore. And this is another image of a leopard which was caught coming out of the Turahalli forest 6 kilometers away from the borders near the Banagata National Park, and it's caught and caged.
When animals move from one side of the forest to the other side of the forest, either they get hit by the vehicle or they get angry and end up attacking the humans inside the vehicle.
In general, there are 3 types of losses: human death, animal death, and loss of human property. These pachyderm intrusions are increasing day by day, particularly in the forest border areas. Continuous monitoring and tracking of animals is difficult due to its large size and fast movement. Methods have been implemented for this, like animal crossing zones and pre-warning systems.
The green dots here represent the access zones in Turahalli Forest. These are a few incidents occurred recently. The first one is a picture of a leopard crossing road in Mysore in the Malathuradi highway in Nagarhole.
This is an image of a bull crossing the road in Nagarhole, which is about to be hit by the vehicle. And this is an image of a tiger crossing the road.
This is an image of an elephant who crossed the road and got hit by the bus. As you can see, the situation of the elephant and the bus in a pretty bad condition. Solution for animals entering the human habitation. As animals were forced to move from forest to new localities in search of food and water, there weren't much resources as it was there in the forest. So animals had no other option except to come into the human habitation in search of food and water. Each and every time, personal visit to the forest or lakes to check the water level is not possible. By helping GIS, we can track a particular area where groundwater level is low or high. By using GIS, in Turahalli Forest, the NGO department went to the forest where the water bodies were dried up completely, and they filled the lakes hoping that the animals won't come and mix with the human habitation again.
These are the images of GIS tracking the groundwater level. The first one is an image of GIS tracking the groundwater level in Bangalore, and this is in the year of 2016-17, and this is an image of the groundwater level in Karnataka. Proposed solution. As we have seen earlier, when animals cross the road from one side of the road to the other side of the road, they get hit by the vehicle and die, or get angry and end up attacking the human inside. With the help of GIS, we can track down these accident-prone zone areas and provide pre-warning system in such cases. There are bridges existing in other countries, including some parts of India, specially built for animals to cross the road, but there are no barricades like structures beside it to show that they have to cross only through the bridges and not through the roads. So we are proposing an idea over here to build bridges along with a barricade-like structures in these accident-prone zones so that the animals don't get hit by the humans and die.
We can also vibrate a static sensor, which will sense the movement of the animals and produce a beeping sound to scare the animals and not let it pass through the barricade. The materials required are Arduino UNO 3, buzzer, barbed wires, motion sensor, vibratory or static sensor. This is the working model. As we can see here, this is the motion sensor. Whenever the animals try to come near the barricade-like structure, the motion sensor will sense the movement and it will send inputs to the Arduino board, which is already programmed using C++, that whenever it senses the motion, it should give buzzing sound along with vibrations to the ground so that the animals won't move further to near the barricade and damage it. In conclusion, we can say that GIS is very helpful in our daily needs, such as hotspot analysis and Google Maps. It will be very helpful to use GIS in such a way that we can save the wildlife. These are all the reference links which we have used to create this presentation.
We would like to thank ISD for giving us the opportunity to express our views and opinions. We would also like to thank our school and our R&D teachers who supported us throughout this project. Last but not the least, we would like to thank our parents who helped us to the end of the project. Thank you.
Yes, sir, we said that it's present in some parts of India.
That was built by addition. Okay, even 100 years is a bullshit. Whereas our fellows are constructing now within 1 year it collapses. And you fellows have to see that it doesn't happen.
Have you done the model of this? You will show it.
Yes.
Do you have a working model with you?
No, sir.
It's not working.
Yeah. No, it works, but we don't have the model. Yes, sir.
It can be done, right?
Yes, yeah, ma'am, it can be done.
Yes, ma'am.
Sir, leopard.
No.
Right.
In front of KSRTC bus. What animal did you tell?
Bull.
Bull. Indian bison, war. Bull is for domestic.
Any more questions from the student? Is there any paper presentation left with you? Okay, then we will move on to poster presentation. For poster presentation, Yaditha and Hamsi, Diversity of Birds in Canada University campus. I will tell the rules.
Excuse me. Have given? Which school?
In 3 minutes, I will give the volume name and 4 minutes is your total time for poster presentation. Okay?
It's visible.
Okay, you can move down now. Scroll down or do this. Scroll down. Okay, you have this laser here and this top. Okay, can you see? If you want to stand it, okay.
Stand here. Good stand. Good afternoon. Good afternoon. Good afternoon to one and all present here. Our topic is diversity of birds in the Bangalore University campus. My name is Hamsini and this is my friend Charita. We are from Vidyaniketan Public School, 8th grade. The main objective of our project is to observe and record the diversity of birds in the Bangalore University campus. The picture in the right side shows the overview of the Bangalore University campus, and this picture shows the area which we have done the study. Methodology. Observation. We went there weekly twice every morning and evening using binoculars and smartphones. We did the 360-degree survey to observe the birds. Identification. We gathered information through articles, books that helped us identify birds through their characters. The main criteria were feathers, neck length, body structure, beak structure, style of feet. Here are the some birds we observed during the survey. House crow, noisy birds with long straight bills, great cockle, shy birds with booming call, rock dove have short bodies with short necks, common prinia have long tails, common moorhen looks like a duck but lack of webbed feet. Indian people, heavy birds that make short flies. Great tit, very active birds with small beaks. Black kite, auk-like birds but often seen around the human habitation. Spotted owlet, mainly nocturnal and grows in large trees and abundant buildings during the day. Common myna make various creaking calls and feed mostly on the ground. They roost large in groups. Indian roller often perch on low branch or even over head buyers for the ground prey. Here are the some birds we also observed during this project. Grey-headed hornbill, Indian grey hornbill, red-vented bulbul, rufous striped, red-viscous bulbul, white-billed kingfisher. Recommendations. Since diversity of birds in Bangalore University campus is agreeable, there is a scope for improvement. This can be done by making anti-pollution areas and encouraging nature lovers and bird watchers and providing open resources and growing more host plants. Conclusion. I would like to conclude, we actually observed 20 to 30 species of birds. We identified birds through their features and it helped us learn and recognize birds. Comparatively, water birds were way less than the migratory birds. Through various articles and books, we can see that the birds in Bangalore University campus are increasing every year. According to reasons why diversity of birds are increasing is pollution-less area, good environment, and growing more host plants. Here are some references. We thank Indian Institution of Science, Energy and Wetland Research Group, Sir T.V. Ramachandra sir, and who will give us chance to learn and present the project as part of Lake Symposium 2022. We also thank our school and teachers for supporting and helping us in this project. Thank you.
Grave on bill. I request you to check cross-verify. Okay, that is not a grave bill. Please check verify and slide upside. And what outlet is that?
Spotted owlet.
I also request and strongly suggest you to check this image. That is not of spotted owlet. Okay.
Sure.
Did you ask for proofreading or did you show to any experts or did you use any field guides to verify these images?
Sir, actually, we went with an photographer, Sir Nash, like wildlife for itself. He told us.
Fine, because grain in oil, as per my knowledge,
that species does not exist in Bangalore or Karnataka town. And also, that is not a spotted owl. Thus, overall good, all the best.
Thank you. Thank you.
Next, Srirangan group.
This is yours, huh? Okay. They are present from there only. Okay.
Professor Rupal, myself, of Mene, Vijay Bhuvan of Mene, and Boyan of grade 7. Our topic is Birds of Ideal Campus. We chose that Ideal Campus because it is surrounded with Areca farm next to our school and mice farm next to our school campus only. Introduction. Birds are very attractive and colorful common creatures living around us. Birds have feathers and beak, making them very unique. They lay hard-shelled eggs. Birds of prey are known to as a powerful vision when compared to human eyes. Different feathers in their bodies form different functions. We take the opportunity to study the bird diversity in our school campus. Objective to create a baseline checklist of the birds seen in the study area to understand the habits of birds seen in and around the school campus, to create awareness amongst the union to start watching the birds in our school campus and create a database of residential bird and migratory bird. Result and discussion. The present study gives a count of 41 species of birds from the study area belonging to 34 different categories of family. The most dominant family was kites, whereas 3 species of kites were recorded, followed by bulbuls, doves, crows, egret, and minors, with 2 species were seen. Rest of the other members were represented by single species. Few water birds were seen flying high over the school buildings. Conclusion. Our school campus has a diverse numbers of birds, which shows that our school campus is rich in the bird diversity. The survey has to be carried out across the different seasons. Recommendation. More fruit-bearing trees has to be planted to attract some more birds to our school campus. References are at the wetland birds of Bengaluru by Nihila MB and Chaturved Shetty. Birds of Indian subcontinent by Grimm. Pocket Guide to Bird of Kaveri Wildlife Sanctuary by NCF.
We are thank you. We are very thankful to our school management, sir, Sri Raghu BC, sir, Secretary, Ideal School, Channagiri, Anusha ma'am, and all other member of management for creating this opportunity to us.
My sincere thanks to Dr. T.V. Ramchandra sir and Vijay sir supervisor, their motivation and constant support. Thank you.
Next, Pragna Bhatt from Baghdadi Dinas.
Have a pointer if you want to use it. But I need to zoom in, right? Okay, fine. Control H, okay, if it goes, if it all goes down.
How do I move?
This one, or you can use this one also.
No.
That's fine. Yes, this side, okay. You see this side, the camera is on. Okay, it's fine. You can.
Good afternoon, everyone present here. I am Prajna Bhatt from class 9, Bagdevi Vilas School, Nelamangala. My topic is Pinamangala Lake Restoration, Management and Conservation. So, Pinamangala Lake is a natural freshwater lake located in Nelamangala, and it is nearby to my home. So, firstly, I would like to highlight the major problems and current status of this lake using the observations and also water tests that was done. So, there is a very less storage capacity in the lake due to excessive growth of plants, weeds, and algae. Sediments and plants have covered the bottom of the lake. So, the lake is under eutrophication, and we observe from the test that the phosphorus and nitrate content is more. So, certain outlets bring polluted sewage water directly into the lake. And during heavy rains, every year the lake overflows and floods the residential area. So the lake has a bad odor, it is muddy and foamy. Urbanization adds to these problems. And from the water test, the ammonia content is more, and therefore the animals find it very hard to live. Yet some kind of fishes, bacteria, algae are found to be living, and some birds surround and live nearby the lake. So moving through these problems, the restoration methods must be discussed accordingly. To restore the lake naturally, in summer, weed control, sediment cleaning, catchment area management can be done. And in monsoon, pure rainwater storage has to be done. The fences can be built and the lake depth has to be managed according to average rainfall. Else, onshore treatment technique can also help us, wherein the water can be pumped onshore. It can be treated by adding dissolved oxygen and removing certain nutrients. So, only proper management will ensure a longer and healthier life of a lake. So, the catchment area must be managed very effectively. Constructions and human activities must be banned, and big trees can be grown on their places. And the pipes or the channels bringing sewage water to the lake must be well built and it has to be diverted. Proper fences have to be managed. Strong regulations will also help in proper management. And the excessive rainwater can be channelized effectively towards agricultural lands. And moving on to conservation, it all lies in people's hands. After once the lake is restored and it is being managed, it should not be allowed to be polluted under people's hands by any chance. So do's and don'ts, boards can be put near the fences of the lake, and education educational efforts, that is environmental education, can help people to understand the importance of the lake and conserve the lake. And regular inspections and monitoring can be done to ensure to take the steps further. And self-service organizations can be formed by students or the people in the society to save the lake. And we can write articles to newspapers and letters to government regarding the lake. So our lake is certainly in a now or never situation. If not restored now, the lake is sure to die. So this has to be done. And this was a wonderful opportunity to bring this lake into this conference. And I hope this beautiful new Binamangala lake becomes reality by the next conference. And I thank IISc for this great opportunity. I thank R&D teachers, my school principal, my parents. Special thanks to each one who has encouraged and supported me. Save lake, save ecosystem, save life. Thank you.
Is there any conservation team ever would be something like that that would affect the neighbors, whoever is staying near the lake?
No, ma'am.
Since it is flooding and negatively affecting the people those who are living nearby. Your home is also there.
Yes, ma'am. The picture
So
is
you
taken.
are also struggling with the you also get affected.
Yes, ma'am. Yes, ma'am. We are trying to. Yes, ma'am. We have talked to the local authorities, and also we have written letters, but the progress is not yet. No.
Otherwise, they will not look into all these problems. That's why our letter important as media.
Yes.
Go to media, take photographs. You said articles, we have to write articles. How many articles TVN has written, how many people have read, how many have been read through?
Yes.
On your Instagram and post all these photos.
No, local people should unite and make representation to the concerned authorities. It is not one authority, one agency handling all these issues. So there are so many interrelated, authorities of the government. You have to write to everyone.
Yes, ma'am.
And you have to follow it up. Just writing and leaving, nothing works.
They will
You
work.
keep on going to them and go behind them, there will be something can happen. Next late conference, you said, no? So you keep working on that.
Yes, ma'am. Very well. Thank you.
Good afternoon, everyone. Good afternoon, one and all who are here. Myself, Disha Patel. I am studying in grade 7 with me, Prithvi and Varshini. We are from Ideal International Techno School, Channagiri. Today, we are explaining about the comparison study of Garga and Shantisagara Lake. Introduction. Channagiri, we are from Channagiri Taluk in Davangere district. Is also known as the land of Arekanat. The taluk is not only the famous for arecanut production, but also hosts Asia's one of the largest man-made lake, that is Shantisagara Lake. Birds have fascinated not only the people but also to us. We took 2 different lakes to study bird diversity near our school. Study site. First one is Shanti Sagara Lake. Second one is Garga Lake. Birds are fine feathered friends of our farmers, which have a great role to play in the ecosystem. Objects to create a baseline checklist of birds seen in the study areas to complete the bird diversity and understand the habitats of birds in 2 different lakes. To know the current status of the birds that is resistant and migratory from these 2 lines. These are the birds which are we noted when we go into the field work of Shantisagara Lake and the Garga Lake. This is the birds which is migratory bird of Shantisagara Lake, common sandpiper. This is the photo of the Shantisagara Lake, Garga Lake, and the study in the Garga Lake. Results and discussion. Totally, we have seen more than 55 species of birds from our study sites. The present study gives a comparison list of 2 lakes from Chanagiri. We have recorded 50 birds from Shantisagara Lake and 38 birds from Garga Lake. Out of 50 birds, 4 were migratory birds from study site 1 and only 1 species were migratory from Garga Lake, that is common sandpiper. The most dominated family were egrets from both the study sites, paradise flycatcher, blue-capped rock thrush, blue rock thrush, and common sandpiper were migratory birds. Conclusion. Both the study sites are attracting fair number of birds. The survey has to be carried out across the different seasons to get a detailed list of migratory birds. We can find the characteristic of birds by their beaks or bills, feathers, and their activities and their diet and their feeding. We can attract the birds by growing lots of trees like lime trees, banana trees, fruits, or etc. In the birds, the types of birds are frugivore, piscivore, and the etc. Now I will give to Varshini. These are the birds which I've seen and some birds she has written.
Common myna, red-belted bulbul, little swift, little cormorant, white-throat wagtail, green bee-eater, white bush chat, Indian pond heron, rose-winged parakeet, white-breasted water hen, jungle crow, little egret, spotted dove, greater cackle, red-wattled lapwing, black-winged kite, black kite. Thank you.
We also use the references for the free guide when you are go to. The references book are like Unearth the Wetland Birds of Bangalore by Rijal and Visar and Chaturved sir, Birds of Indian Subcontinent by the Climate, and we also see the pocket guides for the in the birds when we are go to the field work. I would thankful for our management, Raghu sir, Anusha ma'am, and the member of our management, and giving this opportunity. And I will thank TV Ramchandra sir, Rijal sir, Chaturved sir for this motivation and the constant support. Thank you.
One suggestion to you. To attract the native birds, you need to have the native species of trees, particularly the ficus family. You discuss it with Chaturved and we will help you out in doing that. And to get the migratory birds, it's very difficult because the fish in the lake has to be abundantly available, and in the neighborhood there must be trees for them to nest. They come here for testing. So therefore, and before that, awareness has to be created to the public or the people who live surrounding the village. Okay? Kokre Belluru. Have you heard of that name? Kokre Belluru. Chakravarti will help you in understanding what is that. And maybe probably you can do a field visit to that place. Yeah, so that it will help you. Because those people have taken all the initiatives and they protect all the birds. Right? Okay. Yeah.
Abhinav Kaura of IDS International School. Neha and Dhruv.
Good afternoon to my adult present here. Good afternoon to my adult present here. Myself Sneha of grade 7 from Ideal International Techno School, Chennagiri. I'm very glad to have this opportunity to present my document regarding birds. I have Sinchana and Zina from grade 7 who are accompanying me in this presentation. My topic is birds of Chennagiri Lake. This lake is very polluted and we can't see many birds as much as we see in Shantisagar and Garga Lake. But I still choose this lake as it is highly populated and this lake close by to the lake. We don't see as much birds, so I decided to spread awareness to people that this lake is getting polluted day by day. And if this continues, we cannot see a single bird in this place. Objectives: to create baseline checklist of birds seen in the study area, to understand habitat of birds and seen in and around Chennagiri Lake, to create awareness among people regarding the importance of lake as habitat of birds. Study area, Chennagiri Lake. In the image, when you can see how polluted the lake is, and in the image too, we can see our team members. It is located in the heart of the city, near close to the bus stand. Down we can see the table which tells how much birds we have seen. Results and discussion. The present study area gives us a count of 12 species of birds from the study area. More survey efforts will be made in the future studies. Out of 12 species, 3 were wetland birds, which strongly convey that lake is facing severe threats and not able to attract other very commonly found water birds and other water bodies in and around Chennai. Conclusion: it is very important to understand the importance of lakes as a habitat for birds and other organisms. Strong rules have to be charged and followed strictly to conserve this lake. Recommendation: water analysis has to made to understand the basic features of water. The lake can be adopted by any NGOs or our school for the conservation and development of this particular lake. References. We have used underneath the wetlands of birds of Bengaluru, where vigilance, and certificates and more acknowledgement. We are thankful that this our management has given me this opportunity to present my document here, and I am thankful to Chaturitsa Raghu sir and our ideal management to give me this opportunity. Thank you.
NGO. Should be adopted by any NGOs. That way, any NGOs there. What is NGO? Okay, no problem. Next, here and the ideal.
Okay. ಸರ್ ಸರ್ ಒಂದು ಪ್ರೆಸೆಂಟೇಷನ್ ಇದೆ, ಅದು ಮುಗ್ದಿದ ತಕ್ಷಣ ಕಳಿಸ್ತೀನಿ. ನಡೀತಿದೆ ಸರ್. ಓಕೆ ತಿಂತಾಳು ಫ್ಯಾನು ಎಲ್ಲಿ ಮಾಡ್ತೀನಿ ಮಲ್ಕೊಂಡ್ಬಿಟ್ಟ.
Butterflies are known to know only insects have scales or their wings. We are very proud of our butterflies. Chandrasekhar Lake, which is the ancient Samudra, just man-made lake. There is a walkway path and on the land of this wide lake. Two different types live near the lake. To study butterfly density around the lake, study side one is Chandrasekhar Lake Park and study side two is Gaga Lake Park. Private Areca is only the plant. Areca is only butterfly science, which is the life cycle of butterfly is very interesting and mysterious, as one knows how the change is observation created. Yet of this then the list of butterflies see in the study areas. To compare the butterflies diversity and understand the habitats of butterflies in the two different stages. To know the appearance stages of the butterflies that is very common and rare. In the first image, you can see there is a Chandrasekhar Lake Park, and between it is the Gaga Lake Park. This is our field visit photo, and our common butterfly is crimson rose. It's a swallowtail butterfly, and we can check this about butterflies from the study areas. These are we noted 22 butterflies in our study areas. We can check that. Results and discussions. Totally, we have seen 22 species of butterflies from our study sites. The present study is our composition list of 2 study sites from Changiri. We have recorded 17 butterflies from Chandrasekhar Lake Park and 14
butterflies from Durga Lake found out of the 22 species of butterflies. The family is fresh-footed and dominated in both areas and followed by the whites and yellows. Common silverline is very near butterfly spotted only once near the Durga Lake Park. There were several butterflies like very common. Conclusions. Both the study sites are attractive to great numbers of butterflies. The survey had to be carried out across the different seasons to give a detailed list of butterflies. Our reference books are Bengal butterflies of Chicorates by Chicorates butterflies of India by Isaac Chechumitra. We are very thankful to our school management, Sir Raghusul, Executive Ideal School, Changi, and Harisharma, and all other members of management for creating this opportunity to us. My sincere thanks to Dr. T. Ramachandran sir, Vijay sir, and Chaturaj sir for this motivation and for constant support. Thank you.
Sir, answer with your motivation words. We have selected to adopt Shantisarna Lake, which is Asia's second largest lake and India's largest pent-up lake. And our management has decided we'll adopt that lake. We'll create more awareness and involve more students to take part in that. And of course, sir, all ISC people have visited and given suggestions. And we are happy and thankful also, sir. That lake serves four talukas there, and also 4 districts are benefited because of that lake. And we have a complete Araka farm. Still, the lake is excellent park. So it's a great happen to know what is there. This is the first time our students are participating. Thanks for all the support. We adopt that lake with your words, sir. Thank you so much. Now, we have gone to Channagiri and saw that lake. Let's come to ISC Bengaluru. How beautiful tea now. Delicious tea. We drink our tea like the commission.
Yeah, we'll have a short break now. We'll break half a minute. Right? All of you who have stepped, please wake up. Tea is ready. We will step. We'll meet actually half an hour. 5:15. Further past 5 o'clock, we'll be back here. Anybody who comes late, summarize for the whole day. Please remember that.
ಸರ್ ಬರ್ತಿದ್ದಾರೆ ಹಾ ಬಿಟ್ಟಿದೀವಿ ಬರ್ತಿದ್ದಾರೆ ಯೋಚನೆ ಸರ್ ಓಕೆ ಸೇಮ್ ಟೀಮ್ ಯು ಕ್ಯಾನ್ ಗೋ ಡೌನ್ಲೋಡ್ ಯು ಕ್ಯಾನ್ ಯು ಬಿ ಹಿಯರ್ ನೋ ದೇ ಆರ್ ವೇಟಿಂಗ್ ಡೌನ್ ಫಾರ್ ದ ಟೀಮ್ ಓಕೆ ಯಾವ ಸ್ಕೂಲ ಕೊಡು ಹಾಂ Helper na? He has not come, I think.
The author of this poster is here. Wait. I'm just asking because we wait for all the participants.
Judges cannot come, na?
Fine.
Another 1 hour, ma'am. 1.5 hour. Finish by 6. 6:30.
Good. Are you complete? ಇದಲ್ಲ? sum up ಮಾಡ್ಬೇಕಾಯ್ತು. ಇದಲ್ಲ? Okay. You go ahead with, we have another 13 poster presentations to go on. Let us not waste our time, so that we finish it as early as possible. Right? Okay. Next. 13, 13.
Yeah.
Wait, Mayank. Wait.
No. And these are my teammates.
Okay.
Today we are from the Vijay Teaching School. So the aim of our project is to find and document the types of plants present in the Vijay Teaching. So the major classification of plants in Vijay Teaching are herbs, shrubs, trees, flowers, and creepers.
So let's talk about herbs. We found many herbs in our schools. So what is a herb? Herbs are small plants which are bushy, they are soft stem and they have a very short lifespan. In our school we found many, for example, Simibanekai in Kannada, or in English it's Sayori, Podium Paragidum, Petrus, Ponato is also Solanum, Tuberosum, and also Rio.
So I will be continuing on with the shrubs we found. So we can see the garden croton, also called as the Sodium verangitum. The sorrel, because of the Copernica species. Then the morning glories, also called as the Calistraceous sepium in the scientific language. And the purple leaf, yucca, rose, frangipani, and the garden croton. Yucca is mentioned without the second word because we could not name it 6 or 7 categories.
Now let's talk about trees. Trees are usually very huge and enormous with a large trunk which is hard and gooey. In our schools, we were able to find traveler's palm, fig tree, which is also Ficus racemosa, mango tree, and coconut. And more of it, which is teak plant, sapota, and ashoka tree. Please move to comment for now. And in our school, we were able to find many common fauna.
Here we can observe some relationship among organisms. Here we can see lichens, which are a symbiotic relationship between algae and fungi. Then Monarchus, which is a partially parasitic plant, which we have found as a supportive tree. And then here we can see a Nenuphar flower, which is getting pollinated by the bee.
So let's talk about creepers. Creepers are a plant that is actually on the ground. They have a weak stem which is long and thin and they're very vulnerable. So here are the creepers in our school. We found the fiber petal, morning glories, which is also Calistraceous sepium. So let's go to climbers. Okay. So what are climbers? Climbers are also weak and fragile. To grow, they need any support. For example, let's take a tree. These grow on trees with support. We found climbers, which is the flame vine and the green heart vine.
I'll be continuing with the medicinal plants. These are some of the significant ones, such as the aloe vera, mango, and bamboo, respectively having the medicinal property of soothing burns and being used as dentifrices and stimulating the appetite, preventing constipation.
So what are xerophytes? Xerophytes are plants which actually are able to contain and manage their lives in dry areas with very little water. In our school, we were able to grow cactus, which is also cactus.
So any plants that grow on any dead decaying organic matter known as fungi, then we can see the orange peel fungus.
So these are the references, and we thank our school management, the school, other teachers for giving this opportunity, and ISC for giving us this opportunity. Thank you.
Next presentation, Sanvi Das and group from VJ Christian. I would like to tell you one thing. If you want your poster to be displayed downstairs,
After your presentation, please submit the poster to the table. Then in the management will all the posters download. Okay?
Good evening, everyone. Today, Nandini Singh, Sanvi Das, and I, Shreya Balwadi, of Class 11 from BGS National Public School, will be presenting on the topic Observational Studies on Toad Metamorphosis under Temperature Variation. So the study we did was on the species Dattaphragmus melanostictus, commonly known as the Asian common toad.
So we took home tadpoles from our school pond and we divided them into two groups. One group was placed outside the house where it was relatively colder compared to the group placed inside. And we hypothesized that the group inside and outside would, have not much of a difference in terms of the growth due to the temperature difference not being that high. But what we found out was that the group inside, where it was relatively warmer, metamorphized faster, and we got quite a few toadlets from that as well. And all of the conditions for the tadpoles in the tanks were kept constant. And so what we can conclude from this is that you can see that temperature plays a very important factor in the production of pyroxene in tadpoles. And the availability of water is a very important case in the life cycle and the reproductive cycle of toads and frogs. And climate change is a very big threat to this, and the pollution of water bodies also affects it.
So to begin with, the climatic changes because of noticeable rise in the average temperatures, due to which the optimal body temperatures of the cold-blooded organisms change. This causes a disruption in their hibernation cycle and their breeding seasons. Their breeding cycles are triggered earlier than it is supposed to be, which causes their offspring to face the harsher climatic conditions to survive in. The toads and frogs also have a transdermal uptake system, which makes them sensitive to changes, even the slightest changes in their environment. Considering that they're water breeders, they avoid breeding in polluted water bodies, which causes a decline in the aneirin species around the water bodies, which also indicates the decline in environment and water qualities. A study has shown that higher temperature causes faster metamorphosis and development, but shorter life periods. So hydroperiod refers to the timing of water availability. Water retention is particularly important in lakes, ponds, and other water streams and other water bodies for the breeding of these amphibians. Many tanks are already experiencing early drying of their habitats with mass mortality of eggs, tadpoles, and other metamorphosing animals. So with lesser toads, the insect and pest population will increase, and this can be harmful for the crop fields and farms. Not only this, toads and frogs are assumed to have temperature-dependent sex determination. So this can change the male-female sex ratio, and the ecological balance will not be maintained. So toads and other amphibians like frogs and salamanders, they are basically indicator species. So they are extremely sensitive to climate change and even the slightest changes in the environment, as we have mentioned before. So thus, they can give us a better insight on the functioning of the ecosystem. Therefore, it is very important for us to save these water bodies, and it is our duty to save these water bodies for the survival of these organisms, and also for our benefit and our health. So those are our references. And for the acknowledgements, first of all, we would like to thank Professor S.K. Datta for his valuable time and inputs. Then we would like to thank our school and mentor who gave us a platform to conceptualize our ideas. We would also like to thank IISc for this wonderful lake conference, which promotes knowledge and conservation and gives us opportunity to contribute to helping our environment. Thank you.
These are some pictures that we took while doing the experiment. And actually, yeah, that's a toadlet. And this is a tadpole which got legs.
Yeah, got both the legs.
Yeah,
hind
legs
legs
and
and
yeah.
it was 2 months. It took us 2 months.
Almost 3 if you include December.
Yeah, almost 3 if you include December. But then during the start of December, the Bangalore temperatures suddenly got very cold. And so most of our tadpoles that were kept outside started dying out. So we didn't want, more deaths, so we released them all. Huh? It's because of the temperature. Because if you can see, we have a capacity.
So if you do 2 years of research, I think your conclusion will write a 50 pages.
Yes.
Thank you, ma'am.
Shreya Bharat and he is from Mona Community.
Greetings to everyone. I am Rainey. I am Shreya Bharat. I am Shreya Sandur. So today we will be presenting about millipedes of IISc. We were very fascinated by these millipedes and we used this opportunity to do more research.
So this is the morphology of millipedes. The millipede over here, it starts with the head. The first segment, it starts with the antennae. Sorry, it has 4 segments, many segments, and the antennae is used by the millipedes to sense its surroundings. The next, which is the first segment of the millipede, is called as a column. The column, if we see here, has many dots. They are the eyes of the millipedes. They are compound eyes, also known as ocelli. Later on, we can see all the black and red segments, its legs, etc. And as we go on, in the end, the new legs, we can observe new legs.
The most intriguing thing about millipedes is their abundance of leg and the way they handle so many legs. So it's called the metachronal motion and it's so coordinated that it appears like a wave. 6 legs are there in each wave and as the waves approach the head, it reduces to 4 legs per wave. Then we also calculated the distance between each wave and we noticed that it decreases as the waves approach the head. So, yeah.
So, in consumption, we try to feed them fresh leaves and fresh berries. They ignored it, but then the same millipede we found it eating dead moss. Through verification, we concluded that they eat the dead moss. That's why they are called dead herbivores. Their excretion can be seen there, and its defense. Their skins, their exoskeleton texture is smooth and glossy. It curls up into a spiral to when sense danger. One more defense is when parrot releases some yellow liquid, which contains toxic acid.
So this is the mating of the millipede, and they are intertwined with each other. And when we were observing them, we saw another millipede approaching another millipede, and it was trying to go on top of the other millipede. However, I think they were attempting to mate, but then they were not. And the female millipedes are thicker in weight.
How can we distinguish male and female? The seventh segment in male millipedes doesn't have any legs. It has modified legs called gonopods. They are a genital and they transfer the spermatophore to the female. Now, what is the most vital organ in millipedes? We can see the naturally dead specimen of the millipede here, and the innards were hollow and were eaten away by germs. And the head was not there, but the body was fine and it was dead. When the car crushed the millipede in the picture shown beside, the body was damaged, but the head was fine. In this one, it was alive. So we can conclude that the head is the most vital organ of the millipede. The role in the ecosystem is that it plays a very it completes the carbon and nitrogen cycle. We'd like to thank Vijula Lana, Raghuram Anna, and TVR sir. And we were very fascinated by millipedes. Thank you so much.
Should we explain it in Kannada?
Next, Manasa and Nivedita from BG's National College.
Is it yours? Yeah. Okay. You want to zoom in? Yeah. I can use the pointer if you want to. Okay.
Good evening to one and all present here. Today, my friend Manasa and I, Nivedita from BGS MPS, will be presenting about our research, which is study of butterflies in our school, which is BGS MPS. So, introduction. We all know butterflies are winged insects. They belong to the order Lepidoptera, and they play a very important role in the ecosystem. They are both keystone species and an indicator species. The life cycle has 4 stages, as we all know, and each butterfly sticks to one particular species of plants to lay their eggs, and these species or this plant will be known as the host plant. So the objective of our study was to study the habits and life cycle of butterflies, and then the study of population and species of butterflies in the school, to study the host plants in the school, and to increase the population of butterflies. Now, we divided our study area into 3 with varying conditions such as noise levels, number of host plants, and the health condition of every host plant. The materials we used were the following, and the methods were simply walking, observing, and photography. So we counted every egg, caterpillar, host plant available. And we documented them. So the first part of our project was to rare caterpillars to study their life cycle. So we selected Corvidae and Tailjae as our subjects because we had a large number of champak trees. Here we had 2 samples, and each sample had 14 butterflies.
And the tables show the dates where we, like, the instars where we found them and the dates which they went into the next instar, the day they pupated and the day the day they came out of the pupa. And this table shows the comparison between the time taken for each instar, by sample 1, sample 2, and the average time it takes in a while. So we saw that it takes more time when we are raised in an artificial environment because it's not because of varying temperature mostly. So the next part of our project was to document the butterflies we found in our school. So we found 5 families, and in total we found 17 butterflies. Some of these include common merman, common castor, bellied eggfly, and common sailor. So we identified these butterflies over a period of 4 days at different times of the day. So this graph shows the amount of butterflies we found in each day, with each color representing one day. So this is the life cycle of common jay. This is the life cycle of common jay, and all the pictures were taken when we were rearing the caterpillars at home. The next part is reviewing host plants in school. So our school has a variety of host plants, as you can see, but a very few of them actually, have caterpillars and are good host plants. So we studied this across all the 3 areas, and in every area we basically took the species that were available, the number of species, the butterfly species that were supposed to be on it, the ones that we found, and why, and the reason as to why the species were there or why they were absent. And as you can see, study area 1 has the highest amount, where both champak and lemon, have eggs on them, spotted eggs on them. And we shortened this to the reason as to why this is study area 1 is an ideal environment. It has low noise levels. It has a high number of host plants. As you can see, champak has 17. And the human activity there is very less. Whereas in study area 2 and 3, the human activity is basically a lot because they're basically playgrounds, so there's a lot of noise, and it's not ideal for butterflies. We also did a comparative study of champak in study area 1 and 3, and as 1 is more ideal, we found more caterpillars, as you can see here. So the next part is introducing host plants in school. We introduced Bryophyllum on the day given, and we hope to increase the number, to plant more, so we can, try inviting another species of butterfly. The conclusion of this is the data shows that the population of butterflies increase as the number or health of host plants increase, and they decrease if the number or if the number decreases or if the host plants are unhealthy. And we can also know that noise pollution has an adverse effect on the butterfly population, as we can see in the comparative study between study area 1 and 3, where study area 1 was more ideal. So what we did was just the beginning of our project, and we would like to increase the population, the number of butterflies in our school by introducing new host plants and more nectar plants, and also we'd like to introduce caterpillars into our school project. And first of all, I'd like to thank IISC for conducting this state conference. And next, I would like to thank our principal ma'am and our academic director, sir. Then I'd like to thank Rajeshri ma'am and Chaturved sir for guiding us throughout this project.
Next, Yukta from Freedom International.
Forward, back, point. Okay. And start.
Good evening to one and all present here. I'm Yukta from Freedom International School, and I'll be talking about CRISPR is the gene editing system and how we can use it in the environment. So what is CRISPR? So when invasive species such as viruses, specifically bacteriophages, attack bacteria, part of the DNA is transferred to this bacteria. This bacteria is saved by this DNA is saved by the bacteria, and further through evolution, it makes a kind of defense system which can be which is helpful for the bacteria to fight against these viruses. Now, the catch here is in recent research, scientists have found ways to manipulate this system and make it perform various functions that they desire. And the Nobel Prize for this was given to Emmanuel Charpentier and Jennifer Doudna. So how does this CRISPR system actually work? So when the virus DNA, the target DNA, enters the bacteria, it detects the presence of it. And when it does so, it produces two RNA, one of which is complementary to the DNA of the target invasive species. So this RNA, called as the guide RNA, which is complementary, along with the Cas9 protein, which is an endonuclease, which means that it's able to pack the DNA molecules, form a system together, and are able to cut the DNA and form mutations. This is what disables the DNA, and further it can be replaced with healthy DNA and make it perform what we want it to do. So how can we use this in environment? So the field which I think it will mostly grow is in biofuels. Biofuels in recent years have been really circulating around, and the most famous one thing now is the algae one. Now, the major problem with algae biofuels is the cost it takes to produce. Like the heat is less, but it's also very expensive to produce the amount of biofuels. So how does CRISPR help in this? So with the help of CRISPR, we can coat algae in such a way that the lipid content in it will increase. What's the use of this increased lipid content? When the lipid content of the algae increases, it promotes more amount of biofuels to be produced. So when the amount of biofuels increases, the expense of producing the same amount of biofuels increases, which makes it economical. The second one in agriculture. Now, we've all heard about GMOs and everything till now. But the difference with CRISPR is that the DNA which is in GMO, DNA is introduced from outside species. Like it might be of the same genus, but a different species, which is very controversial in these days. But with the help of CRISPR, we can just manipulate the DNA within the seeds to produce more yield, to be more sensitive to fertilizer, so we can use less fertilizer, which decreases the soil pollution, and also require less amount of water. Further, CRISPR is also well developed in medicine, so it can be used to fight bacterial infections in humans. But this technology can also be adapted for plants, so it can so we can also use to fight like bacterial wilt and also the blight disease. Further research. Now, this is a very theoretical concept, and there's still a lot of research to be done, and it's a very sensitive matter, and it should be done with a lot of precaution. But we can also revive extinct genes. And how is this gene useful to us? Like, if we do so, how is it helpful to us? We can unlock a new set of genes and also find new applications for it, and find new ways to help us. So the disadvantages of it. So this mutation can always be good or bad, and it's sometimes unpredictable. So it can be bad mutation and also harm us. So I said we should always be precautious with it and be very sensitive in this matter. There are also ethical concerns regarding this on the research aspect of CRISPR, which is also what's taking a long time for this to develop. There's also genomic instability in editing, and also even if you produce perfect seedlings and crops for agriculture, if it's not accessible to all farmers and not and then it would be a waste of time and also resources. And there are also research constraints. In conclusion, I think CRISPR can either be a blessing or to us, but we should use it carefully. Thank you.
You said
new genes are introduced from other species. And here it is written. I'm not talking about mutation. Introduction of new character and desirable characters that could be produced, multiplied, thereby increasing the production or whatever, wherever you are introducing the technology. So you said in GMOs it is from a different species. It is how will you quantify if it is from a different species? As long as you get the desired, production and whatever. That is one thing. And why did you took up this topic? No?
If you survey people around you, if you ask them which recipe you want to buy, GMO or the non-GMO one,
It's the mindset. The mindset, no scientific basis for that. Okay, by eating GMO food or anything, nothing will happen. As long as the scientific basis or scientific inputs are concerned. Okay. Mutations are there also and here also. What is the advantage of using this technology over the previous? That's it.
this one, with the DNA from other species, I still not very good things. But this can cause them a lot of damage.
With limited things, so many things have happened, right?
For according to
Why you are interested in this? That's the
growing problem, we always see more ways to solve it. And I think problem also need like we need to adopt other methods to deal with such problems. And I feel especially biology has said that it is a very long way. Even though biology has been also a lot of biology problems, this is as an addition to school.
just for a topic. It takes a long way. By the time new technology
Poonakamati Meghavarma.
Good evening, everyone. My name is Meghavarna, and today I have made my hobby has been bird watching since I was very small. So when I got introduced to the IISc Lake Conference, I thought why not present about birds found in my area, which is very less explored in bird diversity. So the main objective of my study is to document the birds which are found in my area, and to analyze the environmental condition in the area, as birds are very good bioindicators, and to analyze the behavior of some species of birds which I observed. So this is my study area, Girinagar, which is located in South Bangalore, pin code 560085, and study period is from April to December 2022. The vegetation in my area can be described as urban residential vegetation with small gardens and potted plants interspersed with open plots that have ignored growth of weeds and other things. So what I observed, mainly three species of birds, I observed a special behavior. So in the common tailor bird, it has a very unique way of hunting insects in the air. And it is very relatively common tailor bird usually hunts for insects in trees and other bushes. But in the air, it is very less compared to that activity. So when it hunts insects in the air, it flies behind the insect. It goes behind the insect. When the distance is almost minimal, it picks up its speed by with the help of its wings, closes it so it becomes streamlined in shape and so catches the insect. When it catches the insect, it does go a bit down because as it has closed its wings. So, but then too, immediately after biting onto the insect and catching the insect, it starts the flutter of its wings and goes to a nearby place and eats the insect. And next is the red-whiskered bulbul. I observed a interesting behavior in the red-whiskered bulbul when it wants to, in large trees, when it wants to move to the branch above it, it jumps one time and with one flutter of its wings, it moves to the branch above it. And the rose-ringed parakeet is very special in eating fruits like mango, which I have captured here. So what this rose-ringed parakeet does is it clings upside down to the stem and eats all the flesh of the mango, leaves only the seed hanging in the tree. So here you can see in this picture which I have captured, the rose-ringed parakeet is upside down. So these are the other birds which I observed in around my house and in my area. And this is the graph which depicts the height of the bird which I saw them. So you can see the above area where the coconut trees and all, there the black kites and mynas and shikra was found. And in this height where the the length of the tree is there, they're cinerested, flower pecker, and parakeet kind of birds. Here, bulbul, barbet, and tailor bird, and this is the area which I found sunbird and tailor bird. So tailor bird moved in this range on the basis of what I observed. So they were in this height. And the other grain-eating bird like pigeon and all who feed who are ground feeders, they were found near the ground, and crows and bulbuls. So from my study, I can say that the number of bird species were greatest in the morning. They reduced slightly in the afternoon and increased in the evening, but not as much as in the morning. So and small birds in size, they are more active than larger birds. My references are eBird, Birds of the Indian Subcontinent, and to identify bird calls, Merlin app. Acknowledgement for Purnapura. I would really like to thank my school Purnapravati, ISC Lake team, and Ullas Anand who helped me in designing my study and how explaining how to explain, and Raghuram who helped me with the. Thank you.
In Girinagar, where did you observe? Where did you do your observation? If I look at the mango tree, then it is maybe probably closer to your house or within your house compound. Right? Where did you observe all the other ones?
Most of them and most of the birds are in my house because I live there, and I did travel nearby. There is a hill. The map is not visible here. There is a hill and a park nearby. So I found it the
Where did you find the red whiskered bulbul?
bulbul.
In your house? In your house or on the hill?
I didn't find them on the hill. I found them in my house and the park.
Park.
Next, Devashri Yashaswini from Vidyapur.
Good evening to one and all present here. Myself, I am Shrike, with my friend Yashaswini P from Vidyanjali Primary and High. We thank one and all present here for giving us an opportunity to present our study observation and documentation of butterflies in Weaver's colony. Butterflies are the most beautiful and colorful creatures on the earth. The development of butterflies is linked to the evolution of flowering plants. Butterflies also indicate the climate and environment change. Butterflies are dependent on specific plants for food. Thus, the diversity of the butterflies reflects plant diversity in our area. We had a group of 10 members decided to do observation and documentation of butterflies in Weaver's colony. Our study area of 10 places, which are listed, which belongs to Weaver's colony, Bangalore South, Kotigere Coast, Bannerghatta Road, Bangalore 560083 with 2 kilometers square area. Our study sites were Gate Park near Forest Office, 9th Cross South near Shiva Temple, 13th Cross near Ramlingeshwara Temple. This is study map with various locations where we documented this study. We visited this study area every Saturday between 2 p.m. to 4 p.m. and every Sunday between 9 a.m. to 11 a.m. in the month of August to November 2022. Randomly inspecting the study area, identifying the butterflies through visual methods, and with the help of iNaturalist app, and also taking pictures through smartphones provided by our parents. Totally 55 species of butterflies were identified in all the study months. In Gate Park, we could identify all the 4 months. In 9th Cross, which is more butterflies because there is more vegetation. This was the checklist of butterflies which we documented. Common cerulean, Semites seleno, it belongs to family Blues. Common firing, Iptima ballatus, it belongs to family Brush-footed. And finally, Spotted grass yellow, Ulema lata, it belongs to family yellow and whites. The present study reveals that we found 55 species of butterflies. In that, common firing was more in Weaver's colony and common windmill was the rarest butterfly. The highest diversity is in Shiva Temple and the lowest diversity is in 4th Cross. This is crimson rose tip butterfly, common emergent butterfly, Gemides seleno butterfly, common bushfrog, common jezebel, Danny fly, common silver fly, common palm fly, dark blue tiger butterfly. We thank our HM guide teacher for allowing to conduct this study. We also thank all our 10 members who were in various locations for observing and documenting this study. We thank Energy and Wetland Research Group, Center for Ecology Science, Indian Institute of Science, Bangalore, Sri Vijula sir of CEE, and Sri Cheturaj Shet sir for guiding. And we thank for your opportunity. Reference: Arish Bhatt et al. 2015, A brochure of Eyes on Nature, published by Karnataka State for Science, Council and Technology, Butterfly Diversity of Bangalore Urban District by Chaturved Shettar, iNaturalist app for identifying name, for scientific name, we record Wikipedia.
Temple premises has got more trees and plants. What type of trees and plants are there? Because so many butterflies are there. More grass. Okay.
Next, Kavya Iqbal, Cornwall,
Should be facing towards the side of my camera is here. You can see and read, but just make some give you. Scroll down. Want zoomed up. Check करना. Okay. Still là. Again start.
To one and all present here. I am Sadhya M.G. Chaitanya. We are completing English school and studying in 9th standard. We are here to talk about needs of KK school. Unplanned urbanization has led to many problems like loss of flora, internal biodiversity, and has also led to traffic congestion. Most of all the plant varieties in one third are at the verge of extinction. Objective of our study is to understand the different species of plants, conserve biodiversity, and to know the importance of conserving biodiversity. We all know how important are plants to us. They are a primary source of food for all the living organisms. They absorb carbon dioxide and release oxygen that is important for other organisms for cellular respiration, hence maintains gaseous balance, and also helps in carbon sequestration. They prevent soil erosion and help in maintaining water cycle. Many medicines are made directly or indirectly from plants. A single tree provides food and shelter to many species. Plants are important in retaining good soil conditions and maintaining atmospheric temperature, as we all know that. This is our school in 2002, and now this itself shows how green we are. We have 5 acres of land, out of which 3 acres is covered with greenery. But when our school started, it was totally a eucalyptus grove. Now we have 550 trees and all are planted by our seniors. We have rich biodiversity. We have birds, butterflies, and spiders. But the story is different in Vattur. Vattur had many trees, but now we can count the number of trees in our fingers. And we also found that there are trees only in 2 areas. That is Vattur Government Hospital and Government High School. We wanted to know about the trees in our school, so we started numbering them. We knew about their importance, learned their scientific name, and we are going to fix nameplates so others will also learn. This list shows the trees, the scientific name, family, and trees, number of trees present in our school. We have maximum Foster trees. We also have 5 ficus trees. We have many medicinal plants, and the leaves of National fruit, Vigorosia, and Himaruga are used in the treatment of cancer. We also have some rare plants like cinnamon, butterfly, silkworm, we also have 25 different citrus plants. We have red sandalwood and cane hyacinth plants that have come to our school during Lake Symposium from CC. We have perennial plant, coconut plant, tree. Yeah, and I want to share about a pongamia tree in our school. Normally, pongamia trees would grow bushy. I mean, not in height, but width. But this pongamia tree in our school is taller than our ground floor plus two-story building. Yeah, this photo shows the visit of 150 students from RV school to our school. They were mesmerized by seeing the plants, solar panels, water harvesting, and other green initiatives that we have adopted in our school. It was surprising that one of the students didn't even know which is a mango tree. Two other government schools had also visited our school in the month of October. So, by conserving the biodiversity, not only are we benefited, students from other schools are also benefited. And our school premises is much cooler than other parts of Vattur. We have a borewell of 600 feet, but we used to buy water in summer in the early years of our school. Now we have water year through, even during summer, only because of plants and rainwater harvesting. Let's conclude by saying, plant more saplings and children protect Mother Nature. Thank you.
Thank you. We move to next. Arnav from the company.
Okay.
Today 5.5 acres and the forest is more than 140 acres. And agriculture land is 100 acres. So the Andhra is biodiversity and the Andhra is in Puttur Sagal in a national reserve and Puttur Valley. My main objective is to assess the biodiversity of Andhra and differentiate between flowering, fruiting and medicinal plants, medicinal trees of Andhra. And to serve the Samskrita and Punayani, and to assess the unusual list of documented plants, and to document medicinal part of plant and disease cured by Redon plant, and to differentiate between herbs, shrubs, and trees. So, as the my food objective, ISL in this, so Anna has totally the least concern is a 21, and data definition was 1, and not evaluated one remaining. So the 30% is misconcern and 66% are not evaluated and 3% on data deficient. And the flora diversity was flowering, fruiting, and medicinal. So flowering were 5, which means 9%, and fruiting were 24, which means 47%, and 22 medicinal plants, which means 43%. And the trees were 28 and herbs were 7 and shrubs were 10. So shrubs were 37%, herbs were 20%, and trees were 52. And those are the thickness. So total species found are 51 and genus were 48, 3 ficus species are there, and 24 floating plants were there, and 5 flowering plants were there, and 22 medicinal plants were there, and 21 leaf concern plants were there, and 28 trees, and 20 shrubs, and 7 herbs. So, I sincerely thank the Ravana for helping me in this project, and late 2022 team for giving me stage for doing presenting my presentation, and Korea Community team for giving me a wonderful ceremony. nuisance and reduce and reduce website, and FLHT for Sanskrit and Kalamanji, and Planet Ayurveda for some Ayurveda information.
Everything was done by yourself.
Yes.
Everything was done.
No.
How long it took?
It took nearly 2 months. I surveyed for 10 days and I searched some information in internet and it took me 2 to 3 weeks for it. After everything, I presented to Vijayalananda and Raghuravanda for correction. They helped me. Thank you.
Good morning to everyone present. Good evening to everyone present here. Myself, Lavanya. We are studying our topic is diversity. We study about the diversity in ecosystem of Venkatagiri. Venkatagiri Lake is situated near the region of Peninsula, Pathanamthitta, in Kosovo. Venkatagiri Lake has an area of about 8 to 10 acres. Venkatagiri Lake behaves as a home for many aquatic creatures, tiny creatures, and many other plants, birds, etc. Venkatagiri Lake's perimeter is approximately 1.185 km. Its latitude is 77 degrees 39 minutes 40 seconds east. Longitude 12 degrees 52 minutes 42 seconds north. Objectives to study about the diversity and ecosystem of Venkatagiri Lake, to analyze birds present there, to analyze their habitat and features, to analyze plants growing in the lake, to analyze changes in Venkatagiri Lake. We have analyzed totally 8 types of birds. Bird analysis. We have the Great Red Champagne. It is in purple bluish color. They have red beak. Indian pond heron. Scientific name Ardiola ralliodis. They have brownish red color on their upper body. The Eurasian foot. Scientific name Pulcatra. They have white beak with white frontal sheath. The Cathalygate. Scientific name Buculcus ibis. They have yellow beak. Green sandpiper. Scientific name Pinka crocus. They have white belly and greenish brown beak. The red wattle lapwing, they are also known as Asian lapwing. They have red beak with black teeth. The black-headed ibis, they are also known as Indian white ibis. They have black long beaks and black head and neck. The black-winged kite, they are also known as they have owl-like forward-facing eyes.
Plant analysis. We have analyzed 5 types of plants in Venkatagiri Lake. Contadarian grasses, scientific name Ichornia grasses. Paragrass, scientific name Ichornia indica. Polygonum glacial, scientific name Polygonium. Ammophila breviligulata. Fifth one, water granule, scientific name Crasula helsi. Snakes. We have analyzed one type of snake in Venkatagiri Lake, that is the checkered cat. The checkered cat is medium-sized non-venomous snake. Coloration is very variable. It consists of black spots. Insects. We have analyzed one type of insect in Venkatagiri Lake, that is water spider. Scientific name Neridium. Water
ladder is small insects which can walk on top of water by the hydroponics. Then, while going around the lake, we used to get bad smell of sanitary waste. Then we inquired some people present there. Then we got to know that the few buildings are directly connected with underground connection. Then we raised a complaint to our ward corporator. Then we got to know that a few buildings are directly connected with underground connection. They ensured us they will work on the betterment of lake and they will take an action on it. Conclusion. We have for this project, we have gained so much of knowledge. By this, we have studied speeches of 8 birds, 5 plants, 1 insect, and 1 snake. And to our problem, BBMP people ensured that they will work on the betterment of it. Acknowledgement. We thank our principal and administrators and our teacher of our school who helped us and guided us for the successful and supported for the project. Our special thanks to our guided by Ma'am and Sonia Ma'am and our friend Deepika and our parents. We wholeheartedly thank for IASC team for giving an opportunity to work on this project. Use Google Lens for identifying birds and plants. iNaturalist page, eBird organization, Wikipedia. Thank you. Next, Vikas and Murali from Counseling and
PDF na comparative study of soil na PPT ಕಳಿಸಿದ್ರಾ ಇದೊಂದೇನಾ ಇಲ್ಲಿ ನೋಡಿ ಇಲ್ಲಿದೆ ಕೆಳಗಡೆ ಈ ಕಡೆ ಕ್ಯಾಮ್ರಾ ಆನ್ ಇದೆ ಈ ಕಡೆ ನೋಡಿ ಇದಲ್ಲ ಬಂದು ಆಯ್ತು
I am Vikas from grade 9. We are from Vagdevila School, Martal.
Today we are going to discuss about soil analysis. Soil analysis is a chemical process which determines the available nutrients in the soil. It is a valuable tool for agriculture and also increases the economic production. After doing the test, we can ensure that there is a proper nutrients and fertilizers present in the soil, which is required in the vegetation.
We did analyzing the study of soil from different places. As we done, we collected the soil from 5 different places: Martali, Kadbisnalli, HL, Dodnakundi, and Madhepura. Initially, Bengaluru was called as Garden City, and now it is called as Silicon City. To retain the name as Garden City, we are finding a better soil for good ecosystem.
So our study area is Martali, Kadbisnalli, HL, Dodnakundi, and Madhepura. Here are the pictures we are collecting the different types of soil from different places.
The soil which we tested from, which we collected from different places, and we are testing the soil in laboratory. And here are the samples of that image. And we also did nitrogen, phosphorus, and potassium test. And we done pH value of that test. To do nitrogen test, we took 5 cubic centimeter of soil and added 25 ml of nitrogen reagent 1 and mixed it for 5 to 10 minutes. After that, we added decolorizer to that and added nitrogen reagent 2. After that, seeing the decolorizer chart, nitrogen chart, we fixed the value. According to the pH value chart, Dodnakundi has a pH value of 7, where it's neutral in nature. Microbes will develop, and nitrogen content should be high. The Madhepura soil has pH value of 8, which is light neutral, and nitrogen content is high where microbes will develop, and phosphorus is low where phosphorus is high means they do not allow other essential nutrients like zinc and copper to enter, so it should be low, and potassium is medium. If it is high, it did not allow to get root will not develop there, so it should be low or medium. So Madhepura has better soil for good ecosystem. Problem being faced due to the rapid urbanization, the concrete jungles has made the soil suffer from toxic elements and degradation. We also need microorganisms for complete decaying of matter and making the soil more fertile and keeping the soil nutrients. Due to the recent floods, the topsoil was eroded and has been polluted by plastics and toxic metals, and these are the factors which affects the soil fertility.
As the problem set by Dika, we have some solutions. We must grow different types of plants to stop the soil erosion. Adapting the tilling practices help the soil to regain the nutrients. We can produce homemade compost by decomposing kitchen and garden waste, and segregating of waste and developing proper waste management can decrease the degradation of soil. We must support the nearby people or projects who have taken up the initiatives to save our ecosystem, like UTMP workers and shopkeepers, etc.
These are the websites which we use for doing this project. I thank IISc for giving this wonderful opportunity to share our research work.
And we also thank Randelas institutions for giving us an opportunity to be a part of IISc.
I would extend our thanks to the entire R&D department which helped us to do this project.
Next, Rohita and Pavitra from PSBP Learning Leadership Academy.
Poster ಇಲ್ಲ ಒಂದು ಕೇಳಿಲ್ಲ. PPT, okay. Should stand here, okay.
So can you show the buttons?
Forward, back. This is the pointer. No.
Shri Gurubhyo Namaha. I am Rohita and this is my friend Pavitra, and we are here to present our research, which is Insights to Algae. Table of Contents, Introduction. Algae. Algae is a photosynthetic eukaryotic organism. It lacks cell organelles in cell tissues such as stomata, xylem, and phloem. And they are mostly unicellular, but multicellular algae is also found. Objectives. Our study objectives is to relate the growth of algae and the nutrients found in the polluted water bodies. Study area.
This is the first lake, which is the Chikatogo lake, and this is the second one, which is the Basavanapuram lake. We did a manual collection of the water sample, and we did a quantitative analysis of the sample. Further, we did microscopic images of the algae after a centrifuge, and we also identified the algae through the images. These are some of the experiments we did.
Result. These are the algae we identified in the Basavanapuram lake, and these are the algae we identified in the Chikatogo lake.
This is the result we got from the quantitative analysis, which we put in a graph. This is the analytical report from the lab. We found that Chikatogo Lake has a pH which is almost 7, and we found low presence of chlorides and magnesium and a high presence of calcium. It also has low COD and BOD and higher electrical conductivity. We found the predominant algae to be Urea.
In the analysis for the Basavanapuram lake, the pH is alkaline. We have high amounts of chlorides and magnesium, high amount of COD and BOD.
lower electrical conductivity, and low amount of chlorine present. And the predominant algae in the Baswantpura lake is Microcystis. Conclusion. The Baswantpura lake is more polluted than the Chikkatogur lake. We can see a large amount and more number of algal growth in the Baswantpura lake and more than in the Chikkatogur lake. Microcystis is the predominant algae found in the Baswantpura lake. This indicates the pollution in the Baswantpura lake as Microcystis produces a harmful toxin called the heptotoxin, which is harmful to the aquatic organisms living there. This also indicates the presence of iron, cobalt, and manganese because Microcystis thrive under the presence of these nutrients in the water. The second algae we found was the diatoms. Diatoms indicate the presence of silica for the formation of the cell, phosphates, and nitrogen. This also is a reliable indicator of eutrophication. The third algae we found is colapse. Colapse are found in the Baswantpura lake mainly because of the abundant growth of algae. Because colapse usually feed on algae present in the water bodies. The fourth algae we found is the Cynidesmus. Cynidesmus grows in an environment of high temperature, and the Baswantpura lake has 3 degrees Celsius more than Chikkatogur lake, and that's why we can see this growth in the Baswantpura lake. The fifth algae we found, which is common to the both lakes, Chikkatogur lake and the Baswantpura lake, is Cyclotella. This grows because it's an oligotrophic organism, and it also requires an alkaline environment. And as we can see, the Baswantpura lake is alkaline by 8.8, and the Chikkatogur lake is slightly alkaline by 7.4.
We found Euglena common to both lakes. However, we found more number of Euglena in the Chikkatogur lake compared to the Baswantpura lake. We also found multiple species of this genus. Further, the presence of Euglena and Oscillatoria indicates the high amount of organic pollution according to a research paper we referred. Similar conclusions can be taken from Cynidesmus, which also indicates organic pollution. These are the ways to overcome algal blooms. We would like to thank IISc for giving us this opportunity. We would also like to thank Cincy ma'am for help with identification of the algae. We did a lot of tests as Eureka Analytical Labs, and we would like to thank them also. Lastly, I would also like to thank our principals, teachers, and parents for the support and guidance. These are our references. Thank you.
From where you collected the water from different areas or only the one for water analysis?
We took samples thrice from the lake, and it was around similar places.
For the water analysis, we took from 2 places. One at the one corner of the lake and the opposite corner of the other lake. And the same lake. 3 times. Yeah, around the same place.
Yeah, it was a little different. It's not the exact same spot. We did a little different, but not at different corners.
So the water analysis, for water analysis, water and algae also use the same water sample or
Yeah.
Same
one.
Yeah.
Thank you.
Next, Janvi and Anna from the CSC school.
So according to the printer shop, it was AO side. Yeah. Good evening to everyone present. Good evening to everyone present here. My name is Ahana.
And my name is Jackie.
Switch off the mic.
Silence, please. Few more minutes. Please wait.
Keep it near.
Good evening to everyone present here. My name is Ahana.
And my name is Janvi.
And today we will be presenting our study on the habitat of Arikere Lake. The main reason why we chose this lake is actually because we live in its proximity and we have been able to watch how this lake has deteriorated over the years.
So the main objective of our study is to analyze the habitat and the water quality of the lake by visiting the lake, collecting the sample on rainy and rainless days, and comparing and testing the results. So our main study area was Arikere Lake, and according to RTC, the area of the lake is 37 acres and the perimeter is 3 kilometers. And this is a satellite image of Arikere Lake taken in 2022.
So to conduct our study, we visited the lake to get an idea of the surroundings, and we observed that the lake was almost completely covered with macrophytes, and floating water was visible only in the center of the lake. We also observed that sewage was draining directly into the lake through two inlets. There was a construction site right beside the lake, and the barricade around the lake was not well maintained, and there was a lot of garbage down there. So based on our observations, we decided to conduct some water sample tests on Arikere Lake, and we collected the water samples in bottles from the lake on both a rainy and rainless day, and then tested them using a water testing kit and assessed the water quality based on various parameters. These are our results on testing the water samples. We found pH to be slightly acidic on a rainy day as compared to a rainless day. However, nitrate, iron, and fluoride were all decreased on a rainy day, and nitrate was below permissible limits on both days. We also saw a significant increase in dissolved oxygen from 4 to 8 milligrams per liter on a rainy day, and turbidity, fecal pollution, and ammonia remained above the permissible limits and at the same level on both days. So based on our analysis of the habitat and our results, we were able to conclude that the presence of less nitrate basically shows us that excessive macrophyte growth has decreasing influence in this lake, which will explain its decrease. Rain may have further diluted nitrate levels, which is why we observed a decrease on a rainy day. Rain may have also diluted iron and fluoride levels because we observed a decrease in their levels as well. However, excessive iron on a rainless day could be due to construction activities currently ongoing beside the lake. High turbidity, fecal pollution, and ammonia shows human contamination and the impact of sewage draining directly into the lake. We also observed that pH was slightly acidic on a rainy day. We were also able to observe that during the COVID-19 pandemic, garbage dumping beside the lake had actually stopped, and the condition of the lake had improved in the absence of human intervention. However, after the pandemic and the lockdown, the lake has once again returned to a highly polluted state, which goes on to show us that humans are basically causing the pollution in this lake. So our final conclusion is that Arikere Lake is highly polluted and not well maintained, which is affecting the entire ecosystem and habitat. Rainy days do appear to improve the water quality with respect to dissolved oxygen, but it also dilutes nitrate, iron, and fluoride. It's essential to keep in mind that humans and their environment are very closely linked. When we humans pollute our environment, it's likely to have an adverse impact on us in the future, which is clearly seen in the case of Arikere Lake. Therefore, we must take proper steps to conserve Arikere Lake, and steps like banning garbage dumping beside the lake and constructing a proper barricade will go a long way in improving the water.
With this, we would like to conclude our study. Thank you.
Thank you. Thank you very much. 2 3 sections. First of all, a big thank you to all of you who have maintained the time since morning. It's on behalf of all 3 of us, just 4 or 5 of us are there. We are all very happy that we kept up the time. And many of you came back till the last presentation. That's a good indication. But tomorrow morning, we are going to assemble again. And tomorrow, right, for the award ceremony, my sincere request to all of you is, whether you get a prize or not, whether you are identified or not, you will remain in the auditorium till the program gets over. Nobody will get up in the middle and then go. We have asked some of our volunteers to keep a note of those children. By chance, if you get up and walk out of the auditorium, your school name will be noted, and probably you may not get an opportunity to come back again to INC, do your paper presentation. This is a strict instruction from TVS, sir. So tomorrow, don't ever think of getting up and going out of the auditorium before the program concludes. Please, some teachers have said that their school bus and school instructors, please tell your school authorities that till the program gets over tomorrow, they are not allowed to walk out of the auditorium. Please make a note. Some of your friends, if they have already left, they took my permission. You need to inform them also